by Henry Gray, Anatomy of the Human Body. Preface to the 20th edition. Since the publication of the first English edition of this work in 1858, and the first American edition in 1859, great advances in the subject of anatomy have been made, especially in microscopic anatomy and the anatomy of the embryo. This knowledge was embodied from time to time in the successive editions until finally considerable portions of the text, sometimes sections, were devoted to these subjects. However, the main text has always remained primarily a descriptive anatomy of the human body. In the present edition, the special sections on embryology and histology have been distributed among the subjects under which they naturally belong. New matter on physiological anatomy, laws of bone architecture, the mechanics and variations of muscles have been added, occupying much of the space formerly devoted to the sections on applied anatomy. The sections on the ductless glands and the nervous system have been largely rewritten. In the latter, a more rational presentation of the sympathetic nervous system has been achieved through the use of diagrams and descriptions based on physiological and pharmacological work. The central connections of the spinal and cranial nerves are also emphasized. Illustrations have been added wherever important points could be made more clear, and throughout the work colored pictures have been even more extensively used than heretofore. In this respect, special mention might be made of the central nervous system and the section on the muscles. In the section on syndesmology, 6 illustrations are used from Quain's Anatomy through the courtesy of the publishers, Messrs. Longmans, Green & Company of London. The use of the BNA nomenclature in English has been retained practically unchanged in this edition, and important references to the literature have been added at the end of each section. As a practical work on the subject for the student, Gray's Anatomy has always been recognized and appreciated. The plan originally formulated, which has proved so successful, has been adhered to as much as possible. It is interesting to note that although Henry Gray saw only the first edition, much of the original text persists, and many of his illustrations are still in use. Bearing this in mind, it has been the endeavor of the editor to supply only such changes as advances in the science made necessary in order that this work may reflect the latest accessions to anatomical knowledge. W. H. L. Baltimore, 1918. Introduction. The term human anatomy comprises a consideration of the various structures which make up the human organism. In a restricted sense, it deals merely with the parts which form the fully developed individual, and which can be rendered evident to the naked eye by various methods of dissection. Regarded from such a standpoint, it may be studied by 2 methods. 1. The various structures may be separately considered, systematic anatomy. Or, 2. The organs and tissues may be studied in relation to one another, topographical or regional anatomy. It is, however, of much advantage to add to the facts ascertained by naked eye dissection those obtained by the use of the microscope. This introduces 2 fields of investigation, that is, the study of the minute structure of the various component parts of the body, histology, and the study of the human organism in its immature condition, that is, the various stages of its intrauterine development from the fertilized ovum up to the period when it assumes an independent existence, embryology. Owing to the difficulty of obtaining material illustrating all the stages of this early development, gaps must be filled up by observations on the development of lower forms, comparative embryology, or by consideration of adult forms in the line of human ancestry, comparative anatomy. The direct application of the facts of human anatomy to the various pathological conditions which may occur constitutes the subject of applied anatomy. Finally, the appreciation of structures on or immediately underlying the surface of the body is frequently made the subject of special study, surface anatomy. Systematic anatomy. The various systems of which the human body is composed are grouped under the following headings. Osteology, the bony system or skeleton. Syndesmology, the articulations or joints. Myology, the muscles. With the description of the muscles, it is convenient to include that of the fascia, which are so intimately connected with them. Angiology, the vascular system, comprising the heart, blood vessels, lymphatic vessels, and lymph glands. Neurology, the nervous system. The organs of sense may be included in this system. Splanchnology, the visceral system. Topographically, the viscera form two groups, that is, the thoracic viscera and the abdomino-pelvic viscera. The heart, a thoracic viscus, is best considered with the vascular system. The rest of the viscera may be grouped according to their functions. A. The respiratory apparatus, B. The digestive apparatus, and C. The urogenital apparatus. Strictly speaking, the third subgroup should include only such components of the urogenital apparatus as are included within the abdomino-pelvic cavity, but it is convenient to study under this heading certain parts which lie in relation to the surface of the body, that is, the testes and the external organs of generation. For descriptive purposes, the body is supposed to be in the erect posture, with the arms hanging by the sides and the palms of the hands directed forward. The median plane is a vertical antero-posterior plane passing through the center of the trunk. This plane will pass approximately through the sagittal suture of the skull, and hence any plane parallel to it is termed a sagittal plane. A vertical plane at right angles to the median plane passes, roughly speaking, through the central part of the coronal suture, or through a line parallel to it. Such a plane is known as a frontal plane, or sometimes as a coronal plane. A plane at right angles to both the median and frontal planes is termed a transverse plane. The terms anterior or ventral and posterior or dorsal are employed to indicate the relation of parts to the front or back of the body or limbs, and the terms superior or cephalic and inferior or caudal to indicate the relative levels of different structures. Structures nearer to or farther from the median plane are referred to as medial or lateral, respectively. The terms superficial and deep are strictly confined to descriptions of the relative depth from the surface of the various structures. External and internal are reserved almost entirely for describing the walls of cavities or of hollow viscera. In the case of the limbs, the words proximal and distal refer to the relative distance from the attached end of the limb.
Section 1. Embryology. The term embryology in its widest sense is applied to the various changes which take place during the growth of an animal from the egg to the adult condition. It is, however, usually restricted to the phenomena which occur before birth. Embryology may be studied from two aspects. One, that of ontogeny, which deals only with the development of the individual, and two, that of phylogeny, which concerns itself with the evolutionary history of the animal kingdom. In vertebrate animals, the development of a new being can only take place when a female germ cell or ovum has been fertilized by a male germ cell or spermatozoon. The ovum is a nucleated cell, and all the complicated changes by which the various tissues and organs of the body are formed from it after it has been fertilized are the result of two general processes, viz., segmentation and differentiation of cells. Thus, the fertilized ovum undergoes repeated segmentation into a number of cells which at first closely resemble one another, but are, sooner or later, differentiated into two groups. One, somatic cells, the function of which is to build up the various tissues of the body, and two, germinal cells, which become embedded in the sexual glands, the ovaries in the female and the testes in the male, and are destined for the perpetuation of the species. Having regard to the main purpose of this work, it is impossible in the space available in this section to describe fully or illustrate adequately all the phenomena which occur in the different stages of the development of the human body. Only the principal facts are given, and the student is referred for further details to one or other of the textbooks on human embryology. 1. The animal cell. All the tissues and organs of the body originate from a microscopic structure (brackets, the fertilized ovum), close brackets, which consists of a soft jelly-like material enclosed in a membrane and containing a vesicle, or small spherical body, inside which are one or more denser spots. This may be regarded as a complete cell. All the solid tissues consist largely of cells essentially similar to it in nature, but differing in external form. In the higher organisms, a cell may be defined as a nucleated mass of protoplasm of microscopic size. Its two essentials, therefore, are a soft jelly-like material similar to that found in the ovum, and usually styled cytoplasm, and a small spherical body embedded in it, and termed a nucleus. Some of the unicellular protozoa contain no nuclei, but granular particles which, like true nuclei, stain with basic dyes. The other constituents of the ovum, viz., its limiting membrane and the denser spot contained in the nucleus, called the nucleolus, are not essential to the type cell, and in fact many cells exist without them. Cytoplasm (protoplasm) is a material probably of variable constitution during life, but yielding on its disintegration bodies chiefly of proteid nature. Lecithin and cholesterin are constantly found in it, as well as inorganic salts, chief among which are the phosphates and chlorides of potassium, sodium, and calcium. It is of a semi-fluid, viscid consistence, and probably colloidal in nature. The living cytoplasm appears to consist of a homogeneous and structureless ground substance, in which are embedded granules of various types. The mitochondria are the most constant type of granule, and vary in form from granules to rods and threads. Their function is unknown. Some of the granules are proteid in nature and probably essential constituents. Others are fat, glycogen, or pigment granules, and are regarded as adventitious material taken in from without, and hence are styled cell inclusions or paraplasm. When, however, cells have been fixed by reagents, a fibrillar or granular appearance can often be made out under a high power of the microscope. The fibrils are usually arranged in a network or reticulum, to which the term spongioplasm is applied, the clear substance in the meshes being termed hyaloplasm. The size and shape of the meshes of the spongioplasm vary in different cells, and in different parts of the same cell. The relative amounts of spongioplasm and hyaloplasm also vary in different cells, the latter preponderating in the young cell, and the former increasing at the expense of the hyaloplasm as the cell grows. Such appearances in fixed cells are no indication whatsoever of the existence of similar structures in the living, although there must have been something in the living cell to give rise to the fixed structures. The peripheral layer of a cell is in all cases modified either by the formation of a definite cell membrane, as in the ovum, or more frequently in the case of animal cells by a transformation, probably chemical in nature, which is only recognizable by the fact that the surface of the cell behaves as a semi-permeable membrane. Nucleus. The nucleus is a minute body embedded in the protoplasm, and usually of a spherical or oval form, its size having little relation to that of the cell. It is surrounded by a well-defined wall, the nuclear membrane. This encloses the nuclear substance (nuclear matrix), which is composed of a homogeneous material in which is usually embedded one or two nucleoli. In fixed cells, the nucleus seems to consist of a clear substance or karyoplasm, and a network or karyomytome. The former is probably of the same nature as the hyaloplasm of the cell, but the latter, which forms also the wall of the nucleus, differs from the spongioplasm of the cell substance. It consists of fibres or filaments arranged in a reticular manner. These filaments are composed of a homogeneous material known as linin, which stains with acid dyes, and contains embedded in its substance particles which have a strong affinity for basic dyes. These basophil granules have been named chromatin or basichromatin, and owe their staining properties to the presence of nucleic acid. Within the nuclear matrix are one or more highly refracting bodies termed nucleoli, connected with the nuclear membrane by the nuclear filaments. They are regarded as being of two kinds. Some are mere local condensations (net knots) of the chromatin. These are irregular in shape and are termed pseudonucleoli. Others are distinct bodies differing from the pseudonucleoli both in nature and chemical composition. They may be termed true nucleoli and are usually found in resting cells. The true nucleoli are oxyphil, i.e. they stain with acid dyes. Most living cells contain, in addition to their protoplasm and nucleus, a small particle which usually lies near the nucleus and is termed the centrosome. In the middle of the centrosome is a minute body called the centriole, and surrounding this is a clear spherical mass known as the centrosphere. The protoplasm surrounding the centrosphere is frequently arranged in radiating fibrillar rows of granules, forming what is termed the attraction sphere. Reproduction of cells. Reproduction of cells is effected either by direct or by indirect division. In reproduction by direct division, the nucleus becomes constricted in its centre, assuming an hourglass shape, and then divides into two. This is followed by a cleavage or division of the whole protoplasmic mass of the cell, and thus two daughter cells are formed, each containing a nucleus. These daughter cells are at first smaller than the original mother cell, but they grow and the process may be repeated in them, so that multiplication may take place rapidly. Indirect division, or karyokinesis (karyomitosis), has been observed in all the tissues: generative cells, epithelial tissue, connective tissue, muscular tissue, and nerve tissue. It is possible that cell division may always take place by the indirect method. The process of indirect cell division is characterized by a series of complex changes in the nucleus, leading to its subdivision. This is followed by cleavage of the cell protoplasm. Starting with the nucleus in the quiescent or resting stage, these changes may be briefly grouped under the four following phases. 1. Prophase. The nuclear network of chromatin filaments assumes the form of a twisted skein or spirum, while the nuclear membrane and nucleolus disappear. The convoluted skein of chromatin divides into a definite number of V-shaped segments or chromosomes. The number of chromosomes varies in different animals, but is constant for all the cells in an animal of any given species. In man, the number is given by Fleming and Duesberg as 24. Coincidentally with or preceding these changes, the centriole, which usually lies by the side of the nucleus, undergoes subdivision, and the two resulting centrioles, each surrounded by a centrosphere, are seen to be connected by a spindle of delicate achromatic fibres, the achromatic spindle. The centrioles move away from each other, one toward either extremity of the nucleus, and the fibrils of the achromatic spindle are correspondingly lengthened. A line encircling the spindle midway between its extremities or poles is named the equator, and around this the V-shaped chromosomes arrange themselves in the form of a star, thus constituting the mother star or monaster. 2. Metaphase. Each V-shaped chromosome now undergoes longitudinal cleavage into two equal parts or daughter chromosomes, the cleavage commencing at the apex of the V and extending along its divergent limbs. 3. Anaphase. The daughter chromosomes, thus separated, travel in opposite directions along the fibrils of the achromatic spindle toward the centrioles, around which they group themselves, and thus two star-like figures are formed, one at either pole of the achromatic spindle. This constitutes the diaster. The daughter chromosomes now arrange themselves into a skein or spirum, and eventually form the network of chromatin which is characteristic of the resting nucleus. 4. Telophase. The cell protoplasm begins to appear constricted around the equator of the achromatic spindle, where double rows of granules are also sometimes seen. The constriction deepens, and the original cell gradually becomes divided into two new cells, each with its own nucleus and centrosome, which assume the ordinary positions occupied by such structures in the resting stage. The nuclear membrane and nucleolus are also differentiated during this phase. Embryology. 2. The ovum. The ova are developed from the primitive germ cells which are embedded in the substance of the ovaries. Each primitive germ cell gives rise, by repeated divisions, to a number of smaller cells termed oogonia, from which the ova or primary oocytes are developed. Human ova are extremely minute, measuring about 0.2 millimeters in diameter, and are enclosed within the egg follicles of the ovaries. As a rule, each follicle contains a single ovum, but sometimes two or more are present. Footnote. See description of the ovary on a future page. End of footnote. By the enlargement and subsequent rupture of a follicle at the surface of the ovary, an ovum is liberated and conveyed by the uterine tube to the cavity of the uterus. Unless it be fertilized, it undergoes no further development and is discharged from the uterus. But if fertilization take place, it is retained within the uterus and is developed into a new being. In appearance and structure, the ovum differs little from an ordinary cell, but distinctive names have been applied to its several parts. Thus, the cell substance is known as the yolk or ooplasm, the nucleus as the germinal vesicle, and the nucleolus as the germinal spot. The ovum is enclosed within a thick, transparent envelope, the zona striata or zona pellucida, adhering to the outer surface of which are several layers of cells derived from those of the follicle, and collectively constituting the corona radiata. Yolk. The yolk comprises 1. The cytoplasm of the ordinary animal cell with its spongioplasm and hyaloplasm. This is frequently termed the formative yolk. 2. The nutritive yolk or deutoplasm, which consists of numerous rounded granules of fatty and albuminoid substances embedded in the cytoplasm. In the mammalian ovum, the nutritive yolk is extremely small in amount, and is of service in nourishing the embryo in the early stages of its development only, whereas in the egg of the bird there is sufficient to supply the chick with nutriment throughout the whole period of incubation. The nutritive yolk not only varies in amount, but in its mode of distribution within the egg. Thus, in some animals it is almost uniformly distributed throughout the cytoplasm. In some it is centrally placed and is surrounded by the cytoplasm. In others it is accumulated at the lower pole of the ovum, while the cytoplasm occupies the upper pole. A centrosome and centriole are present and lie in the immediate neighbourhood of the nucleus. Germinal vesicle. The germinal vesicle, or nucleus, is a large spherical body which at first occupies a nearly central position, but becomes eccentric as the growth of the ovum proceeds. Its structure is that of an ordinary cell nucleus, viz. It consists of a reticulum or karyomitome, the meshes of which are filled with karyoplasm, while connected with, or embedded in, the reticulum are a number of chromatin masses or chromosomes, which may present the appearance of a skein, or may assume the form of rods or loops. The nucleus is enclosed by a delicate nuclear membrane, and contains in its interior a well-defined nucleolus or germinal spot. Coverings of the ovum. The zona striata or zona pellucida is a thick membrane which, under the higher powers of the microscope, is seen to be radially striated. It persists for some time after fertilization has occurred, and may serve for protection during the earlier stages of segmentation. It is not yet determined whether the zona striata is a product of the cytoplasm of the ovum, or of the cells of the corona radiata, or both. The corona radiata consists of 2 or 3 strata of cells. They are derived from the cells of the follicle, and adhere to the outer surface of the zona striata when the ovum is set free from the follicle. The cells are radially arranged around the zona, those of the innermost layer being columnar in shape. The cells of the corona radiata soon disappear. In some animals they secrete, or are replaced by, a layer of adhesive protein, which may assist in protecting and nourishing the ovum. The phenomena attending the discharge of the ova from the follicles belong more to the ordinary functions of the ovary than to the general subject of embryology, and are therefore described with the anatomy of the ovaries. Footnote: see description of the ovary on a future page. End of footnote. Maturation of the ovum. Before an ovum can be fertilized, it must undergo a process of maturation or ripening. This takes place previous to or immediately after its escape from the follicle, and consists essentially of an unequal subdivision of the ovum, first into 2 and then into 4 cells. 3 of the 4 cells are small, incapable of further development, and are termed polar bodies or polar-cytes, while the 4th is large and constitutes the mature ovum. The process of maturation has not been observed in the human ovum, but has been carefully studied in the ova of some of the lower animals, to which the following description applies. It was pointed out on page 37 that the number of chromosomes found in the nucleus is constant for all the cells in an animal of any given species, and that in man the number is probably 24. This applies not only to the somatic cells, but to the primitive ova and their descendants. For the purpose of illustrating the process of maturation, a species may be taken in which the number of nuclear chromosomes is 4. If an ovum from such be observed at the beginning of the maturation process, it will be seen that the number of its chromosomes is apparently reduced to 2. In reality, however, the number is doubled, since each chromosome consists of 4 granules grouped to form a tetrad. During the metaphase, see page 37, each tetrad divides into two dyads, which are equally distributed between the nuclei of the two cells formed by the first division of the ovum. One of the cells is almost as large as the original ovum, and is named the secondary oocyte. The other is small, and is termed the first polar body. The secondary oocyte now undergoes subdivision, during which each dyad divides and contributes a single chromosome to the nucleus of each of the two resulting cells. This second division is also unequal, producing a large cell, which constitutes the mature ovum, and a small cell, the second polar body. The first polar body frequently divides while the second is being formed, and as a final result, 4 cells are produced, viz., the mature ovum and 3 polar bodies, each of which contains 2 chromosomes, i.e., 1/2 the number present in the nuclei of the somatic cells of members of the same species. The nucleus of the mature ovum is termed the female pronucleus. Embryology. 3. The spermatozoon. The spermatozoa or male germ cells are developed in the testes and are present in enormous numbers in the seminal fluid. Each consists of a small but greatly modified cell. The human spermatozoon possesses a head, a neck, a connecting piece or body, and a tail. The head is oval or elliptical but flattened, so that when viewed in profile it is pear-shaped. Its anterior 2/3 are covered by a layer of modified protoplasm, which is named the head cap. This in some animals, e.g. the salamander, is prolonged into a barbed spear-like process or perforator, which probably facilitates the entrance of the spermatozoon into the ovum. The posterior part of the head exhibits an affinity for certain reagents and presents a transversely striated appearance, being crossed by 3 or 4 dark bands. In some animals, a central rod-like filament extends forward for about 2/3 of the length of the head, while in others a rounded body is seen near its centre. The head contains a mass of chromatin, and is generally regarded as the nucleus of the cell surrounded by a thin envelope. The neck is less constricted in the human spermatozoon than in those of some of the lower animals. The anterior centriole, represented by 2 or 3 rounded particles, is situated at the junction of the head and neck, and behind it is a band of homogeneous substance. The connecting piece or body is rod-like and is limited behind by a terminal disc. The posterior centriole is placed at the junction of the body and neck, and like the anterior, consists of 2 or 3 rounded particles. From this centriole, an axial filament, surrounded by a sheath, runs backward through the body and tail. In the body, the sheath of the axial filament is encircled by a spiral thread, around which is an envelope containing mitochondria granules, and termed the mitochondria sheath. The tail is of great length, and consists of the axial thread or filament surrounded by its sheath, which may contain a spiral thread or may present a striated appearance. The terminal portion or end piece of the tail consists of the axial filament only. Krause gives the length of the human spermatozoon as between 52 μ and 62 μ, the head measuring 4 to 5 μ, the connecting piece 6 μ, and the tail from 41 μ to 52 μ. By virtue of their tails, which act as propellers, the spermatozoa are capable of free movement, and if placed in favourable surroundings, e.g. in the female passages, will retain their vitality and power of fertilizing for several days. In certain animals, e.g. bats, It has been proved that spermatozoa retained in the female passages for several months are capable of fertilizing. The spermatozoa are developed from the primitive germ cells which have become embedded in the testes, and the stages of their development are very similar to those of the maturation of the ovum. The primary germ cells undergo division and produce a number of cells termed spermatogonia, and from these the primary spermatocytes are derived. Each primary spermatocyte divides into two secondary spermatocytes, and each secondary spermatocyte into two spermatids or young spermatozoa. From this it will be seen that a primary spermatocyte gives rise to 4 spermatozoa. On comparing this process with that of the maturation of the ovum, it will be observed that the primary spermatocyte gives rise to 2 cells, the secondary spermatocytes, and the primary oocyte to 2 cells, the secondary oocyte and the first polar body. Again, the 2 secondary spermatocytes, by their subdivision, give origin to 4 spermatozoa, and the secondary oocyte and first polar body to 4 cells, the mature ovum and 3 polar bodies. In the development of the spermatozoa, as in the maturation of the ovum, there is a reduction of the nuclear chromosomes to 1/2 of those present in the primary spermatocyte. But here the similarity ends, for it must be noted that the 4 spermatozoa are of equal size, and each is capable of fertilizing a mature ovum, whereas the 3 polar bodies are not only very much smaller than the mature ovum, but are incapable of further development, and may be regarded as abortive ova. 4. Fertilization of the ovum. Fertilization consists in the union of the spermatozoon with the mature ovum. Nothing is known regarding the fertilization of the human ovum, but the various stages of the process have been studied in other mammals, and from the knowledge so obtained, it is believed that fertilization of the human ovum takes place in the lateral or ampullary part of the uterine tube, and the ovum is then conveyed along the tube to the cavity of the uterus, a journey probably occupying 7 or 8 days, and during which the ovum loses its corona radiata and zona striata, and undergoes segmentation. Sometimes the fertilized ovum is arrested in the uterine tube and there undergoes development, giving rise to a tubal pregnancy. Or it may fall into the abdominal cavity and produce an abdominal pregnancy. Occasionally the ovum is not expelled from the follicle when the latter ruptures, but is fertilized within the follicle and produces what is known as an ovarian pregnancy. Under normal conditions, only one spermatozoon enters the yolk and takes part in the process of fertilization. At the point where the spermatozoon is about to pierce, the yolk is drawn out into a conical elevation termed the cone of attraction. As soon as the spermatozoon has entered the yolk, the peripheral portion of the latter is transformed into a membrane, the vitelline membrane, which prevents the passage of additional spermatozoa. Occasionally a second spermatozoon may enter the yolk, thus giving rise to a condition of polyspermy. When this occurs, the ovum usually develops in an abnormal manner and gives rise to a monstrosity. Having pierced the yolk, the spermatozoon loses its tail, while its head and connecting piece assume the form of a nucleus containing a cluster of chromosomes. This constitutes the male pronucleus, and associated with it there are a centriole and centrosome. The male pronucleus passes more deeply into the yolk, and coincidentally with this, the granules of the cytoplasm surrounding it become radially arranged. The male and female pronuclei migrate toward each other, and, meeting near the centre of the yolk, fuse to form a new nucleus, the segmentation nucleus, which therefore contains both male and female nuclear substance. The former transmits the individualities of the male ancestors, the latter those of the female ancestors, to the future embryo. By the union of the male and female pronuclei, the number of chromosomes is restored to that which is present in the nuclei of the somatic cells. Embryology. 5. Segmentation of the fertilized ovum. The early segmentation of the human ovum has not yet been observed, but judging from what is known to occur in other mammals, it may be regarded as certain that the process starts immediately after the ovum has been fertilized, i.e., while the ovum is in the uterine tube. The segmentation nucleus exhibits the usual mitotic changes, and these are succeeded by a division of the ovum into two cells of nearly equal size. Footnote: in the mammalian ova, the nutritive yolk or deutoplasm is small in amount and uniformly distributed throughout the cytoplasm. Such ova undergo complete division during the process of segmentation and are therefore termed holoblastic. In the ova of birds, reptiles, and fishes, where the nutritive yolk forms by far the larger portion of the egg, the cleavage is limited to the formative yolk and is therefore only partial. Such ova are termed meroblastic. Again, it has been observed in some of the lower animals that the pronuclei do not fuse, but merely lie in apposition. At the commencement of the segmentation process, the chromosomes of the two pronuclei group themselves around the equator of the nuclear spindle and then divide. An equal number of male and female chromosomes travel to the opposite poles of the spindle, and thus the male and female pronuclei contribute equal shares of chromatin to the nuclei of the two cells which result from the subdivision of the fertilized ovum. End of footnote. The process is repeated again and again, so that the two cells are succeeded by 4, 8, 16, 32, and so on, with the result that a mass of cells is found within the zona striata, and to this mass the term morula is applied. The segmentation of the mammalian ovum may not take place in the regular sequence of 2, 4, 8, etc., since one of the two first formed cells may subdivide more rapidly than the other, giving rise to a 3 or a 5 cell stage. The cells of the morula are at first closely aggregated, but soon they become arranged into an outer or peripheral layer, the trophoblast, which does not contribute to the formation of the embryo proper, and an inner cell mass from which the embryo is developed. Fluid collects between the trophoblast and the greater part of the inner cell mass, and thus the morula is converted into a vesicle, the blastodermic vesicle. The inner cell mass remains in contact, however, with the trophoblast at one pole of the ovum. This is named the embryonic pole, since it indicates the situation where the future embryo will be developed. The cells of the trophoblast become differentiated into two strata. An outer, termed the syncytium or syncytiotrophoblast, so named because it consists of a layer of protoplasm studded with nuclei but showing no evidence of subdivision into cells, and an inner layer, the cytotrophoblast or layer of Langhans, in which the cell outlines are defined. As already stated, the cells of the trophoblast do not contribute to the formation of the embryo proper. They form the ectoderm of the chorion and play an important part in the development of the placenta. On the deep surface of the inner cell mass, a layer of flattened cells, the entoderm, is differentiated and quickly assumes the form of a small sac, the yolk sac. Spaces appear between the remaining cells of the mass, and by the enlargement and coalescence of these spaces, a cavity, termed the amniotic cavity, is gradually developed. The floor of this cavity is formed by the embryonic disc, composed of a layer of prismatic cells, the embryonic ectoderm, derived from the inner cell mass and lying in apposition with the entoderm. The primitive streak, formation of the mesoderm. The embryonic disc becomes oval and then pear-shaped, the wider end being directed forward. Near the narrow posterior end, an opaque streak, the primitive streak, makes its appearance, and extends along the middle of the disc for about 1/2 of its length. At the anterior end of the streak there is a knob-like thickening, termed Henson's knot. A shallow groove, the primitive groove, appears on the surface of the streak, and the anterior end of this groove communicates, by means of an aperture, the blastophore, with the yolk sac. The primitive streak is produced by a thickening of the axial part of the ectoderm, the cells of which multiply, grow downward, and blend with those of the subjacent entoderm. From the sides of the primitive streak, a third layer of cells, the mesoderm, extends lateralward between the ectoderm and entoderm. The caudal end of the primitive streak forms the cloacal membrane. The extension of the mesoderm takes place throughout the whole of the embryonic and extra-embryonic areas of the ovum, except in certain regions. One of these is seen immediately in front of the neural tube. Here the mesoderm extends forward in the form of two crescentic masses, which meet in the middle line so as to enclose behind them an area which is devoid of mesoderm. Over this area the ectoderm and entoderm come into direct contact with each other and constitute a thin membrane, the buccopharyngeal membrane, which forms a septum between the primitive mouth and pharynx. In front of the buccopharyngeal area, where the lateral crescents of mesoderm fuse in the middle line, the pericardium is afterward developed, and this region is therefore designated the pericardial area. A second region where the mesoderm is absent, at least for a time, is that immediately in front of the pericardial area. This is termed the proamniotic area, and is the region where the proamnion is developed. In man, however, a proamnion is apparently never formed. A third region is at the hind end of the embryo, where the ectoderm and entoderm come into apposition and form the cloacal membrane. The blastoderm now consists of 3 layers, named from without inward: ectoderm, mesoderm, and entoderm. Each has distinctive characteristics and gives rise to certain tissues of the body. Footnote: the mode of formation of the germ layers in the human ovum has not yet been observed. In the youngest known human ovum, viz. That described by Bryce and Teacher, all 3 layers are already present, and the mesoderm is split into its 2 layers. The extra-embryonic coelom is of considerable size, and scattered mesodermal strands are seen stretching between the mesoderm of the yolk sac and that of the chorion. End of footnote. Ectoderm. The ectoderm consists of columnar cells which are, however, somewhat flattened or cubical toward the margin of the embryonic disc. It forms the whole of the nervous system, the epidermis of the skin, the lining cells of the sebaceous, sudoriferous and mammary glands, the hairs and nails, the epithelium of the nose and adjacent air sinuses, and that of the cheeks and roof of the mouth. From it also are derived the enamel of the teeth and the anterior lobe of the hypophysis cerebri, the epithelium of the cornea, conjunctiva and lacrimal glands, and the neuroepithelium of the sense organs. Entoderm. The entoderm consists at first of flattened cells which subsequently become columnar. It forms the epithelial lining of the whole of the digestive tube, excepting part of the mouth and pharynx and the terminal part of the rectum, which are lined by involutions of the ectoderm, the lining cells of all the glands which open into the digestive tube, including those of the liver and pancreas, the epithelium of the auditory tube and tympanic cavity, of the trachea, bronchi, and air cells of the lungs, of the urinary bladder and part of the urethra, and that which lines the follicles of the thyroid glands and thymus. Mesoderm. The mesoderm consists of loosely arranged branched cells surrounded by a considerable amount of intercellular fluid. From it the remaining tissues of the body are developed. The endothelial lining of the heart and blood vessels and the blood corpuscles are, however, regarded by some as being of entodermal origin. As the mesoderm develops between the ectoderm and entoderm, it is separated into lateral halves by the neural tube and notochord, presently to be described. A longitudinal groove appears on the dorsal surface of either half, and divides it into a medial column, the paraxial mesoderm, lying on the side of the neural tube, and a lateral portion, the lateral mesoderm. The mesoderm in the floor of the groove connects the paraxial with the lateral mesoderm, and is known as the intermediate cell mass. In it, the genito-urinary organs are developed. The lateral mesoderm splits into two layers, an outer or somatic, which becomes applied to the inner surface of the ectoderm, and with it forms the somatopleure, and an inner or splanchnic, which adheres to the entoderm, and with it forms the splanchnoplure. The space between the two layers of the lateral mesoderm is termed the coelom.
Part 6. The neural groove and tube. In front of the primitive streak, two longitudinal ridges, caused by a folding up of the ectoderm, make their appearance, one on either side of the middle line. These are named the neural folds. They commence some little distance behind the anterior end of the embryonic disc, where they are continuous with each other, and from there gradually extend backward, one on either side of the anterior end of the primitive streak. Between these folds is a shallow median groove, the neural groove. The groove gradually deepens as the neural folds become elevated, and ultimately the folds meet and coalesce in the middle line and convert the groove into a closed tube, the neural tube or canal, the ectodermal wall of which forms the rudiment of the nervous system. After the coalescence of the neural folds over the anterior end of the primitive streak, the blastopore no longer opens on the surface, but into the closed canal of the neural tube, and thus a transitory communication, the neuronteric canal, is established between the neural tube and the primitive digestive tube. The coalescence of the neural folds occurs first in the region of the hind brain, and from there extends forward and backward. Toward the end of the third week, the front opening, anterior neuropore, of the tube finally closes at the anterior end of the future brain, and forms a recess which is in contact for a time with the overlying ectoderm. The hinder part of the neural groove presents for a time a rhomboidal shape, and to this expanded portion the term sinus rhomboidalis has been applied. Before the neural groove is closed, a ridge of ectodermal cells appears along the prominent margin of each neural fold. This is termed the neural crest or ganglion ridge, and from it the spinal and cranial nerve ganglia and the ganglia of the sympathetic nervous system are developed. By the upward growth of the mesoderm, the neural tube is ultimately separated from the overlying ectoderm. The cephalic end of the neural groove exhibits several dilatations, which, when the tube is closed, assume the form of three vesicles. These constitute the three primary cerebral vesicles, and correspond respectively to the future forebrain, prosencephalon, midbrain, mesencephalon, and hindbrain, rhombencephalon. The walls of the vesicles are developed into the nervous tissue and neuroglia of the brain, and their cavities are modified to form its ventricles. The remainder of the tube forms the medulla spinalis, or spinal cord. From its ectodermal wall, the nervous and neuroglial elements of the medulla spinalis are developed while the cavity persists as the central canal. Part 7. The notochord. The notochord consists of a rod of cells situated on the ventral aspect of the neural tube. It constitutes the foundation of the axial skeleton, since around it the segments of the vertebral column are formed. Its appearance synchronizes with that of the neural tube. On the ventral aspect of the neural groove an axial thickening of the entoderm takes place. This thickening assumes the appearance of a furrow, the chordal furrow, the margins of which come into contact and so convert it into a solid rod of cells, the notochord, which is then separated from the entoderm. It extends throughout the entire length of the future vertebral column and reaches as far as the anterior end of the midbrain, where it ends in a hook-like extremity in the region of the future dorsum celli of the sphenoid bone. It lies at first between the neural tube and the entoderm of the yolk sac, but soon becomes separated from them by the mesoderm, which grows medialward and surrounds it. From the mesoderm, surrounding the neural tube and notochord, the skull and vertebral column, and the membranes of the brain and medulla spinalis are developed. Part 8. The primitive segments. Toward the end of the second week, transverse segmentation of the paraxial mesoderm begins, and it is converted into a series of well-defined, more or less cubical masses, the primitive segments, which occupy the entire length of the trunk on either side of the middle line from the occipital region of the head. Each segment contains a central cavity, myocele, which, however, is soon filled with angular and spindle-shaped cells. The primitive segments lie immediately under the ectoderm on the lateral aspect of the neural tube and notochord, and are connected to the lateral mesoderm by the intermediate cell mass. Those of the trunk may be arranged in the following groups, viz. cervical 8, thoracic 12, lumbar 5, sacral 5, and coccygeal from 5 to 8. Those of the occipital region of the head are usually described as being 4 in number. In mammals, primitive segments of the head can be recognized only in the occipital region, but a study of the lower vertebrates leads to the belief that they are present also in the anterior part of the head, and that altogether 9 segments are represented in the cephalic region. Part 9. Separation of the embryo. The embryo increases rapidly in size, but the circumference of the embryonic disc, or line of meeting of the embryonic and amniotic parts of the ectoderm, is of relatively slow growth, and gradually comes to form a constriction between the embryo and the greater part of the yolk sac. By means of this constriction, which corresponds to the future umbilicus, a small part of the yolk sac is enclosed within the embryo and constitutes the primitive digestive tube. The embryo increases more rapidly in length than in width, and its cephalic and caudal ends soon extend beyond the corresponding parts of the circumference of the embryonic disc and are bent in a ventral direction to form the cephalic and caudal folds respectively. The cephalic fold is first formed, and as the proamniotic area, lying immediately in front of the pericardial area, forms the anterior limit of the circumference of the embryonic disc, the forward growth of the head necessarily carries with it the posterior end of the pericardial area, so that this area and the buccopharyngeal membrane are folded back under the head of the embryo, which now encloses a diverticulum of the yolk sac named the foregut. The caudal end of the embryo is at first connected to the chorion by a band of mesoderm called the body stalk, but with the formation of the caudal fold the body stalk assumes a ventral position. A diverticulum of the yolk sac extends into the tail fold and is termed the hindgut. Between the foregut and the hindgut there exists for a time a wide opening into the yolk sac, but the latter is gradually reduced to a small pear-shaped sac, sometimes termed the umbilical vesicle, and the channel of communication is at the same time narrowed and elongated to form a tube called the vitelline duct. Part 10. The yolk sac. The yolk sac is situated on the ventral aspect of the embryo. It is lined by entoderm, outside of which is a layer of mesoderm. It is filled with fluid, the vitelline fluid, which possibly may be utilized for the nourishment of the embryo during the earlier stages of its existence. Blood is conveyed to the wall of the sac by the primitive aortae, and after circulating through a wide-meshed capillary plexus, is returned by the vitelline veins to the tubular heart of the embryo. This constitutes the vitelline circulation, and by means of it nutritive material is absorbed from the yolk sac and conveyed to the embryo. At the end of the 4th week, the yolk sac presents the appearance of a small pear-shaped vesicle, umbilical vesicle, opening into the digestive tube by a long narrow tube, the vitelline duct. The vesicle can be seen in the afterbirth as a small, somewhat oval-shaped body whose diameter varies from 1 mm to 5 mm. It is situated between the amnion and the chorion, and may lie on or at a varying distance from the placenta. As a rule, the duct undergoes complete obliteration during the 7th week, but in about 3% of cases its proximal part persists as a diverticulum from the small intestine. Meckel's diverticulum, which is situated about 3 or 4 feet above the ileocolic junction, and may be attached by a fibrous cord to the abdominal wall at the umbilicus. Sometimes a narrowing of the lumen of the ileum is seen opposite the site of the attachment of the duct. Part 11. Development of the fetal membranes and placenta. The allantois arises as a tubular diverticulum of the posterior part of the yolk sac. When the hind gut is developed, the allantois is carried backward with it, and then opens into the cloaca, or terminal part of the hindgut. It grows out into the body stalk, a mass of mesoderm, which lies below and around the tail end of the embryo. The diverticulum is lined by entoderm and covered by mesoderm, and in the latter are carried the allantoic or umbilical vessels. In reptiles, birds, and many mammals, the allantois becomes expanded into a vesicle which projects into the extraembryonic coelom. If its further development be traced in the bird, it is seen to project to the right side of the embryo, and gradually expanding, it spreads over its dorsal surface as a flattened sac between the amnion and the serosa, and extending in all directions, ultimately surrounds the yolk. Its outer wall becomes applied to and fuses with the serosa, which lies immediately inside the shell membrane. Blood is carried to the allantoic sac by the 2 allantoic or umbilical arteries, which are continuous with the primitive aortae, and after circulating through the allantoic capillaries, is returned to the primitive heart by the 2 umbilical veins. In this way the allantoic circulation, which is of the utmost importance in connection with the respiration and nutrition of the chick, is established. Oxygen is taken from, and carbonic acid is given up to the atmosphere through the eggshell, while nutritive materials are at the same time absorbed by the blood from the yolk. In man and other primates, the nature of the allantois is entirely different from that just described. Here it exists merely as a narrow, tubular diverticulum of the hindgut, and never assumes the form of a vesicle outside the embryo. With the formation of the amnion, the embryo is, in most animals, entirely separated from the chorion, and is only again united to it when the allantoic mesoderm spreads over and becomes applied to its inner surface. The human embryo, on the other hand, as was pointed out by His, is never wholly separated from the chorion, its tail end being from the first connected with the chorion by means of a thick band of mesoderm named the body stalk. Into this stalk the tube of the allantois extends. The amnion. The amnion is a membranous sac which surrounds and protects the embryo. It is developed in reptiles, birds, and mammals, which are hence called amniota, but not in amphibia and fishes, which are consequently termed anamnia. In the human embryo the earliest stages of the formation of the amnion have not been observed. In the youngest embryo which has been studied, the amnion was already present as a closed sac, and is indicated on page 46 appears in the inner cell mass as a cavity. This cavity is roofed in by a single stratum of flattened ectodermal cells, the amniotic ectoderm, and its floor consists of the prismatic ectoderm of the embryonic disc, the continuity between the roof and floor being established at the margin of the embryonic disc. Outside the amniotic ectoderm is a thin layer of mesoderm, which is continuous with that of the soma teplur and is connected by the body stalk with the mesodermal lining of the chorion. When first formed, the amnion is in contact with the body of the embryo, but about the 4th or 5th week, fluid, liquor amnii, begins to accumulate within it. This fluid increases in quantity and causes the amnion to expand and ultimately to adhere to the inner surface of the chorion, so that the extra embryonic part of the coelom is obliterated. The liquor amnii increases in quantity up to the 6th or 7th month of pregnancy, after which it diminishes somewhat. At the end of pregnancy it amounts to about 1 liter. It allows of the free movements of the fetus during the later stages of pregnancy, and also protects it by diminishing the risk of injury from without. It contains less than 2% of solids, consisting of urea and other extractives, inorganic salts, a small amount of protein, and frequently a trace of sugar. That some of the liquor amnii is swallowed by the fetus is proved by the fact that epidermal debris and hairs have been found among the contents of the fetal alimentary canal. In reptiles, birds, and many mammals, the amnion is developed in the following manner. At the point of constriction, where the primitive digestive tube of the embryo joins the yolk sac, a reflection or folding upward of the soma teplur takes place. This, the amniotic fold, first makes its appearance at the cephalic extremity, and subsequently at the caudal end and sides of the embryo, and gradually rising more and more, its different parts meet and fuse over the dorsal aspect of the embryo, and enclose a cavity, the amniotic cavity. After the fusion of the edges of the amniotic fold, the 2 layers of the fold become completely separated, the inner forming the amnion, the outer the false amnion, or serosa. The space between the amnion and the serosa constitutes the extraembryonic coelom, and for a time communicates with the embryonic coelom. The umbilical cord and body stalk. The umbilical cord attaches the fetus to the placenta. Its length at full time, as a rule, is about equal to the length of the fetus, that is, about 50 centimeters, but it may be greatly diminished or increased. The rudiment of the umbilical cord is represented by the tissue which connects the rapidly growing embryo with the extraembryonic area of the ovum. Included in this tissue are the body stalk and the vitelline duct, the former containing the allantoic diverticulum and the umbilical vessels, the latter forming the communication between the digestive tube and the yolk sac. The body stalk is the posterior segment of the embryonic area, and is attached to the chorion. It consists of a plate of mesoderm covered by thickened ectoderm, on which a trace of the neural groove can be seen, indicating its continuity with the embryo. Running through its mesoderm are the 2 umbilical arteries and the 2 umbilical veins, together with the canal of the allantois, the last being lined by entoderm. Its dorsal surface is covered by the amnion, while its ventral surface is bounded by the extraembryonic coelom and is in contact with the vitelline duct and yolk sac. With the rapid elongation of the embryo and the formation of the tail fold, the body stalk comes to lie on the ventral surface of the embryo, where its mesoderm blends with that of the yolk sac and the vitelline duct. The lateral leaves of soma teplur then grow round on each side, and meeting on the ventral aspect of the allantois, enclose the vitelline duct and vessels, together with a part of the extraembryonic coelom. The latter is ultimately obliterated. The cord is covered by a layer of ectoderm which is continuous with that of the amnion, and its various constituents are enveloped by embryonic gelatinous tissue, jelly of Wharton. The vitelline vessels and duct, together with the right umbilical vein, undergo atrophy and disappear, and thus the cord, at birth, contains a pair of umbilical arteries and one, the left, umbilical vein. Implantation or embedding of the ovum. As described, fertilization of the ovum occurs in the lateral or ampullary end of the uterine tube and is immediately followed by segmentation. On reaching the cavity of the uterus, the segmented ovum adheres like a parasite to the uterine mucous membrane, destroys the epithelium over the area of contact, and excavates for itself a cavity in the mucous membrane in which it becomes embedded. In the ovum described by Bryce and Teacher, the point of entrance was visible as a small gap closed by a mass of fibrin and leukocytes. In the ovum described by Peters, the opening was covered by a mushroom-shaped mass of fibrin and blood clot, the narrow stalk of which plugged the aperture in the mucous membrane. Soon, however, all trace of the opening is lost, and the ovum is then completely surrounded by the uterine mucous membrane. The structure actively concerned in the process of excavation is the trophoblast of the ovum, which possesses the power of dissolving and absorbing the uterine tissues. The trophoblast proliferates rapidly and forms a network of branching processes which cover the entire ovum and invade and destroy the maternal tissues and open into the maternal blood vessels, with the result that the spaces in the trophoblastic network are filled with maternal blood. These spaces communicate freely with one another and become greatly distended and form the intervillous space. The decidua. Before the fertilized ovum reaches the uterus, the mucous membrane of the body of the uterus undergoes important changes, and is then known as the decidua. The thickness and vascularity of the mucous membrane are greatly increased. Its glands are elongated and open on its free surface by funnel-shaped orifices, while their deeper portions are tortuous and dilated into irregular spaces. The interglandular tissue is also increased in quantity, and is crowded with large, round, oval, or polygonal cells termed decidual cells. These changes are well advanced by the second month of pregnancy, when the mucous membrane consists of the following strata. 1. Stratum compactum. Next, the free surface. In this the uterine glands are only slightly expanded and are lined by columnar cells. 2. Stratum spongiosum, in which the gland tubes are greatly dilated and very tortuous, and are ultimately separated from one another by only a small amount of interglandular tissue, while their lining cells are flattened or cubical. 3. A thin, unaltered or boundary layer. Next, the uterine muscular fibers, containing the deepest parts of the uterine glands, which are not dilated and are lined with columnar epithelium. It is from this epithelium that the epithelial lining of the uterus is regenerated after pregnancy. Distinctive names are applied to different portions of the decidua. The part which covers in the ovum is named the decidua capsularis, the portion which intervenes between the ovum and the uterine wall is named the decidua basalis, or decidua placentalis. It is here that the placenta is subsequently developed. The part of the decidua which lines the remainder of the body of the uterus is known as the decidua vera or decidua parietalis. Coincidentally with the growth of the embryo, the decidua capsularis is thinned and extended, and the space between it and the decidua vera is gradually obliterated, so that by the third month of pregnancy the two are in contact. By the fifth month of pregnancy, the decidua capsularis has practically disappeared, while during the succeeding months the decidua vera also undergoes atrophy, owing to the increased pressure. The glands of the stratum compactum are obliterated, and their epithelium is lost. In the stratum spongiosum, the glands are compressed and appear as slit-like fissures, while their epithelium undergoes degeneration. In the unaltered or boundary layer, however, the glandular epithelium retains a columnar or cubical form. The chorion. The chorion consists of two layers, an outer formed by the primitive ectoderm or trophoblast, and an inner by the somatic mesoderm. With this latter the amnion is in contact. The trophoblast is made up of an internal layer of cubical or prismatic cells, the cytotrophoblast, or layer of Langhans, and an external layer of richly nucleated protoplasm devoid of cell boundaries, the syncytiotrophoblast. It undergoes rapid proliferation and forms numerous processes: the chorionic villi, which invade and destroy the uterine decidua and at the same time absorb from it nutritive materials for the growth of the embryo. The chorionic villi are at first small and non-vascular, and consist of trophoblast only, but they increase in size and ramify, while the mesoderm, carrying branches of the umbilical vessels, grows into them, and in this way they are vascularized. Blood is carried to the villi by the branches of the umbilical arteries, and after circulating through the capillaries of the villi, is returned to the embryo by the umbilical veins. Until about the end of the second month of pregnancy, the villi cover the entire chorion and are almost uniform in size, but after this they develop unequally. The greater part of the chorion is in contact with the decidua capsularis, and over this portion of the villi, with their contained vessels, undergo atrophy, so that by the fourth month scarcely a trace of them is left, and hence this part of the chorion becomes smooth, and is named the chorion live. As it takes no share in the formation of the placenta, it is also named the non-placental part of the chorion. On the other hand, the villi on that part of the chorion which is in contact with the decidua placentalis increase greatly in size and complexity, and hence this part is named the chorion frondosum. The placenta. The placenta connects the fetus to the uterine wall and is the organ by means of which the nutritive, respiratory, and excretory functions of the fetus are carried out. It is composed of fetal and maternal portions. Fetal portion. The fetal portion of the placenta consists of the villi of the chorion frondosum. These branch repeatedly and increase enormously in size. These greatly ramified villi are suspended in the intervillous space and are bathed in maternal blood, which is conveyed to the space by the uterine arteries and carried away by the uterine veins. A branch of an umbilical artery enters each villus and ends in a capillary plexus from which the blood is drained by a tributary of the umbilical vein. The vessels of the villus are surrounded by a thin layer of mesoderm consisting of gelatinous connective tissue, which is covered by two strata of ectodermal cells derived from the trophoblast. The deeper stratum, next the mesodermic tissue, represents the cytotrophoblast or layer of Langhans. The superficial, in contact with the maternal blood, the syncytiotrophoblast. After the fifth month the two strata of cells are replaced by a single layer of somewhat flattened cells. Maternal portion. The maternal portion of the placenta is formed by the decidua placentalis containing the intervillous space. As already explained, this space is produced by the enlargement and intercommunication of the spaces in the trophoblastic network. The changes involve the disappearance of the greater portion of the stratum compactum, but the deeper part of this layer persists and is condensed to form what is known as the basal plate. Between this plate and the uterine muscular fibers are the stratum spongiosum and the boundary layer. Through these and the basal plate, the uterine arteries and veins pass to and from the intervillous space. The endothelial lining of the uterine vessels ceases at the point where they terminate in the intervillous space, which is lined by the syncytiotrophoblast. Portions of the stratum compactum persist and are condensed to form a series of septa, which extend from the basal plate through the thickness of the placenta and subdivide it into the lobules or cotyledons seen on the uterine surface of the detached placenta. The fetal and maternal blood currents traverse the placenta, the former passing through the blood vessels of the placental villi, and the latter through the intervillous space. The two currents do not intermingle, being separated from each other by the delicate walls of the villi. Nevertheless, the fetal blood is able to absorb, through the walls of the villi, oxygen and nutritive materials from the maternal blood, and give up to the latter its waste products. The blood, so purified, is carried back to the fetus by the umbilical vein. It will thus be seen that the placenta not only establishes a mechanical connection between the mother and the fetus, but subserves for the latter the purposes of nutrition, respiration, and excretion. In favor of the view that the placenta possesses certain selective powers, may be mentioned the fact that glucose is more plentiful in the maternal than in the fetal blood. It is interesting to note also that the proportion of iron and of lime and potash in the fetus is increased during the last months of pregnancy. Further, there is evidence that the maternal leukocytes may migrate into the fetal blood, since leukocytes are much more numerous in the blood of the umbilical vein than in that of the umbilical arteries. The placenta is usually attached near the fundus uteri and more frequently on the posterior than on the anterior wall of the uterus. It may, however, occupy a lower position, and in rare cases its site is close to the orificium internum uteri, which it may occlude, thus giving rise to the condition known as placenta previa. Separation of the placenta. After the child is born, the placenta and membranes are expelled from the uterus as the afterbirth. The separation of the placenta from the uterine wall takes place through the stratum spongiosum and necessarily causes rupture of the uterine vessels. The orifices of the torn vessels are, however, closed by the firm contraction of the uterine muscular fibers, and thus postpartum hemorrhage is controlled. The epithelial lining of the uterus is regenerated by the proliferation and extension of the epithelium, which lines the persistent portions of the uterine glands in the unaltered layer of the decidua. The expelled placenta appears as a discoid mass which weighs about 450 grams and has a diameter of from 15 to 20 centimeters. Its average thickness is about 3 centimeters, but this diminishes rapidly toward the circumference of the disc, which is continuous with the membranes. Its uterine surface is divided by a series of fissures into lobules or cotyledons, the fissures containing the remains of the septa, which extended between the maternal and fetal portions. Most of these septa end in irregular or pointed processes. Others, especially those near the edge of the placenta, pass through its thickness and are attached to the chorion. In the early months these septa convey branches of the uterine arteries which open into the intervillous space on the surfaces of the septa. The fetal surface of the placenta is smooth, being closely invested by the amnion. Seen through the latter, the chorion presents a mottled appearance, consisting of gray, purple, or yellowish areas. The umbilical cord is usually attached near the center of the placenta, but may be inserted anywhere between the center and the margin. In some cases it is inserted into the membranes, that is, the filamentous insertion. From the attachment of the cord, the longer branches of the umbilical vessels radiate under the amnion, the veins being deeper and larger than the arteries. The remains of the vitelline duct and yolk sac may be sometimes observed beneath the amnion, close to the cord, the former as an attenuated thread, the latter as a minute sac. On section, the placenta presents a soft, spongy appearance, caused by the greatly branched villi. Surrounding them is a varying amount of maternal blood, giving the dark red color to the placenta. Many of the larger villi extend from the chorionic to the decidual surface, while others are attached to the septa, which separate the cotyledons, but the great majority of the villi hang free in the intervillous space. Part 12. The branchial region. The branchial or visceral arches and pharyngeal pouches. In the lateral walls of the anterior part of the foregut, 5 pharyngeal pouches appear. Each of the upper 4 pouches is prolonged into a dorsal and a ventral diverticulum. Over these pouches corresponding indentations of the ectoderm occur, forming what are known as the branchial or outer pharyngeal grooves. The intervening mesoderm is pressed aside and the ectoderm comes for a time into contact with the entodermal lining of the foregut, and the 2 layers unite along the floors of the grooves to form thin closing membranes between the foregut and the exterior. Later, the mesoderm again penetrates between the entoderm and the ectoderm. In gill-bearing animals, the closing membranes disappear and the grooves become complete clefts, the gill clefts, opening from the pharynx onto the exterior. Perforation, however, does not occur in birds or mammals. The grooves separate a series of rounded bars or arches, the branchial or visceral arches, in which thickening of the mesoderm takes place. The dorsal ends of these arches are attached to the sides of the head, while the ventral extremities ultimately meet in the middle line of the neck. In all, 6 arches make their appearance, but of these only the first 4 are visible externally. The first arch is named the mandibular, and the second the hyoid. The others have no distinctive names. In each arch a cartilaginous bar consisting of right and left halves is developed, and with each of these there is one of the primitive aortic arches. The mandibular arch lies between the first branchial groove and the stomotium. From it are developed the lower lip, the mandible, the muscles of mastication, and the anterior part of the tongue. Its cartilaginous bar is formed by what are known as Meckel's cartilages. Above this the incus is developed. The dorsal end of each cartilage is connected with the ear capsule and is ossified to form the malleus. The ventral ends meet each other in the region of the symphysis menti, and are usually regarded as undergoing ossification to form that portion of the mandible which contains the incisor teeth. The intervening part of the cartilage disappears. The portion immediately adjacent to the malleus is replaced by fibrous membrane, which constitutes the sphenomandibular ligament, while from the connective tissue covering the remainder of the cartilage, the greater part of the mandible is ossified. From the dorsal ends of the mandibular arch a triangular process, the maxillary process, grows forward on either side and forms the cheek and lateral part of the upper lip. The second or hyoid arch assists in forming the side and front of the neck. From its cartilage are developed the styloid process, stylohyoid ligament, and lesser cornu of the hyoid bone. The stages probably arises in the upper part of this arch. The cartilage of the third arch gives origin to the greater cornu of the hyoid bone. The ventral ends of the second and third arches unite with those of the opposite side, and form a transverse band, from which the body of the hyoid bone and the posterior part of the tongue are developed. The ventral portions of the cartilages of the fourth and fifth arches unite to form the thyroid cartilage. From the cartilages of the sixth arch, the cricoid and arytenoid cartilages and the cartilages of the trachea are developed. The mandibular and hyoid arches grow more rapidly than those behind them, with the result that the latter become, to a certain extent, telescoped within the former, and a deep depression, the sinus cervicalis, is formed on either side of the neck. This sinus is bounded in front by the hyoid arch, and behind by the thoracic wall. It is ultimately obliterated by the fusion of its walls. From the first branchial groove, the concha auriculae and external acoustic meatus are developed, while around the groove there appear on the mandibular and hyoid arches a number of swellings from which the auricula or pinna is formed. The first pharyngeal pouch is prolonged dorsally to form the auditory tube and the tympanic cavity. The closing membrane between the mandibular and hyoid arches is invaded by mesoderm and forms the tympanic membrane. No traces of the second, third, and fourth branchial grooves persist. The inner part of the second pharyngeal pouch is named the sinus tonsillaris. In it the tonsil is developed, above which a trace of the sinus persists as the supratonsillar fossa. The fossa of Rosenmuller, or lateral recess of the pharynx, is by some regarded as a persistent part of the second pharyngeal pouch, but it is probably developed as a secondary formation. From the third pharyngeal pouch the thymus arises as an entodermal diverticulum on either side, and from the fourth pouches small diverticula project and become incorporated with the thymus. But in man these diverticula probably never form true thymus tissue. The parathyroids also arise as diverticula from the third and fourth pouches. From the fifth pouches the ultimobranchial bodies originate and are enveloped by the lateral prolongations of the median thyroid rudiment. They do not, however, form true thyroid tissue, nor are any traces of them found in the human adult. The nose and face. During the third week 2 areas of thickened ectoderm, the olfactory areas, appear immediately under the forebrain in the anterior wall of the stomotium, one on either side of a region termed the frontonasal process. By the upgrowth of the surrounding parts, these areas are converted into pits, the olfactory pits, which indent the frontonasal process and divide it into a medial and two lateral nasal processes. The rounded lateral angles of the medial process constitute the globular processes of His. The olfactory pits form the rudiments of the nasal cavities, and from their ectodermal lining the epithelium of the nasal cavities, with the exception of that of the inferior metases, is derived. The globular processes are prolonged backward as plates, termed the nasal laminae. These laminae are at first some distance apart, but gradually approaching, they ultimately fuse and form the nasal septum. The processes themselves meet in the middle line and form the premaxillae and the philtrum or central part of the upper lip. The depressed part of the medial nasal process between the globular processes forms the lower part of the nasal septum or columella, while above this is seen a prominent angle, which becomes the future apex, and still higher a flat area, the future bridge, of the nose. The lateral nasal processes form the aili of the nose. Continuous with the dorsal end of the mandibular arch, and growing forward from its cephalic border, is a triangular process, the maxillary process, the ventral extremity of which is separated from the mandibular arch by a shaped notch. The maxillary process forms the lateral wall and floor of the orbit, and in it are ossified the zygomatic bone and the greater part of the maxilla. It meets with the lateral nasal process, from which, however, it is separated for a time by a groove, the naso-optic furrow, that extends from the furrow encircling the eyeball to the olfactory pit. The maxillary processes ultimately fuse with the lateral nasal and globular processes, and form the lateral parts of the upper lip and the posterior boundaries of the nares. From the 3rd to the 5th month the nares are filled by masses of epithelium, on the breaking down and disappearance of which the permanent openings are produced. The maxillary process also gives rise to the lower portion of the lateral wall of the nasal cavity. The roof of the nose and the remaining parts of the lateral wall, viz., the ethmoidal labyrinth, the inferior nasal concha, the lateral cartilage, and the lateral crus of the alar cartilage, are developed in the lateral nasal process. By the fusion of the maxillary and nasal processes in the roof of the stomodium, the primitive palate is formed, and the olfactory pits extend backward above it. The posterior end of each pit is closed by an epithelial membrane, the bucco-nasal membrane, formed by the apposition of the nasal and stomodial epithelium. By the rupture of these membranes, the primitive choanae, or openings between the olfactory pits and the stomodium, are established. The floor of the nasal cavity is completed by the development of a pair of shelf-like palatine processes, which extend medialward from the maxillary processes. These coalesce with each other in the middle line, and constitute the entire palate, except a small part in front which is formed by the premaxillary bones. 2 apertures persist for a time between the palatine processes and the premaxillae, and represent the permanent channels which in the lower animals connect the nose and mouth. The union of the parts which form the palate commences in front, the premaxillary and palatine processes joining in the 8th week, while the region of the future hard palate is completed by the 9th, and that of the soft palate by the 11th week. By the completion of the palate the permanent choanae are formed, and are situated a considerable distance behind the primitive choanae. The deformity known as cleft palate results from a non-union of the palatine processes, and that of hare lip through a non-union of the maxillary and globular processes. See page 199. The nasal cavity becomes divided by a vertical septum, which extends downward and backward from the medial nasal process and nasal laminae, and unites below with the palatine processes. Into this septum a plate of cartilage extends from the under aspect of the ethmoid plate of the chondrocranium. The anterior part of this cartilaginous plate persists as the septal cartilage of the nose and the medial crus of the alar cartilage, but the posterior and upper parts are replaced by the vomer and perpendicular plate of the ethmoid. On either side of the nasal septum, at its lower and anterior part, the ectoderm is invaginated to form a blind pouch or diverticulum, which extends backward and upward into the nasal septum and is supported by a curved plate of cartilage. These pouches form the rudiments of the vomeronasal organs of Jacobson, which open below, close to the junction of the premaxillary and maxillary bones. The limbs. The limbs begin to make their appearance in the third week as small elevations or buds at the side of the trunk. Prolongations from the muscle and cutis plates of several primitive segments extend into each bud, and carry with them the anterior divisions of the corresponding spinal nerves. The nerves supplying the limbs indicate the number of primitive segments which contribute to their formation, the upper limb being derived from 7, viz. 4th cervical to 2nd thoracic inclusive, and the lower limb from 10, viz. 12th thoracic to 4th sacral inclusive. The axial part of the mesoderm of the limb bud becomes condensed and converted into its cartilaginous skeleton, and by the ossification of this, the bones of the limbs are formed. By the 6th week, the 3 chief divisions of the limbs are marked off by furrows, the upper into arm, forearm, and hand, the lower into thigh, leg, and foot. The limbs are at first directed backward nearly parallel to the long axis of the trunk, and each presents 2 surfaces and 2 borders. Of the surfaces, one, the future flexor surface of the limb, is directed ventrally; the other, the extensor surface, dorsally. One border, the preaxial, looks forward toward the cephalic end of the embryo, and the other, the postaxial, backward toward the caudal end. The lateral epicondyle of the humerus, the radius, and the thumb lie along the preaxial border of the upper limb, and the medial epicondyle of the femur, the tibia, and the great toe along the corresponding border of the lower limb. The preaxial part is derived from the anterior segments, the postaxial from the posterior segments of the limb bud, and this explains to a large extent the innervation of the adult limb, the nerves of the more anterior segments being distributed along the preaxial, radial or tibial, and those of the more posterior along the postaxial, ulnar or fibular border of the limb. The limbs next undergo a rotation or torsion through an angle of 90 degrees around their long axes, the rotation being effected almost entirely at the limb girdles. In the upper limb the rotation is outward and forward; in the lower limb, inward and backward. As a consequence of this rotation, the preaxial, radial border of the forelimb is directed lateralward, and the preaxial, tibial border of the hind limb is directed medialward. Thus, the flexor surface of the forelimb is turned forward, and that of the hind limb backward.
Development of the body cavities. In the human embryo described by Peters, the mesoderm outside the embryonic disc is split into 2 layers, enclosing an extra embryonic column. There is no trace of an intra-embryonic column. At a later stage, 4 cavities are formed within the embryo, viz., 1 on either side within the mesoderm of the pericardial area, and 1 in either lateral mass of the general mesoderm. All these are at first independent of each other and of the extra-embryonic coelom, but later they become continuous. The 2 cavities in the general mesoderm unite on the ventral aspect of the gut and form the pleuroperitoneal cavity, which becomes continuous with the remains of the extra-embryonic coelom around the umbilicus. The 2 cavities in the pericardial area rapidly join to form a single pericardial cavity, and this from 2 lateral diverticula extend caudalward to open into the pleuroperitoneal cavity. Between the 2 latter diverticula is a mass of mesoderm containing the ducts of Cuvier, and this is continuous ventrally with the mesoderm, in which the umbilical veins are passing to the sinus venosus. A septum of mesoderm thus extends across the body of the embryo. It is attached in front to the body wall, between the pericardium and umbilicus, behind the body wall at the level of the second cervical segment. Laterally, it is deficient where the pericardial pleural peritoneal cavities communicate, while it is perforated in the middle line by the foregut. This partition is termed septum transversum, and is at first a bulky plate of tissue. As development proceeds, the dorsal end of the septum is carried gradually caudalward, and when it reaches the fifth cervical segment, muscular tissue with the phrenic nerve grows into it. It continues to recede, however, until it reaches the position of the adult diaphragm on the bodies of the upper lumbar vertebrae. The liver buds grow into the septum transversum and undergo development there. The lung buds, meantime, have grown out from the foregut and project laterally into the forepart of the pleural peritoneal cavity. The developing stomach and liver are embedded in the septum transversum. Caudal to this, the intestines project into the back part of the pleural peritoneal cavity. Owing to the descent of the dorsal end of the septum transversum, the lung buds come to lie above the septum, and thus pleural and peritoneal portions of the pleural peritoneal cavity, still, however, in free communication with one another, may be recognized. The pericardial cavity opens into the pleural part. The ultimate separation of the permanent cavities from one another is effected by the growth of a ridge of tissue on either side of the mesoderm surrounding the duct of Cuvier. The front part of this ridge grows across and obliterates the pleural pericardial opening. The hinder part grows across the pleural peritoneal opening. With the continued growth of the lungs, the pleural cavities are pushed forward in the body wall towards the ventral median line, thus separating the pericardium from the lateral thoracic walls. The further development of the peritoneal cavity has been described with the development of the digestive tube. The form of the embryo at different stages of its growth. First week. During this period, the ovum is in the uterine tube. Having been fertilized in the upper part of the tube, it slowly passes down, undergoing segmentation, and reaches the uterus. Peters describes a specimen, the age of which he reckoned as from 3 to 4 days. Footnote. Bryce and Teacher, Early Development and Embedding of the Human Ovum, 1908, have described an ovum which they regard as 13 to 14 days old. In it, the two vesicles, the amnion and yolk sac, were present, but there was no trace of a layer of embryonic ectoderm. They are of opinion that the age of Peter's ovum has been understated and estimate it as between 13 1/2 and 14 1/2 days. End footnote. It was embedded in the decidua on the posterior wall of the uterus and enveloped by a decidua capsularis, the central part of which, however, consisted merely of a layer of fibrin. The ovum was in the form of a sac, the outer wall of which consisted of a layer of trophoblast. Inside this was a thin layer of mesoderm composed of round, oval and spindle-shaped cells. Numerous villous processes, some consisting of trophoblast only, others possessing a core of mesoderm, projected from the surface of the ovum into the surrounding decidua. Inside this sac, the rudiment of the embryo was found in the form of a patch of ectoderm, covered by a small but completely closed amnion. It possessed a minute yolk sac and was surrounded by mesoderm, which was connected by a band to that lining the trophoblast. Second week. By the end of this week, the ovum has increased considerably in size, and the majority of its villi are vascularized. The embryo has assumed a definite form, and its cephalic and caudal extremities are easily distinguished. The neural folds are partly united. The embryo is more completely separated from the yolk sac, and the paraxial mesoderm is being divided into the primitive segments. Third week. By the end of the third week, the embryo is strongly curved, and the primitive segments number about 30. The primary divisions of the brain are visible, and the optic and auditory vesicles are formed. 4 branchial grooves are present. The stomadeum is well marked, and the bucopharyngeal membrane has disappeared. The rudiments of the limbs are seen as short buds, and the Wolffian bodies are visible. Fourth week. The embryo is markedly curved on itself, and when viewed in profile is almost circular in outline. The cerebral hemispheres appear as hollow buds, and the elevations, which form the rudiments of the auricula, are visible. The limbs now appear as oval, flattened projections. 5th week. The embryo is less curved, and the head is relatively of large size. Differentiation of the limbs into their segments occurs. The nose forms a short, flattened projection. The cloacal tubercle is evident. 6th week. The curvature of the embryo is further diminished. The branchial grooves, except the first, have disappeared, and the rudiments of the fingers and toes can be recognized. 7th and 8th weeks. The flexure of the head is gradually reduced, and the neck is somewhat lengthened. The upper lip is completed, and the nose is more prominent. The nostrils are directed forward, and the palate is not completely developed. The eyelids are present in the shape of folds above and below the eye, and the different parts of the auricula are distinguishable. By the end of the 2nd month, the fetus measures from 28 to 30 millimeters in length. 3rd month. The head is extended, and the neck is lengthened. The eyelids meet and fuse, remaining closed until the end of the 6th month. The limbs are well developed, and nails appear on the digits. The external generative organs are so far differentiated that it is possible to distinguish the sex. By the end of this month, the length of the fetus is about 7 centimeters, but if the legs be included, it is from 9 to 10 centimeters. 4th month. The loop of gut which projected into the umbilical cord is withdrawn within the fetus. The hairs begin to make their appearance. There is a general increase in size, so that by the end of the 4th month, the fetus is from 12 to 13 centimeters in length, but if the legs be included, it is from 16 to 20 centimeters. 5th month. It is during this month that the first movements of the fetus are usually observed. The eruption of hair on the head commences, and the vernix caseosa begins to be deposited. By the end of this month, the total length of the fetus, including the legs, is from 25 to 27 centimeters. 6th month. The body is covered by fine hairs, lanugo, and the deposit of vernix caseosa is considerable. The papillae of the skin are developed, and the free border of the nail projects from the corium of the dermis. Measured from vertex to heels, the total length of the fetus at the end of this month is from 30 to 32 centimeters. 7th month. The pupillary membrane atrophies, and the eyelids are open. The testes descends with the vaginal sac of the peritoneum. From vertex to heels, the total length at the end of the 7th month is from 35 to 36 centimeters. The weight is a little over 3 pounds. 8th month. The skin assumes a pink colour, and is now entirely coated with vernix caseosa, and the langua begins to disappear. Subcutaneous fat has been developed to a considerable extent, and the fetus presents a plump appearance. The total length, that is, from head to heels, at the end of the 8th month is about 40 centimeters, and the weight varies between 4 1/2 and 5 1/2 pounds. 9th month. The langua has largely disappeared from the trunk. The umbilicus is almost in the middle of the body, and the testes are in the scrotum. At full time, the fetus weighs from 6 1/2 to 8 pounds, and measures from head to heels about 50 centimeters.
Osteology. Introduction. Development of the skeleton. The general framework of the body is built up mainly of a series of bones, supplemented, however, in certain regions by pieces of cartilage. The bony part of the framework constitutes the skeleton. In the skeleton of the adult there are 206 distinct bones, as follows. The axial skeleton, divided into the vertebral column with 26 bones, the skull with 22 bones, the hyoid bone 1, the ribs and sternum 25, 74 bones in total. The appendicular skeleton, upper extremities with 64 bones, lower extremities with 62 bones, 126 altogether, and the auditory ossicles 6, total of 206. The patellae are included in this enumeration, but the smaller sesamoid bones are not reckoned. Bones are divisible into 4 classes: long, short, flat, and irregular. Long bones. The long bones are found in the limbs, and each consists of a body or shaft and 2 extremities. The body, or diaphysis, is cylindrical, with a central cavity termed the medullary canal. The wall consists of dense, compact tissue of considerable thickness in the middle part of the body, but becoming thinner toward the extremities. Within the medullary canal is some cancellous tissue, scanty in the middle of the body, but greater in amount towards the ends. The extremities are generally expanded for the purpose of articulation and to afford broad surfaces for muscular attachment. They are usually developed from separate centres of ossification termed epiphyses, and consist of cancellous tissue surrounded by thin compact bone. The medullary canal and the spaces in the cancellous tissue are filled with marrow. The long bones are not straight, but curved, the curve generally taking place in 2 planes, thus affording greater strength to the bone. The bones belonging to this class are the clavicle, humerus, radius, ulna, femur, tibia, fibula, metacarpals, metatarsals, and phalanges. Short bones. Where a part of the skeleton is intended for strength and compactness combined with limited movement, it is constructed of a number of short bones, as in the carpus and tarsus. These consist of cancellous tissue, covered by a thin crust of compact substance. The patellae, together with the other sesamoid bones, are by some regarded as short bones. Flat bones. Where the principal requirement is either extensive protection or the provision of broad surfaces for muscular attachment, the bones are expanded into broad, flat plates, as in the skull and the scapula. These bones are composed of 2 thin layers of compact tissue enclosing between them a variable quantity of cancellous tissue. In the cranial bones, the layers of compact tissue are familiarly known as the tables of the skull. The outer one is thick and tough, the inner is thin, dense, and brittle, and hence is termed the vitreous table. The intervening cancellous tissue is called the diploe, and this, in certain regions of the skull, becomes absorbed so as to leave spaces filled with air sinuses, between the 2 tables. The flat bones are the occipital, parietal, frontal, nasal, lacrimal, vomer, scapular, os coxae, hip bone, sternum, ribs, and, according to some, the patella. Irregular bones. The irregular bones are such as, from their peculiar form, cannot be grouped under the preceding heads. They consist of cancellous tissue enclosed within a thin layer of compact bone. The irregular bones are the vertebrae, sacrum, coccyx, temporal, sphenoid, ethmoid, zygomatic, maxilla, mandible, palatine, inferior nasal concha, and hyoid. Surfaces of bones. If the surface of a bone be examined, certain eminences and depressions are seen. These eminences and depressions are of 2 kinds, articular and non-articular. Well-marked examples of articular eminences are found in the heads of the humerus and femur, and of articular depressions in the glenoid cavity of the scapula and the acetabulum of the hip bone. Non-articular eminences are designated according to their form. Thus a broad, rough, uneven elevation is called a tuberosity, protuberance, or process; a small, rough prominence, a tubercle; a sharp, slender, pointed eminence, a spine; a narrow, rough elevation, running some way along the surface, a ridge, crest, or line. Non-articular depressions are also of variable form and are described as fossae, pits, depressions, grooves, furrows, fissures, notches, etc. These non-articular eminences and depressions serve to increase the extent of surface for the attachment of ligaments and muscles, and are usually well marked in proportion to the muscularity of the subject. A short perforation is called a foramen, a longer passage a canal. 1. Development of the skeleton. The skeleton. The skeleton is of mesodermal origin, and may be divided into A. That of the trunk, axial skeleton, comprising the vertebral column, skull, ribs, and sternum, and B. That of the limbs, appendicular skeleton. The vertebral column. The notochord is a temporary structure and forms a central axis, around which the segments of the vertebral column are developed. Note 11. In the Amphioxus the notochord persists and forms the only representative of a skeleton in that animal. End of note. It is derived from the entoderm and consists of a rod of cells which lies on the ventral aspect of the neural tube and reaches from the anterior end of the midbrain to the extremity of the tail. On either side of it is a column of paraxial mesoderm which becomes subdivided into a number of more or less cubical segments, the primitive segments. These are separated from one another by intersegmental septa, and are arranged symmetrically on either side of the neural tube and notochord. To every segment a spinal nerve is distributed. At first each segment contains a central cavity, the myocoil, but this is soon filled with a core of angular and spindle-shaped cells. The cells of the segment become differentiated into three groups, which form respectively the cutis plate or dermatome, the muscle plate or myotome, and the sclerotome. The cutis plate is placed on the lateral and dorsal aspect of the myocoil, and from it the true skin of the corresponding segment is derived. The muscle plate is situated on the medial side of the cutis plate and furnishes the muscles of the segment. The cells of the sclerotome are largely derived from those forming the core of the myocoil, and lie next to the notochord. Fusion of the individual sclerotomes in an antero-posterior direction soon takes place, and thus a continuous strand of cells, the sclerotogenous layer, is formed along the ventrolateral aspects of the neural tube. The cells of this layer proliferate rapidly, and extending medialward surround the notochord. At the same time they grow backward on the lateral aspects of the neural tube and eventually surround it, and thus the notochord and neural tube are enveloped by a continuous sheath of mesoderm, which is termed the membranous vertebral column. In this mesoderm the original segments are still distinguishable, but each is now differentiated into two portions, an anterior, consisting of loosely arranged cells, and a posterior, of more condensed tissue. Between the two portions the rudiment of the intervertebral fibrocartilage is laid down. Cells from the posterior mass grow into the intervals between the myotomes of the corresponding and succeeding segments, and extend both dorsally and ventrally. The dorsal extensions surround the neural tube and represent the future vertebral arch, while the ventral extend into the body wall as the costal processes. The hinder part of the posterior mass joins the anterior mass of the succeeding segment to form the vertebral body. Each vertebral body is therefore a composite of two segments, being formed from the posterior portion of one segment and the anterior part of that immediately behind it. The vertebral and costal arches are derivatives of the posterior part of the segment in front of the intersegmental septum with which they are associated. This stage is succeeded by that of the cartilaginous vertebral column. In the fourth week two cartilaginous centres make their appearance, one on either side of the notochord. These extend around the notochord and form the body of the cartilaginous vertebra. A second pair of cartilaginous foci appear in the lateral parts of the vertebral bow, and grow backward on either side of the neural tube to form the cartilaginous vertebral arch, and a separate cartilaginous centre appears for each costal process. By the 8th week the cartilaginous arch has fused with the body, and in the 4th month the two halves of the arch are joined on the dorsal aspect of the neural tube. The spinous process is developed from the junction of the two halves of the vertebral arch. The transverse process grows out from the vertebral arch behind the costal process. In the upper cervical vertebrae a band of mesodermal tissue connects the ends of the vertebral arches across the ventral surfaces of the intervertebral fibrocartilages. This is termed the hypocaudal bar or brace. In all except the first it is transitory and disappears by fusing with the fibrocartilages. In the atlas, however, the entire bow persists and undergoes chondrification. It develops into the anterior arch of the bone, while the cartilage representing the body of the atlas forms the dens or odontoid process which fuses with the body of the second cervical vertebra. The portions of the notochord which are surrounded by the bodies of the vertebrae atrophy and ultimately disappear. while those which lie in the centres of the intervertebral fibrocartilages undergo enlargement, and persist throughout life as the central nucleus pulposus of the fibrocartilages. The ribs. The ribs are formed from the ventral or costal processes of the primitive vertebral bowels, the process extending between the muscle plates. In the thoracic region of the vertebral column, the costal processes grow lateralward to form a series of arches, the primitive costal arches. As already described, the transverse process grows out behind the vertebral end of each arch. It is at first connected to the costal process by continuous mesoderm, but this becomes differentiated later to form the costo-transverse ligament. Between the costal process and the tip of the transverse process, the costo-transverse joint is formed by absorption. The costal process becomes separated from the vertebral bow by the development of the costocentral joint. In the cervical vertebrae, the transverse process forms the posterior boundary of the foramen transversarium, while the costal process corresponding to the head and neck of the rib fuses with the body of the vertebra, and forms the anterolateral boundary of the foramen. The distal portions of the primitive costal arches remain undeveloped. Occasionally the arch of the seventh cervical vertebra undergoes greater development, and by the formation of costovertebral joints is separated off as a rib. In the lumbar region the distal portions of the primitive costal arches fail. The proximal portions fuse with the transverse processes to form the transverse processes of descriptive anatomy. Occasionally a movable rib is developed in connection with the first lumbar vertebra. In the sacral region costal processes are developed only in connection with the upper three, or it may be four, vertebrae. The processes of adjacent segments fuse with one another to form the lateral part of the sacrum. The coccygeal vertebrae are devoid of costal processes. The sternum. The ventral ends of the ribs become united to one another by a longitudinal bar termed the sternal plate, and opposite the first 7 pairs of ribs these sternal plates fuse in the middle line to form the manubrium and body of the sternum. The xiphoid process is formed by a backward extension of the sternal plates. The skull. Up to a certain age the development of the skull corresponds with that of the vertebral column, but it is modified later in association with the expansion of the brain vesicles, the formation of the organs of smell, sight, and hearing, and the development of the mouth and pharynx. The notochord extends as far forward as the anterior end of the midbrain, and becomes partly surrounded by mesoderm. The posterior part of this mesodermal investment corresponds with the basilar part of the occipital bone, and shows a subdivision into 4 segments, which are separated by the roots of the hypoglossal nerve. The mesoderm then extends over the brain vesicles, and thus the entire brain is enclosed by a mesodermal investment, which is termed the membraneous cranium. From the inner layer of this the bones of the skull and the membranes of the brain are developed. From the outer layer the muscles, blood vessels, true skin, and subcutaneous tissues of the scalp. In the shark and dogfish this membranous cranium undergoes complete chondrification and forms the cartilaginous skull or chondrocranium of these animals. In mammals, on the other hand, the process of chondrification is limited to the base of the skull, the roof and sides being covered in by membrane. Thus the bones of the base of the skull are preceded by cartilage, those of the roof and sides by membrane. The posterior part of the base of the skull is developed around the notochord and exhibits a segmented condition analogous to that of the vertebral column, while the anterior part arises in front of the notochord and shows no regular segmentation. The base of the skull may therefore be divided into a caudal or vertebral, and b. a precaudal or prevertebral portion. In the lower vertebrates 2 pairs of cartilages are developed, viz., a pair of paracaudal cartilages, one on either side of the notochord, and a pair of precaudal cartilages, the trabeculi cranii, in front of the notochord. The paracaudal cartilages unite to form a basilar plate, from which the cartilaginous part of the occipital bone and the bassus sphenoid are developed. On the lateral aspects of the paracaudal cartilages the auditory vesicles are situated, and the mesoderm enclosing them is soon converted into cartilage, forming the cartilaginous ear capsules. These cartilaginous ear capsules, which are of an oval shape, fuse with the sides of the basilar plate, and from them arise the petrous and mastoid portions of the temporal bones. The trabeculi cranii are two curved bars of cartilage which embrace the hypophysis cerebri. Their posterior ends soon unite with the basilar plate, while their anterior ends join to form the ethmoidal plate, which extends forwards between the forebrain and the olfactory pits. Later the trabeculi meet and fuse below the hypophysis, forming the floor of the fossa hypophyseos and so cutting off the anterior lobe of the hypophysis from the stomodeum. The median part of the ethmoidal plate forms the bony and cartilaginous parts of the nasal septum. From the lateral margins of the trabeculi cranii, three processes grow out on either side. The anterior forms the ethmoidal labyrinth, and the lateral and alar cartilages of the nose. The middle gives rise to the small wing of the sphenoid, while from the posterior the great wing and lateral pterygoid plate of the sphenoid are developed. The bones of the vault are of membranous formation, and are termed dermal or covering bones. They are partly developed from the mesoderm of the membranous cranium, and partly from that which lies outside the entoderm of the foregut. They comprise the upper part of the occipital squama, interparietal, the squamae and tympanic parts of the temporals, the parietals, the frontal, the vomer, the medial pterygoid plates, and the bones of the face. Some of them remain distinct throughout life, e.g. parietal and frontal, while others join with the bones of the chondrocranium, e.g. interparietal, squamae of the temporals, and medial pterygoid plates. Recent observations have shown that, in mammals, the basicranial cartilage, both in the caudal and precaudal regions of the base of the skull, is developed as a single plate which extends from behind forward. In man, however, its posterior part shows an indication of being developed from two chondrifying centres which fuse rapidly in front and below. The anterior and posterior thirds of the cartilage surround the notochord, but its middle third lies on the dorsal aspect of the notochord, which in this region is placed between the cartilage and the wall of the pharynx. Bone. Part 1. Structure and physical properties. Bone is one of the hardest structures of the animal body. It possesses also a certain degree of toughness and elasticity. Its colour, in a fresh state, is pinkish-white externally and deep red within. On examining a section of any bone, it is seen to be composed of two kinds of tissue, one of which is dense in texture, like ivory, and is termed compact tissue. The other consists of slender fibres and lamellae, which join to form a reticular structure. This, from its resemblance to lattice work, is called cancellous tissue. The compact tissue is always placed on the exterior of the bone, the cancellous in the interior. The relative quality of these two kinds of tissue varies in different bones, and in different parts of the same bone, according as strength or lightness is requisite. Close examination of the compact tissue shows it to be extremely porous, so that the difference in structure between it and the cancellous tissue depends merely upon the different amounts of solid matter, and the size and number of spaces in each. The cavities are small in the compact tissue and the solid matter between them abundant, while in cancellous tissue the spaces are large and the solid matter is in smaller quantity. Bone during life is permeated by vessels, and is enclosed, except where it is coated with articular cartilage, in a fibrous membrane, the periosteum, by means of which many of these vessels reach the hard tissue. If the periosteum be stripped from the surface of the living bone, small bleeding points are seen which mark the entrance of the periosteal vessels, and on section during life every part of the bone exudes blood from the minute vessels which ramify in it. The interior of each of the long bones of the limbs presents a cylindrical cavity filled with marrow and lined by a highly vascular areolar structure, called the medullary membrane. The strength of bone compared with other materials. Substance. Medium steel. Weight in pounds per cubic foot. 490. Ultimate strength pounds per square inch. Tension 65,000. Compression 60,000. Shear 40,000. Substance. Granite. Weight in pounds per cubic foot. 170. Ultimate strength pounds per square inch. Tension 1,500. Compression 15,000. Shear 2,000. Substance, oak, white. Weight in pounds per cubic foot, 46. Ultimate strength, pounds per square inch. Tension, 12,500. Footnote. Indicates stresses with the grain, i.e., when the load is parallel to the long axis of the material, or parallel to the direction of the fibres of the material. End footnote. Compression, 7,000. Shear, 4,000. Footnote. Indicates unit stresses across the grain, i.e., at right angles to the direction of the fibres of the material. End footnote. Substance, compact bone, low. Weight in pounds per cubic foot, 119. Ultimate strength, pounds per square inch. Tension, 13,200. Compression, 18,000. Shear, 11,800. Substance, compact bone, high. Weight in pounds per cubic foot. Left blank. Ultimate strength, pounds per square inch. Tension, 17,700. Compression, 24,000. Shear, 7,150. Periosteum. The periosteum adheres to the surface of each of the bones in nearly every part, but not to cartilaginous extremities. When strong tendons or ligaments are attached to a bone, the periosteum is incorporated with them. It consists of two layers closely united together, the outer one formed chiefly of connective tissue, containing occasionally a few fat cells, the inner one of elastic fibres of the finer kind, forming dense membranous networks, which again can be separated into several layers. In young bones the periosteum is thick and very vascular, and is intimately connected at either end of the bone with the epiphyseal cartilage, but less closely with the body of the bone, from which it is separated by a layer of soft tissue, containing a number of granular corpuscles or osteoblasts, by which ossification proceeds on the exterior of the young bone. Later in life the periosteum is thinner and less vascular, and the osteoblasts are converted into an epithelioid layer on the deep surface of the periosteum. The periosteum serves as a nidus for the ramification of the vessels previous to their distribution in the bone, hence the liability of bone to exfoliation or necrosis when denuded of this membrane by injury or disease. Fine nerves and lymphatics, which generally accompany the arteries, may also be demonstrated in the periosteum. Marrow. The marrow not only fills up the cylindrical cavities in the bodies of the long bones, but also occupies the spaces of the cancellous tissue and extends into the larger bony canals, Haversian canals, which contain the blood vessels. It differs in composition in different bones. In the bodies of the long bones the marrow is of a yellow colour and contains, in 100 parts, 96 of fat, 1 of areolar tissue and vessels, and 3 of fluid with extractive matter. It consists of a basis of connective tissue supporting numerous blood vessels and cells, most of which are fat cells, but some are marrow cells, such as occur in the red marrow to be immediately described. In the flat and short bones, the articular ends of the long bones, in the bodies of the vertebrae, in the cranial diploe, and in the sternum and ribs of the marrow is of a red colour, and contains in 100 parts, 75 of water, and 25 of solid matter consisting of cell globulin, nucleoprotein, extractives, salts, and only a small proportion of fat. The red marrow consists of a small quantity of connective tissue, blood vessels, and numerous cells, some few of which are fat cells, but the great majority are roundish nucleated cells, the true marrow cells of Colica. These marrow cells proper, or myelocytes, resemble in appearance lymphoid corpuscles, and like them are amoeboid. They generally have a hyaline protoplasm, though some show granules either oxyphil or basophil in reaction. A number of eosinophil cells are also present. Among the marrow cells may be seen smaller cells, which possess a slightly pinkish hue. These are the erythroblasts or normoblasts, from which the red corpuscles of the adult are derived, and which may be regarded as descendants of the nucleated coloured corpuscles of the embryo. Giant cells, myeloplaxes, osteoclasts, large, multinucleated protoplasmic masses are also to be found in both sorts of adult marrow, but more particularly in red marrow. They were believed by Kolliker to be concerned in the absorption of bone matrix, and hence the name which he gave to them, osteoclasts. They excavate in the bone small shallow pits or cavities, which are named house-ships foveolae, and in these they are found lying. Vessels and nerves of bone. The blood vessels of bone are very numerous. Those of the compact tissue are derived from a close and dense network of vessels ramifying in the periosteum. From this membrane vessels pass into the minute orifices in the compact tissue, and run through the canals which traverse its substance. The cancellous tissue is supplied in a similar way, but by less numerous and larger vessels, which, perforating the outer compact tissue, are distributed to the cavities of the spongy portion of the bone. In the long bones, numerous apertures may be seen at the ends near the articular surfaces. Some of these give passage to the arteries of the larger set of vessels referred to, but the most numerous and largest apertures are for some of the veins of the cancellous tissue, which emerge apart from the arteries. The marrow in the body of a long bone is supplied by one large artery, or sometimes more, which enters the bone at the nutrient foramen, situated in most cases near the centre of the body, and perforates obliquely the compact structure. The medullary or nutrient artery, usually accompanied by one or two veins, sends branches upward and downward, which ramify in the medullary membrane, and give twigs to the adjoining canals. The ramifications of this vessel anastomose with the arteries of the cancellous and compact tissues. In most of the flat, and in many of the short spongy bones, one or more large apertures are observed, which transmit to the central parts of the bone vessels corresponding to the nutrient arteries and veins. The veins emerge from the long bones in three places. Kolliker. 1. One or two large veins accompany the artery. 2. Numerous large and small veins emerge at the articular extremities. 3. Many small veins pass out of the compact substance. In the flat cranial bones the veins are large, very numerous, and run in tortuous canals in the diploic tissue, the sides of the canals being formed by thin lamellae of bone, perforated here and there for the passage of branches from the adjacent cancelli. The same condition is also found in all cancellous tissue, the veins being enclosed and supported by osseous material and having exceedingly thin coats. When a bone is divided, the vessels remain patulous, and do not contract in the canals in which they are contained. Lymphatic vessels, in addition to those found in the periosteum, have been traced by Cruikshank into the substance of bone, and Klein describes them as running in the Haversian canals. Nerves are distributed freely to the periosteum, and accompany the nutrient arteries into the interior of the bone. They are said by Kolliker to be most numerous in the articular extremities of the long bones, in the vertebrae, and in the larger flat bones. Minute anatomy. A transverse section of dense bone may be cut with a saw and ground down until it is sufficiently thin. If this be examined with a rather low power, the bone will be seen to be mapped out into a number of circular districts, each consisting of a central hole surrounded by a number of concentric rings. These districts are termed Haversian systems. The central hole is an Haversian canal, and the rings are layers of bony tissue arranged concentrically around the central canal and termed lamellae. Moreover, on closer examination it will be found that between these lamellae, and therefore also arranged concentrically around the central canal, are a number of little dark spots, the lacunae, and that these lacunae are connected with each other and with the central Haversian canal by a number of fine dark lines, which radiate like the spokes of a wheel and are called canaliculi. Filling in the irregular intervals which are left between these circular systems are other lamellae, with their lacunae and canaliculi running in various directions, but more or less curved. They are termed interstitial lamellae. Again, other lamellae, found on the surface of the bone, are arranged parallel to its circumference. They are termed circumferential, or by some authors primary or fundamental lamellae, to distinguish them from those laid down around the axis of the Haversian canals, which are then termed secondary, or special lamellae. The Haversian canals, seen in a transverse section of bone as round holes at or about the centre of each Haversian system, may be demonstrated to be true canals if a longitudinal section be made. It will then be seen that the canals run parallel with the longitudinal axis of the bone for a short distance and then branch and communicate. They vary considerably in size, some being as much as 0.12 mm in diameter. The average size is, however, about 0.05 mm. Near the medullary cavity the canals are larger than those near the surface of the bone. Each canal contains 1 or 2 blood vessels, with a small quantity of delicate connective tissue and some nerve filaments. In the larger ones there are also lymphatic vessels, and cells with branching processes which communicate, through the canaliculi, with the branched processes of certain bone cells in the substance of the bone. Those canals near the surface of the bone open upon it by minute orifices, and those near the medullary cavity open in the same way into this space, so that the whole of the bone is permeated by a system of blood vessels running through the bony canals in the centres of the Haversian systems. The lamellae are thin plates of bony tissue encircling the central canal, and may be compared, for the sake of illustration, to a number of sheets of paper pasted one over another around a central hollow cylinder. After macerating a piece of bone in dilute mineral acid, these lamellae may be stripped off in a longitudinal direction as thin films. If one of these be examined with a high power of the microscope, it will be found to be composed of a finely reticular structure, made up of very slender transparent fibres, decussating obliquely, and coalescing at the points of intersection. These fibres are composed of fine fibrils identical with those of white connective tissue. The intercellular matrix between the fibres is impregnated by calcareous deposit which the acid dissolves. In many places the various lamellae may be seen to be held together by tapering fibres, which run obliquely through them, pinning or bolting them together. They were first described by Sharpie, and were named by him perforating fibres. The lacunae are situated between the lamellae, and consist of a number of oblong spaces. In an ordinary microscopic section, viewed by transmitted light, they appear as fusiform opaque spots. Each lacuna is occupied during life by a branched cell, termed a bone cell or bone corpuscle, the processes from which extend into the canaliculi. The canaliculi are exceedingly minute channels, crossing the lamellae and connecting the lacunae with neighbouring lacunae and also with the Haversian canal. From the Haversian canal a number of canaliculi are given off, which radiate from it and open into the first set of lacunae between the first and second lamellae. From these lacunae a second set of canaliculi is given off. These run outward to the next series of lacunae, and so on until the periphery of the Haversian system is reached. Here the canaliculi given off from the last series of lacunae do not communicate with the lacunae of neighbouring Haversian systems, but after passing outward for a short distance form loops and return to their own lacunae. Thus every part of an Haversian system is supplied with nutrient fluids derived from the vessel in the Haversian canal and distributed to the canaliculi and lacunae. The bone cells are contained in the lacunae, which however they do not completely fill. They are flattened and nucleated branched cells, homologous with those of connected tissue. The branches, especially in young bones, pass into the canaliculi from the lacunae. In thin plates of bone, as in the walls of the spaces of cancellous tissue, the Haversian canals are absent, and the canaliculi open into the spaces of the cancellous tissue, medullary spaces, which thus have the same function as the Haversian canals. Bone, Part 2. Chemical composition. Bone consists of an animal and an earthy part intimately combined together. The animal part may be obtained by immersing a bone for a considerable time in dilute mineral acid, after which process the bone comes out exactly the same shape as before, but perfectly flexible, so that a long bone, one of the ribs for example, can easily be tied in a knot. If now a transverse section is made, the same general arrangement of the Haversian canals, lamellae, lacunae, and canaliculi is seen. The earthy part may be separately obtained by calcination, by which the animal matter is completely burnt out. The bone will still retain its original form, but it will be white and brittle, will have lost about 1/3 of its original weight, and will crumble down with the slightest force. The earthy matter is composed chiefly of calcium phosphate, about 58% of the weight of the bone, calcium carbonate about 7%, calcium fluoride and magnesium phosphate from 1 to 2% each, and sodium chloride less than 1%. They confer on bone its hardness and rigidity, while the animal matter, ossein, determines its tenacity. Ossification. Some bones are preceded by membrane, such as those forming the roof and sides of the skull. Others, such as the bones of the limbs, are preceded by rods of cartilage. Hence two kinds of ossification are described, the intramembranous and the intracartilaginous. Intramembranous ossification. In the case of bones which are developed in membrane, no cartilaginous mould precedes the appearance of the bony tissue. The membrane which occupies the place of the future bone is of the nature of connective tissue, and ultimately forms the periosteum. It is composed of fibres and granular cells in a matrix. The peripheral portion is more fibrous, while, in the interior, the cells or osteoblasts predominate. The whole tissue is richly supplied with blood vessels. At the outset of the process of bone formation, a little network of spicules is noticed radiating from the point or centre of ossification. These rays consist at their growing points of a network of fine clear fibres and granular corpuscles with an intervening ground substance. The fibres are termed osteogenic fibres, and are made up of fine fibrils differing little from those of white fibrous tissue. The membrane soon assumes a dark and granular appearance from the deposition of calcareous granules in the fibres and in the intervening matrix, and in the calcified material some of the granular corpuscles or osteoblasts are enclosed. By the fusion of the calcareous granules the tissue again assumes a more transparent appearance, but the fibres are no longer so distinctly seen. The involved osteoblasts from the corpuscles of the future bone, the spaces in which they are enclosed constituting the lacunae. The involved osteoblasts form the corpuscles of the future bone, the spaces in which they are enclosed constituting the lacunae. As the osteogenetic fibres grow out to the periphery they continue to calcify and give rise to fresh bone spicules. Thus a network of bone is formed, the meshes of which contain the blood vessels and a delicate connective tissue crowded with osteoblasts. The bony trabeculi thicken by the addition of fresh layers of bone formed by the osteoblasts on the surface, and the meshes are correspondingly encroached upon. Subsequently, successive layers of bony tissue are deposited under the periosteum and around the larger vascular channels which become the Haversian canals, so that the bone increases much in thickness. Intercartilaginous ossification. Just before ossification begins the mass is entirely cartilaginous, and in a long bone, which may be taken as an example, the process commences in the centre and proceeds towards the extremities, which for some time remain cartilaginous. Subsequently a similar process commences in one or more places in those extremities and gradually extends through them. The extremities do not, however, become joined to the body of the bone by bony tissue until growth has ceased. Between the body and either extremity a layer of cartilaginous tissue, termed the epiphyseal cartilage, persists for a definite period. The first step in the ossification of the cartilage is that the cartilage cells, at the point where ossification is commencing and which is termed a centre of ossification, enlarge and arrange themselves in rows. The matrix in which they are embedded increases in quantity, so that the cells become further separated from each other. A deposit of calcareous material now takes place in this matrix, between the rows of cells, so that they become separated from each other by longitudinal columns of calcified matrix, presenting a granular and opaque appearance. Here and there the matrix between two cells of the same row also become calcified, and transverse bars of calcified substance stretch from one calcareous column to another. Thus there are longitudinal groups of the cartilage cells enclosed in oblong cavities, the walls of which are formed of calcified matrix which cuts off all nutrition from the cells. The cells in consequence atrophy, leaving spaces called the primary areoli. At the same time that this process is going on in the centre of the solid bar of cartilage, certain changes are taking place on its surface. This is covered by a very vascular membrane, the perichondrium, entirely similar to the embryonic connective tissue already described as constituting the basis of membrane bone. On the inner surface of this, that is to say, on the surface in contact with the cartilage, are gathered the formative cells, the osteoblasts. By the agency of these cells a thin layer of bony tissue is formed between the perichondrium and the cartilage, by the intramembranous mode of ossification just described. There are then, in this first stage of ossification, two processes going on simultaneously in the centre of the cartilage, the formation of a number of oblong spaces, formed of calcified matrix and containing the withered cartilage cells, and on the surface of the cartilage the formation of a layer of true membrane bone. The second stage consists in the prolongation into the cartilage of processes of the deeper or osteogenetic layer of the perichondrium, which has now become periosteum. The processes consist of blood vessels and cells, osteoblasts or bone formers, and osteoclasts or bone destroyers. The latter are similar to the giant cells, myeloplaxes, found in marrow, and they excavate passages to the new formed bony layer by absorption, and pass through it into the calcified matrix. Wherever these processes come in contact with the calcified walls of the primary areolae, they absorb them, and thus cause a fusion of the original cavities and the formation of larger spaces, which are termed the secondary areolae or medullary spaces. These secondary spaces become filled with embryonic marrow, consisting of osteoblasts and vessels, derived, in the manner described above, from the osteogenetic layer of the periosteum. Thus far there has been traced the formation of enlarged spaces, secondary areolae, the perforated walls of which are still formed by calcified cartilage matrix, containing an embryonic marrow derived from the processes sent in from the osteogenetic layer of the periosteum, and consisting of blood vessels and osteoblasts. The walls of these secondary areolae are at this time of only inconsiderable thickness, but they become thickened by the deposition of layers of true bone on their surface. The process takes place in the following manner. Some of the osteoblasts of the embryonic marrow, after undergoing rapid division, arrange themselves as an epithelioid layer on the surface of the wall of the space. This layer of osteoblasts forms a bony stratum, and thus the wall of the space becomes gradually covered with a layer of true osseous substance, in which some of the bone-forming cells are included as bone corpuscles. The next stage in the process consists in the removal of these primary bone spicules by the osteoclasts. One of these giant cells may be found lying in a house ship's foveola at the free end of each spicule. The removal of the primary spicules go on pari passu with the formation of permanent bone by the periosteum, and in this way the medullary cavity of the body of the bone is formed. This series of changes has been gradually proceeding towards the end of the body of the bone, so that in the ossifying bone all the changes described above may be seen in different parts, from the true bone at the centre of the body to the hyaline cartilages at the extremities. While the ossification of the cartilaginous body is extending towards the articular ends, the cartilage immediately in advance of the osseous tissue continues to grow until the length of the adult bone is reached. During the period of growth the articular end, or epiphysis, remains for some time entirely cartilaginous. Then a bony centre appears, and initiates in it the process of intracartilaginous ossification, but this process never extends to any great distance. The epiphysis remains separated from the body by a narrow cartilaginous layer for a definite time. This layer ultimately ossifies. The distinction between body and epiphysis is obliterated, and the bone assumes its completed form and shape. The same remarks also apply to such processes of bone as are separately ossified, e.g. the trochanters of the femur. The bones therefore continue to grow until the body has acquired its full stature. They increase in length by ossification continuing to extend behind the epiphyseal cartilage, which goes on growing in advance of the ossifying process. They increase in circumference by deposition of new bone from the deeper layer of the periosteum on their external surface, and at the same time an absorption takes place from within, by which the medullary cavities are increased. The permanent bone formed by the periosteum when first laid down is cancellous in structure. Later the osteoblasts contained in its spaces become arranged in the concentric layers characteristic of the Haversian systems, and are included as bone corpuscles. The number of ossific centres varies in different bones. In most of the short bones ossification commences at a single point near the centre, and proceeds towards the surface. In the long bones there is a central point of ossification for the body or diaphysis, and one or more for each extremity, the epiphysis. That for the body is the first to appear. The times of union of the epiphysis with the body vary inversely with the dates at which their ossifications begin, with the exception of the fibula, and regulate the direction of the nutrient arteries of the bones. Thus the nutrient arteries of the bones of the arm and forearm are directed towards the elbow, since the epiphyses at this joint become united to the bodies before those at the opposite extremities. In the lower limb, on the other hand, the nutrient arteries are directed away from the knee, that is, upwards in the femur, downward in the tibia and fibula, and in them it is observed that the upper epiphyses of the femur and the lower epiphyses of the tibia and fibula unite first with the bodies. Where there is only one epiphysis, the nutrient artery is directed towards the other end of the bone, as towards the acromial end of the clavicle, towards the distal ends of the metacarpal bone of the thumb and the metatarsal bone of the great toe, and towards the proximal ends of the other metacarpal and metatarsal bones. Parsons groups epiphyses under three headings, viz. 1. Pressure epiphyses, appearing at the articular ends of the bones and transmitting the weight of the body from bone to bone. 2. Traction epiphyses, associated with the insertion of muscles and originally sesamoid structures though not necessarily sesamoid bones. And 3. Atavistic epiphyses, representing parts of the skeleton which at one time formed separate bones but which have lost their function and only appear as separate ossifications in early life.
The vertebral column. Columna vertebralis, spinal column. The vertebral column is a flexuous and flexible column formed of a series of bones called vertebrae. The vertebrae are 33 in number and are grouped under the names cervical, thoracic, lumbar, sacral, and coccygeal, according to the regions they occupy. There are 7 in the cervical region, 12 in the thoracic, 5 in the lumbar, 5 in the sacral, and 4 in the coccygeal. This number is sometimes increased by an additional vertebra in one region, or it may be diminished in one region, the deficiency often being supplied by an additional vertebra in another. The number of cervical vertebrae is, however, very rarely increased or diminished. The vertebrae in the upper 3 regions of the column remain distinct throughout life, and are known as true or movable vertebrae. Those of the sacral and coccygeal regions, on the other hand, are termed false or fixed vertebrae, because they are united with one another in the adult to form 2 bones, 5 forming the upper bone or sacrum, and 4 the terminal bone or coccyx. With the exception of the first and second cervical, the true or movable vertebrae present certain common characteristics, which are best studied by examining one from the middle of the thoracic region. General characteristics of a vertebra. A typical vertebra consists of 2 essential parts, namely, an anterior segment, the body, and a posterior part, the vertebral or neural arch. These enclose a foramen, the vertebral foramen. The vertebral arch consists of a pair of pedicles and a pair of laminae, and supports 7 processes, namely, 4 articular, 2 transverse, and 1 spinous. When the vertebrae are articulated with each other, the bodies form a strong pillar for the support of the head and trunk, and the vertebral foramina constitute a canal for the protection of the medulla spinalis, or spinal cord, while between every pair of vertebrae are 2 apertures, the intervertebral foramina, 1 on each side, for the transmission of the spinal nerves and vessels. Body, corpus vertebrae. The body is the largest part of a vertebra, and is more or less cylindrical in shape. Its upper and lower surfaces are flattened and rough, and give attachment to the intervertebral fibrocartilages, and each presents a rim around its circumference. In front, the body is convex from side to side, and concave from above downward. Behind, it is flat from above downward, and slightly concave from side to side. Its anterior surface presents a few small apertures for the passage of nutrient vessels. On the posterior surface is a single large irregular aperture, or occasionally more than one, for the exit of the basovertebral veins from the body of the vertebra. Pedicles. Radices archi vertebrae. The pedicles are 2 short, thick processes which project backward, one on either side, from the upper part of the body, at the junction of its posterior and lateral surfaces. The concavities above and below the pedicles are named the vertebral notches, and when the vertebrae are articulated, the notches of each contiguous pair of bones form the intervertebral foramina already referred to. Laminae. The laminae are 2 broad plates directed backward and medialward from the pedicles. They fuse in the middle line posteriorly, and so complete the posterior boundary of the vertebral foramen. Their upper borders and the lower parts of their anterior surfaces are rough for the attachment of the ligmenta flava. Processes. Spinous process. Processus spinosus. The spinous process is directed backward and downward from the junction of the laminae, and serves for the attachment of muscles and ligaments. Articular processes. The articular processes, two superior and two inferior, spring from the junctions of the pedicles and laminae. The superior project upward, and their articular surfaces are directed more or less backward. The inferior project downward, and their surfaces look more or less forward. The articular surfaces are coated with hyaline cartilage. Transverse processes. Processus transversi. The transverse processes, two in number, project one at either side from the point where the lamina joins the pedicle, between the superior and inferior articular processes. They serve for the attachment of muscles and ligaments. Structure of a vertebra. The body is composed of cancellous tissue covered by a thin coating of compact bone. The latter is perforated by numerous orifices, some of large size for the passage of vessels. The interior of the bone is traversed by one or two large canals for the reception of veins, which converge toward a single large irregular aperture, or several small apertures at the posterior part of the body. The thin bony lamellae of the cancellous tissue are more pronounced in lines perpendicular to the upper and lower surfaces, and are developed in response to greater pressure in this direction. The arch and processes projecting from it have thick coverings of compact tissue. The cervical vertebrae. Vertebrae cervicales. Cervical vertebrae are the smallest of the true vertebrae, and can be readily distinguished from those of the thoracic or lumbar regions by the presence of a foramen in each transverse process. The first, second, and seventh present exceptional features and must be separately described. The following characteristics are common to the remaining four. The body is small and broader from side to side than from before backward. The anterior and posterior surfaces are flattened and of equal depth. The former is placed on a lower level than the latter, and its inferior border is prolonged downward, so as to overlap the upper and fore part of the vertebra below. The upper surface is concave transversely, and presents a projecting lip on either side. The lower surface is concave from before backward, convex from side to side, and presents laterally shallow concavities which receive the corresponding projecting lips of the subjacent vertebra. The pedicles are directed lateralward and backward, and are attached to the body midway between its upper and lower borders, so that the superior vertebral notch is as deep as the inferior, but it is at the same time narrower. The laminae are narrow and thinner above than below. The vertebral foramen is large and of a triangular form. The spinous process is short and bifid, the two divisions being often of unequal size. The superior and inferior articular processes on either side are fused to form an articular pillar, which projects lateralward from the junction of the pedicle and lamina. The articular facets are flat and of an oval form. The superior look backward, upward, and slightly medialward. The inferior forward, downward, and slightly lateralward. The transverse processes are each pierced by the foramen transversarium, which in the upper six vertebrae gives passage to the vertebral artery and vein and a plexus of sympathetic nerves. Each process consists of an anterior and a posterior part. The anterior portion is the homologue of the rib in the thoracic region, and is therefore named the costal process or costal element. It arises from the side of the body, and is directed lateralward in front of the foramen, and ends in a tubercle, the anterior tubercle. The posterior part, the true transverse process, springs from the vertebral arch behind the foramen, and is directed forward and lateralward. It ends in a flattened vertical tubercle, the posterior tubercle. These two parts are joined outside the foramen by a bar of bone which exhibits a deep sulcus on its upper surface for the passage of the corresponding spinal nerve. Footnote: the costal element of a cervical vertebra not only includes the portion which springs from the side of the body, but the anterior and posterior tubercles and the bar of bone which connects them. End of footnote. First cervical vertebra. The first cervical vertebra is named the atlas because it supports the globe of the head. Its chief peculiarity is that it has no body, and this is due to the fact that the body of the atlas has fused with that of the next vertebra. Its other peculiarities are that it has no spinous process, is ring-like, and consists of an anterior and a posterior arch and two lateral masses. The anterior arch forms about 1/5 of the ring. Its anterior surface is convex and presents at its center the anterior tubercle for the attachment of the longus colli muscles. Posteriorly it is concave and marked by a smooth oval or circular facet, fovea dentis, for articulation with the odontoid process, dens, of the axis. The upper and lower borders respectively give attachment to the anterior atlanto-occipital membrane and the anterior atlantoaxial ligament. The former connects it with the occipital bone above, and the latter with the axis below. The posterior arch forms about 2/5 of the circumference of the ring. It ends behind in the posterior tubercle, which is the rudiment of a spinous process, and gives origin to the recti capitis posterioris minoris. The diminutive size of this process prevents any interference with the movements between the atlas and the skull. The posterior part of the arch presents above and behind a rounded edge for the attachment of the posterior atlanto-occipital membrane, while immediately behind each superior articular process is a groove, sulcus arteriae vertebralis, sometimes converted into a foramen by a delicate bony spiculum which arches backward from the posterior end of the superior articular process. This groove represents the superior vertebral notch, and serves for the transmission of the vertebral artery, which, after ascending through the foramen in the transverse process, winds around the lateral mass in a direction backward and medialward. It also transmits the suboccipital first spinal nerve. On the undersurface of the posterior arch, behind the articular facets, are two shallow grooves, the inferior vertebral notches. The lower border gives attachment to the posterior atlantoaxial ligament, which connects it with the axis. The lateral masses are the most bulky and solid parts of the atlas, in order to support the weight of the head. Each carries two articular facets, a superior and an inferior. The superior facets are of large size, oval, concave, and approach each other in front, but diverge behind. They are directed upward, medialward, and a little backward, each forming a cup for the corresponding condyle of the occipital bone, and are admirably adapted to the nodding movements of the head. Not infrequently they are partially subdivided by indentations which encroach upon their margins. The inferior articular facets are circular in form, flattened or slightly convex, and directed downward and medialward, articulating with the axis and permitting the rotary movements of the head. Just below the medial margin of each superior facet is a small tubercle, for the attachment of the transverse atlantal ligament which stretches across the ring of the atlas and divides the vertebral foramen into two unequal parts, the anterior or smaller receiving the odontoid process of the axis, the posterior transmitting the medulla spinalis and its membranes. This part of the vertebral canal is of considerable size, much greater than is required for the accommodation of the medulla spinalis, and hence lateral displacement of the atlas may occur without compression of this structure. The transverse processes are large, they project lateralward and downward from the lateral masses, and serve for the attachment of muscles which assist in rotating the head. They are long, and their anterior and posterior tubercles are fused into one mass. The foramen transversarium is directed from below, upward, and backward. Second cervical vertebra. The second cervical vertebra is named the epistrophius or axis because it forms the pivot upon which the first vertebra, carrying the head, rotates. The most distinctive characteristic of this bone is the strong odontoid process which rises perpendicularly from the upper surface of the body. The body is deeper in front than behind, and prolonged downward anteriorly so as to overlap the upper and fore part of the third vertebra. It presents in front a median longitudinal ridge separating two lateral depressions for the attachment of the longus colli muscles. Its undersurface is concave from before backward and convex from side to side. The dens or odontoid process exhibits a slight constriction or neck where it joins the body. On its anterior surface is an oval or nearly circular facet for articulation with that on the anterior arch of the atlas. On the back of the neck and frequently extending onto its lateral surfaces is a shallow groove for the transverse atlantal ligament, which retains the process in position. The apex is pointed and gives attachment to the apical odontoid ligament. Below the apex the process is somewhat enlarged and presents on either side a rough impression for the attachment of the alar ligament. These ligaments connect the process to the occipital bone. The internal structure of the odontoid process is more compact than that of the body. The pedicles are broad and strong, especially in front, where they coalesce with the sides of the body and the root of the odontoid process. They are covered above by the superior articular surfaces. The laminae are thick and strong, and the vertebral foramen large, but smaller than that of the atlas. The transverse processes are very small, and each ends in a single tubercle. Each is perforated by the foramen transversarium, which is directed obliquely upward and lateralward. The superior articular surfaces are round, slightly convex, directed upward and lateralward, and are supported on the body, pedicles, and transverse processes. The inferior articular surfaces have the same direction as those of the other cervical vertebrae. The superior vertebral notches are very shallow and lie behind the articular processes. The inferior lie in front of the articular processes, as in the other cervical vertebrae. The spinous process is large, very strong, deeply channeled on its under surface, and presents a bifid, tuberculated extremity. The seventh cervical vertebra. The most distinct characteristic of this vertebra is the existence of a long and prominent spinous process, hence the name vertebra prominens. This process is thick, nearly horizontal in direction, not bifurcated, but terminating in a tubercle into which the lower end of the ligamentum nuchae is attached. The transverse processes are of considerable size, their posterior roots are large and prominent, while the anterior are small and faintly marked. The upper surface of each has usually a shallow sulcus for the eighth spinal nerve, and its extremity seldom presents more than a trace of bifurcation. The foramen transversarium may be as large as that in the other cervical vertebrae, but is generally smaller on one or both sides. Occasionally it is double, sometimes it is absent. On the left side it occasionally gives passage to the vertebral artery. More frequently the vertebral vein traverses it on both sides, but the usual arrangement is for both artery and vein to pass in front of the transverse process and not through the foramen. Sometimes the anterior root of the transverse process attains a large size and exists as a separate bone, which is known as a cervical rib. The thoracic vertebrae. Vertebrae thoracales. The thoracic vertebrae are intermediate in size between those of the cervical and lumbar regions. They increase in size from above downward, the upper vertebrae being much smaller than those in the lower part of the region. They are distinguished by the presence of facets on the sides of the bodies for articulation with the heads of the ribs, and facets on the transverse processes of all, except the 11th and 12th, for articulation with the tubercles of the ribs. The bodies in the middle of the thoracic region are heart-shaped, and as broad in the anterior-posterior as in the transverse direction. At the ends of the thoracic region they resemble respectively those of the cervical and lumbar vertebrae. They are slightly thicker behind than in front, flat above and below, convex from side to side in front, deeply concave behind, and slightly constricted laterally and in front. They present on either side two costal demifacets, one above near the root of the pedicle, the other below, in front of the inferior vertebral notch. These are covered with cartilage in the fresh state, and when vertebrae are articulated with one another, form, with the intervening intervertebral fibrocartilages, oval surfaces for the reception of the heads of the ribs. The pedicles are directed backward and slightly upward, and the inferior vertebral notches are of a large size, deeper than in any other region of the vertebral column. The laminae are broad, thick, and imbricated, that is to say, they overlap those of subadjacent vertebrae like tiles on a roof. The vertebral foramen is small and of a circular form. The spinous process is long, triangular on coronal section, directed obliquely downward, and ends in a tuberculated extremity. These processes overlap from the 5th to the 8th, but are less oblique in direction above and below. The superior articular processes are thin plates of bone projecting upward from the junctions of the pedicles and laminae. Their articular facets are practically flat, and are directed backward and a little lateralward and upward. The inferior articular processes are fused to a considerable extent with the laminae, and project but slightly beyond their lower borders. Their facets are directed forward and a little medialward and downward. The transverse processes arise from the arch behind the superior articular processes and pedicles. They are thick, strong, and of considerable length, directed obliquely backward and lateralward, and each ends in a clubbed extremity, on the front of which is a small concave surface for articulation with the tubercle of a rib. The 1st, 9th, 10th, 11th, and 12th thoracic vertebrae present certain peculiarities and must be specially considered. The first thoracic vertebra has on either side of the body an entire articular facet for the head of the first rib, and a demi-facet for the upper half of the head of the second rib. The body is like that of a cervical vertebra, being broad transversely, its upper surface is concave, and lipped on either side. The superior articular surfaces are directed upward and backward. The spinous process is thick, long, and almost horizontal. The transverse processes are long, and the upper vertebral notches are deeper than those of the other thoracic vertebrae. The 9th thoracic vertebra may have no demi-facets below. In some subjects, however, it has 2 demi-facets on either side. When this occurs, the 10th has only demi-facets at the upper part. The 10th thoracic vertebra has, except in the cases just mentioned, an entire articular facet on either side, which is placed partly on the lateral surface of the pedicle. In the 11th thoracic vertebra, the body approaches in its form and size to that of the lumbar vertebrae. The articular facets for the heads of the ribs are of large size and placed chiefly on the pedicles, which are thicker and stronger in this and the next vertebra than in any other part of the thoracic region. The spinous process is short and nearly horizontal in direction. The transverse processes are very short, tuberculated at their extremities, and have no articular facets. The 12th thoracic vertebra has the same general characteristics as the 11th, but may be distinguished from it by its inferior articular surfaces being convex and directed lateralward, like those of the lumbar vertebrae. By the general form of the body, laminae and spinous process, in which it resembles the lumbar vertebrae, and by each transverse process being subdivided into 3 elevations, the superior, inferior, and lateral tubercles, the superior and inferior correspond to the mammillary and accessory processes of the lumbar vertebrae. Traces of similar elevations are found on the transverse processes of the 10th and 11th thoracic vertebrae. The lumbar vertebrae. Vertebrae lumbalis. The lumbar vertebrae are the largest segments of the movable part of the vertebral column, and can be distinguished by the absence of a foramen in the transverse process, and by the absence of facets on the sides of the body. The body is large, wider from side to side than from before backward, and a little thicker in front than behind. It is flattened or slightly concave above and below, concave behind, and deeply constricted in front and at the sides. The pedicles are very strong, directed backward from the upper part of the body. Consequently, the inferior vertebral notches are of considerable depth. The laminae are broad, short, and strong. The vertebral foramen is triangular, larger than in the thoracic, but smaller than in the cervical region. The spinous process is thick, broad, and somewhat quadrilateral. It projects backward and ends in a rough, uneven border, thickest below, where it is occasionally notched. The superior and inferior articular processes are well defined, projecting respectively upward and downward from the junctions of pedicles and laminae. The facets on the superior processes are concave and look backward and medialward. Those on the inferior are convex and are directed forward and lateralward. The former are wider apart than the latter, since in the articulated column the inferior articular processes are embraced by the superior processes of the subadjacent vertebra. The transverse processes are long, slender, and horizontal in the upper 3 lumbar vertebrae. They incline a little upward in the lower 2. In the upper 3 vertebrae they arise from the junctions of the pedicles and laminae, but in the lower 2 they are set farther forward and spring from the pedicles and posterior parts of the bodies. They are situated in front of the articular processes instead of behind them, as in the thoracic vertebrae, and are homologous with the ribs. Of the 3 tubercles noticed in connection with the transverse processes of the lower thoracic vertebrae, the superior one is connected in the lumbar region with the back part of the superior articular process, and is named the mammillary process. The inferior is situated at the back part of the base of the transverse process and is called the accessory process. The fifth lumbar vertebra is characterized by its body being much deeper in front than behind, which accords with the prominence of the sacrovertebral articulation, by the smaller size of its spinous process, by the wide interval between the inferior articular processes, and by the thickness of its transverse processes, which spring from the body as well as from the pedicles. The sacral and coccygeal vertebrae. The sacral and coccygeal vertebrae consist at an early period of life of 9 separate segments, which are united in the adult, so as to form 2 bones, 5 entering into the formation of the sacrum, 4 into that of the coccyx. Sometimes the coccyx consists of 5 bones, occasionally the number is reduced to 3. The sacrum, os sacrum. The sacrum is a large triangular bone, situated in the lower part of the vertebral column and at the upper and back part of the pelvic cavity, where it is inserted like a wedge between the 2 hip bones. Its upper part or base articulates with the last lumbar vertebra, its apex with the coccyx. It is curved upon itself and placed very obliquely, its base projecting forward and forming the prominent sacrovertebral angle when articulated with the last lumbar vertebra. Its central part is projected backward so as to give increased capacity to the pelvic cavity. Pelvic surface, facies pelvina. The pelvic surface is concave from above downward and slightly so from side to side. Its middle part is crossed by 4 transverse ridges, the positions of which correspond with the original planes of separation between the 5 segments of the bone. The portions of bone intervening between the ridges are the bodies of the sacral vertebrae. The body of the first segment is of large size, and in form resembles that of a lumbar vertebra. The succeeding ones diminish from above downward, are flattened from before backward, and curved so as to accommodate themselves to the form of the sacrum, being concave in front, convex behind. At the ends of the ridges are seen the anterior sacral foramina, 4 in number on either side, somewhat rounded in form, diminishing in size from above downward, and directed lateralward and forward. They give exit to the anterior divisions of the sacral nerves and entrance to the lateral sacral arteries. Lateral to these foramina are the lateral parts of the sacrum, each consisting of 5 separate segments at an early period of life. In the adult, these are blended with the bodies and with each other. Each lateral part is traversed by 4 broad, shallow grooves, which lodge the anterior divisions of the sacral nerves, and are separated by prominent ridges of bone, which give origin to the piriformis muscle. If a sagittal section be made through the center of the sacrum, the bodies are seen to be united at their circumferences by bone, wide intervals being left centrally, which, in the fresh state, are filled by the intervertebral fibrocartilages. In some bones, this union is more complete between the lower than the upper segments. Dorsal surface, facies dorsalis. The dorsal surface is convex and narrower than the pelvic. In the middle line it displays a crest, the middle sacral crest, surmounted by 3 or 4 tubercles, the rudimentary spinous processes of the upper 3 or 4 sacral vertebrae. On either side of the middle sacral crest is a shallow groove, the sacral groove, which gives origin to the multifidus, the floor of the groove being formed by the united laminae of the corresponding vertebrae. The laminae of the 5th sacral vertebra, and sometimes those of the 4th, fail to meet behind, and thus a hiatus or deficiency occurs in the posterior wall of the sacral canal. On the lateral aspect of the sacral groove is a linear series of tubercles, produced by the fusion of the articular processes, which together form the indistinct sacro-articular crests. The articular processes of the 1st sacral vertebra are large and oval in shape. Their facets are concave from side to side, look backward and medialward, and articulate with the facets on the inferior processes of the 5th lumbar vertebra. The tubercles which represent the inferior articular processes of the 5th sacral vertebra are prolonged downward as rounded processes, which are named the sacrocornua, and are connected to the cornua of the coccyx. Lateral to the articular processes are the 4 posterior sacral foramina. They are smaller in size and less regular in form than the anterior, and transmit the posterior divisions of the sacral nerves. On the lateral side of the posterior sacral foramina is a series of tubercles, which represent the transverse processes of the sacral vertebrae and form the lateral crest of the sacrum. The transverse tubercles of the first sacral vertebra are large and very distinct. They, together with the transverse tubercles of the second vertebra, give attachment to the horizontal parts of the posterior sacroiliac ligaments. Those of the third vertebra give attachment to the oblique fasciculi of the posterior sacroiliac ligaments, and those of the fourth and fifth to the sacrotuberous ligaments. Lateral surface. The lateral surface is broad above, but narrowed into a thin edge below. The upper half presents in front an ear-shaped surface, the auricular surface, covered with cartilage in the fresh state, for articulation with the ilium. Behind is a rough surface, the sacral tuberosity, on which are 3 deep and uneven impressions for the attachment of the posterior sacroiliac ligament. The lower half is thin and ends in a projection called the inferior lateral angle. Medial to this angle is a notch, which is converted into a foramen by the transverse process of the first piece of the coccyx, and transmits the anterior division of the fifth sacral nerve. The thin lower half of the lateral surface gives attachment to the sacrotuberous and sacrospinous ligaments, to some fibers of the gluteus maximus behind, and to the coccygeus in front. Base. Basis os sacri. The base of the sacrum, which is broad and expanded, is directed upward and forward. In the middle is a large oval articular surface, the upper surface of the body of the first sacral vertebra, which is connected with the under surface of the body of the last lumbar vertebra by an intervertebral fibrocartilage. Behind this is the large triangular orifice of the sacral canal, which is completed by the laminae and spinous process of the first sacral vertebra. The superior articular processes project from it on either side. They are oval, concave, directed backward and medialward, like the superior articular processes of a lumbar vertebra. They are attached to the body of the first sacral vertebra and to the alae by short, thick pedicles. On the upper surface of each pedicle is a vertebral notch, which forms the lower part of the foramen between the last lumbar and first sacral vertebrae. On either side of the body is a large triangular surface, which supports the psoas major and lumbosacral trunk, and in the articulated pelvis is continuous with the iliac fossa. This is called the ala. It is slightly concave from side to side, convex from before backward, and gives attachment to a few of the fibers of the iliacus. The posterior fourth of the ala represents the transverse process, and its anterior 3/4 the costal process of the first sacral segment. Apex. Apex os sacri. The apex is directed downward and presents an oval facet for articulation with the coccyx. Vertebral canal. Canalis sacralis. Sacral canal. The vertebral canal runs throughout the greater part of the bone. Above it is triangular in form, below its posterior wall is incomplete, from the non-development of the laminae and spinous processes. It lodges the sacral nerves, and its walls are perforated by the anterior and posterior sacroforamina through which these nerves pass out. Structure. The sacrum consists of cancellous tissue enveloped by a thin layer of compact bone. Articulations. The sacrum articulates with 4 bones, the last lumbar vertebra above, the coccyx below, and the hip bone on either side. Differences in the sacrum of the male and female. In the female, the sacrum is shorter and wider than in the male. The lower half forms a greater angle with the upper half is nearly straight, the lower half presenting the greatest amount of curvature. The bone is also directed more obliquely backward. This increases the size of the pelvic cavity and renders the sacrovertebral angle more prominent. In the male, the curvature is more evenly distributed over the whole length of the bone, and is altogether greater than in the female. Variations. The sacrum in some cases consists of 6 pieces. Occasionally the number is reduced to 4. The bodies of the first and second vertebrae may fail to unite. Sometimes the uppermost transverse tubercles are not joined to the rest of the ala on one or both sides, or the sacral canal may be open through a considerable part of its length, in consequence of the imperfect development of the laminae and spinous processes. The sacrum also varies considerably with respect to its degree of curvature. The coccyx, os coccygeus. The coccyx is usually formed of 4 rudimentary vertebrae. The number may, however, be increased to 5 or diminished to 3. In each of the first 3 segments may be traced a rudimentary body and articular and transverse processes. The last piece, sometimes the third, is a mere nodule of bone. All the segments are destitute of pedicles, laminae, and spinous processes. The first is the largest. It resembles the lowest sacral vertebra and often exists as a separate piece. The last 3 diminish in size from above downward and are usually fused with one another. Surfaces. The anterior surface is slightly concave and marked with 3 transverse grooves, which indicate the junctions of the different segments. It gives attachment to the anterior sacrococcygeal ligament and the levatorus ani, and supports part of the rectum. The posterior surface is convex, marked by transverse grooves similar to those on the anterior surface, and presents on either side a linear row of tubercles, the rudimentary articular processes of the coccygeal vertebrae. Of these, the superior pair are large, and are called the coccygeal cornua. They project upward, and articulate with the cornua of the sacrum, and on either side complete the foramen for the transmission of the posterior division of the 5th sacral nerve. Borders. The lateral borders are thin and exhibit a series of small eminences, which represent the transverse processes of the coccygeal vertebrae. Of these, the first is the largest. It is flattened from before backward, and often ascends to join the lower part of the thin lateral edge of the sacrum, thus completing the foramen for the transmission of the anterior division of the 5th sacral nerve. The others diminish in size from above downward, and are often wanting. The borders of the coccyx are narrow, and give attachment to either side to the sacrotuberous and sacrospinous ligaments, to the coccygeus in front of the ligaments, and to the gluteus maximus behind them. Base. The base presents an oval surface for articulation with the sacrum. Apex. The apex is rounded, and has attached to it the tendon of the sphincter ani externus. It may be bifid, and is sometimes deflected to one or the other side. Ossification of the vertebral column. Each cartilaginous vertebra is ossified from 3 primary centers, 2 for the vertebral arch and 1 for the body. Footnote. A vertebra is occasionally found in which the body consists of 2 lateral portions, a condition which proves that the body is sometimes ossified from 2 primary centers, 1 on either side of the middle line. End of footnote. Ossification of the vertebral arches begins in the upper cervical vertebrae about the 7th or 8th week of fetal life and gradually extends down the column. The ossific granules first appear in the situations where the transverse processes afterward project, and spread backward to the spinous process, forward into the pedicles, and lateralward into the transverse and articular processes. Ossification of the bodies begins about the 8th week in the lower thoracic region, and subsequently extends upward and downward along the column. The center for the body does not give rise to the whole of the body of the adult vertebra, the posterior lateral portions of which are ossified by extensions from the vertebral arch centers. The body of the vertebra during the first few years of life shows, therefore, 2 synchondroses. Neurocentral synchondroses, transversing it along the planes of junction of the 3 centers. In the thoracic region, the facets for the heads of the ribs lie behind the neurocentral synchondroses and are ossified from the centers for the vertebral arch. At birth, the vertebra consists of 3 pieces, the body and the halves of the vertebral arch. During the first year, the halves of the arch unite behind, union taking place first in the lumbar region and then extending upward through the thoracic and cervical regions. About the 3rd year, the bodies of the upper cervical vertebrae are joined to the arches on either side. In the lower lumbar vertebrae, the union is not completed until the 6th year. Before puberty, no other changes occur, excepting a gradual increase of these primary centers, the upper and under surfaces of the bodies, and the ends of the transverse and spinous processes being cartilaginous. About the 16th year, 5 secondary centers appear, 1 for the tip of each transverse process, 1 for the extremity of the spinous process, 1 for the upper and 1 for the lower surface of the body. These fuse with the rest of the bone about the age of 25. Exceptions to this mode of development occur in the 1st, 2nd, and 7th cervical vertebrae and in the lumbar vertebrae. Atlas. The atlas is usually ossified from 3 centers. Of these, one appears in each lateral mass about the 7th week of fetal life and extends backward. At birth, these portions of bone are separated from one another behind by a narrow interval filled with cartilage. Between the 3rd and 4th years, they unite either directly or through the medium of a separate center developing in the cartilage. At birth, the anterior arch consists of cartilage. In this, a separate center appears about the end of the 1st year after birth, and joins the lateral masses from the 6th to the 8th year, the lines of union extending across the anterior portions of the superior articular facets. Occasionally there is no separate center, the anterior arch being formed by the forward extension and ultimate junction of the 2 lateral masses. Sometimes this arch is ossified from 2 centers, one on either side of the middle line. Epistrophius or axis. The axis is ossified from 5 primary and 2 secondary centers. The body and vertebral arch are ossified in the same manner as the corresponding parts in the other vertebrae, namely, 1 center for the body and 2 for the vertebral arch. The centers for the arch appear about the 7th or 8th week of fetal life, that for the body about the 4th or 5th month. The dens or odontoid process consists originally of a continuation upward of the cartilaginous mass, in which the lower part of the body is formed. About the 6th month of fetal life, 2 centers make their appearance in the base of this process. They are placed laterally, and join before birth to form a conical bilobed mass deeply cleft above. The interval between the sides of the cleft and the summit of the process is formed by a wedge-shaped piece of cartilage. The base of the process is separated from the body by a cartilaginous disc, which gradually becomes ossified at its circumference, but remains cartilaginous in its center until advanced age. In this cartilage, rudiments of the lower epiphyseal lamella of the atlas and the upper epiphyseal lamella of the axis may sometimes be found. The apex of the odontoid process has a separate center which appears in the 2nd and joins about the 12th year. This is the upper epiphyseal lamella of the atlas. In addition to these, there is a secondary center for a thin epiphyseal plate on the undersurface of the body of the bone. The 7th cervical vertebra. The anterior or costal part of the transverse process of this vertebra is sometimes ossified from a separate center which appears about the 6th month of fetal life, and joins the body and posterior part of the transverse process between the 5th and 6th years. Occasionally the costal part persists as a separate piece, and becoming lengthened lateral and forward, constitutes what is known as a cervical rib. Separate ossific centers have also been found in the costal processes of the 4th, 5th, and 6th cervical vertebrae. Lumbar vertebrae. The lumbar vertebrae have each 2 additional centers for the mammillary processes. The transverse process of the first lumbar is sometimes developed as a separate piece, which may remain permanently ununited with the rest of the bone, thus forming a lumbar rib, a peculiarity, however, rarely met with. Sacrum. The body of each sacral vertebra is ossified from a primary center and 2 epiphyseal plates, one for its upper and another for its under surface, while each vertebral arch is ossified from 2 centers. The anterior portions of the lateral parts have 6 additional centers, 2 for each of the first 3 vertebrae. These represent the costal elements, and make their appearance above and lateral to the anterior sacral foramina. On each lateral surface 2 epiphyseal plates are developed, one for the auricular surface and another for the remaining part of the thin lateral edge of the bone. Footnote. The ends of the spinous processes of the 3 sacral vertebrae are sometimes developed from 3 separate epiphyses, and Fawcett states that a number of epiphyseal nodules may be seen in the sacrum at the age of 18 years. These are distributed as follows: 1 for each of the mammillary processes of the first sacral vertebra, 12, 6 on either side, in connection with the costal elements, 2 each for the first and second, and 1 each for the third and fourth, and 8 for the transverse processes, 4 on either side, 1 each for the first, third, fourth, and fifth. He is further of opinion that the lower part of each lateral surface of the sacrum is formed by the extension and union of the third and fourth costal and fourth and fifth transverse epiphyses. End of footnote. Periods of ossification. About the 8th or 9th week of fetal life, ossification of the central part of the body of the first sacrovertebra commences, and is rapidly followed by deposit of ossific matter in the second and third. Ossification does not commence in the bodies of the lower two segments until between the 5th and 8th months of fetal life. Between the 6th and 8th months, ossification of the vertebral arches takes place, and about the same time the costal centers for the lateral parts make their appearance. The junctions of the vertebral arches with the bodies take place in the lower vertebrae as early as the 2nd year, but are not affected in the uppermost until the 5th or 6th year. About the 16th year, the epiphyseal plates for the upper and under surfaces of the bodies are formed, and between the 18th and 20th years, those for the lateral surfaces make their appearance. The bodies of the sacrovertebrae are, during early life, separated from each other by intervertebral fibrocartilages, but about the 18th year the two lowest segments become united by bone, and the process of bony union gradually extends upward, with the result that between the 25th and 30th years of life all the segments are united. On examining a sagittal section of the sacrum, the situations of the intervertebral fibrocartilages are indicated by a series of oval cavities. Coccyx. The coccyx is ossified from 4 centers, one for each segment. The ossific nuclei make their appearance in the following order: in the first segment between the 1st and 4th years, in the second between the 5th and 10th years, in the third between the 10th and 15th years, in the fourth between the 14th and 20th years. As age advances, the segments unite with one another, the union between the first and second segments being frequently delayed until after the age of 25 or 30. At a late period of life, especially in females, the coccyx often fuses with the sacrum. The vertebral column as a whole. The vertebral column is situated in the median line as the posterior part of the trunk. Its average length in the male is about 71 centimeters. Of this length, the cervical part measures 12.5 centimeters, the thoracic about 28 centimeters, the lumbar 18 centimeters, and the sacrum and coccyx 12.5 centimeters. The female column is about 61 centimeters in length. Curves. Viewed laterally, the vertebral column represents several curves, which correspond to the different regions of the column, and are called cervical, thoracic, lumbar, and pelvic. The cervical curve, convex forward, begins at the apex of the odontoid process and ends at the middle of the second thoracic vertebra. It is the least marked of all the curves. The thoracic curve, concave forward, begins at the middle of the second and ends at the middle of the 12th thoracic vertebra. Its most prominent point behind corresponds to the spinous process of the 7th thoracic vertebra. The lumbar curve is more marked in the female than in the male. It begins at the middle of the last thoracic vertebra and ends at the sacrovertebral angle. It is convex anteriorly, the convexity of the lower 3 vertebrae being much greater than that of the upper 2. The pelvic curve begins at the sacrovertebral articulation and ends at the point of the coccyx. Its concavity is directed downward and forward. The thoracic and pelvic curves are termed primary curves because they alone are present during fetal life. The cervical and lumbar curves are compensatory or secondary and are developed after birth, the former when the child is able to hold up its head at 3 or 4 months and to sit upright at 9 months, the latter at 12 or 18 months when the child begins to walk. The vertebral column also has a slight lateral curvature, the convexity of which is directed toward the right side. This may be produced by muscular action, most persons using the right arm in preference to the left, especially in making long continued efforts, when the body is curved to the right side. In support of this explanation, it has been found that in 1 or 2 individuals who were left-handed, the convexity was to the left side. By others, this curvature is regarded as being produced by the aortic arch, an upper part of the descending thoracic aorta, a view which is supported by the fact that in cases where the viscera are transposed and the aorta is on the right side, the convexity of the curve is directed to the left side. Surfaces. Anterior surface. When viewed from in front, the width of the bodies of the vertebrae is seen to increase from the second cervical to the first thoracic. There is then a slight diminution in the next 3 vertebrae. Below this there is again a gradual and progressive increase in width as low as the sacrovertebral angle. From this point there is rapid diminution to the apex of the coccyx. Posterior surface. The posterior surface of the vertebral column represents in the median line the spinous processes. In the cervical region, with the exception of the second and seventh vertebrae, these are short and horizontal, with bifid extremities. In the upper part of the thoracic region they are directed obliquely downward, in the middle they are almost vertical, and in the lower part they are nearly horizontal. In the lumbar region they are nearly horizontal. The spinous processes are separated by considerable intervals in the lumbar region, by narrower intervals in the neck, and are closely approximated in the middle of the thoracic region. Occasionally one of these processes deviates a little from the median line, a fact to be remembered in practice, as irregularities of this sort are attendant also on fractures or displacements of the vertebral column. On either side of the spinous processes is the vertebral groove, formed by the laminae in the cervical and lumbar regions, where it is shallow, and by the laminae and transverse processes in the thoracic region, where it is deep and broad. These grooves lodge the deep muscles of the back. Lateral to the vertebral grooves are the articular processes, and still more laterally the transverse processes. In the thoracic region, the transverse processes stand backward, on a plane considerably behind that of the same processes in the cervical and lumbar regions. In the cervical region, the transverse processes are placed in front of the articular processes, lateral to the pedicles, and between the intervertebral foramina. In the thoracic region, they are posterior to the pedicles, intervertebral foramina, and articular processes. In the lumbar region, they are in front of the articular processes, but behind the intervertebral foramina. Lateral surfaces. The lateral surfaces are separated from the posterior surface by the articular processes in the cervical and lumbar regions, and by the transverse processes in the thoracic region. They present, in front, the sides of the bodies of the vertebrae, marked in the thoracic region by the facets for articulation with the heads of the ribs. More posteriorly are the intervertebral foramina, formed by the juxtaposition of the vertebral notches, oval in shape, smallest in the cervical and upper part of the thoracic regions, and gradually increasing in size to the last lumbar. They transmit the spinal nerves and are situated between the transverse processes in the cervical region and in front of them in the thoracic and lumbar regions. Vertebral canal. The vertebral canal follows the different curves of the column. It is large and triangular in those parts of the column which enjoy the greatest freedom of movement, namely the cervical and lumbar regions, and is small and rounded in the thoracic region where motion is more limited. Abnormalities. Occasionally the coalescence of the laminae is not completed, and consequently a cleft is left in the arches of the vertebrae, through which a protrusion of the spinal membranes, dura mater and arachnoid, and generally the medulla spinalis itself takes place, constituting the malformation known as spina bifida. This condition is most common in the lumbosacral region, but it may occur in the thoracic or cervical region, or the arches throughout the whole length of the canal may remain incomplete.
The thorax. The skeleton of the thorax or chest is an osseo-cartilaginous cage, containing and protecting the principal organs of respiration and circulation. It is conical in shape, being narrow above and broad below, flattened from before backward, and longer behind than in front. It is somewhat reniform on transverse section on account of the projection of the vertebral bodies into the cavity. Boundaries. The posterior surface is formed by the 12 thoracic vertebrae and the posterior parts of the ribs. It is convex from above downward and presents on either side of the middle line a deep groove, in consequence of the lateral and backward direction which the ribs take from the vertebral extremities to their angles. The anterior surface, formed by the sternum and costal cartilages, is flattened and slightly convex, and inclined from above downward and forward. The lateral surfaces are convex. They are formed by the ribs, separated from each other by the intercostal spaces, 11 in number, which are occupied by the intercostal muscles and membranes. The upper opening of the thorax is reniform in shape, being broader from side to side than from before backward. It is formed by the first thoracic vertebra behind, the upper margin of the sternum in front, and the first rib on either side. It slopes downward and forward, so that the anterior part of the opening is on a lower level than the posterior. Its antero-posterior diameter is about 5 centimeters, and its transverse diameter about 10 centimeters. The lower opening is formed by the 12th thoracic vertebra behind, by the 11th and 12th ribs at the sides, and in front by the cartilages of the 10th, 9th, 8th, and 7th ribs, which ascend on either side and form an angle, the subcostal angle, into the apex of which the xiphoid process projects. The lower opening is wider transversely than from before backward, and slopes obliquely downward and backward. It is closed by the diaphragm, which forms the floor of the thorax. The thorax of the female differs from that of the male as follows. 1. Its capacity is less. 2. The sternum is shorter. 3. The upper margin of the sternum is on a level with the lower part of the body of the 3rd thoracic vertebra, whereas in the male it is on a level with the lower part of the body of the 2nd. 4. The upper ribs are more movable, and so allow a greater enlargement of the upper part of the thorax. The sternum. Breastbone. The sternum is an elongated flattened bone forming the middle portion of the anterior wall of the thorax. Its upper end supports the clavicles, and its margins articulate with the cartilages of the first 7 pairs of ribs. It consists of 3 parts, named from above downward: the manubrium, the body or gladiolus, and the xiphoid process. In early life the body consists of 4 segments, or sternebrae. In its natural position the inclination of the bone is oblique from above, downward and forward. It is slightly convex in front and concave behind, broad above, becoming narrowed at the point where the manubrium joins the body, after which it again widens a little below the middle of the body, and then narrows to its lower extremity. Its average length in the adult is about 17 centimeters, and is rather greater in the male than in the female. Manubrium. Manubrium sterni. The manubrium is of a somewhat quadrangular form, broad and thick above, narrow below at its juncture with the body. Surfaces. Its anterior surface, convex from side to side, concave from above downward, is smooth, and affords attachment on either side to the sternal origins of the pectoralis major and sternocleidomastoidus. Sometimes the ridges limiting the attachments of these muscles are very distinct. Its posterior surface, concave and smooth, affords attachment on either side to the sternohyoidus and sternothyreoidus. Borders. The superior border is the thickest, and presents at its center the jugular or peristernal notch. On either side of the notch is an oval, articular surface, directed upward, backward, and lateralward, for articulation with the sternal end of the clavicle. The inferior border, oval and rough, is covered in a fresh state with a thin layer of cartilage, for articulation with the body. The lateral borders are each marked above by a depression for the first costal cartilage, and below by a small facet, which, with a similar facet on the upper angle of the body, forms a notch for the reception of the costal cartilage of the 2nd rib. Between the depression for the first costal cartilage and the demi-facet for the 2nd is a narrow curved edge, which slopes from above downward and medialward. Body. Corpus sterni. Gladiolus. The body, considerably longer, narrower, and thinner than the manubrium, attains its greatest breadth close to the lower end. Surfaces. Its anterior surface is nearly flat, directed upward and forward, and marked by 3 transverse ridges, which cross the bone opposite the 3rd, 4th, and 5th articular depressions. Footnote. Patterson, The Human Sternum, 1904, who examined 524 specimens, points out that these ridges are altogether absent in 23.7%, that in 69% a ridge exists opposite the 3rd costal attachment, and 39% opposite the 4th, 4% only opposite the 5th. End footnote. It affords attachment on either side of the sternal origin of the pectoralis major. At the junction of the 3rd and 4th pieces of the body is occasionally seen an orifice, the sternal foramen, of varying size and form. The posterior surface, slightly concave, is also marked by 3 transverse lines, less distinct, however, than those in front. From its lower part on either side, the transversus thoracis takes origin. Borders. The superior border is oval and articulates with the manubrium junction of the two forming the sternal angle, angulus Ludovici. Footnote. Named after the French surgeon Antoine Louis, 1723 1792. The Latin name angulus Ludovici is not infrequently mistranslated into English as the angle of Ludwig. End footnote. The inferior border is narrow and articulates with the xiphoid process. Each lateral border, at its superior angle, has a small facet, with which a similar facet on the manubrium forms a cavity for the cartilage of the second rib. Below this are four angular depressions which receive the cartilages of the third, fourth, fifth, and sixth ribs, while the inferior angle has a small facet, which, with a corresponding one on the xiphoid process, forms a notch for the cartilage of the second rib. These articular depressions are separated by a series of curved interarticular intervals, which diminish in length from above downward, and correspond to the intercostal spaces. Most of the cartilages belong to the true ribs, as will be seen from the foregoing description, articulate with the sternum at the lines of junction of its permuted component segments. This is well seen in many of the lower animals, where the parts of the bone remain ununited longer than in man. Xiphoid process. Processus xiphoideus. Ensiform or xiphoid appendix. The xiphoid process is the smallest of the three pieces. It is thin and elongated, cartilaginous in structure in youth, but more or less ossified at its upper part in the adult. Surfaces. Its anterior surface affords attachment on either side to the anterior costoxiphoid ligament, and a small part of the rectus abdominis, its posterior surface, to the posterior costoxiphoid ligament, and to some of the fibers of the diaphragm and transversus thoracis, its lateral borders, to the aponeurosis of the abdominal muscles. Above, it articulates with the lower end of the body, and on the front of each superior angle presents a facet for part of the cartilage of the seventh rib. Below, by its pointed extremity, it gives attachment to the linea alba. The xiphoid process varies much in form. It may be broad and thin, pointed, bifid, perforated, curved, or deflected considerably to one or other side. Structure. The sternum is composed of highly vascular cancellous tissue, covered by a thin layer of compact bone, which is thickest in the manubrium between the articular facets for the clavicles. Ossification. The sternum originally consists of two cartilaginous bars, situated one on either side of the median plane and connected with the cartilages of the upper 9 ribs of its own side. These two bars fuse with each other along the middle line to form the cartilaginous sternum, which is ossified from 6 centers, 1 for the manubrium, 4 for the body, and 1 for the xiphoid process. The ossific centers appear in the intervals between the articular depressions for the costal cartilages, in the following order: in the manubrium and first piece of the body during the 6th month, in the second and third pieces of the body during the 7th month of fetal life, in its 4th piece during the 1st year after birth, and in the xiphoid process between the 5th and 18th years. The centers make their appearance at the upper parts of the segments and proceed gradually downward. Footnote. Out of 141 sterna between the time of birth and the age of 16 years, Patterson found the 4th or lowest center for the body present only in 38 cases, that is, 26.9%. End footnote. To these may be added the occasional existence of 2 small episternal centers, which make their appearance one on either side of the jugular notch. They are probably vestiges of the episternal bone of the monotremata and lizards. Occasionally some of the segments are formed from more than one center, the number and position of which vary. Thus the first piece may have 2, 3, or even 6 centers. When 2 are present, they are generally situated one above the other, the upper being the larger. The second piece has seldom more than one. The third, fourth, and fifth pieces are often formed from 2 centers placed laterally, the irregular union of which explains the rare occurrence of the sternal foramen, or of the vertical fissure which which occasionally intersects this part of the bone constituting the malformation known as fissurus sterni. These conditions are further explained by the manner in which the cartilaginous sternum is formed. More rarely still, the upper end of the sternum may be divided by a fissure. Union of the various centres of the body begins about puberty, and proceeds from below upward. By the age of 25 they are all united. The xiphoid process may become joined to the body before the age of 30, but this occurs more frequently after 40. On the other hand, it sometimes remains ununited in old age. In advanced life the manubrium is occasionally joined to the body by bone. When this takes place, however, the body tissue is generally only superficial. The central portion of the interfering cartilage remains unossified. Articulations. The sternum articulates on either side with the clavicle and upper 7 costal cartilages. Section 18. The ribs. Costae. The ribs are elastic arches of bone which form a large part of the thoracic skeleton. They are 12 in number on either side, but this number may be increased by the development of a cervical or lumbar rib, or may be diminished to 11. The first 7 are connected behind with a vertebral column, and in front, through the intervention of the costal cartilages, with the sternum. They are called true or vertebro-sternal ribs. Footnote. Sometimes the 8th rib cartilage articulates with the sternum. This condition occurs more frequently on the right than on the left side. End footnote. The remaining 5 are false ribs. Of these, the first 3 have the cartilages attached to the cartilage of the rib above, vertebrochondral. The last 2 are free at their anterior extremities and are termed floating or vertebral ribs. The ribs vary in their direction, the upper ones being less oblique than the lower. The obliquity reaches its maximum at the 9th rib and gradually decreases from that rib to the 12th. The ribs are situated one below the other in such a manner that spaces called intercostal spaces are left between them. The length of each space corresponds to that of the adjacent ribs and their cartilages. The breadth is greater in front than behind, and between the upper and the lower ribs. The ribs increase in length from the 1st to the 7th, below which they diminish to the 12th. In breadth they decrease from above downward. In the upper 10, the greatest breadth is at the sternal extremity. Common characteristics of the ribs. A rib from the middle of the series should be taken in order to study the common characteristics of these bones. Each rib has 2 extremities, a posterior or vertebral and an anterior or sternal, and an intervening portion, the body or shaft. Posterior extremity. The posterior or vertebral extremity presents for examination a head, neck, and tubercle. The head is marked by a kidney-shaped articular surface, divided by a horizontal crest into 2 facets for articulation, with a depression formed on the bodies of 2 adjacent thoracic vertebrae. The upper facet is the smaller. To the crest is attached the interarticular ligament. The neck is the flattened portion which extends lateralward from the head. It is about 2.5 cm long and is placed in front of the transverse process of the lower of the 2 vertebrae with which the head articulates. Its anterior surface is flat and smooth, its posterior rough for the attachment of the ligament of the neck, and perforated by numerous foramina. Of its 2 borders, the superior presents a rough crest, crista colle costo, for the attachment of the anterior costo-transverse ligament. Its inferior border is rounded. On the posterior surface, at the juncture of the neck and body, and nearer the lower than the upper border, is an eminence, the tubercle. It consists of an articular and non-articular portion. The articular portion, the lower and more medial of the 2, presents a small oval surface for articulation with the end of the transverse process of the lower of the 2 vertebrae to which the head is connected. The non-articular portion is a rough elevation, and affords attachment to the ligament of the tubercle. The tubercle is much more prominent in the upper than in the lower ribs. Body. The body or shaft is thin and flat, with 2 surfaces, an external and an internal, and 2 borders, a superior and an inferior. The external surface is convex, smooth, and marked, a little in front of the tubercle, by a prominent line. Directed downward and lateralward, this gives attachment to a tendon of the iliocostalis, and is called the angle. At this point the rib is bent in two directions, and at the same time twisted on its long axis. If the rib be laid upon its lower border, the portion of the body in front of the angle rests upon this border, while the portion behind the angle is bent medialward, and at the same time tilted upward. As a result of the twisting, the external surface behind the angle looks downward, and in front of the angle slightly upward. The distance between the angle and the tubercle is progressively greater from the second to the 10th ribs. The portion between the angle and the tubercle is rounded, rough, and irregular, and serves for the attachment of the longissimus dorsi. The internal surface is concave, smooth, directed a little upward behind the angle, a little downward in front of it, and is marked by a ridge which commences at the lower extremity of the head. This ridge is strongly marked as far as the angle, and gradually becomes lost at the junction of the anterior and middle thirds of the bone. Between it and the inferior border is a groove, the costal groove, for the intercostal vessels and nerve. At the back part of the bone, this groove belongs to the inferior border, but just in front of the angle, where it is deepest and broadest, it is on the internal surface. The superior edge of the groove is rounded and serves for the attachment of an intercostalis internus. The inferior edge corresponds to the lower margin of the rib, and gives attachment to an intercostalis externus. Within the groove are seen the orifices of numerous small foramina for nutrient vessels which traverse the shaft obliquely from before backward. The superior border, thick and rounded, is marked by an external and an internal lip, more distinct behind than in front, which serve for the attachment of intercostalis externus and internus. The inferior border is thin and has attached to it an intercostalis externus. Anterior extremity. The anterior or sternal extremity is flattened and presents a porous, oval, concave depression into which the costal cartilage is received. Peculiar ribs. The first, second, 10th, 11th, and 12th ribs present certain variations from the common characteristics described above, and require special consideration. First rib. The first rib is the most curved and usually the shortest of all the ribs. It is broad and flat, its surfaces looking upward and downward, and its borders inward and outward. The head is small, rounded, and possesses only a single articular facet, for articulation with the body of the first thoracic vertebra. The neck is narrow and rounded. The tubercle, thick and prominent, is placed on the outer border. There is no angle, but at the tubercle the rib is slightly bent, with its convexity upward, so that the head of the bone is directed downward. The upper surface of the body is marked by two shallow grooves, separated from each other by a slight ridge prolonged internally into a tubercle, the scalene tubercle, for the attachment of the scalenus anterior. The anterior groove transmit the subclavian vein, the posterior and subclavian artery, and the lowest trunk of the brachial plexus. Behind the posterior groove is a rough area for the attachment of the scalenus medius. The under surface is smooth and destitute of a costal groove. The outer border is convex, thick, and rounded, and at its posterior part gives attachment to the first digitation of the serratus anterior. The inner border is concave, thin and sharp, and marked about its center by the scalene tubercle. The anterior extremity is larger and thicker than that of any of the other ribs. The second rib. The second rib is much longer than the first, but has a very similar curvature. The non-articular portion of the tubercle is occasionally only feebly marked. The angle is slight and situated close to the tubercle. The body is not twisted, so that both ends touch any plane surface upon which it may be laid, but there is a bend, with its convexity upward, similar to, though smaller than, that found in the first rib. The body is not flattened horizontally like that of the first rib. Its external surface is convex and looks upward and a little outward. Near the middle of it is a rough eminence for the origin of the lower part of the first and the whole of the second digitation of the serratus anterior. Behind and above this is attached the scalenus posterior. The internal surface, smooth and concave, is directed downward and a little inward. On its posterior part there is a short costal groove. 10th rib. The 10th rib has only a single articular facet on its head. 11th and 12th ribs. The 11th and 12th ribs have each a single articular facet on the head, which is of rather large size. They have no necks or tubercles, and are pointed at their anterior ends. The 11th has a slight angle and a shallow costal groove. The 12th has neither. It is much shorter than the 11th, and its head is inclined slightly downward. Sometimes the 12th rib is even shorter than the first. Structure. The ribs consist of highly vascular cancellous tissue, enclosed in a thin layer of compact bone. Ossification. Each rib, with the exception of the last two, is ossified from 4 centers, a primary center for the body, and 3 epiphyseal centers, 1 for the head, and 1 each for the articular and non-articular parts of the tubercle. The 11th and 12th ribs have each only 2 centers, those for the tubercles being wanting. Ossification begins near the angle toward the end of the 2nd month of fetal life, and is seen first in the 6th and 7th ribs. The epiphyses for the head and tubercle make their appearance between the 16th and 20th years, and are united to the body about the 25th year. Fawcett states that in all probability there is usually no epiphysis on the non-articular part of the tuberosity below the 6th or 7th rib. Section 19. The costal cartilages. Cartilaginous costalis. The costal cartilages are bars of hyaline cartilage which serve to prolong the ribs forward and contribute very materially to the elasticity of the walls of the thorax. The first 7 pairs are connected with the sternum. The next 3 are each articulated with the lower border of the cartilage of the preceding rib. The last 2 have pointed extremities, which end in the wall of the abdomen. Like the ribs, the costal cartilages vary in their length, breadth, and direction. They increase in length from the 1st to the 7th, then gradually decrease to the 12th. Their breadth, as well as that of the intervals between them, diminishes from the first to the last. They are broad in their attachments to the ribs, and taper toward their sternal extremities, excepting the first 2, which are of the same breadth throughout, and the 6th, 7th, and 8th, which are enlarged where their margins are in contact. They also vary in direction. The first descends a little, the second is horizontal, the third ascends slightly, while the others are angular, following the course of the ribs for a short distance, and then ascending to the sternum or preceding cartilage. Each costal cartilage presents 2 surfaces, 2 borders, and 2 extremities. Surfaces. The anterior surface is convex, and looks forward and upward. That of the first gives attachment to the costal clavicular ligament and the subclavius muscle. Those of the first 6 or 7, at their sternal ends, to the pectoralis major. The others are covered by, and give partial attachment to, some of the flat muscles of the abdomen. The posterior surface is concave, and directed backward and downward. That of the first gives attachment to the sternothyroides, those of the third to the 6th inclusive of the transversus thoracis, and the 6 or 7 inferior ones to the transversus abdominis and the diaphragm. Borders. Of the 2 borders the superior is concave, the inferior convex. They afford attachment to the intercostalis interni. The upper border of the 6 gives attachment also to the pectoralis major. The inferior borders of the 6th, 7th, 8th, and 9th cartilages present heel-like projections at the points of greatest convexity. These projections carry smooth oblong facets, which articulate respectively with facets and slight projections from the upper borders of the 7th, 8th, 9th, and 10th cartilages. Extremities. The lateral end of each cartilage is continuous with the osseous tissue of the rib to which it belongs. The medial end of the first is continuous with the sternum. The medial ends of the 6 succeeding ones are rounded and are received into shallow concavities on the lateral margins of the sternum. The medial ends of the 8th, 9th, and 10th costal cartilages are pointed, and are connected each with a cartilage immediately above. Those of the 11th and 12th are pointed and free. In old age, the costal cartilages are prone to undergo superficial ossification. Cervical ribs, derived from the 7th cervical vertebra, are of not infrequent occurrence, and are important clinically because they may give rise to obscure nervous or vascular symptoms. The cervical rib may be a mere epiphysis, articulating only with the transverse process of the vertebra, but more commonly it consists of a defined head, neck, and tubercle, with or without a body. It extends lateralward, or forward and lateralward, into the posterior triangle of the neck, where it may terminate in a free end or may join the first thoracic rib, the first costal cartilage, or the sternum. It varies much in shape, size, direction, and mobility. If it reach far enough forward, part of the brachial plexus and the subclavian artery and vein cross over it, and are apt to suffer compression in so doing. Pressure on the artery may obstruct the circulation so much that arterial thrombosis results, causing gangrene of the fingertips. Pressure on the nerves is commoner, and affects the 8th cervical and 1st thoracic nerves, causing paralysis of the muscles they supply, and neuralgic pain and paresthesia in the area of skin to which they are distributed. No ocular pupillary changes are to be found. The thorax is frequently found to be altered in shape in certain diseases. In rickets, the ends of the ribs, where they join the costal cartilages, become enlarged, giving rise to the so-called rickety rosary, which in mild cases is only found on the internal surface of the thorax. Lateral to these enlargements, the softened ribs sink in, so as to present a groove passing downward and lateralward on either side of the sternum. This bone is forced forward by the bending of the ribs, and the antero-posterior diameter of the chest is increased. The ribs affected, of the 2nd to the 8th, the lower ones being prevented from falling in by the presence of the liver, stomach, and spleen, and when the abdomen is distended, as it often is in rickets, the lower ribs may be pushed outward, causing a transverse groove, Harrison's sulcus, just above the costal arch. This deformity, or forward projection of the sternum, often asymmetrical, is known as pigeon breast, and may be taken as evidence of active or old rickets, except in cases of primary spinal curvature. In many instances, it is associated in children with obstruction in the upper air passages, due to enlarged tonsils or adenoid growths. In some rickety children or adults, and also in others who give no history or further evidence of having had rickets, an opposite condition obtains. The lower part of the sternum, and often the xiphoid process, as well, are deeply depressed backward, producing an oval hollow in the lower sternal and upper epigastric regions. This is known as funnel breast, German, Trickerbrust, and never appears to produce the least disturbance of any of the vital functions. The fistical chest is often long and narrow, and with great obliquity of the ribs and projection of the scapulae. In pulmonary emphysema, the chest is enlarged in all its diameters, and presents on section an almost circular outline. It has received the name of the barrel-shaped chest. In severe cases of lateral curvature of the vertebral column, the thorax becomes much distorted, in consequence of the rotation of the bodies of the vertebra, which takes place in this disease. The ribs opposite the convexity of the dorsal curve become extremely convex behind, being thrown out and bulging, and at the same time flattened in front, so that the two ends of the same rib are almost parallel. Coincidently with this, the ribs on the opposite side, on the concavity of the curve, are sunk and depressed behind, and bulging and convex in front. Section 20. The skull. The skull is supported on the summit of the vertebral column, and is of an oval shape, wider behind than in front. It is composed of a series of flattened or irregular bones which, with one exception, the mandible, are immovably jointed together. It is divisible into two parts. One, the cranium, which lodges and protects the brain, consists of 8 bones, and 2, the skeleton of the face, of 14, as follows: skull, 22 bones; cranium, 8 bones; occipital, 2 parietals; frontal, 2 temporals; sphenoidal, ethmoidal; face, 14 bones; 2 nasals, 2 maxillae, 2 lacrymals, 2 zygomatics, 2 palatines, 2 inferior nasal conchae, vomer, mandible. In the basal nomenclature, certain bones developed in association with the nasal capsule, the inferior nasal conchae, the lacrymals, the nasals, and the vomer, are grouped as cranial and not as facial bones. The hyoid bone, situated at the root of the tongue and attached to the base of the skull by ligaments, is described in this section. The occipital bone, ossa cranii, and os occipitali. The occipital bone, situated at the back and lower part of the cranium, is trapezoid in shape and curved on itself. It is pierced by a large oval aperture, the foramen magnum, through which the cranial cavity communicates with the vertebral canal. The curved expanded plate behind the foramen magnum is named the squama. The thick, somewhat quadrilateral piece in front of the foramen is called the basilar part, whilst on either side of the foramen is the lateral portion, the squama, squama occipitalis. The squama, situated above and behind the foramen magnum, is curved from above downward and from side to side. Surfaces. The external surface is convex and presents midway between the summit of the bone and the foramen magnum a prominence, the external occipital protuberance. Extending lateralward from this on either side are 2 curved lines, one a little above the other. The upper, often faintly marked, is named the highest neutral line, and to it the galea aponeurotica is attached. The lower is termed the superior neutral line. That part of the squama which lies above the highest neutral lines is named the planum occipitale, and is covered by the occipitalis muscle. That below, termed the planum nuchale, is rough and irregular for the attachment of several muscles. From the external occipital protuberance, a ridge or crest, the median neutral line, often faintly marked, descends to the foramen magnum, and affords attachment to the ligamentum nuchae. Running from the middle of this line across either half of the neutral plane is the inferior neutral line. Several muscles are attached to the outer surface of the squama. Thus the superior neutral line gives origin to the occipitalis trapezius, and insertion to the sternocleidomastoides and splenius capitis. Into the surface between the superior and inferior neutral lines, semispinalis capitis and the obliquus capitis superior are inserted, while the inferior neutral line and the area below it receive the insertions of the recti capitis posterioris major and minor. The posterior atlanto-occipital membrane is attached around the posterolateral part of the foramen magnum, just outside the margin of the foramen. The internal surface is deeply concave and divided into 4 fossae cruciate eminence. The upper 2 fossae are triangular and lodge the occipital bones of the cerebrum. The lower 2 are quadrilateral and accommodate the hemispheres of the cerebellum. At the point of intersection of the 4 divisions of the cruciate eminence is the internal occipital protuberance. From this protuberance, the upper division of the cruciate eminence runs to the superior angle of the bone, and on one side of it, generally the right, is a deep groove, the sagittal sulcus, which lodges the hinder part of the superior sagittal sinus. To the margins of this sulcus the falx cerebri is attached. The lower division of the cruciate eminence is prominent, and is named the internal occipital crest. It bifurcates near the foramen magnum, and gives attachment to the falx cerebelli. In the attached margin of this falx is the occipital sinus, which is sometimes duplicated. In the upper part of the internal occipital crest, a small depression is sometimes distinguishable. It is termed the foramen fossa, since it is occupied by part of the vermis of the cerebellum. Transverse grooves, one on either side, extend from the internal occipital protuberance to the lateral angles of the bone. Those grooves accommodate the transverse sinuses, and their prominent margins give attachment to the tentorium cerebelli. The groove on the right side is usually larger than that on the left, and is continuous with that for the superior sagittal sinus. Exceptions to this condition are, however, not infrequent. The left may be larger than the right, or the two may be almost equal in size. The angle of union of the superior sagittal and transverse sinuses is named the confluence of the sinuses. Torcular hierophili. Footnote. The columns of blood coming in different directions were supposed to be pressed together at this point. Turcular, a wine press. End footnote. And its position is indicated by a depression situated on one or other side of the protuberance. Lateral parts. Pars lateralis. The lateral parts are situated at the sides of the foramen magnum. On their under surfaces are the condyles for articulation with the superior facets of the atlas. The condyles are oval or reniform in shape, and their interior extremities, directed forward and medialward, are closer together than their posterior, and encroach on the basilar portion of the bone. The posterior extremities extend back to the level of the middle of the foramen magnum. The articular surfaces of the condyles are convex from before backward and from side to side, and look downward and lateralward. To their margins are attached the capsules of the atlanto-occipital articulations, and on the medial side of each is a rough impression or tubercle for the alar ligament. At the base of either condyle, the bone is tunneled by a short canal, the hypoglossal canal, anterior condyloid foramen. This begins on the cranial surface of the bone immediately above the foramen magnum, and is directed lateralward and forward above the condyle. It may be partially or completely divided into two by a spicule of bone. It gives exit to the hypoglossal or 12th cerebral nerve, and entrance to a meningeal branch of the ascending pharyngeal artery. Behind either condyle is a depression, the condyloid fossa, which receives the posterior margin of the superior facet of the atlas when the head is bent backward. The floor of this fossa is sometimes perforated by the condyloid canal, through which an emissary vein passes from the transverse sinus. Extending lateralward from the posterior half of the condyle is a quadrilateral plate of bone, the jugular process, excavated in front by the jugular notch, which, in the articulated skull, forms the posterior part of the jugular foramen. The jugular notch may be divided into two by a bony spicule, the intrajugular process, which projects lateralward above the hypoglossal canal. The under surface of the jugular process is rough, and gives attachment to the rectus capitis lateralis muscle, and the lateral atlanto-occipital ligament. From this surface in eminence, the paramastoid process sometimes projects downwards, and may be of sufficient length to reach, and articulate with, the transverse process of the atlas. Laterally, the jugular process presents a rough quadrilateral or triangular area, which is joined to the jugular surface of the temporal bone by a plate of cartilage. After the age of 25, this plate tends to ossify. The upper surface of the lateral part presents an oval eminence, the jugular tubercle, which overlies the hypoglossal canal, and is sometimes crossed by an oblique groove for the glossopharyngeal, vagus, and accessory nerves. On the upper surface of the jugular process is a deep groove which curves medialward and forward and is continuous with the jugular notch. This groove lodges the terminal part of the transverse sinus and opening into it, close to the medial margin, is the orifice of the condyloid canal. Basilar part, pars basilaris. The basilar part extends forward and upward from the foramen magnum, and presents in front an area more or less quadrilateral in outline. In the young skull, this area is rough and uneven, and is joined to the body of the sphenoid by a plate of cartilage. By the 25th year, this cartilaginous plate is ossified, and the occipital and sphenoid form a continuous bone. Surfaces. On its lower surface, about 1 centimeter in front of the foramen magnum, is a pharyngeal tubercle, which gives attachment to the fibrous raphe of the pharynx. On either side of the middle line, the longus capitis and rectus capitis anterior are inserted, and immediately in front of the foramen magnum, the anterior atlanto-occipital membrane is attached. The upper surface presents a broad, shallow groove, which inclines upward and forward from the foramen magnum. It supports the medulla oblongata, and near the margin of the foramen magnum gives attachment to the membrana tectoria. On the lateral margins of this surface are faint grooves for the inferior petrocell sinuses. Foramen magnum. The foramen magnum is a large oval aperture with its long diameter anterior-posterior. It is wider behind than in front, where it is encroached upon by the condyles. It transmits the medulla oblongata and its membranes, the accessory nerves, the vertebral arteries, the anterior and posterior spinal arteries, and the membrana tectoria and alar ligaments. Angles. The superior angle of the occipital bone articulates with the occipital angles of the parietal bones, and, in the fetal skull, corresponds in position with the posterior fontanelle. The inferior angle is fused with the body of the sphenoid. The lateral angles are situated at the extremities of the grooves for the transverse sinuses. Each is received into the interval between the mastoid angle of the parietal and mastoid part of the temporal. Borders. The superior borders extend from the superior to the lateral angles. They are deeply serrated for articulation with the occipital borders of the parietals, and form by this union the lambdoidal suture. The inferior borders extend from the lateral angles to their inferior angle. The upper half of each articulates with the mastoid portion of the corresponding temporal, the lower half with the petrous part of the same bone. These two portions of the inferior border are separated from one another by the jugular process, the notch on the anterior surface of which forms the posterior part of the jugular foramen. Structure. The occipital, like the other cranial, the outer and inner tables, between which is the cancellous tissue or diploid. The bone is especially thick at the ridges, protuberances, condyles, and an anterior part of the basilar part. In the inferior fossae it is thin, semi-transparent, and destitute of diploid. Ossification. The planum occipitale of the squama is developed in membrane, and may remain separate throughout life when it constitutes the interparietal bone. The rest of the bone is developed in cartilage. The number of nuclei for the planum occipitale is usually given as 4, 2 appearing near the middle line about the 2nd month, and 2 some little distance from the middle line about the 3rd month of fetal life. The planum nuchale of the squama is ossified from 2 centers, which appear about the 7th week of fetal life, and soon unite to form a single piece. Union of the upper and lower portions of the squama takes place in the 3rd month of fetal life. An occasional center, Kirchrig, appears in the posterior margin of the foramen magnum during the 5th month. This forms a separate ossicle, sometimes double, which unites with the rest of the squama before birth. Each of the lateral parts begins to ossify from a single center during the 8th week of fetal life. The basilar portion is ossified from 2 centers, one in front of the other. These appear about the 6th week of fetal life and rapidly coalesce. Mall states that the planum occipitale is ossified from 2 centers and the basilar portion from 1. About the 4th year, the squama and the 2 lateral portions unite, and about the 6th year the bone consists of a single piece. Between the 18th and 25th years, the occipital and sphenoid become united, forming a single bone. The parietal bone, os parietale. The parietal bones form, by their union, the sides and roof of the cranium. Each bone is irregularly quadrilateral in form, and has 2 surfaces, 4 borders, and 4 angles. Surfaces. The external surface is convex, smooth, and marked near the center by an eminence, the parietal eminence, tuber parietale, which indicates the point where ossification commenced. Crossing the middle of the bone in an arched direction are 2 curved lines, the superior and inferior temporal lines. The former gives attachment to the temporal fascia, and the latter indicates the upper limit of the muscular origin of the temporalis. Above these lines the bone is covered by the galea aponeurotica. Below them it forms part of the temporal fossa, and affords attachment to the temporalis muscle. At the back part and close to the upper or sagittal border is the parietal foramen, which transmit a vein to the superior sagittal sinus, and sometimes a small branch of the occipital artery. It is not constantly present, and its size varies considerably. The internal surface is concave. It presents depressions corresponding to the cerebral convolutions, and numerous furrows for the ramifications of the middle meningeal vessel. The latter run upward and backward from the sphenoidal angle, and from the central and posterior part of the squamous border. Along the upper margin is a shallow groove, which, together with that on the opposite parietal, forms a channel, the sagittal sulcus, for the superior sagittal sinus. The edges of the sulcus afford attachment to the falx cerebri. Near the groove are several depressions, best marked in the skulls of old persons, for the arachnoid granulations, Pacchionian bodies. In the groove is the internal opening of the parietal foramen when the aperture exists. Borders. The sagittal border, the longest and thickest, is dentated and articulates with its fellow of the opposite side, forming the sagittal suture. The squamous border is divided into 3 parts. Of these, the anterior is thin and pointed, beveled at the expense of the outer surface, and overlapped by the tip of the great wing of the sphenoid. The middle portion is arched, beveled at the expense of the outer surface, and overlapped by the squama of the temporal. The posterior part is thick and serrated for articulation with the mastoid portion of the temporal. The frontal border is deeply serrated and beveled at the expense of the outer surface above and of the inner below. It articulates with the frontal bone, forming 1/2 of the coronal suture. The occipital border, deeply denticulated, articulates with the occipital, forming 1/2 of the lambdoidal suture. Angles. The frontal angle is practically a right angle, and corresponds with the point of meeting of the sagittal and coronal sutures. This point is named the bregma. In the fetal skull, and for about a year and a half after birth, this region is membranous, and is called the anterior fontanelle. The sphenoidal angle, thin and acute, is received into the interval between the frontal bone and the great wing of the sphenoid. Its inner surface is marked by a deep groove, sometimes a canal, for the anterior divisions of the middle meningeal artery. The occipital angle is rounded, and corresponds with the point of meeting of the sagittal and lambdoidal sutures, a point which is termed the lambda. In the fetus this part of the skull is membranous, and is called the posterior fontanelle. The mastoid angle is truncated. It articulates with the occipital bone and with the mastoid portion of the temporal, and presents on its inner surface a broad, shallow groove which lodges part of the transverse sinus. The point of meeting of this angle where the occipital and mastoid part of the temporal is named the asterion. Ossification. The parietal bone is ossified in membrane from a single centre, which appears at the parietal eminence about the 8th week of fetal life. Ossification gradually extends in a radial manner from the centre toward the margins of the bone. The angles are consequently the parts last formed, and it is here that the fontanelles exist. Occasionally the parietal bone is divided into two parts, upper and lower, by an antero-posterior suture. Articulations. The parietal articulates with 5 bones: the opposite parietal, the occipital, frontal, temporal, and sphenoid. The frontal bone, os frontale. The frontal bone resembles a cockle shell in form, and consists of 2 portions: a vertical portion, the squama, corresponding with the region of the forehead, and an orbital or horizontal portion, which enters into the formation of the roofs of the orbital and nasal cavities. Squama, squama frontalis. Surfaces. The external surface of this portion is convex, and usually exhibits, in the lower part of the middle line, the remains of the frontal or metopic suture. In infancy, this suture divides the bone into 2, a condition which may persist throughout life. On either side of this suture, about 3 cm above the supraorbital margin, is a rounded elevation, the frontal eminence, tuber frontale. These eminences vary in size in different individuals, are occasionally unsymmetrical, and are especially prominent in young skulls. The surface of the bone above them is smooth, and covered by the galea aponeurotica. Below the frontal eminences, and separated from them by a shallow groove, are 2 arched elevations, the superciliary arches. These are prominent medially, and are joined to one another by a smooth elevation named the glabella. They are larger in the male than in the female, and their degree of prominence depends to some extent on the size of the frontal air sinuses. Footnote: Some confusion is occasioned to students commencing the study of anatomy by the name sinus, having been given to 2 different kinds of space connected with the skull. It may be as well, therefore, to state here that the sinuses in the interior of the cranium which produce the grooves on the inner surfaces of the bones are venous channels which convey the blood from the brain, while the sinuses external to the cranial cavity, the frontal, sphenoidal, ethmoidal, and maxillary, are hollow spaces in the bones themselves. They communicate with the nasal cavities and contain air. End footnote. Prominent ridges are, however, occasionally associated with small air sinuses. Beneath each superciliary arch is a curved and prominent margin, the supraorbital margin, which forms the upper boundary of the base of the orbit, and separates the squama from the orbital portion of the bone. The lateral part of this margin is sharp and prominent, affording to the eye, in that situation, considerable protection from injury. The medial part is rounded. At the junction of its medial and intermediate thirds is a notch, sometimes converted into a foramen, the supraorbital notch or foramen, which transmits the supraorbital vessels and nerve. A small aperture in the upper part of the notch transmits a vein from the dipoi to join the supraorbital vein. The supraorbital margin ends laterally in the zygomatic process, which is strong and prominent, and articulates with the zygomatic bone. Running upward and backward from this process is a well-marked line, the temporal line, which divides into the upper and lower temporal lines, continuous, in the articulated skull, with the corresponding lines on the parietal bone. The area below and behind the temporal line forms the anterior part of the temporal fossa, and gives origin to the temporalis muscle. Between the supraorbital margins, the squama projects downward to a level below that of the zygomatic processes. This portion is known as the nasal part and presents a rough, uneven interval, the nasal notch, which articulates on either side of the middle line with the nasal bone, and laterally with the frontal process of the maxilla and with the lacrimal. The term nation is applied to the middle of the frontonasal suture. From the center of the notch, the nasal process projects downward and forward beneath the nasal bones and frontal processes of the maxillae, and supports the bridge of the nose. The nasal process ends below in a sharp spine, and on either side of this is a small grooved surface, which enters into the formation of the roof of the corresponding nasal cavity. The spine forms part of the septum of the nose, articulating in front with the crest of the nasal bones and behind with the perpendicular plate of the ethmoid. The internal surface of the squama is concave and presents in the upper part of the middle line a vertical groove, the sagittal sulcus, the edges of which unite below to form a ridge, the frontal crest. The sulcus lodges the superior sagittal sinus, while its margins and the crest afford attachment to the falx cerebri. The crest ends below in a small notch which is converted into a foramen, the foramen cecum, by articulation with the ethmoid. This foramen varies in size in different subjects, and is frequently impervious. When open, it transmits a vein from the nose to the superior sagittal sinus. On either side of the middle line, the bone presents depressions for the convolutions of the brain, and numerous small furrows for the anterior branches of the middle meningeal vessels. Several small irregular fossae may also be seen on either side of the sagittal sulcus, for the reception of the arachnoid granulations. Orbital or horizontal part. Pars orbitalis. This portion consists of two thin triangular plates, the orbital plates, which form the vaults of the orbits, and are separated from one another by a median gap, the ethmoidal notch. Surfaces. The inferior surface of each orbital plate is smooth and concave, and presents laterally, under cover of the zygomatic process, a shallow depression, the lacrimal fossa, for the lacrimal gland. Near the nasal part is a depression, the fovea trochlearis, or occasionally a small trochlear spine, for the attachment of the cartilaginous pulley of the obliquus oculi superior. The superior surface is convex and marked by depressions for the convolutions of the frontal lobes of the brain, and faint grooves for the meningeal branches of the ethmoidal vessels. The ethmoidal notch separates the two orbital plates. It is quadrilateral and filled in the articulated skull by the cribriform plate of the ethmoid. The margins of the notch present several half cells, which, when united with corresponding half cells on the upper surface of the ethmoid, complete the ethmoidal air cells. Two grooves cross these edges transversely. They are converted into the anterior and posterior ethmoidal canals by the ethmoid, and open on the medial wall of the orbit. The anterior canal transmits the nasociliary nerve and anterior ethmoidal vessels, the posterior ethmoidal nerve and vessels. In front of the ethmoidal notch, on either side of the frontal spine, are the openings of the frontal air sinuses. These are two irregular cavities, which extend backward, upward, and lateralward for a variable distance between the two tables of the skull. They are separated from one another by a thin, bony septum, which often deviates to one or other side, with the result that the sinuses are rarely symmetrical. Absent of birth, they are usually fairly well developed between the 7th and 8th years, but only reach their full size after puberty. They vary in size in different persons, and are larger in men than in women. Footnote: Aldrin Turner, The Accessory Sinuses of the Nose, 1901, gives the following measurements for a sinus of average size: height 1 1/4 inches, Breadth 1 inch. Depth from before backward 1 inch. End footnote. They are lined by mucous membrane, and each communicates with the corresponding nasal cavity by means of a passage called the frontal nasal duct. Borders. The border of the squama is thick, strongly serrated, beveled at the expense of the inner table above, where it rests upon the parietal bones, and at the expense of the outer table on either side, where it receives the lateral pressure of those bones. This border is continued below into a triangular rough surface, which articulates with the great wing of the sphenoid. The posterior borders of the orbital plates are thin and serrated, and articulate with the small wings of the sphenoid. Structure. The squama and the zygomatic processes are very thick, consisting of diploic tissue contained between two compact laminae. Diploic tissue is absent in the regions occupied by the frontal air sinuses. The orbital portion is thin, translucent, and composed entirely of compact bone. Hence, the facility with which instruments can penetrate the cranium through this part of the orbit. When the frontal sinuses are exceptionally large, they may extend backward for a considerable distance into the orbital portion, which in such cases also consists of only 2 tables. Ossification. The frontal bone is ossified in membrane from 2 primary centers, one for each half, which appear toward the end of the 2nd month of fetal life, one above each supraorbital margin. From each of these centers, ossification extends upward to form the corresponding half of the squama, and backward to form the orbital plate. The spine is ossified from a pair of secondary centers, on either side of the middle line. Similar centers appear in the nasal part and zygomatic processes. At birth the bone consists of 2 pieces, separated by the frontal suture, which is usually obliterated, except at its lower part, by the 8th year, but occasionally persists throughout life. It is generally maintained that the development of the frontal sinuses begins at the end of the 1st or beginning of the 2nd year, but Onodi's researches indicate that development begins at birth. The sinuses are of considerable size by the 7th or 8th year, but do not attain the full proportions until after puberty. Articulations. The frontal articulates with 12 bones: the sphenoid, the ethmoid, the 2 parietals, the 2 nasals, the 2 maxillae, the 2 lacrimals, and the 2 zygomatics.
The temporal bone, Part 1. Os temporale. The temporal bones are situated at the sides and base of the skull. Each consists of 5 parts, viz., the squama, the petrous, mastoid, and tympanic parts, and the styloid process. The squama temporalis. The squama forms the anterior and upper part of the bone and is scale-like, thin, and translucent. Surfaces. Its outer surface is smooth and convex. It affords attachment to the temporalis muscle, and forms part of the temporal fossa. On its hinder part is a vertical groove for the middle temporal artery. A curved line, the temporal line, or supramastoid crest, runs backward and upward across its posterior part. It serves for the attachment of the temporal fascia, and limits the origin of the temporalis muscle. The boundary between the squama and the mastoid portion of the bone, as indicated by traces of the original suture, lies about 1 centimeter below this line. Projecting from the lower part of the squama is a long arched process, the zygomatic process. This process is at first directed lateralward, its 2 surfaces looking upward and downward. It then appears as if twisted inward upon itself, and runs forward, its surfaces now looking medialward and lateralward. The superior border is long, thin, and sharp, and serves for the attachment of the temporal fascia. The inferior, short, thick, and arched, has attached to it some fibers of the masseter. The lateral surface is convex and subcutaneous. The medial is concave and affords attachment to the masseter. The anterior end is deeply serrated and articulates with the zygomatic bone. The posterior end is connected to the squama by two roots, the anterior and posterior roots. The posterior root, a prolongation of the upper border, is strongly marked. It runs backward across the external acoustic meatus, and is continuous with the temporal line. The anterior root, continuous with the lower border, is short but broad and strong. It is directed medialward and ends in a rounded eminence. The articular tubercle, eminentia articularis. This tubercle forms the front boundary of the mandibular fossa, and in the fresh state is covered with cartilage. In front of the anticular tubercle is a small triangular area which assists in forming the infratemporal fossa. This area is separated from the outer surface of the squama by a ridge which is continuous behind, with the anterior root of the zygomatic process, and in front, in the articulated skull, with the infratemporal crest on the great wing of the sphenoid. Between the posterior wall of the external acoustic meatus and the posterior root of the zygomatic process is the area called the suprameatal triangle, or mastoid fossa, through which an instrument may be pushed into the tympanic antrum. At the junction of the anterior root with the zygomatic process is a projection for the attachment of the temporomandibular ligament, and behind the anterior root is an oval depression, forming part of the mandibular fossa, for the reception of the condyle of the mandible. The mandibular fossa, glenoid fossa, is bound in front by the articular tubercle, behind by the tympanic part of the bone, which separates it from the external acoustic meatus. It is divided into two parts by a narrow slit, the petrotympanic fissure, Glaserian fissure. The anterior part, formed by the squama, is smooth, covered in the fresh state with cartilage, and articulates with the condyle of the mandible. Behind this part of the fossa is a small conical eminence. This is the representative of a prominent tubercle, which, in some mammals, descends behind the condyle of the mandible, and prevents its backward displacement. The posterior part of the mandibular fossa, formed by the tympanic part of the bone, is non-articular, and sometimes lodges a portion of the parotid gland. The petrotympanic fissure leads into the middle ear or tympanic cavity. It lodges the anterior process of the malleus, and transmits the tympanic branch of the interior maxillary artery. The chorda tympani nerve passes through a canal of Hugulaire, separated from the anterior edge of the petrotympanic fissure by a thin scale of bone, and situated on the lateral side of the auditory tube, in a retiring angle between the squama and the petrous portion of the temporal. The internal surface of the squama is concave. It presents depressions corresponding to the convolutions of the temporal lobe of the brain, and grooves for the branches of the middle meningeal vessels. Borders. The superior border is thin and beveled at the expense of the internal table, so as to overlap the squamous border of the parietal bone, forming with it the squamosal suture. Posteriorly, the superior border forms an angle, the parietal notch, with the mastoid portion of the bone. The antero-inferior border is thick, serrated, and beveled at the expense of the inner table above and of the outer below, for articulation with the great wing of the sphenoid. Mastoid portion, pars mastoidea. The mastoid portion forms the posterior part of the bone. Surfaces. Its outer surface is rough and gives attachment to the occipitalis and auricularis posterior. It is perforated by numerous foramina. One of these, of large size, situated near the posterior border, is termed the mastoid foramen. It transmits the vein to the transverse sinus and a small branch of the occipital artery to the dura mater. The position and size of this foramen are very variable. It is not always present. Sometimes it is situated in the occipital bone, or in the suture between the temporal and the occipital. The mastoid portion is continued below into a conical projection, the mastoid process, the size and form of which varies somewhat. It is larger in the male than in the female. This process serves for the attachment of the sternocleidomastoideus, splenius capitis, and the longissimus capitis. On the medial side of the process is a deep groove, the mastoid notch, digastric fossa, for the attachment of the digastricus. Medial to this is a shallow furrow, the occipital groove, which lodges the occipital artery. The inner surface of the mastoid portion presents a deep curved groove, the sigmoid sulcus, which lodges part of the transverse sinus. In it may be seen the opening of the mastoid foramen. The groove for the transverse sinus is separated from the innermost of the mastoid air cells by a very thin lamina of bone, and even this may be partially deficient. Borders. The superior border of the mastoid portion is broad and serrated, for articulation with the mastoid angle of the parietal. The posterior border, also serrated, articulates with the inferior border of the occipital between the lateral angle and the jugular process. Anteriorly, the mastoid portion is fused with the descending process of the squama above. Below, it enters into the formation of the external acoustic meatus and the tympanic cavity. A section of the mastoid process shows it to be hollowed out into a number of spaces, the mastoid cells, which exhibit the greatest possible variety as to their size and number. At the upper and front part of the process, they are large and irregular, and contain air. But toward the lower part they diminish in size, while those at the apex of the process are frequently quite small and contain marrow. Occasionally they are entirely absent, and the mastoid is then solid throughout. In addition to these, a large irregular cavity is situated at the upper and front part of the bone. It is called the tympanic antrum, and must be distinguished from the mastoid cells, though it communicates with them. Like the mastoid cells, it is filled with air and lined by a prolongation of the mucous membrane of the tympanic cavity, with which it communicates. The tympanic antrum is bound above by a thin plate of bone, the tegmen tympani, which separates it from the middle fossa of the base of the skull. Below, by the mastoid process, laterally by the squama just below the temporal line, and medially by the lateral semicircular canal of the inner ear, which projects into its cavity. It opens in front into that portion of the tympanic cavity which is known as the attic, or epitympanic recess. The tympanic antrum is a cavity of some considerable size at the time of birth. The mastoid air cells may be regarded as diverticula from the antrum, and begin to appear at or before birth. By the 5th year they are well marked, but their development is not completed until toward puberty.
The temporal bone. Part 2. Petrous portion. Pars petrosa, pyramis. The petrous portion or pyramid is pyramidal and is wedged in at the base of the skull between the sphenoid and occipital. Directed medialward, forward, and a little upward, it presents for examination a base, an apex, 3 surfaces, and 3 angles, and contains in its interior the essential parts of the organ of hearing. Base. The base is fused with the internal surfaces of the squama and mastoid portion. Apex. The apex, rough and uneven, is received into the angular interval between the posterior border of the great wing of the sphenoid and the basilar part of the occipital. It presents the anterior or internal orifice of the carotid canal, and forms the posterolateral boundary of the forum lacerum. Surfaces. The anterior surface forms the posterior part of the middle fossa of the base of the skull, and is continuous with the inner surface of the squamous portion, to which it is united by the petrosquamous suture, remains of which are distinct even at a late period of life. It is marked by depressions for the convolutions of the brain, and presents 6 points for examination. 1. Near the centre an eminence, eminentia arcuata, which indicates the situation of the superior semicircular canal. 2. In front of and a little lateral to this eminence, a depression indicating the position of the tympanic cavity. Here the layer of bone which separates the tympanic from the cranial cavity is extremely thin, and is known as the tegmen tympani. 3. A shallow groove, sometimes double, leading lateralwards and backwards to an oblique opening, the hiatus of the facial canal, for the passage of the greater superficial petrosal nerve and the petrosal branch of the middle meningeal artery. 4. Lateral to the hiatus, a smaller opening, occasionally seen, for the passage of the lesser superficial petrosal nerve. 5. Near the apex of the bone, the termination of the carotid canal, the wall of which in this situation is deficient in front. 6. Above this canal the shallow trigeminal impression for the reception of the semilunar ganglion. The posterior surface, Fig. 138, forms the front portion of the posterior fossa of the base of the skull and is continuous with the inner surface of the mastoid portion. Near the centre is a large orifice, the internal acoustic meatus, the size of which varies considerably. Its margins are smooth and rounded, and it leads into a short canal about 1 cm in length, which runs lateralward. It transmits the facial and acoustic nerves and the internal auditory branch of the basilar artery. The lateral end of the canal is closed by a vertical plate, which is divided by a horizontal crest, the crista falciformis, into 2 unequal portions. Fig. 140. Each portion is further subdivided by a vertical ridge into an anterior and a posterior part. In the portion beneath the crista falciformis are 3 sets of foramina. One group, just below the posterior part of the crest, situated in the area cribrosa media, consists of several small openings for the nerves to the saccule. Below and behind this area is the foramen singulare, or opening for the nerve to the posterior semicircular duct. In front of and below the first is the tractus spiralis foraminosus, consisting of a number of small spirally arranged openings, which encircle the canalis centralis cochleae. These openings together with the central canal transmit the nerves to the cochlea. The portion above the crista falciformis presents behind the area cribrosa superior, pierced by a series of small openings, for the passage of the nerves to the utricle and the superior and lateral semicircular ducts, and in front the area faciens, with one large opening, the commencement of the canal for the facial nerve, aqueductus fallopii. Behind the internal acoustic meatus is a small slit almost hidden by a thin plate of bone, leading to a canal, the aqueductus vestibuli, which transmits the ductus endolymphaticus together with a small artery and vein. Above and between these 2 openings is an irregular depression which lodges a process of the dura mater and transmits a small vein. In the infant this depression is represented by a large fossa, the subarcuate fossa, which extends backwards as a blind tunnel under the superior semicircular canal. The inferior surface, Fig. 141, is rough and irregular, and forms part of the exterior of the base of the skull. It presents 11 points for examination. 1. Near the apex is a rough surface, quadrilateral in form, which serves partly for the attachment of the levator veli palatini and the cartilaginous portion of the auditory tube, and partly for connection with the basilar part of the occipital bone through the intervention of some dense fibrous tissue. 2. Behind this is the large circular aperture of the carotid canal, which descends at first vertically, and then, making a bend, runs horizontally forward and medialward. It transmits into the cranium the internal carotid artery, and the carotid plexus of nerves. 3. Medial to the opening for the carotid canal and close to its posterior border, in front of the jugular fossa, is a triangular depression. At the apex of this is a small opening, the aqueductus cochleae, which lodges a tubular prolongation of the dura mater establishing a communication between the perilymphatic space and the subarachnoid space, and transmits a vein from the cochlea to join the internal jugular. 4. Behind these openings is a deep depression, the jugular fossa, of variable depth and size in different skulls. It lodges the bulb of the internal jugular vein. 5. In the bony ridge dividing the carotid canal from the jugular fossa is a small inferior tympanic canaliculus, for the passage of the tympanic branch of the glossopharyngeal nerve. 6. In the lateral part of the jugular fossa is the mastoid canaliculus, for the entrance of the auricular branch of the vagus nerve. 7. Behind the jugular fossa is a quadrilateral area, the jugular surface, covered with cartilage in the fresh state, and articulating with the jugular process of the occipital bone. 8. Extending backward from the carotid canal is the vaginal process, a sheath-like plate of bone which divides behind into two laminae. The lateral lamina is continuous with the tympanic part of the bone, the medial with the lateral margin of the jugular surface. 9. Between these laminae is the styloid process, a sharp spine about 2.5 cm in length. 10. Between the styloid and mastoid processes is the stylomastoid foramen. It is the termination of the facial canal and transmits the facial nerve and stylomastoid artery. 11. Situated between the tympanic portion and the mastoid process is the tympanomastoid fissure, for the exit of the auricular branch of the vagus nerve. Angles. The superior angle, the longest, is grooved for the superior petrosal sinus, and gives attachment to the tentorium cerebelli. At its medial extremity is a notch, in which the trigeminal nerve lies. The posterior angle is intermediate in length between the superior and the anterior. Its medial half is marked by a sulcus, which forms, with a corresponding sulcus on the occipital bone, the channel for the inferior petrosal sinus. Its lateral half presents an excavation, the jugular fossa, which, with the jugular notch on the occipital, forms the jugular foramen. An eminence occasionally projects from the centre of the fossa and divides the foramen into two. The anterior angle is divided into two parts: a lateral joined to the squama by a suture, petrosquamous, the remains of which are more or less distinct; a medial, free, which articulates with the spinous process of the sphenoid. At the angle of junction of the petrous part and the squama are two canals, one above the other, and separated by a thin plate of bone, the septum canalis musculotubarii, processus cochleariformis. Both canals lead into the tympanic cavity. The upper one, semicanalis tensoris tympani, transmits the tensor tympani. The lower one, semicanalis tubi auditativae, forms the bony part of the auditory tube. The tympanic cavity, auditory ossicles, and internal ear are described with the organ of hearing. Tympanic part, pars tympanica. The tympanic part is a curved plate of bone lying below the squama and in front of the mastoid process. Surfaces. Its postero-superior surface is concave, and forms the anterior wall, the floor and part of the posterior wall of the bony external acoustic meatus. Medially, it presents a narrow furrow, the tympanic sulcus, for the attachment of the tympanic membrane. Its antero-inferior surface is quadrilateral and slightly concave. It constitutes the posterior boundary of the mandibular fossa, and is in contact with the retromandibular part of the parotid gland. Borders. Its lateral border is free and rough, and gives attachment to the cartilaginous part of the external acoustic meatus. Internally, the tympanic part is fused with the petrous portion, and appears in the retreating angle between it and the squama, where it lies below and lateral to the orifice of the auditory tube. Posteriorly it blends with the squama and mastoid part, and forms the anterior boundary of the tympanomastoid fissure. Its upper border fuses laterally with the back of the postglenoid process, while medially it bounds with the petrotympanic fissure. The medial part of the lower border is thin and sharp. Its lateral part splits to enclose the root of the styloid process, and is therefore named the vaginal process. The central portion of the tympanic part is thin, and in a considerable percentage of skulls is perforated by a hole, the foramen of Huschke. The external acoustic meatus is nearly 2 cm long and is directed inwards and slightly forward. At the same time it forms a slight curve, so that the floor of the canal is convex upward. In sagittal section it presents an oval or elliptical shape, with the long axis directed downward and slightly backward. Its anterior wall and floor and the lower part of its posterior wall are formed by the tympanic part, the roof and upper part of the posterior wall by the squama. Its inner end is closed, in the recent state, by the tympanic membrane. The upper limit of its outer orifice is formed by the posterior root of the zygomatic process, immediately below which there is sometimes seen a small spine, the suprameatal spine, situated at the upper and posterior part of the orifice. Styloid process, processus styloideus. The styloid process is slender, pointed, and of varying length. It projects downwards and forwards from the under surface of the temporal bone. Its proximal part, tympanohyal, is ensheathed by the vaginal process of the tympanic portion, while its distal part, stylohyal, gives attachment to the stylohyoid and stylomandibular ligaments, and to the styloglossus, stylohyoideus, and stylopharyngeus muscles. The stylohyoid ligament extends from the apex of the process to the letter cornu of the hyoid bone, and in some instances is partially, in others completely, ossified. Structure. The structure of the squama is like that of the other cranial bones. The mastoid portion is spongy, and the petrous portion is dense and hard. Ossification. The temporal bone is ossified from 8 centres, exclusive of those for the internal ear and the tympanic ossicles, viz., 1 for the squama including the zygomatic process, 1 for the tympanic part, 4 for the petrous and mastoid parts, and 2 for the styloid process. Just before the close of fetal life, Figure 142, the temporal bone consists of 3 principal parts. 1. The squama is ossified in membrane from a single nucleus, which appears near the root of the zygomatic process about the 2nd month. 2. The petromastoid part is developed from 4 centres, which make their appearance in a cartilaginous ear capsule about the 5th or 6th month. 1. Prootic, appears in the neighbourhood of the eminentia arcuata, spreads in front and above the internal acoustic meatus, and extends to the apex of the bone. It forms part of the cochlea, vestibule, superior semicircular canal, and medial wall of the tympanic cavity. A second, opisthotic, appears at the promontory of the medial wall of the tympanic cavity and surrounds the fenestra cochleae. It forms the floor of the tympanic cavity and vestibule, surrounds the carotid canal, invests the lateral and lower part of the cochlea, and spreads medially below the internal acoustic meatus. A third, pterotic, roofs in the tympanic cavity and antrum, while the fourth, epiotic, appears near the posterior semicircular canal and extends to form the mastoid process. Frolic. 3. The tympanic ring is an incomplete circle, in the concavity of which is a groove, the tympanic sulcus, for the attachment of the circumference of the tympanic membrane. This ring expands to form the tympanic part, and is ossified in membrane from a single centre which appears about the 3rd month. The styloid process is developed from the proximal part of the cartilage of the 2nd branchial or hyoid arch by 2 centres. 1 for the proximal part, the tympanohyal, appears before birth. The other, comprising the rest of the process, is named the stylohyal, and does not appear until after birth. The tympanic ring unites with the squama shortly before birth. The pteromastoid part and squama join during the 1st year, and the tympanohyal portion of the styloid process about the same time. Figures 143 and 144. The stylohyal does not unite with the rest of the bone until after puberty, and in some skulls never at all. The chief subsequent changes in the temporal bone apart from increase in size are 1. The tympanic ring extends outwards and backwards to form the tympanic part. This extension does not, however, take place at an equal rate all round the circumference of the ring, but occurs most rapidly on its anterior and posterior portions. And these outgrowths meet and blend, and thus for a time there exists in the floor of the meatus a foramen, the foramen of Huschke. This foramen is usually closed about the 5th year, but may persist throughout life. 2. The mandibular fossa is at first extremely shallow, and looks lateralward as well as downward. It becomes deeper and ultimately directed downward. Its change in direction is accounted for as follows. The part of the squama which forms the fossa lies at first below the level of the zygomatic process. As, however, the base of the skull increases in width, this lower part of the squama is directed horizontally inward to contribute to the middle fossa of the skull, and its surfaces therefore come to look upward and downward. The attached portion of the zygomatic process also becomes everted, and projects like a shelf at right angles to the squama. 3. The mastoid portion is at first quite flat, and the stylomastoid foramen and rudimentary styloid process lie immediately behind the tympanic ring. With the development of the air cells, the outer part of the mastoid portion grows downward and forward to form the mastoid process. and the styloid process and stylomastoid foramen now come to lie on the under surface. The descent of the foramen is necessarily accompanied by a corresponding lengthening of the facial canal. 4. The downward and forward growth of the mastoid process also pushes forward the tympanic part, so that the portion of it which formed the original floor of the meatus and contained the foramen of Huschka is ultimately found in the interior wall. 5. The fossa subarcuata becomes filled up and almost obliterated. Articulations. The temporal articulates with 5 bones: occipital, parietal, sphenoid, mandible, and zygomatic.
The sphenoid bone, os sphenoidale. The sphenoid bone is situated at the base of the skull in front of the temporals and basilar part of the occipital. It somewhat resembles a bat with its wings extended, and is divided into a median portion or body, 2 great and 2 small wings extending outward from the sides of the body, and 2 pterygoid processes which project from it below. Body, corpus sphenoidale. The body, more or less cubical in shape, is hollowed out in its interior to form 2 large cavities, the sphenoidal air sinuses, which are separated from each other by a septum. Surfaces. The superior surface of the body presents in front a prominent spine, the ethmoidal spine, for articulation with the cribriform plate of the ethmoid. Behind this is a smooth surface slightly raised in the middle line, and grooved on either side for the olfactory lobes of the brain. This surface is bounded behind by a ridge, which forms the anterior border of a narrow transverse groove, the chiasmatic groove, or optic groove, above and behind which lies the optic chiasma. The groove ends on either side in the optic foramen, which transmits the optic nerve and ophthalmic artery into the orbital cavity. Behind the chiasmatic groove is an elevation, the tuberculum sellae, and still more posteriorly a deep depression, the sella turcica, the deepest part of which lodges the hypophysis cerebri and is known as the fossa hypophyseus. The anterior boundary of the sella turcica is completed by 2 small eminences, one on either side, called the middle clinoid processes, while the posterior boundary is formed by a square-shaped plate of bone, the dorsum celli, ending at its superior angles in 2 tubercles, the posterior clinoid processes, the size and form of which vary considerably in different individuals. The posterior clinoid processes deepen the sella turcica, and give attachment to the tentorium cerebelli. On either side of the dorsum celli is a notch for the passage of the abducent nerve, and below the notch a sharp process, the petrosal process, which articulates with the apex of the petrous portion of the temporal bone, and forms the medial boundary of the foramen lacerum. Behind the dorsum celli is a shallow depression, the clevus, which slopes obliquely backward and is continuous with the groove on the basilar portion of the occipital bone. It supports the upper part of the pons. The lateral surfaces of the body are united with the great wings and the medial pterygoid plates. Above the attachment of each great wing is a broad groove, curved something like the italic letter F. It lodges the internal carotid artery and the cavernous sinus, and is named the carotid groove. Along the posterior part of the lateral margin of this groove, in the angle between the body and great wing, is a ridge of bone called the lingula. The posterior surface, quadrilateral in form, is joined, during infancy and adolescence, to the basilar part of the occipital bone by a plate of cartilage. Between the 18th and 25th years this becomes ossified, ossification commencing above and extending downward. The anterior surface of the body presents, in the middle line, a vertical crest, the sphenoidal crest, which articulates with the perpendicular plate of the ethmoid, and forms part of the septum of the nose. On either side of the crest is an irregular opening leading into the corresponding sphenoidal air sinus. These sinuses are 2 large irregular cavities hollowed out of the interior of the body of the bone, and separated from one another by a bony septum, which is commonly bent to one or the other side. They vary considerably in form and size, are seldom symmetrical, and are often partially subdivided by irregular bony laminae. Footnote. Aldrin Turner gives the following as their average measurements: vertical height 7/8 inch, antero-posterior depth 7/8 inch, transverse breadth 3/4 inch. End footnote. Occasionally they extend into the basilar part of the occipital nearly as far as the foramen magnum. They begin to be developed before birth, and are of a considerable size by the age of 6. They are partially closed, in front and below, by 2 thin, curved plates of bone, the sphenoidal conchae, leaving in the articulated skull a round opening at the upper part of each sinus, by which it communicates with the upper and back part of the nasal cavity, and occasionally with the posterior ethmoidal air cells. The lateral margin of the anterior surface is serrated, and articulates with the lamina papyracea of the ethmoid, completing the posterior ethmoidal cells. The lower margin articulates with the orbital process of the palatine bone, and the upper with the orbital plate of the frontal bone. The inferior surface presents, in the middle line, a triangular spine, the sphenoidal rostrum, which is continuous with the sphenoidal crest on the anterior surface, and is received in a deep fissure between the alae of the vomer. On either side of the rostrum is a projecting lamina, the vaginal process, directed medialward from the base of the medial pterygoid plate, with which it will be described. The great wings, ali magni. The great wings, or allisphenoids, are 2 strong processes of bone, which arise from the sides of the body, and are curved upward, lateralward, and backward. The posterior part of each projects as a triangular process, which fits into the angle between the squama and the petrous portion of the temporal, and presents at its apex a downwardly directed process, the spina angularis, or sphenoidal spine. Surfaces. The superior or cerebral surface of each great wing forms part of the middle fossa of the skull. It is deeply concave, and presents depressions for the convolutions of the temporal lobe of the brain. At its anterior and medial part is a circular aperture, the foramen rotundum, for the transmission of the maxillary nerve. Behind and lateral to this is the foramen ovale, for the transmission of the mandibular nerve, the accessory meningeal artery, and sometimes the lesser superficial petrosal nerve. Footnote. The lesser superficial petrosal nerve sometimes passes through a special canal, canaliculus innominatus of Arnold, situated medial to the foramen spinosum. End footnote. Medial to the foramen ovale, a small aperture, the foramen vessalii, may occasionally be seen opposite the root of the pterygoid process. It opens below near the scaphoid fossa, and transmits a small vein from the cavernous sinus. Lastly, in the posterior angle, near to and in front of the spine, is a short canal, sometimes double, the foramen spinosum, which transmits the medial meningeal vessels and a recurrent branch from the mandibular nerve. The lateral surface is convex and divided by a transverse ridge, the infratemporal crest, into 2 portions. The superior or temporal portion, convex from above downward, concave from before backward, forms a part of the temporal fossa, and gives attachment to the temporalis. The inferior or infratemporal, smaller in size and concave, enters into the formation of the infratemporal fossa, and, together with the infratemporal crest, affords attachment to the pterygoideus externus. It is pierced by the foramen ovale and foramen spinosum, and at its posterior part is the spina angularis, which is frequently grooved on its medial surface for the chorda tympani nerve. To the spina angularis are attached the sphenomandibular ligament and the tensor veli palatini. Medial to the anterior extremity of the infratemporal crest is a triangular process which serves to increase the attachment of the pterygoideus externus. Extending downward and medialward from this process onto the front part of the lateral pterygoid plate is a ridge which forms the anterior limit of the infratemporal surface, and, in the articulated skull, the posterior boundary of the pterygomaxillary fissure. The orbital surface of the great wing, smooth and quadrilateral in shape, is directed forward and medialward, and forms the posterior part of the lateral wall of the orbit. Its upper serrated edge articulates with the orbital plate of the frontal. Its inferior rounded border forms the posterolateral boundary of the inferior orbital fissure. Its medial sharp margin forms the lower boundary of the superior orbital fissure, and has projecting from about its center a little tubercle which gives attachment to the inferior head of the rectus lateralis oculi. At the upper part of this margin is a notch for the transmission of a recurrent branch of the lacrimal artery. Its lateral margin is serrated and articulates with the zygomatic bone. Below the medial end of the superior orbital fissure is a grooved surface which forms the posterior wall of the pterygopalatine fossa and is pierced by the foramen rotundum. Margin. Commencing from behind, that portion of the circumference of the great wing which extends from the body to the spine is irregular. Its medial half forms the anterior boundary of the foramen lacerum, and presents the posterior aperture of the pterygoid canal for the passage of the corresponding nerve and artery. Its lateral half articulates, by means of a synchondrosis, with the petrous portion of the temporal, and between the two bones on the under surface of the skull is a furrow, the sulcus tubi, for the lodgment of the cartilaginous part of the auditory tube. In front of the spine the circumference presents a concave serrated edge, beveled at the expense of the inner table below and of the outer table above, for articulation with the temporal squama. At the tip of the great wing is a triangular portion, beveled at the expense of the internal surface, for articulation with the sphenoidal angle of the parietal bone. This region is named the pterion. Medial to this is a triangular serrated surface for articulation with the frontal bone. This surface is continuous medially with the sharp edge, which forms the lower boundary of the superior orbital fissure, and laterally with the serrated margin for articulation with the zygomatic bone. The small wings, ali parvi. The small wings, or orbitosphenoids, are two thin triangular plates, which arise from the upper and anterior parts of the body, and, projecting lateralward, end in sharp points. Surfaces. The superior surface of each is flat, and supports part of the frontal lobe of the brain. The inferior surface forms the back part of the roof of the orbit, and the upper boundary of the superior orbital fissure. This fissure is of a triangular form, and leads from the cavity of the cranium into that of the orbit. It is bounded medially by the body, above by the small wing, below by the medial margin of the orbital surface of the great wing, and is completed laterally by the frontal bone. It transmits the oculomotor, trochlear, and abducent nerves, the three branches of the ophthalmic division of the trigeminal nerve, some filaments from the cavernous plexus of the sympathetic, the orbital branch of the middle meningeal artery, a recurrent branch from the lacrimal artery to the dura mater, and the ophthalmic vein. Borders. The anterior border is serrated for articulation with the frontal bone. The posterior border, smooth and rounded, is received into the lateral fissure of the brain. The medial end of this border forms the anterior clinoid process, which gives attachment to the tentorium cerebelli. It is sometimes joined to the middle clinoid process by a spicule of bone, and when this occurs, the termination of the groove for the internal carotid artery is converted into a foramen, caroticoclinoid. The small wing is connected to the body by two roots, the upper thin and flat, the lower thick and triangular. Between the two roots is the optic foramen for the transmission of the optic nerve and ophthalmic artery. Pterygoid processes, processus pterygoidii. The pterygoid processes, one on either side, descend perpendicularly from the regions where the body and great wings unite. Each process consists of a medial and a lateral plate, the upper parts of which are fused anteriorly. A vertical sulcus, the pterygopalatine groove, descends on the front of the line of fusion. The plates are separated below by an angular cleft, the pterygoid fissure, the margins of which are rough for articulation with the pyramidal process of the palatine bone. The two plates diverge behind and enclose between them a V-shaped fossa, the pterygoid fossa, which contains the pterygoideus internus and tensor veli palatini. Above this fossa is a small, oval, shallow depression, the scaphoid fossa, which gives origin to the tensor veli palatini. The anterior surface of the pterygoid process is broad and triangular near its root, where it forms the posterior wall of the pterygopalatine fossa, and presents the anterior orifice of the pterygoid canal. Lateral pterygoid plate. The lateral pterygoid plate is broad, thin, and everted. Its lateral surface forms part of the medial wall of the infratemporal fossa, and gives attachment to the pterygoideus externus. Its medial surface forms part of the pterygoid fossa, and gives attachment to the pterygoideus internus. Medial pterygoid plate. The medial pterygoid plate is narrower and longer than the lateral. It curves lateralward at its lower extremity into a hook-like process, the pterygoid hamulus, around which the tendon of the tensor veli palatini glides. The lateral surface of this plate forms part of the pterygoid fossa. The medial surface constitutes the lateral boundary of the choana, or posterior aperture of the corresponding nasal cavity. Superiorly, the medial plate is prolonged onto the undersurface of the body as a thin lamina, named the vaginal process, which articulates in front with the sphenoidal process of the palatine, and behind this with the ala of the vomer. The angular prominence between the posterior margin of the vaginal process and the medial border of the scaphoid fossa is named the pterygoid tubercle, and immediately above this is the posterior opening of the pterygoid canal. On the undersurface of the vaginal process is a furrow, which is converted into a canal by the sphenoidal process of the palatine bone, for the transmission of the pharyngeal branch of the internal maxillary artery and the pharyngeal nerve from the sphenopalatine ganglion. The pharyngeal aponeurosis is attached to the entire length of the posterior edge of the medial plate, and the constrictor pharyngeus superior takes origin from its lower third. Projecting backward from near the middle of the posterior edge of this plate is an angular process, the processus tubarius, which supports the pharyngeal end of the auditory tube. The anterior margin of the plate articulates with the posterior border of the vertical part of the palatine bone. The sphenoidal conchi, conchi sphenoidalis, sphenoidal turbinated processes. The sphenoidal conchi are two thin curved plates situated at the anterior and lower part of the body of the sphenoid. An aperture of variable size exists in the anterior wall of each, and through this the sphenoidal sinus opens into the nasal cavity. Each is irregular in form and tapers to a point behind, being broader and thinner in front. Its upper surface is concave and looks toward the cavity of the sinus. Its undersurface is convex and forms part of the roof of the corresponding nasal cavity. Each bone articulates in front with the ethmoid, laterally with the palatine. Its pointed posterior extremity is placed above the vomer, and is received between the root of the pterygoid process laterally and the rostrum of the sphenoid medially. A small portion of the sphenoidal concha sometimes enters into the formation of the medial wall of the orbit, between the lamina papyracea of the ethmoid in front, the orbital plate of the palatine below, and the frontal bone above. Ossification. Until the 7th or 8th month of fetal life, the body of the sphenoid consists of two parts, that is, one in front of the tuberculum celli, the pre-sphenoid, with which the small wings are continuous, the other comprising the sella turcica and dorsum celli, the post-sphenoid, with which are associated the great wings and pterygoid processes. The greater part of the bone is ossified in cartilage. There are 14 centers in all, 6 for the pre-sphenoid and 8 for the post-sphenoid. Pre-sphenoid. About the 9th week of fetal life an ossific center appears for each of the small wings, orbitosphenoids, just lateral to the optic foramen. Shortly afterward 2 nuclei appear in the pre-sphenoid part of the body. The sphenoidal conchi are each developed from a center which makes its appearance about the 5th month. At birth they consist of small triangular laminae, and it is not until the 3rd year that they become hollowed out and cone-shaped. About the 4th year they fuse with the labyrinths of the ethmoid, and between the 9th and 12th years they unite with the sphenoid. Footnote: according to Cleland, each sphenoidal concha is ossified from 4 centers. End footnote. Post-sphenoid. The first ossific nuclei are those for the great wings, allospheноids. Footnote: Mall, American Journal of Anatomy, 1906, states that the pterygoid center appears first in an embryo 57 days old. End footnote. One makes its appearance in each wing between the foramen rotundum and foramen ovale about the 8th week. The orbital plate and that part of the sphenoid which is found in the temporal fossa, as well as the lateral pterygoid plate, are ossified in membrane. Fawcett. Soon after, the centers for the post-sphenoid part of the body appear, one on either side of the sella turcica, and become blended together about the middle of fetal life. Each medial pterygoid plate, with the exception of its hamulus, is ossified in membrane, and its center probably appears about the 9th or 10th week. The hamulus becomes chondrified during the 3rd month, and almost at once undergoes ossification. Fawcett. The medial joins the lateral pterygoid plate about the 6th month. About the 4th month a center appears for each lingula, and speedily joins the rest of the bone. The presphenoid is united to the postsphenoid about the 8th month, and at birth the bone is in 3 pieces, a central, consisting of the body and small wings, and 2 lateral, each comprising a great wing and pterygoid process. In the first year after birth the great wings and body unite, and the small wings extend inward above the anterior part of the body, and, meeting with each other in the middle line, form an elevated smooth surface termed the jugum sphenoidale. By the 25th year the sphenoid and occipital are completely fused. Between the pre- and post-sphenoid there are occasionally seen the remains of a canal, the canalus craniopharyngeus, through which, in early fetal life, the hypophyseal diverticulum of the buccal ectoderm is transmitted. The sphenoidal sinuses are present as minute cavities at the time of birth, onodi, but do not attain their full size until after puberty. Intrinsic ligaments of the sphenoid. The more important of these are the pterygospinous, stretching between the spina angularis and the lateral pterygoid plate, see cervical fascia; the interclinoid, a fibrous process joining the anterior to the posterior clinoid process; and the caroticoclinoid, connecting the anterior to the middle clinoid process. These ligaments occasionally ossify. Articulations. The sphenoid articulates with 12 bones, 4 single, the vomer, ethmoid, frontal, and occipital, and 4 paired, the parietal, temporal, zygomatic, and palatine. Footnote: It also sometimes articulates with the tuberosity of the maxilla. End footnote. Section 25. Ethmoid bone. Os ethmoidale. The ethmoid bone is exceedingly light and spongy, and cubical in shape. It is situated at the anterior part of the base of the cranium, between the 2 orbits, at the roof of the nose, and contributes to each of these cavities. It consists of 4 parts, a horizontal or cribriform plate, forming part of the base of the cranium, a perpendicular plate, constituting part of the nasal septum, and 2 lateral masses or labyrinths. Cribriform plate, lamina cribrosa, horizontal lamina. The cribriform plate is received into the ethmoidal notch of the frontal bone and roofs in the nasal cavities. Projecting upward from the middle line of this plate is a thick, smooth, triangular process, the crista galli, so called from its resemblance to a cock's comb. The long, thin posterior border of the crista galli serves for the attachment of the falx cerebri. Its anterior border, short and thick, articulates with the frontal bone, and presents 2 small projecting alli, which are received into corresponding depressions in the frontal bone, and complete the foramen cecum. Its sides are smooth, and sometimes bulging from the presence of a small air sinus in the interior. On either side of the crista galli the cribriform plate is narrow and deeply grooved. It supports the olfactory bulb and is perforated by foramina for the passage of the olfactory nerves. The foramina in the middle of the groove are small and transmit the nerves to the roof of the nasal cavity. Those at the medial and lateral parts of the groove are larger. The former transmit the nerves to the upper part of the nasal septum, the latter those to the superior nasal concha. At the front part of the cribriform plate, on either side of the crista galli, is a small fissure which is occupied by a process of dura mater. Lateral to this fissure is a notch or foramen which transmits the nasociliary nerve. From this notch a groove extends backward to the anterior ethmoidal foramen. Perpendicular plate, lamina perpendicularis, vertical plate. The perpendicular plate is a thin, flattened lamina, polygonal in form, which descends from the under surface of the cribriform plate and assists in forming the septum of the nose. It is generally deflected a little to one or the other side. The anterior border articulates with the spine of the frontal bone and the crest of the nasal bones. The posterior border articulates by its upper half with the sphenoidal crest, by its lower with the vomer. The inferior border is thicker than the posterior, and serves for the attachment of the septal cartilage of the nose. The surfaces of the plate are smooth, except above, where numerous grooves and canals are seen. These lead from the medial foramina on the cribriform plate and lodge filaments of the olfactory nerves. The labyrinth or lateral mass, Labyrinthus ethmoidalis, consists of a number of thin-walled cellular cavities, the ethmoidal cells, arranged in three groups, anterior, middle, and posterior, and interposed between two vertical plates of bone. The lateral plate forms part of the orbit, the medial part of the corresponding nasal cavity. In the disarticulated bone many of these cells are opened into, but when the bones are articulated they are closed in at every part, except where they open into the nasal cavity. Surfaces. The upper surface of the labyrinth presents a number of half-broken cells, the walls of which are completed, in the articulated skull, by the edges of the ethmoidal notch of the frontal bone. Crossing this surface are two grooves, converted into canals by articulation with the frontal. They are the anterior and posterior ethmoidal canals, and open on the inner wall of the orbit. The posterior surface presents large irregular cellular cavities, which are closed in by articulation with the sphenoidal concha and orbital process of the palatine. The lateral surface is formed of a thin, smooth, oblong plate, the lamina papyracea, os planum, which covers in the middle and posterior ethmoidal cells, and forms a large part of the medial wall of the orbit. It articulates above with the orbital plate of the frontal bone, below with the maxilla and orbital process of the palatine, in front with the lacrimal, and behind with the sphenoid. In front of the lamina papyracea are some broken air cells which are overlapped and completed by the lacrimal bone and the frontal process of the maxilla. A curved lamina, the uncinate process, projects downward and backward from this part of the labyrinth. It forms a small part of the medial wall of the maxillary sinus, and articulates with the ethmoidal process of the inferior nasal concha. The medial surface of the labyrinth forms part of the lateral wall of the corresponding nasal cavity. It consists of a thin lamella which descends from the under surface of the cribriform plate, and ends below in a free, convoluted margin, the middle nasal concha. It is rough, and marked above by numerous grooves, directed nearly vertically downward from the cribriform plate. They lodge branches of the olfactory nerves, which are distributed to the mucous membrane covering the superior nasal concha. The back part of the surface is subdivided by a narrow oblique fissure, the superior meatus of the nose, bounded above by a thin curved plate, the superior nasal concha. The posterior ethmoidal cells open into this meatus. Below and in front of the superior meatus is the convex surface of the middle nasal concha. It extends along the whole length of the medial surface of the labyrinth, and its lower margin is free and thick. The lateral surface of the middle concha is concave, and assists in forming the middle meatus of the nose. The middle ethmoidal cells open into the central part of this meatus, and a sinuous passage, termed the infundibulum, extends upward and forward through the labyrinth and communicates with the anterior ethmoidal cells, and in about 50% of skulls is continued upward as the frontonasal duct into the frontal sinus. Ossification. The ethmoid is ossified in the cartilage of the nasal capsule by three centers, one for the perpendicular plate and one for each labyrinth. The labyrinths are first developed, ossific granules making their appearance in the region of the lamina papyracea between the 4th and 5th months of fetal life, and extending into the conchae. At birth the bone consists of the two labyrinths, which are small and ill developed. During the first year after birth the perpendicular plate and crista galli begin to ossify from a single center, and are joined to the labyrinths about the beginning of the second year. The cribriform plate is ossified partly from the perpendicular plate and partly from the labyrinths. The development of the ethmoidal cells begins during fetal life. Articulations. The ethmoid articulates with 15 bones, 4 of the cranium, the frontal, the sphenoid, and the 2 sphenoidal conchae, and 11 of the face, the 2 nasals, 2 maxillae, 2 lacrymals, 2 palatines, 2 inferior nasal conchae, and the vomer. Sutural or Wormian bones. Footnote: Ole Wurm, professor of anatomy at Copenhagen, 1624 to 1639, was erroneously supposed to have given the first detailed description of these bones. End footnote. In addition to the usual centers of ossification of the cranium, others may occur in the course of the sutures, giving rise to irregular, isolated bones termed sutural or Wormian bones. They occur most frequently in the course of the lambdoidal suture, but are occasionally seen at the fontanelles, especially the posterior. 1. The pterion ossicle sometimes exists between the sphenoidal angle of the parietal and the great wing of the sphenoid. They have a tendency to be more or less symmetrical on the two sides of the skull, and vary much in size. Their number is generally limited to 2 or 3, but more than 100 have been found in the skull of an adult hydrocephalic subject.
5b. The facial bones. 1. The nasal bones. Ossa faciae and ossa nasalia. The nasal bones are 2 small oblong bones, varying in size and form in different individuals. They are placed side by side at the middle and upper part of the face, and form, by their junction, the bridge of the nose. Each has 2 surfaces and 4 borders. Surfaces. The outer surface is concavo-convex from above downward, convex from side to side. It is covered by the procerus and compressor naris, and perforated about its centre by a foramen for the transmission of a small vein. The inner surface is concave from side to side, and is traversed from above downward by a groove for the passage of a branch of the nasociliary nerve. Borders. The superior border is narrow, thick, and serrated for articulation with the nasal notch of the frontal bone. The inferior border is thin, and gives attachment to the lateral cartilage of the nose. Near its middle is a notch which marks the end of the groove just referred to. The lateral border is serrated, beveled at the expense of the inner surface above, and of the outer below, to articulate with the frontal process of the maxilla. The medial border, thicker above than below, articulates with its fellow of the opposite side, and is prolonged behind into a vertical crest, which forms part of the nasal septum. This crest articulates, from above downward, with the spine of the frontal, the perpendicular plane of the ethmoid, and the septal cartilage of the nose. Ossification. Each bone is ossified from one centre, which appears at the beginning of the 3rd month of fetal life in the membrane overlying the front part of the cartilaginous nasal capsule. Articulations. The nasal articulates with 4 bones, 2 of the cranium, the frontal and ethmoid, and 2 of the face, the opposite nasal and the maxilla. 5b. The maxillae, upper jaw. The maxillae are the largest bones of the face, excepting the mandible, and form, by their union, the whole of the upper jaw. Each assists in forming the boundaries of 3 cavities, viz., the roof of the mouth, the floor and lateral wall of the nose, and the floor of the orbit. It also enters into the formation of 2 fossae, the infratemporal and pterygopalatine, and 2 fissures, the inferior orbital and pterygomaxillary. Each bone consists of a body and 4 processes, zygomatic, frontal, alveolar, and palatine. The body, corpus maxillae. The body is somewhat pyramidal in shape, and contains a large cavity, the maxillary sinus, antrum of Highmore. It has 4 surfaces, an anterior, a posterior or infratemporal, a superior or orbital, and a medial or nasal. Surfaces. The anterior surface is directed forward and lateralward. It presents at its lower part a series of eminences corresponding to the positions of the roots of the teeth. Just above those of the incisor teeth is a depression, the incisive fossa, which gives origin to the depressor alae nasi. The alveolar border below the fossa is attached a slip of the orbicularis oris. Above and a little lateral to it, the nasalis arises. Lateral to the incisive fossa is another depression, the canine fossa. It is larger and deeper than the incisive fossa, and is separated from it by a vertical ridge, the canine eminence, corresponding to the socket of the canine tooth, and the canine fossa gives origin to the caninus. Above the fossa is the infraorbital foramen, the end of the infraorbital canal. It transmits the infraorbital vessels and nerve. Above the foramen is the margin of the orbit, which affords attachment to part of the quadratus labii superioris. Medially, the anterior surface is limited by a deep concavity, the nasal notch, the margin of which gives attachment to the dilator naris posterior, and ends below in a pointed process, which with its fellow of the opposite side forms the anterior nasal spine. The infratemporal surface is convex, directed backward and lateralward, and forms part of the infratemporal fossa. It is separated from the anterior surface by the zygomatic process and by a strong ridge, extending upward from the socket of the first molar tooth. It is pierced about its centre by the apertures of the alveolar canals, which transmit the posterior superior alveolar vessels and nerves. At the lower part of this surface is a rounded eminence, the maxillary tuberosity, especially prominent after the growth of the wisdom tooth. It is rough on the lateral side for articulation with the pyramidal process of the palatine bone, and in some cases articulates with the lateral pterygoid plate of the sphenoid. It gives origin to a few fibres of the pterygoideus internus. Immediately above this is a smooth surface, which forms the anterior boundary of the pterygopalatine fossa, and presents a groove for the maxillary nerve. This groove is directed lateralward and slightly upward, and is continuous with the infraorbital groove on the orbital surface. The orbital surface is smooth and triangular, and forms the greater part of the floor of the orbit. It is bounded medially by an irregular margin which in front presents a notch, the lacrimal notch. Behind this notch the margin articulates with the lacrimal, the lamina papyracea of the ethmoid, and the orbital process of the palatine. It is bounded behind by a smooth rounded edge which forms the anterior margin of the inferior orbital fissure, and sometimes articulates at its lateral extremity with the orbital surface of the great wing of the sphenoid. It is limited in front by part of the circumference of the orbit, which is continuous medially with the frontal process, and laterally with the zygomatic process. Near the middle of the posterior part of the orbital surface is the infraorbital groove, for the passage of the infraorbital vessels and nerve. The groove begins at the middle of the posterior border, where it is continuous with that near the upper edge of the infratemporal surface, and passing forward ends in a canal which subdivides into 2 branches. One of the canals, the infraorbital canal, opens just below the margin of the orbit. The other, which is smaller, runs downward in the substance of the anterior wall of the maxillary sinus, and transmits the anterior superior alveolar vessels and nerve to the front teeth of the maxilla. From the back part of the infraorbital canal, a second small canal is sometimes given off. It runs downward in the lateral wall of the sinus, and conveys the middle alveolar nerve to the premolar teeth. At the medial and fore part of the orbital surface, just lateral to the lacrimal groove, is a depression, which gives origin to the obliquus oculi inferior. The nasal surface presents a large irregular opening leading into the maxillary sinus. At the upper border of this aperture are some broken air cells, which in the articulated skull are closed in by the ethmoid and lacrimal bones. Below the aperture is a smooth concavity which forms part of the inferior meatus of the nasal cavity, and behind it is a rough surface for articulation with the perpendicular part of the palatine bone. This surface is traversed by a groove commencing near the middle of the posterior border and running obliquely downward and forward. The groove is converted into a canal, the pterygopalatine canal, by the palatine bone. In front of the opening of the sinus is a deep groove, the lacrimal groove, which is converted into the nasolacrimal canal by the lacrimal bone and inferior nasal concha. This canal opens into the inferior meatus of the nose and transmits the nasolacrimal duct. More anteriorly is an oblique ridge, the conchal crest, for articulation with the inferior nasal concha. The shallow concavity above this ridge forms part of the atrium of the middle meatus of the nose, and that below it part of the inferior meatus. The maxillary sinus, or antrum of Highmore, sinus maxillaris. The maxillary sinus is a large pyramidal cavity within the body of the maxilla. Its apex, directed lateralward, is formed by the zygomatic process, its base, directed medialward, by the lateral wall of the nose. Its walls are everywhere exceedingly thin, and correspond to the nasal orbit, anterior and infratemporal surfaces of the body of the bone. Its nasal wall or base presents, in the disarticulated bone, a large irregular aperture communicating with the nasal cavity. In the articulated skull this aperture is much reduced in size by the following bones: the unsonate process of the ethmoid bone, the ethmoidal process of the inferior nasal concha below, the vertical part of the palatine behind, and a small part of the lacrimal above and in front. The sinus communicates with the middle meatus of the nose, generally by two small apertures left between the above-mentioned bones. In the fresh state, usually only one small opening exists, near the upper part of the cavity, the other is closed by mucous membrane. On the posterior wall are the alveolar canals, transmitting the posterior superior alveolar vessels and nerves to the molar teeth. The floor is formed by the alveolar process of the maxilla, and if the sinus be of an average size, is on a level with the floor of the nose. If the sinus be large, it reaches below this level. Projecting into the floor of the antrum are several conical processes, corresponding to the roots of the first and second molar teeth. In some case the floor is perforated by the fangs of the teeth. The infraorbital canal usually projects into the cavity as a well-marked ridge extending from the roof to the anterior wall. Additional ridges are sometimes seen in the posterior wall of the cavity and are caused by the alveolar canals. The size of the cavity varies in different skulls, and even on the two sides of the same skull. The zygomatic process, Processus zygomaticus, malar process. The zygomatic process is a rough triangular eminence, situated at the angle of separation of the anterior, zygomatic and orbital surfaces. In front it forms part of the anterior surface, behind it is concave, and forms part of the infratemporal fossa. Above it is rough and serrated for articulation with the zygomatic bone, while below it presents the prominent arched border which marks the division between the anterior and infratemporal surfaces. The frontal process, Processus frontalis, nasal process. The frontal process is a strong plate which projects upward, medialward, and backwards, by the side of the nose, forming part of its lateral boundary. Its lateral surface is smooth, continuous with the anterior surface of the body, and gives attachment to the quadratus labii superioris, the orbicularis oculi, and the medial palpebral ligament. Its medial surface forms part of the lateral wall of the nasal cavity. At its upper part is a rough, uneven area, which articulates with the ethmoid, closing in the anterior ethmoidal cells. Below this is an oblique ridge, the ethmoidal crest, the posterior end of which articulates with the middle nasal concha, while the anterior part is termed the agonaceae. The crest forms the upper limit of the atrium of the middle meatus. The upper border articulates with the frontal bone and the anterior with the nasal. The posterior border is thick and hollowed into a groove, which is continuous below with the lacrimal groove on the nasal surface of the body. By the articulation of the medial margin of the groove with the anterior border of the lacrimal, a corresponding groove on the lacrimal is brought into continuity, and together they form the lacrimal fossa for the lodgment of the lacrimal sac. The lateral margin of the groove is named the anterior lacrimal crest, and continues below with the orbital margin. At its junction with the orbital surface is a small tubercle, the lacrimal tubercle, which serves as a guide to the position of the lacrimal sac. The alveolar process, Processus alveolaris. The alveolar process is the thickest and most spongy part of the bone. It is broader behind than in front, and excavated into deep cavities for the reception of the teeth. These cavities are 8 in number and vary in size and depth according to the teeth they contain. That for the canine tooth is the deepest. Those for the molars are the widest and are subdivided into minor cavities by septa. Those for the incisors are single but deep and narrow. The buccinator arises from the outer surface of this process, as far forward as the first molar tooth. When the maxillae are articulated with each other, their alveolar processes together form the alveolar arch. The centre of the anterior margin of this arch is named the alveolar point. The palatine process. Processus palatinus, palatal process. The palatine process, thick and strong, is horizontal and projects medialward from the nasal surface of the bone. It forms a considerable part of the floor of the nose and the roof of the mouth and is much thicker in front than behind. Its inferior surface is concave, rough and uneven, and forms, with the palatine process of the opposite bone, the anterior 3/4 of the hard plate. It is perforated by numerous foramina for the passage of the nutrient vessels. Is channeled at the back part of its lateral border by a groove, sometimes a canal, for the transmission of the descending palatine vessels and the anterior palatine nerve from the sphenopalatine ganglion, and presents little depressions for the lodgment of the palatine glands. When the two maxillae are articulated, a funnel-shaped opening, the incisive foramen, is seen in the middle line, immediately behind the incisor teeth. In this opening the orifices of 2 lateral canals are visible. They are named the incisive canals or foramina of Stenson. Through each of them passes the terminal branch of the descending palatine artery and the nasopalatine nerve. Occasionally 2 additional canals are present in the middle line. They are termed the foramina of Scarpa, and when present transmit the nasopalatine nerves, the left passing through the anterior and the right through the posterior canal. On the undersurface of the palatine process, a delicate linear suture, well seen in young skulls, may sometimes be noticed extending lateralward and forward on either side from the incisive foramen to the interval between the lateral incisor and the canine tooth. The small part in front of this suture constitutes the premaxilla, or incisivum, which in most vertebrates forms an independent bone. It includes the whole thickness of the alveolus, the corresponding part of the floor of the nose and the anterior nasal spine, and contains the sockets of the incisor teeth. The upper surface of the palatine process is concave from side to side, smooth, and forms the greater part of the floor of the nasal cavity. It presents, close to its medial margin, the upper orifice of the incisive canal. The lateral border of the process is incorporated with the rest of the bone. The medial border is thicker in front than behind, and is raised above into a ridge, the nasal crest, which, with the corresponding ridge of the opposite bone, forms a groove for the reception of the voma. The front part of this ridge rises to a considerable height, and is named the incisor crest. It is prolonged forward into a sharp process, which forms, together with a similar process of the opposite bone, the anterior nasal spine. The posterior border is serrated for articulation with the horizontal part of the palatine bone. Ossification. The maxilla is ossified in membrane. Mull and Fawcett maintain that it is ossified from 2 centres only, one for the maxilla proper and one for the premaxilla. These centres appear during the 6th week of fetal life, and unite in the beginning of the 3rd month, but the suture between the 2 portions persists on the palate until nearly middle life. Mull states that the frontal process is developed from both centres. The maxillary sinus appears as a shallow groove on the nasal surface of the bone about the 4th month of fetal life, but does not reach its full size until after the 2nd dentition. The maxilla was formerly described as ossifying from 6 centres, viz. 1. The orbiter nasal forms that part of the body of the bone which lies medial to the infraorbital canal, including the medial part of the floor of the orbit and the lateral wall of the nasal cavity. A second, the zygomatic, gives origin to the portion which lies lateral to the infraorbital canal, including the zygomatic process. From the third, the palatine, is developed the palatine process posterior to the incisive canal together with the adjoining part of the nasal wall. A fourth, the premaxillary, forms the incisive bone which carries the incisor teeth and corresponds to the premaxilla of the lower vertebrates. Some anatomists believe that the premaxillary bone is ossified by 2 centres. A fifth, the nasal, gives rise to the frontal process and the portion above the canine tooth. And a sixth, the infravomarine, lies between the palatine and the premaxillary centres and beneath the vomer. This centre, together with the corresponding centre of the opposite bone, separates the incisive canals from each other. Articulations. The maxilla articulates with 9 bones, 2 of the cranium, the frontal and ethmoid, and 7 of the face, viz. The nasal, zygomatic, lacrimal, inferior nasal concha, palatine, vomer, and its fellow of the opposite side. Sometimes it articulates with the orbital surface, and sometimes with the lateral pterygoid plate of the sphenoid. Changes produced in the maxilla by age. At birth, the transverse and antero-posterior diameters of the bone are each greater than the vertical. The frontal process is well marked, and the body of the bone consists of little more than the alveolar process, the teeth sockets reaching almost to the floor of the orbit. The maxillary sinus presents the appearance of a furrow on the lateral wall of the nose. In the adult, the vertical diameter is the greatest, owing to the development of the alveolar process and the increase in size of the sinus. In old age, the bone reverts in some measure to the infantile condition. Its height is diminished, and after the loss of the teeth the alveolar process is absorbed, and the lower part of the bone contracted and reduced in thickness.
The lacrimal bone, os lacrimale. The lacrimal bone, the smallest and most fragile bone of the face, is situated at the front part of the medial wall of the orbit. It has 2 surfaces and 4 borders. Surfaces. The lateral or orbital surface is divided by a vertical ridge, the posterior lacrimal crest, into 2 parts. In front of this crest is a longitudinal groove, the lacrimal sulcus, sulcus lacrimalis, the inner margin of which unites with the frontal process of the maxilla, and the lacrimal fossa is thus completed. The upper part of this fossa lodges the lacrimal sac, the lower part the nasolacrimal duct. The portion behind the crest is smooth, and forms part of the medial wall of the orbit. The crest, with a part of the orbital surface immediately behind it, gives origin to the lacrimal part of the orbicularis oculi, and ends below in a small hook-like projection, the lacrimal hamulus, which articulates with the lacrimal tubercle of the maxilla, and completes the upper orifice of the lacrimal canal. It sometimes exists as a separate piece, and is then called the lesser lacrimal bone. The medial or nasal surface presents a longitudinal furrow corresponding to the crest on the lateral surface. The area in front of this furrow forms part of the middle meatus of the nose. That behind it articulates with the ethmoid, and completes some of the anterior ethmoidal cells. Borders. Of the 4 borders, the anterior articulates with the frontal process of the maxilla, the posterior with the lamina papyracea of the ethmoid, the superior with the frontal bone. The inferior is divided by the lower edge of the posterior lacrimal crest into 2 parts. The posterior part articulates with the orbital plate of the maxilla. The anterior is prolonged downwards as the descending process, which articulates with the lacrimal process of the inferior nasal concha, and assists in forming the canal for the nasolacrimal duct. Ossification. The lacrimal is ossified from a single centre, which appears about the 12th week in the membrane covering the cartilaginous nasal capsule. Articulations. The lacrimal articulates with 4 bones, 2 of the cranium, the frontal and ethmoid, and 2 of the face, the maxilla and the inferior nasal concha. The zygomatic bone. Os zygomaticum, malar bone. The zygomatic bone is small and quadrangular, and is situated at the upper and lateral part of the face. It forms the prominence of the cheek, part of the lateral wall and floor of the orbit, and parts of the temporal and infratemporal fossae. It presents a malar and a temporal surface. 4 processes, the frontosphenoidal, orbital, maxillary, and temporal, and 4 borders. Surfaces. The malar surface is convex and perforated near its centre by a small aperture, the zygomaticofacial foramen, for the passage of the zygomaticofacial nerve and vessels. Below this foramen is a slight elevation, which gives origin to the zygomaticus. The temporal surface, directed backward and medialward, is concave, presenting medially a rough triangular area for articulation with the maxilla, and laterally a smooth concave surface, the upper part of which forms the anterior boundary of the temporal fossa, the lower a part of the infratemporal fossa. Near the centre of this surface is the zygomaticotemporal foramen, for the transmission of the zygomaticotemporal nerve. Processes. The frontosphenoidal process is thick and serrated, and articulates with the zygomatic process of the frontal bone. On its orbital surface, just within the orbital margin, and about 11 mm below the zygomaticofrontal suture, is a tubercle of varying size and form, but present in 95% of skulls. The orbital process is a thick, strong plate, projecting backward and medialward from the orbital margin. Its anteromedial surface forms, by its junction with the orbital surface of the maxilla and with the great wing of the sphenoid, part of the floor and lateral wall of the orbit. On it are seen the orifices of 2 canals, the zygomatico-orbital foramina. One of these canals opens into the temporal fossa, the other on the malar surface of the bone. The former transmits the zygomatico-temporal, the latter the zygomatico-facial nerve. Its posterolateral surface, smooth and convex, forms part of the temporal and infratemporal fossae. Its anterior margin, smooth and rounded, is part of the circumference of the orbit. Its superior margin, rough and directed horizontally, articulates with the frontal bone behind the zygomatic process. Its posterior margin is serrated for articulation with the great wing of the sphenoid and the orbital surface of the maxilla. At the angle of junction of the sphenoidal and maxillary portions, a short, concave, non-articular part is generally seen. This forms the anterior boundary of the inferior orbital fissure. Occasionally this non-articular part is absent, the fissure then being completed by the junction of the maxilla and sphenoid, or by the interposition of a small sutural bone in the angular interval between them. The maxillary process presents a rough triangular surface which articulates with the maxilla. The temporal process, long, narrow, and serrated, articulates with the zygomatic process of the temporal. Borders. The antero-superior or orbital border is smooth, concave, and forms a considerable part of the circumference of the orbit. The antero-inferior or maxillary border is rough, and bevelled at the expense of its inner table, to articulate with the maxilla. Near the orbital margin it gives origin to the quadratus labii superioris. The postero-superior or temporal border, curved like an italic letter F, is continuous above with the commencement of the temporal line, and below with the upper border of the zygomatic arch. The temporal fascia is attached to it. The postero-inferior or zygomatic border affords attachment by its rough edge to the masseter. Ossification. The zygomatic bone is generally described as ossifying from 3 centres, 1 for the malar and 2 for the orbital portion. These appear about the 8th week, and fuse about the 5th month of fetal life. Mal describes it as being ossified from 1 centre which appears just beneath and to the lateral side of the orbit. After birth the bone is sometimes divided by a horizontal suture into an upper larger and a lower smaller division. In some quadrumana the zygomatic bone consists of 2 parts, an orbital and a malar. Articulations. The zygomatic articulates with 4 bones, the frontal, sphenoidal, temporal, and maxilla. The palatine bone, os palatinum, palate bone. The palatine bone is situated at the back part of the nasal cavity between the maxilla and the pterygoid process of the sphenoid. It contributes to the walls of 3 cavities, the floor and lateral wall of the nasal cavity, the roof of the mouth, and the floor of the orbit. It enters into the formation of 2 fossae, the pterygopalatine and the pterygoid fossae, and 1 fissure, the inferior orbital fissure. The palatine bone somewhat resembles the letter L, and consists of a horizontal and a vertical part, and 3 outstanding processes, viz. The pyramidal process, which is directed backward and lateralward from the junction of the 2 parts, and the orbital and sphenoidal processes, which surmount the vertical part, and are separated by a deep notch, the sphenopalatine notch. The horizontal part, pars horizontalis, horizontal plate. The horizontal part is quadrilateral and has 2 surfaces and 4 borders. Surfaces. The superior surface, concave from side to side, forms the back part of the floor of the nasal cavity. The inferior surface, slightly concave and rough, forms, with the corresponding surface of the opposite bone, the posterior fourth of the hard palate. Near its posterior margin may be seen a more or less marked transverse ridge for the attachment of part of the aponeurosis of the tensor veli palatini. Borders. The anterior border is serrated and articulates with the palatine process of the maxilla. The posterior border is concave, free, and serves for the attachment of the soft palate. Its medial end is sharp and pointed, and when united with that of the opposite bone forms a projecting process, the posterior nasal spine, for the attachment of the musculus uvulae. The lateral border is united with the lower margin of the perpendicular part, and is grooved by the lower end of the pterygopalatine canal. The medial border, the thickest, is serrated for articulation with its fellow of the opposite side. Its superior edge is raised into a ridge, which, united with the ridge of the opposite bone, forms the nasal crest for articulation with the posterior part of the lower edge of the vomer. The vertical part, pars perpendicularis, perpendicular plate. The vertical part is thin, of an oblong form, and presents 2 surfaces and 4 borders. Surfaces. The nasal surface exhibits at its lower part a broad, shallow depression, which forms part of the inferior meatus of the nose. Immediately above this is a well-marked horizontal ridge, the conchal crest, for articulation with the inferior nasal concha. Still higher is a second broad, shallow depression, which forms part of the middle meatus, and is limited above by a horizontal crest less prominent than the inferior, the ethmoidal crest, for articulation with the middle nasal concha. Above the ethmoidal crest is a narrow horizontal groove, which forms part of the superior meatus. The maxillary surface is rough and irregular throughout the greater part of its extent, for articulation with the nasal surface of the maxilla. Its upper and back part is smooth where it enters into the formation of the pterygopalatine fossa. It is also smooth in front, where it forms the posterior part of the medial wall of the maxillary sinus. On the posterior part of this surface is a deep vertical groove, converted into the pterygopalatine canal by articulation with the maxilla. This canal transmits the descending palatine vessels and the anterior palatine nerve. Borders. The anterior border is thin and irregular, Opposite the conchal crest is a pointed projecting lamina, the maxillary process, which is directed forward and closes in the lower and back part of the opening of the maxillary sinus. The posterior border presents a deep groove, the edges of which are serrated for articulation with the medial pterygoid plate of the sphenoid. This border is continuous above with the sphenoidal process. Below it expands into the pyramidal process. The superior border supports the orbital process in front and the sphenoidal process behind. These processes are separated by the sphenopalatine notch, which is converted into the sphenopalatine foramen by the undersurface of the body of the sphenoid. In the articulated skull, this foramen leads from the pterygopalatine fossa into the posterior part of the superior meatus of the nose, and transmits the sphenopalatine vessels and the superior nasal and nasopalatine nerves. The inferior border is fused with the lateral edge of the horizontal part, and immediately in front of the pyramidal process is grooved by the lower end of the pterygopalatine canal. The pyramidal process or tuberosity. Processus pyramidalis. The pyramidal process projects backward and lateralward from the junction of the horizontal and vertical parts, and is received into the angular interval between the lower extremities of the pterygoid plates. On its posterior surface is a smooth grooved triangular area, limited on either side by a rough articular furrow. The furrows articulate with the pterygoid plates, while the grooved intermediate area completes the lower part of the pterygoid fossa and gives origin to a few fibres of the pterygoideus internus. The anterior part of the lateral surface is rough for articulation with the tuberosity of the maxilla. Its posterior part consists of a smooth triangular area, which appears in the articulated skull between the tuberosity of the maxilla and the lower part of the lateral pterygoid plate, and completes the lower part of the infratemporal fossa. On the base of the pyramidal process, close to its union with the horizontal part, are the lesser palatine foramina for the transmission of the posterior and middle palatine nerves. The orbital process. Processus orbitalis. The orbital process is placed on a higher level than the sphenoidal, and is directed upward and lateralward from the front of the vertical part, to which it is connected by a constricted neck. It presents 5 surfaces which enclose an air cell. Of these surfaces, 3 are articular and 2 non-articular. The articular surfaces are 1. The anterior or maxillary, directed forward, lateralward, and downward, of an oblong form and rough for articulation with the maxilla. 2. The posterior or sphenoidal, directed backward, upward, and medialward. It presents the opening of the air cell, which usually communicates with the sphenoidal sinus. The margins of the opening are serrated for articulation with the sphenoidal concha. 3. The medial or ethmoidal, directed forward, articulates with the labyrinth of the ethmoid. In some cases the air cell opens on this surface of the bone, and then communicates with the posterior ethmoidal cells. More rarely it opens on both surfaces, and then communicates with the posterior ethmoidal cells and the sphenoidal sinus. The non-articular surfaces are 1. The superior or orbital, directed upward and lateralward. It is triangular in shape and forms the back part of the floor of the orbit, and 2. The lateral, of an oblong form, directed toward the pterygopalatine fossa. It is separated from the orbital surface by a rounded border, which enters into the formation of the inferior orbital fissure. The sphenoidal process. Processus sphenoidalis. The sphenoidal process is a thin compressed plate, much smaller than the orbital, and directed upward and medialward. It presents 3 surfaces and 2 borders. The superior surface articulates with the root of the pterygoid process and the under surface of the sphenoidal concha, its medial border reaching as far as the ala of the voma. It presents a groove which contributes to the formation of the pharyngeal canal. The medial surface is concave and forms part of the lateral wall of the nasal cavity. The lateral surface is divided into an articular and a non-articular portion. The former is rough, for articulation with the medial pterygoid plate. The latter is smooth, and forms part of the pterygopalatine fossa. The anterior border forms the posterior boundary of the sphenopalatine notch. The posterior border, serrated at the expense of the outer table, articulates with the medial pterygoid plate. The orbital and sphenoidal processes are separated from one another by the sphenopalatine notch. Sometimes the 2 processes are united above and form between them a complete foramen, or the notch may be crossed by 1 or more spicules of bone, giving rise to 2 or more foramina. Ossification. The palatine bone is ossified in membrane from a single centre, which makes its appearance about the 6th or 8th week of fetal life at the angle of junction of the 2 parts of the bone. From this point ossification spreads medialward to the horizontal part, downward into the pyramidal process, and upward into the vertical part. Some authorities describe the bone as ossifying from 4 centres, 1 for the pyramidal process and portion of the vertical part behind the pterygopalatine groove, a 2nd for the rest of the vertical and the horizontal parts, a 3rd for the orbital, and a 4th for the sphenoidal process. At the time of birth, the height of the vertical part is about equal to the transverse width of the horizontal part, whereas in the adult the former measures about twice as much as the latter. Articulations. The palatine articulates with 6 bones: the sphenoid, ethmoid, maxilla, inferior nasal concha, voma, and opposite palatine.
Section 29. The inferior nasal concha, the voma. Concha nasalis inferior, inferior turbinated bone. The inferior nasal concha extends horizontally along the lateral wall of the nasal cavity, and consists of a lamina of spongy bone curled upon itself like a scroll. It has 2 surfaces, 2 borders, and 2 extremities. The medial surface is convex, perforated by numerous apertures and traversed by longitudinal grooves for the lodgment of vessels. The lateral surface is concave, and forms part of the inferior meatus. Its upper border is thin, irregular, and connected to various bones along the lateral wall of the nasal cavity. It may be divided into 3 portions. Of these, the anterior articulates with the conchal crest of the maxilla, the posterior with the conchal crest of the palatine. The middle portion represents 3 well-marked processes, which vary much in their size and form. Of these, the anterior or lacrimal process is small and pointed, and is situated at the junction of the anterior 4th with the posterior 3/4 of the bone. It articulates by its apex with the descending process of the lacrimal bone, and by its margins with the groove on the back of the frontal processes of the maxilla, and thus assists in forming the canal for the nasolacrimal duct. Behind this process a broad, thin plate, the ethmoidal process, ascends to join the uncinate process of the ethmoid. From its lower border a thin lamina, the maxillary process, curves downward and lateralward. It articulates with the maxilla and forms a part of the medial wall of the maxillary sinus. The inferior border is free, thick, and cellular in structure, more especially in the middle of the bone. Both extremities are more or less pointed, the posterior being the more tapering. Ossification. The inferior nasal concha is ossified from a single centre, which appears about in the 5th month of fetal life in the lateral wall of the cartilaginous nasal capsule. Articulations. The inferior nasal concha articulates with 4 bones, the ethmoid, maxilla, lacrimal, and palatine. 5b7. The vomer. The vomer is situated in the median plane, but its anterior portion is frequently bent to one or other side. It is thin, somewhat quadrilateral in shape, and forms the hinder and lower parts of the nasal septum. It has 2 surfaces and 4 borders. The surfaces are marked by small furrows for blood vessels, and on each is the nasopalatine groove, which runs obliquely downward and forward, and lodges the nasopalatine nerve and vessels. The superior border, the thickest, presents a deep furrow, bounded on either side by a horizontal projecting ala of bone. The furrow receives the rostrum of the sphenoid, while the margins of the alae articulate with the vaginal process of the medial pterygoid plates of the sphenoid behind, and with the sphenoidal process of the palatine bones in front. The inferior border articulates with the crest formed by the maxilla and palatine bones. The anterior border is the longest and slopes downward and forward. Its upper half is fused with the perpendicular plate of the ethmoid. Its lower half is grooved for the inferior margin of the septal cartilage of the nose. The posterior border is free, concave, and separates the choani. It is thick and bifid above, thin below. Ossification. At an early period, the septum of the nose consists of a plate of cartilage, the ethmovomerian cartilage. The postero-superior part of this cartilage is ossified to form the perpendicular plate of the ethmoid. Its antero-inferior portion persists as the septal cartilage, while the vomer is ossified in the membrane covering its postero-inferior part. 2 ossific centres, one on either side of the middle line, appear about the 8th week of fetal life in this part of the membrane, and hence the vomer consists primarily of 2 lamellae. About the 3rd month these unite below, and thus a deep groove is formed in which the cartilage is lodged. As growth proceeds, the union of the lamellae extend upward and forward, and at the same time the intervening plate of cartilage undergoes absorption. By the age of puberty, the lamellae are almost completely united to form a median plate, but evidence of the bilaminar origin of the bone is seen in the inverted alae of its upper border and the groove on its anterior margin. Articulation. The vomer articulates with 6 bones, 2 of the cranium, the sphenoid and ethmoid, and 4 of the face, the 2 maxillae and 2 palatine bones. It also articulates with the septal cartilage of the nose.
The mandible. 5b, Part 8. The mandible, lower jaw. Inferior maxillary bone. The mandible, the largest and strongest bone of the face, serves for the reception of the lower teeth. It consists of a curved horizontal portion, the body, and 2 perpendicular portions, the rami, which unite with the ends of the body nearly at right angles. The body, corpus mandibuli. The body is curved somewhat like a horseshoe and has 2 surfaces and 2 borders. Surfaces. The external surface is marked in the median line by a faint ridge indicating the symphysis or line of junction of the 2 pieces of which the bone is composed at an early period of life. This ridge divides below and encloses a triangular eminence, the mentoprotuberance, the base of which is depressed in the center but raised on either side to form the mental tubercle. On either side of the symphysis, just below the incisor teeth, is a depression, the incisive fossa, which gives origin to the mentalis and a small portion of the orbicularis oris. Below the second premolar tooth on either side, midway between the upper and lower borders of the body, is the mental foramen, for the passage of the mental vessels and nerve. Running backward and upward from each mental tubercle is a faint ridge, the oblique line, which is continuous with the anterior border of the ramus. It affords attachment to the quadratus labii inferioris and triangularis. The platysma is attached below it. The internal surface is concave from side to side. Near the lower part of the symphysis is a pair of laterally placed spines, termed the mental spines, which give origin to the genioglossae. Immediately below these is a second pair of spines, or more frequently a median ridge or impression, for the origin of the geniohyoidii. In some cases the mental spines are fused to form a single eminence, in others they are absent and their position is indicated merely by an irregularity of the surface. Above the mental spines a median foramen and furrow are sometimes seen. They mark the line of union of the halves of the bone. Below the mental spines, on either side of the middle line, is an oval depression for the attachment of the anterior belly of the digastricus. Extending upward and backward on either side from the lower part of the symphysis is the myeloid line, which gives origin to the mylohyoideus. The posterior part of this line, near the alveolar margin, gives attachment to a small part of the constrictor pharyngeus superior, and to the pterygomandibular raphe. Above the anterior part of this line is a smooth triangular area against which the sublingual gland rests, and below the hinder part an oval fossa for the submaxillary gland. Borders. The superior or alveolar border, wider behind than in front, is hollowed into cavities for the reception of the teeth. These cavities are 16 in number and vary in depth and size according to the teeth which they contain. To the outer lip of the superior border on either side, the buccinator is attached as far forward as the first molar tooth. The inferior border is rounded, longer than the superior, and thicker in front than behind. At the point where it joins the lower border of the ramus a shallow groove, for the external maxillary artery, may be present. The ramus. Ramus mandibuli, perpendicular portion. The ramus is quadrilateral in shape and has two surfaces, four borders, and two processes. Surfaces. The lateral surface is flat and marked by oblique ridges at its lower part. It gives attachment throughout nearly the whole of its extent to the masseter. The medial surface presents about its center the oblique mandibular foramen, for the entrance of the inferior alveolar vessels and nerve. The margin of this opening is irregular. It presents in front a prominent ridge, surmounted by a sharp spine, the lingula mandibuli, which gives attachment to the sphenomandibular ligament. At its lower and back part is a notch from which the myloid groove runs obliquely downward and forward, and lodges the myloid vessels and nerve. Behind this groove is a rough surface, for the insertion of the pterygoideus internus. The mandibular canal runs obliquely downward and forward in the ramus, and then horizontally forward in the body, where it is placed under the alveoli and communicates with them by small openings. On arriving at the incisor teeth it turns back to communicate with the mental foramen, giving off two small canals which run to the cavities containing the incisor teeth. In the posterior 2/3 of the bone the canal is situated nearer the internal surface of the mandible, and in the anterior third nearer its external surface. It contains the inferior alveolar vessels and nerve, from which branches are distributed to the teeth. The lower border of the ramus is thick, straight, and continuous with the inferior border of the body of the bone. At its junction with the posterior border is the angle of the mandible, which may be either inverted or everted, and is marked by rough, oblique ridges on each side for the attachment of the masseter laterally and the pterygoideus internus medially. The stylomandibular ligament is attached to the angle between these muscles. The anterior border is thin above, thicker below, and continuous with the oblique line. The posterior border is thick, smooth, rounded, and covered by the parotid gland. The upper border is thin and is surmounted by two processes, the coronoid in front and the condyloid behind, separated by a deep concavity, the mandibular notch. The coronoid process, processus coronoideus, is a thin, triangular eminence which is flattened from side to side and varies in shape and size. Its anterior border is convex and is continuous below with the anterior border of the ramus. Its posterior border is concave and forms the anterior boundary of the mandibular notch. Its lateral surface is smooth and affords insertion to the temporalis and masseter. Its medial surface gives insertion to the temporalis and presents a ridge which begins near the apex of the process and runs downward and forward to the inner side of the last molar tooth. Between this ridge and the anterior border is a grooved triangular area, the upper part of which gives attachment to the temporalis, the lower part to some fibers of the buccinator. The condyloid process, processus condyloideus, is thicker than the coronoid and consists of two portions, the condyle and the constricted portion which supports it, the neck. The condyle presents an articular surface for articulation with the articular disc of the temporomandibular joint. It is convex from before backward and from side to side, and extends farther on the posterior than on the anterior surface. Its long axis is directed medialward and slightly backward, and if prolonged to the middle line will meet that of the opposite condyle near the anterior margin of the foramen magnum. At the lateral extremity of the condyle is a small tubercle for the attachment of the temporomandibular ligament. The neck is flattened from before backward and strengthened by ridges which descend from the forepart and sides of the condyle. Its posterior surface is convex, its anterior presents a depression for the attachment of the pterygoideus externus. The mandibular notch, separating the two processes, is a deep semilunar depression and is crossed by the masseteric vessels and nerve. Ossification. The mandible is ossified in the fibrous membrane covering the outer surfaces of Meckel's cartilages. These cartilages form the cartilaginous bar of the mandibular arch and are two in number, a right and a left. Their proximal or cranial ends are connected with the ear capsules, and their distal extremities are joined to one another at the symphysis by mesodermal tissue. They run forward immediately below the condyles and then, bending downward, lie in a groove near the lower border of the bone. In front of the canine tooth they incline upward to the symphysis. From the proximal end of each cartilage, the malleus and incus, two of the bones of the middle ear, are developed. The next succeeding portion, as far as the lingula, is replaced by fibrous tissue, which persists to form the sphenomandibular ligament. Between the lingula and the canine tooth the cartilage disappears, while the portion of it below and behind the incisor teeth becomes ossified and incorporated with this part of the mandible. Ossification takes place in the membrane covering the outer surface of the ventral end of Meckel's cartilage, and each half of the bone is formed from a single center which appears, near the mental foramen, about the 6th week of fetal life. By the 10th week the portion of Meckel's cartilage which lies below and behind the incisor teeth is surrounded and invaded by the membrane bone. Somewhat later, accessory nuclei of cartilage make their appearance, viz. a wedge-shaped nucleus in the condyloid process, and extending downward through the ramus, a small strip along the anterior border of the coronoid process, and smaller nuclei in the front part of both alveolar walls and along the front of the lower border of the bone. These accessory nuclei possess no separate ossific centers, but are invaded by the surrounding membrane bone and undergo absorption. The inner alveolar border, usually described as arising from a separate ossific center, splenial center, is formed in the human mandible by an ingrowth from the main mass of the bone. At birth the bone consists of two parts, united by a fibrous symphysis, in which ossification takes place during the first year. The foregoing description of the ossification of the mandible is based on the researches of Lowe and Fawcett, and differs somewhat from that usually given. Articulations. The mandible articulates with the two temporal bones. Changes produced in the mandible by age. At birth the body of the bone is a mere shell, containing the sockets of the two incisor, the canine, and the two deciduous molar teeth, imperfectly partitioned off from one another. The mandibular canal is of large size, and runs near the lower border of the bone. The mental foramen opens beneath the socket of the first deciduous molar tooth. The angle is obtuse, 175 degrees, and the condyloid portion is nearly in line with the body. The coronoid process is of comparatively large size, and projects above the level of the condyle. After birth, the two segments of the bone become joined at the symphysis, from below upward, in the first year, but a trace of separation may be visible in the beginning of the second year, near the alveolar margin. The body becomes elongated in its whole length, but more especially behind the mental foramen, to provide space for the 3 additional teeth developed in this part. The depth of the body increases owing to increased growth of the alveolar part, to afford room for the roots of the teeth, and by thickening of the subdental portion which enables the jaw to withstand the powerful action of the masticatory muscles. But the alveolar portion is the deeper of the two, and consequently the chief part of the body lies above the oblique line. The mandibular canal, after the second dentition, is situated just above the level of the myloioid line, and the mental foramen occupies the position usual to it in the adult. The angle becomes less obtuse, owing to the separation of the jaws by the teeth. About the 4th year it is 140 degrees. In the adult, the alveolar and subdental portions of the body are usually of equal depth. The mental foramen opens midway between the upper and lower borders of the bone, and the mandibular canal runs nearly parallel with the myloioid line. The ramus is almost vertical in direction, the angle measuring from 110 to 120 degrees. In old age the bone becomes greatly reduced in size, for with the loss of the teeth the alveolar process is absorbed, and consequently the chief part of the bone is below the oblique line. The mandibular canal, with the mental foramen opening from it, is close to the alveolar border. The ramus is oblique in direction, the angle measures about 140 degrees, and the neck of the condyle is more or less bent backward. 5b, Section 9, the hyoid bone. Os hyoideum, lingual bone. The hyoid bone is shaped like a horseshoe, and is suspended from the tips of the styloid processes of the temporal bones by the styloid ligaments. It consists of 5 segments, viz., a body, 2 greater cornua, and 2 lesser cornua. The body, or basihil, corpus os hyoidei. The body or central part is of a quadrilateral form. Its anterior surface is convex and directed forward and upward. It is crossed in its upper half by a well-marked transverse ridge with a slight downward convexity, and in many cases a vertical median ridge divides it into 2 lateral halves. The portion of the vertical ridge above the transverse line is present in a majority of specimens, but the lower portion is evident only in rare cases. The anterior surface gives insertion to the geniohyoideus in the greater part of its extent, both above and below the transverse ridge. A portion of the origin of the hyoglossus notches the lateral margin of the geniohyoideus attachment. Below the transverse ridge the mylohyoideus, sternohyoideus, and omohyoideus are inserted. The posterior surface is smooth, concave, directed backward and downward, and separated from the epiglottis by the hyothyroid membrane and a quantity of loose areolar tissue. A bursa intervenes between it and the hyothyroid membrane. The superior border is rounded and gives attachment to the hyothyroid membrane and some aponeurotic fibers of the genioglossus. The inferior border affords insertion medially to the sternohyoideus and laterally to the omohyoideus, and occasionally a portion of the thyrohyoideus. It also gives attachment to the levator glandulae thyroidei, when this muscle is present. In early life the lateral borders are connected to the greater cornua by synchondroses, after middle life usually by bony union. The greater cornua, or thyrohyals, cornua majora. The greater cornua project backward from the lateral borders of the body. They are flattened from above downward and diminish in size from before backward. Each ends in a tubercle to which is fixed the lateral hyothyroid ligament. The upper surface is rough close to its lateral border for muscular attachments. The largest of these are the origins of the hyoglossus and constrictor pharyngeus medius, which extend along the whole length of the cornu. The digastricus and stylohyoideus have small insertions in front of these near the junction of the body with the cornu. To the medial border the hyothyroid membrane is attached, while the anterior half of the lateral border gives insertion to the thyrohyoideus. The lesser cornua, or ceratohyals, cornua minora. The lesser cornu are two small conical eminences attached by their bases to the angles of junction between the body and greater cornua. They are connected to the body of the bone by fibrous tissue, and occasionally to the greater cornua by distinct diarthrodial joints, which usually persist throughout life, but occasionally become ankylosed. The lesser cornua are situated in the line of the transverse ridge on the body, and appear to be morphological continuations of it. The apex of each cornu gives attachment to the stylohyoid ligament. Footnote: These ligaments in many animals are distinct bones, and in man may undergo partial ossification. End footnote. The chondroglossus rises from the medial side of the base. Ossification. The hyoid is ossified from 6 centers, 2 for the body and 1 for each cornu. Ossification commences in the greater cornua toward the end of fetal life, in the body shortly afterward, and in the lesser cornua during the first or second year after birth.
The exterior of the skull. Part 1. The skull as a whole may be viewed from different points, and the views so obtained are termed the normae of the skull. Thus, it may be examined from above, norma verticalis, from below, norma basalis, from the side, norma lateralis, from behind, norma occipitalis, or from the front, norma frontalis. Norma verticalis. When viewed from above, the outline presented varies greatly in different skulls. In some it is more or less oval, in others more nearly circular. The surface is traversed by 3 sutures, viz. 1. The coronal sutures, nearly transverse in direction, between the frontal and parietals. 2. The sagittal sutures, medially placed, between the parietal bones, and deeply serrated in its anterior 2/3. And 3. The upper part of the lambdoidal suture, between the parietals and the occipital. The point of junction of the sagittal and coronal suture is named the bregma, that of the sagittal and lambdoid sutures the lambda. They indicate respectively the positions of the anterior and posterior fontanelles in the fetal skull. On either side of the sagittal suture are the parietal eminence and parietal foramen. The latter, however, is frequently absent on one or both sides. The skull is often somewhat flattened in the neighbourhood of the parietal foramina, and the term obelian is applied to that point of the sagittal suture which is on a level with the foramina. In front is the glabella, and on its lateral aspects are the superciliary arches, and above these the frontal eminences. Immediately above the glabella may be seen the remains of the frontal suture. In a small percentage of skulls this suture persists and extends along the middle line to the bregma. Passing backward and upward from the zygomatic processes of the frontal bone are the temporal lines, which mark the upper limits of the temporal fossae. The zygomatic arches may or may not be seen projecting beyond the anterior portion of these lines. Norma basalis. The inferior surface of the base of the skull, exclusive of the mandible, is bounded in front by the incisor teeth in the maxillae, behind by the superior nuchal lines of the occipital, and laterally by the alveolar arch, the lower border of the zygomatic bone, the zygomatic arch, and an imaginary line extending from it to the mastoid process and extremity of the superior nuchal line of the occipital. It is formed by the palatine processes of the maxillae and palatine bones, the vomer, the pterygoid processes, the under surfaces of the great wings, spinous processes, and part of the body of the sphenoid, the under surfaces of the squamae and mastoid and petrous portions of the temporals, and the undersurface of the occipital bone. The anterior part or hard palate projects below the level of the rest of the surface, and is bounded in front and laterally by the alveolar arch containing the 16 teeth of the maxillae. Immediately behind the incisor teeth is the incisive foramen. In this foramen are two lateral apertures, the openings of the incisive canals, foramina of Stenson, which transmit the anterior branches of the descending palatine vessels and the nasopalatine nerves. Occasionally, two additional canals are present in the incisive foramen. They are termed the foramina of Scarpa and are situated in the middle line. When present, they transmit the nasopalatine nerves. The vault of the hard palate is concave, uneven, perforated by numerous foramina, marked by depressions for the palatine glands, and traversed by a crucial suture formed by the junction of the four bones of which it is composed. In the young skull a suture may be seen extending on either side from the incisive foramen to the interval between the lateral incisor and canine teeth, and marking off the os incisivum or premaxillary bone. At either posterior angle of the hard palate is the greater palatine foramen, for the transmission of the descending palatine vessels and anterior palatine nerve, and running forward and medialward from it is a groove, for the same vessels and nerve. Behind the posterior palatine foramen is the pyramidal process of the palatine bone, perforated by one or more lesser palatine foramina, and marked by the commencement of a transverse ridge, for the attachment of the tendinous expansion of the tensor veli palatini. Projecting backward from the centre of the posterior border of the hard palate is the posterior nasal spine, for the attachment of the musculus uvulae. Behind and above the hard palate are the coni, measuring about 2.5 cm in their vertical and 1.25 cm in their transverse diameters. They are separated from one another by the vomer, and each is bounded above by the body of the sphenoid, below by the horizontal part of the palatine bone, and laterally by the medial pterygoid plate of the sphenoid. At the superior border of the vomer may be seen the expanded alae of this bone, receiving between them the rostrum of the sphenoid. Near the lateral margins of the alae of the vomer, at the roots of the pterygoid processes, are the pharyngeal canals. The pterygoid process presents near its base the pterygoid canal, for the transmission of a nerve and artery. The medial pterygoid plate is long and narrow. On the lateral side of its base is the scaphoid fossa, for the origin of the tensor veli palatini, and at its lower extremity the hamulus, around which the tendon of this muscle turns. The lateral pterygoid plate is broad, its lateral surface forms the medial boundary of the infratemporal fossa, and affords attachment of the pterygoideus externus. Behind the nasal cavities is the basilar portion of the occipital bone, presenting near its centre the pharyngeal tubercle for the attachment of the fibrous raphe of the pharynx, with depressions on either side for the insertions of the rectus capitis anterior and longus capitis. At the base of the lateral pterygoid plate is the foramen ovale, for the transmission of the mandibular nerve, the accessory meningeal artery, and sometimes the lesser superficial petrosal nerve. Behind this are the foramen spinosum, which transmits the middle meningeal vessels, and the prominent spina angularis, sphenoidal spine, which gives attachment to the sphenomandibular ligament and the tensor veli palatini. Lateral to the spina angularis is the mandibular fossa, divided into two parts by the petrotympanic fissure, the anterior portion, concave, smooth, bounded in front by the articular tubercle, serves for the articulation of the condyle of the mandible. The posterior portion, rough and bounded behind by the tympanic part of the temporal, is sometimes occupied by a part of the parotid gland. Emerging from between the laminae of the vaginal process of the tympanic part is the styloid process, and at the base of this process is the stylomastoid foramen for the exit of the facial nerve and entrance of the stylomastoid artery. Lateral to the stylomastoid foramen, between the tympanic part and the mastoid process, is the tympanomastoid fissure, for the auricular branch of the vagus. Upon the medial side of the mastoid process is the mastoid notch, for the posterior belly of the digastricus, and medial to the notch, the occipital groove for the occipital artery. At the base of the medial pterygoid plate is a large and somewhat triangular aperture, the foramen lacerum, bounded in front by the great wing of the sphenoid, behind by the apex of the petrous portion of the temporal bone, and medially by the body of the sphenoid and basilar portion of the occipital bone. It presents in front the posterior orifice of the pterygoid canal, behind the aperture of the carotid canal. The lower part of this opening is filled up in the fresh state by a fibrocartilaginous plate, across the upper or cerebral surface of which the internal carotid artery passes. Lateral to this aperture is a groove, the sulcus tubi auditivi, between the petrous part of the temporal and the great wing of the sphenoid. This sulcus is directed lateralward and backward from the root of the medial pterygoid plate and lodges the cartilaginous part of the auditory tube. It is continuous behind with the canal in the temporal bone which forms the bony part of the same tube. At the bottom of this sulcus is a narrow cleft, the petrosphenoidal fissure, which is occupied, in the fresh condition, by a plate of cartilage. Behind this fissure is the undersurface of the petrous portion of the temporal bone, presenting, near its apex, the quadrilateral rough surface, part of which affords attachment to the levator veli palatini. Lateral to this surface is the orifice of the carotid canal, and medial to it the depression leading to the aqueductus cochleae, the former transmitting the internal carotid artery and the carotid plexus of the sympathetic, the latter serving for the passage of a vein from the cochlea. Behind the carotid canal is the jugular foramen, a large aperture, formed in front by the petrous portion of the temporal and behind by the occipital. It is generally larger on the right than on the left side, and may be subdivided into 3 compartments. The anterior compartment transmits the inferior petrosal sinus, the intermediate, the glossopharyngeal, vagus, and accessory nerves, the posterior, the transverse sinus, and some meningeal branches from the occipital and ascending pharyngeal arteries. On the ridge of bone dividing the carotid canal from the jugular foramen is the inferior tympanic canaliculus for the transmission of the tympanic branch of the glossopharyngeal nerve, and on the wall of the jugular foramen, near the root of the styloid process, is the mastoid canaliculus for the passage of the auricular branch of the vagus nerve. Extending forward from the jugular foramen to the foramen lacerum is the petrooccipital fissure, occupied, in the fresh state, by a plate of cartilage. Behind the basilar portion of the occipital bone is the foramen magnum, bounded laterally by the occipital condyles, the medial sides of which are rough for the attachment of the alar ligaments. Lateral to each condyle is the jugular process which gives attachment to the rectus capitis lateralis muscle and the lateral atlanto-occipital ligament. The foramen magnum transmits the medulla oblongata and its membranes, the accessory nerves, the vertebral arteries, the anterior and posterior spinal arteries, and the ligaments connecting the occipital bone with the axis. The midpoints on the anterior and posterior margins of the foramen magnum are respectively termed the basion and the opistheon. In front of each condyle is the canal for the passage of the hypoglossal nerve and a meningeal artery. Behind each condyle is the condyloid fossa, perforated on one or both sides by the condyloid canal for the transmission of a vein from the transverse sinus. Behind the foramen magnum is the medium nuchal line, extending above at the external occipital protuberance, while on either side are the superior and inferior nuchal lines. These, as well as the surfaces of the bone between them, are rough for the attachment of the muscles which are enumerated on pages 129 and 130. Norma lateralis. When viewed from the side, the skull is seen to consist of the cranium above and behind, and of the face below and in front. The cranium is somewhat ovoid in shape, but its contour varies in different cases and depends largely on the length and height of the skull and on the degree of prominence of the superciliary arches and frontal eminences. Entering into its formation are the frontal, the parietal, the occipital, the temporal, and the great wing of the sphenoid. These bones are joined to one another and to the zygomatic by the following sutures: the zygomatico-temporal between the zygomatic process of the temporal and the temporal process of the zygomatic, the zygomatico-frontal uniting the zygomatic bone with the zygomatic process of the frontal, the sutures surrounding the great wing of the sphenoid, viz., the sphenozygomatic in front, the sphenofrontal and sphenoparietal above, and the sphenosquamosal behind. The sphenoparietal suture varies in length in different skulls, and is absent in those cases where the frontal articulates with the temporal squama. The point corresponding with the posterior end of the sphenoparietal suture is named the pterion. It is situated about 3 cm behind, and a little above the level of the zygomatic process of the frontal bone. The squamosal suture arches backward from the pterion and connects the temporal squama with the lower border of the parietal. This suture is continuous behind with the short, nearly horizontal parieto-mastoid suture, which unites the mastoid process of the temporal with the region of the mastoid angle of the parietal. Extending from above downward and forward across the cranium are the coronal and lambdoidal sutures. The former connects the parietals with the frontal, the latter the parietals with the occipital. The lambdoidal suture is continuous below with the occipito-mastoid suture between the occipital and mastoid portion of the temporal. In or near the last suture is the mastoid foramen, for the transmission of an emissary vein. The point of meeting of the parieto-mastoid, occipito-mastoid, and lambdoidal sutures is known as the asterion. Immediately above the orbital margin is the superciliary arch, and at a higher level the frontal eminence. Near the centre of the parietal bone is the parietal eminence. Posteriorly is the external occipital protuberance, from which the superior nuchal line may be followed forward to the mastoid process. Arching across the side of the cranium are the temporal lines, which mark the upper limit of the temporal fossa. The temporal fossa, fossa temporalis. The temporal fossa is bounded above and behind by the temporal lines, which extend from the zygomatic process of the frontal bone upward and backward across the frontal and parietal bones, and then curve downward and forward to become continuous with the supramastoid crest and the posterior root of the zygomatic arch. The point where the upper temporal line cuts the coronal suture is named the Stephanion. The temporal fossa is bounded in front by the frontal and zygomatic bones, and opening on the back of the latter is the zygomatico-temporal foramen. Laterally the fossa is limited by the zygomatic arch, formed by the zygomatic and temporal bones. Below it is separated from the infratemporal fossa by the infratemporal crest on the great wing of the sphenoid, and by a ridge, continuous with this crest, which is carried backward across the temporal squama to the anterior root of the zygomatic process. In front and below, the fossa communicates with the orbital cavity through the inferior orbital or sphenomaxillary fissure. The floor of the fossa is deeply concave in front and convex behind, and is formed by the zygomatic, frontal, parietal, sphenoid, and temporal bones. It is traversed by vascular furrows. One, usually well marked, runs upwards above and in front of the external acoustic meatus, and lodges the middle temporal artery. Two others, frequently indistinct, may be observed on the anterior part of the floor, and are for the anterior and posterior deep temporal arteries. The temporal fossa contains the temporalis muscle and its vessels and nerves, together with the zygomatico-temporal nerve. The zygomatic arch is formed by the zygomatic process of the temporal and the temporal process of the zygomatic, the two being united by an oblique suture. The tendon of the temporalis passes medial to the arch to gain insertion into the coronoid process of the mandible. The zygomatic process of the temporal arises by two routes, an anterior, directed inward in front of the mandibular fossa, where it expands to form the articular tubercle, and a posterior, which runs backwards above the external acoustic meatus and is continuous with the supramastoid crest. The upper border of the arch gives attachment to the temporal fascia. The lower border and medial surface give origin to the masseter. Below the posterior root of the zygomatic arch is the elliptical orifice of the external acoustic meatus, bounded in front, below, and behind by the tympanic part of the temporal bone. To its outer margin the cartilaginous segment of the external acoustic meatus is attached. The small triangular area between the posterior root of the zygomatic arch and the postero-superior part of the orifice is termed the suprameatal triangle, on the anterior border of which a small spinous process, the suprameatal spine, is sometimes seen. Between the tympanic part and the articular tubercle is the mandibular fossa, divided into two parts by the petrotympanic fissure. The anterior and larger part of the fossa articulates with the condyle of the mandible and is limited behind by the external acoustic meatus. The posterior part sometimes lodges a portion of the parotid gland. The styloid process extends downward and forward for a variable distance from the lower part of the tympanic part, and gives attachment to the styloglossus, stylohyoideus, and stylopharyngeus, and to the stylohyoid and stylomandibular ligaments. Projecting downward behind the external acoustic meatus is the mastoid process, to the outer surface of which the sternocleidomastoideus, splenius capitis, and longissimus capitis are attached. The exterior of the skull, Part 2. The infratemporal fossa, fossa infratemporalis, zygomatic fossa, The infratemporal fossa is an irregularly shaped cavity, situated below and medial to the zygomatic arch. It is bounded, in front, by the infratemporal surface of the maxilla and the ridge which descends from its zygomatic process; behind, by the articular tubercle of the temporal and the spinal angularis of the sphenoid; above, by the great wing of the sphenoid below the infratemporal crest, and by the under surface of the temporal squama; below, by the alveolar border of the maxilla; medially, by the lateral pterygoid plate. It contains the lower part of the temporalis, the pterygoideae internus and externus, the internal maxillary vessels, and the mandibular and maxillary nerves. The foramen ovale and foramen spinosum open on its roof, and the alveolar canals on its anterior wall. At its upper and medial part are two fissures, which together form a T-shaped fissure, the horizontal limb being named the inferior orbital, and the vertical one the pterygomaxillary. The inferior orbital fissure, fissura orbitalis inferior, sphenomaxillary fissure, horizontal in direction, opens into the lateral and back part of the orbit. It is bounded above by the lower border of the orbital surface of the great wing of the sphenoid, below by the lateral border of the orbital surface of the maxilla and the orbital process of the palatine bone, laterally by a small part of the zygomatic bone. Note 48. Occasionally the maxilla and the sphenoid articulate with each other at the interior extremity of this fissure. The zygomatic is then excluded from it. Medially it joins at right angles with the pterygomaxillary fissure. Through the inferior orbital fissure the orbit communicates with the temporal, infratemporal, and pterygopalatine fossae. The fissure transmits the maxillary nerve and its zygomatic branch, the infraorbital vessels, the ascending branches from the sphenopalatine ganglion, and a vein which connects the inferior ophthalmic vein with the pterygoid venous plexus. The pterygomaxillary fissure is vertical and descends at right angles from the medial end of the preceding. It is a triangular interval, formed by the divergence of the maxilla from the pterygoid process of the sphenoid. It connects the infratemporal with the pterygopalatine fossa and transmits the terminal part of the internal maxillary artery. The pterygopalatine fossa, fossa pterygopalatina, sphenomaxillary fossa. The pterygopalatine fossa is a small triangular space at the angle of junction of the inferior orbital and pterygomaxillary fissures, and placed beneath the apex of the orbit. It is bounded above by the under surface of the body of the sphenoid and by the orbital process of the palatine bone, in front by the infratemporal surface of the maxilla, behind by the base of the pterygoid process and lower part of the anterior surface of the great wing of the sphenoid, medially by the vertical part of the palatine bone with its orbital and sphenoidal processes. This fossa communicates with the orbit by the inferior orbital fissure, with the nasal cavity by the sphenopalatine foramen, and with the infratemporal fossa by the pterygomaxillary fissure. 5 foramina open into it. Of these, 3 are on the posterior wall, viz., the foramen rotundum, the pterygoid canal, and the pharyngeal canal, in this order downward and medialward. On the medial wall is the sphenopalatine foramen, and below is the superior orifice of the pterygopalatine canal. The fossa contains the maxillary nerve, the sphenopalatine ganglion, and the terminal part of the internal maxillary artery. Norma occipitalis. When viewed from behind, the cranium presents a more or less circular outline. In the middle line is the posterior part of the sagittal suture connecting the parietal bones. Extending downward and lateralward from the hinder end of the sagittal suture is the deeply serrated lambdoidal suture, joining the parietals to the occipital, and continuous below with the parietomastoid and occipitomastoid sutures. It frequently contains one or more sutural bones. Near the middle of the occipital squama is the external occipital protuberance or inion, and extending lateralward from it on either side is the superior nuchal line, and above this the faintly marked highest nuchal line. The part of the squama above the inion and highest lines is named the planum occipitali, and is covered by the occipitalis muscle. The part below is termed the planum nuchale, and is divided by the median nuchal line which runs downward and forward from the inion to the foramen magnum. This ridge gives attachment to the ligamentum nuchae. The muscles attached to the planum nuchale are enumerated on page 130. Below and in front are the mastoid processes, convex laterally and grooved medially by the mastoid notches. In or near the occipital mastoid suture is the mastoid foramen for the passage of the mastoid emissary vein. Norma frontalis. When viewed from the front, the skull exhibits a somewhat oval outline, limited above by the frontal bone, below by the body of the mandible, and laterally by the zygomatic bones and the mandibular rami. The upper part, formed by the frontal squama, is smooth and convex. The lower part, made up of the bones of the face, is irregular. It is excavated laterally by the orbital cavities, and presents in the middle line the anterior nasal aperture leading to the nasal cavities, and below this the transverse slit between the upper and lower dental arcades. Above, the frontal eminences stand out more or less prominently, and beneath these are the superciliary arches, joined to one another in the middle by the glabella. On and above the glabella a trace of the frontal suture sometimes persists. Beneath is the frontonasal suture, the midpoint of which is termed the nasion. Behind and below the frontonasal suture the frontal articulates with the frontal process of the maxilla and with the lacrimal. Arching transversely below the superciliary arches is the upper part of the margin of the orbit, thin and prominent in its lateral 2/3, rounded in its medial third, and presenting at the junction of these two portions the supraorbital notch or foramen for the supraorbital nerve and vessels. The supraorbital margin ends laterally in the zygomatic process which articulates with the zygomatic bone, and from it the temporal line extends upward and backward. Below the frontonasal suture is the bridge of the nose, convex from side to side, concavo-convex from above downward, and formed by the two nasal bones supported in the middle line by the perpendicular plate of the ethmoid, and laterally by the frontal processes of the maxillae which are prolonged upward between the nasal and lacrimal bones and form the lower and medial part of the circumference of each orbit. Below the nasal bones and between the maxillae is the anterior aperture of the nose, piriform in shape, with the narrow end directed upward. Laterally this opening is bounded by sharp margins, to which the lateral and alar cartilages of the nose are attached. Below, the margins are thicker and curve medialward and forward to end in the anterior nasal spine. On looking into the nasal cavity, the bony septum which separates the nasal cavities presents, in front, a large triangular deficiency. This, in the fresh state, is filled up by the cartilage of the nasal septum. On the lateral wall of each nasal cavity the anterior part of the inferior nasal concha is visible. Below and lateral to the anterior nasal aperture are the anterior surfaces of the maxillae, each perforated, near the lower margin of the orbit, by the infraorbital foramen for the passage of the infraorbital nerve and vessels. Below and medial to this foramen is the canine eminence separating the incisive from the canine fossa. Beneath these fossae are the alveolar processes of the maxillae containing the upper teeth, which overlap the teeth of the mandible in front. The zygomatic bone on either side forms the prominence of the cheek, the lower and lateral portion of the orbital cavity, and the anterior part of the zygomatic arch. It articulates medially with the maxilla, behind with the zygomatic process of the temporal, and above with the great wing of the sphenoid and the zygomatic process of the frontal. It is perforated by the zygomaticofacial foramen for the passage of the zygomaticofacial nerve. On the body of the mandible is a median ridge, indicating the position of the symphysis. This ridge divides below to enclose the mental protuberance, the lateral angles of which constitute the mental tubercles. Below the incisor teeth is the incisive fossa, and beneath the second premolar tooth the mental foramen which transmits the mental nerve and vessels. The oblique line runs upward from the mental tubercle and is continuous behind with the anterior border of the ramus. The posterior border of the ramus runs downward and forward from the condyle to the angle, which is frequently more or less everted. The orbits. The orbits are two quadrilateral pyramidal cavities, situated at the upper and anterior part of the face, their bases being directed forward and lateralward, and their apices backward and medialward, so that their long axes, if continued backward, would meet over the body of the sphenoid. Each presents for examination a roof, a floor, a medial and a lateral wall, a base, and an apex. The roof is concave, directed downward and slightly forward, and formed in front by the orbital plate of the frontal, behind by the small wing of the sphenoid. It presents medially the trochlea fovea for the attachment of the cartilaginous pulley of the obliquus oculi superior, laterally the lacrimal fossa for the lacrimal gland, and posteriorly the suture between the frontal bone and the small wing of the sphenoid. The floor is directed upward and lateralward, and is of less extent than the roof. It is formed chiefly by the orbital surface of the maxilla, in front and laterally by the orbital process of the zygomatic bone, and behind and medially, to a small extent, by the orbital process of the palatine. At its medial angle is the upper opening of the nasolacrimal canal, immediately to the lateral side of which is a depression for the origin of the obliquus oculi inferior. On its lateral part is the suture between the maxilla and zygomatic bone, and at its posterior part that between the maxilla and the orbital process of the palatine. Running forward near the middle of the floor is the infraorbital groove, ending in front in the infraorbital canal and transmitting the infraorbital nerve and vessels. The medial wall is nearly vertical, and is formed from before backward by the frontal process of the maxilla, the lacrimal, the lamina papyrachia of the ethmoid, and a small part of the body of the sphenoid in front of the optic foramen. Sometimes the sphenoidal concha forms a small part of this wall. It exhibits 3 vertical sutures, viz. the lacromaxillary, lacrimo-ethmoidal, and sphenoethmoidal. In front is seen the lacrimal groove, which lodges the lacrimal sac, and behind the groove is the posterior lacrimal crest, from which the lacrimal part of the orbicularis oculi arises. At the junction of the medial wall and the roof are the frontomaxillary, frontolacrimal, frontoethmoidal, and sphenofrontal sutures. The point of junction of the anterior border of the lacrimal with the frontal is named the dacrion. In the frontoethmoidal suture are the anterior and posterior ethmoidal foramina, the former transmitting the nasociliary nerve and anterior ethmoidal vessels, the latter the posterior ethmoidal nerve and vessels. The lateral wall, directed medialward and forward, is formed by the orbital process of the zygomatic and the orbital surface of the great wing of the sphenoid. These are united by the sphenozygomatic suture which terminates below at the front end of the inferior orbital fissure. On the orbital process of the zygomatic bone are the orbital tubercle, Whitnell, and the orifices of 1 or 2 canals, which transmit the branches of the zygomatic nerve. Between the roof and the lateral wall, near the apex of the orbit, is the superior orbital fissure. Through this fissure the oculomotor, the trochlea, the ophthalmic division of the trigeminal, and the adjacent nerves enter the orbital cavity. Also some filaments from the cavernous plexus of the sympathetic and the orbital branches of the middle meningeal artery. Passing backward through the fissure are the ophthalmic vein and the recurrent branch from the lacrimal artery to the dura mater. The lateral wall and floor are separated posteriorly by the inferior orbital fissure, which transmits the maxillary nerve and its zygomatic branch, the infraorbital vessels, and the ascending branches from the sphenopalatine ganglion. The base of the orbit, quadrilateral in shape, is formed above by the supraorbital arch of the frontal bone, in which is supraorbital notch or foramen for the passage of the supraorbital vessels and nerve. Below by the zygomatic bone and maxilla, united by the zygomatico-maxillary suture, laterally by the zygomatic bone and the zygomatic process of the frontal joined by the zygomatico-frontal suture, medially by the frontal bone and the frontal process of the maxilla, united by the frontomaxillary suture. The apex, situated at the back of the orbit, corresponds to the optic foramen, a short, cylindrical canal which transmits the optic nerve and ophthalmic artery. Some anatomists describe the apex of the orbit as corresponding with the medial end of the superior orbital fissure. It seems better, however, to adopt the statement in the text, since the ocular muscles take origin around the optic foramen and diverge from it to the bulb of the eye. It will thus be seen that there are 9 openings communicating with each orbit, viz. the optic foramen, superior and inferior orbital fissures, supraorbital foramen, infraorbital canal, anterior and posterior ethmoidal foramina, zygomatic foramen, and the canal for the nasolacrimal duct. The interior of the skull, Part 1. Inner surface of the skull cap. The inner surface of the skull cap is concave and presents depressions for the convolutions of the cerebrum, together with numerous furrows for the lodgment of branches of the middle meningeal vessels. Along the middle line is a longitudinal groove, narrow in front, where it commences at the frontal crest, but broader behind. It lodges the superior sagittal sinus, and its margins afford attachment to the falx cerebri. On either side of it are several depressions for the arachnoid granulations, and, at its back part, the openings of the parietal foramina when these are present. It is crossed in front by the coronal suture, and behind by the lambdoidal, while the sagittal lies in the medial plane between the parietal bones. Upper surface of the base of the skull. The upper surface of the base of the skull or floor of the cranial cavity presents three fossae, called the anterior, middle, and posterior cranial fossae. Anterior fossa. Fossa cranii anterior. The floor of the anterior fossa is formed by the orbital plates of the frontal, the cribriform plate of the ethmoid, and the small wings and front part of the body of the sphenoid. It is limited behind by the posterior borders of the small wings of the sphenoid and by the anterior margin of the chiasmatic groove. It is transversed by the frontoethmoidal, sphenoethmoidal, and sphenofrontal sutures. Its lateral portions roof in the orbital cavities and support the frontal lobes of the cerebrum. They are convex and marked by depressions for the brain convolutions, and grooves for the branches of the meningeal vessels. The central portion corresponds with the roof of the nasal cavity, and is markedly depressed on either side of the crista galli. It presents, in and near the median line, from before backward, the commencement of the frontal crest for the attachment of the falx cerebri, the foramen caecum, between the frontal bone and the crista galli of the ethmoid, which usually transmits a small vein from the nasal cavity to the superior sagittal sinus. Behind the foramen caecum, the crista galli, the free margin of which affords attachment to the falx cerebri on either side of the crista galli, the olfactory groove formed by the cribriform plate, which supports the olfactory bulb and presents foramina for the transmission of the olfactory nerves, and in front a slit-like opening for the nasociliary nerve. Lateral to either olfactory groove are the internal openings of the anterior and posterior ethmoidal foramina. The anterior, situated about the middle of the lateral margin of the olfactory groove, transmits the anterior ethmoidal vessels and the nasociliary nerve. The nerve runs in a groove along the lateral edge of the cribriform plate to the slit-like opening above mentioned. The posterior ethmoidal foramen opens at the back part of this margin under cover of the projecting lamina of the sphenoid, and transmits the posterior ethmoidal vessels and nerve. Farther back in the middle line is the ethmoidal spine, bounded behind by a slight elevation separating two shallow longitudinal grooves which support the olfactory lobes. Behind this is the anterior margin of the chiasmatic groove, running lateralward on either side to the upper margin of the optic foramen. The middle fossa, fossa cranii media. The middle fossa, deeper than the preceding, is narrow in the middle and wide at the sides of the skull. It is bounded in front by the posterior margins of the small wings of the sphenoid, the anterior clinoid processes, and the ridge forming the anterior margin of the chiasmatic groove. Behind, by the superior angles of the petrous portions of the temporals and the dorsum celli. Laterally by the temporal squamae. Sphenoidal angles of the parietals, and great wings of the sphenoid. It is traversed by the squamosal, sphenoparietal, sphenosquamosal, and sphenopetrosal sutures. The middle part of the fossa presents, in front, the chiasmatic groove and tuberculum celli. The chiasmatic groove ends on either side at the optic foramen, which transmits the optic nerve and ophthalmic artery to the orbital cavity. Behind the optic foramen the anterior clinoid process is directed backward and medialward and gives attachment to the tentorium cerebelli. Behind the tuberculum celli is a deep depression, the sella turcica, containing the fossa hypophyseos, which lodges the hypophysis, and presents on its anterior wall the middle clinoid processes. The sella turcica is bounded posteriorly by a quadrilateral plate of bone, the dorsum celli, the upper angles of which are surmounted by the posterior clinoid processes. These afford attachment to the tentorium cerebelli, and below each is a notch for the abducent nerve. On either side of the sella turcica is the carotid groove, which is broad, shallow, and curved, somewhat like the italic letter F. It begins behind at the foramen lacerum and ends on the medial side of the anterior clinoid process, where it is sometimes converted into a foramen, caroticoclinoid, by the union of the anterior with the middle clinoid process. Posteriorly it is bound laterally by the lingula. The groove lodges the cavernous sinus and the internal carotid artery, the latter being surrounded by a plexus of sympathetic nerves. The lateral parts of the middle fossa are of considerable depth and support the temporal lobes of the brain. They are marked by depressions for the brain convolutions and traversed by furrows for the anterior and posterior branches of the middle meningeal vessels. These furrows begin near the foramen spinosum, and the anterior runs forward and upward to the sphenoidal angle of the parietal, where it is sometimes converted into a bony canal. The posterior runs lateralward and backward across the temporal squama and passes on to the parietal near the middle of its lower border. The following apertures are also to be seen. In front is the superior orbital fissure, bounded above by the small wing, below by the great wing, and medially by the body of the sphenoid. It is usually completed laterally by the orbital plate of the frontal bone. It transmits to the orbital cavity the oculomotor, the trochlea, the ophthalmic division of the trigeminal, and the adjacent nerves. Some filaments from the cavernous plexus of the sympathetic, and the orbital branch of the middle meningeal artery, and from the orbital cavity a recurrent branch from the lacrimal artery to the dura mater, and the ophthalmic veins. Behind the middle end of the superior orbital fissure is the foramen rotundum, for the passage of the maxillary nerve. Behind and lateral to the foramen rotundum is the foramen ovale, which transmits the mandibular nerve, the accessory meningeal artery, and the lesser superficial petrosal nerve. Medial to the foramen ovale is the foramen versalii, which varies in size in different individuals, and is often absent. When present it opens below at the lateral side of the scaphoid fossa, and transmits a small vein. Lateral to the foramen ovale is the foramen spinosum, for the passage of the middle meningeal vessels, and a recurrent branch from the mandibular nerve. Medial to the foramen ovale is the foramen lacerum. In the fresh state the lower part of this aperture is filled up by a layer of fibrocartilage, while its upper and inner parts transmit the internal carotid artery surrounded by a plexus of sympathetic nerves. The nerve of the pterygoid canal and a meningeal branch from the ascending pharyngeal artery pierce the layer of fibrocartilage. On the anterior surface of the petrous portion of the temporal bone are seen the eminence caused by the projection of the superior semicircular canal, in front of and a little lateral to this depression corresponding to the roof of the tympanic cavity, the groove leading to the hiatus of the facial canal, for the transmission of the greater superficial petrosal nerve and the petrosal branch of the middle meningeal artery. Beneath it, the smaller groove, for the passage of the lesser superficial petrosal nerve, and, near the apex of the bone, the depression for the semilunar ganglion and the orifice of the carotid canal. The posterior fossa, fossa cranii posterior. The posterior fossa is the largest and deepest of the three. It is formed by the dorsum sellae and clivus of the sphenoid, the occipital, the petrous and mastoid portions of the temporals, and the mastoid angles of the parietal bones. It is crossed by the occipito-mastoid and parieto-mastoid sutures, and lodges the cerebellum, pons, and medulla oblongata. It is separated from the middle fossa in and near the median line by the dorsum sellae of the sphenoid and on either side by the superior angle of the petrous portion of the temporal bone. This angle gives attachment to the tentorum cerebelli, is grooved for the superior petrosal sinus, and presents at its medial end a notch upon which the trigeminal nerve rests. The fossa is limited behind by the grooves for the transverse sinuses. In its centre is the foramen magnum, on either side of which is a rough tubercle for the attachment of the alar ligaments. A little above this tubercle is the canal, which transmits the hypoglossal nerve and a meningeal branch from the ascending pharyngeal artery. In front of the foramen magnum, the basilar portion of the occipital and the posterior part of the body of the sphenoid form a grooved surface which supports the medulla oblongata and pons. In the young skull these bones are joined by a synchondrosis. This grooved surface is separated on either side from the petrous portion of the temporal by the petro-occipital fissure, which is occupied in the fresh state by a plate of cartilage. The fissure is continuous behind with the jugular foramen, and its margins are grooved for the inferior petrosal sinus. The jugular foramen is situated between the lateral part of the occipital and the petrous portion of the temporal. The anterior portion of this foramen transmits the inferior petrosal sinus, the posterior portion, the transverse sinus and some meningeal branches from the occipital and ascending pharyngeal arteries, and the intermediate portion, the glossopharyngeal, vagus and accessory nerves. Above the jugular foramen is the internal acoustic meatus, for the facial and acoustic nerves and internal auditory artery. Behind and lateral to this is the slit-like opening leading into the aqueductus vestibuli, which lodges the ductus endolymphaticus, while between these and near the superior angle of the petrous portion is a small triangular depression, the remains of the fossa subarcuata, which lodges a process of the dura mater and occasionally transmits a small vein. Behind the foramen magnum are the inferior occipital fossae, which support the hemispheres of the cerebellum, separated from one another by the internal occipital crest, which serves for the attachment of the falx cerebelli and lodges the occipital sinus. The posterior fossae are surmounted by the deep grooves for the transverse sinuses. Each of these channels, in its passage to the jugular foramen, grooves the occipital, the mastoid angle of the parietal, the mastoid portion of the temporal, and the jugular process of the occipital, and ends at the back part of the jugular foramen. Where this sinus grooves the mastoid portion of the temporal, the orifice of the mastoid foramen may be seen, and, just previous to its termination, the condyloid canal opens into it. Neither opening is constant. The nasal cavity. Cavum nasi, nasal fossa. The nasal cavities are two irregular spaces, situated one on either side of the middle line of the face, extending from the base of the cranium to the roof of the mouth, and separated from each other by a thin vertical septum. They open on the face through the pear-shaped anterior nasal aperture, and their posterior openings or coni communicate, in the fresh state, with the nasal part of the pharynx. They are much narrower above than below, and in the middle than at the anterior or posterior openings. Their depth, which is considerable, is greatest in the middle. They communicate with the frontal, ethmoidal, sphenoidal, and maxillary sinuses. Each cavity is bounded by a roof, a floor, a medial and a lateral wall. The roof is horizontal in its central part, but slopes downward in front and behind. It is formed in front by the nasal bone and the spine of the frontal, in the middle by the cribriform plate of the ethmoid, and behind by the body of the sphenoid, the sphenoidal concha, the ali of the vomer, and the sphenoidal process of the palatine bone. In the cribriform plate of the ethmoid are the foramina for the olfactory nerves, and on the posterior part of the roof is the opening into the sphenoidal sinus. The floor is flattened from before backward and concave from side to side. It is formed by the palatine process of the maxilla and the horizontal part of the palatine bone. Near its anterior end is the opening of the incisive canal. The medial wall, septum nasi, is frequently deflected to one or other side, more often to the left than to the right. It is formed in front by the crest of the nasal bones and frontal spine, in the middle by the perpendicular plate of the ethmoid, behind by the vomer and the rostrum of the sphenoid, below by the crest of the maxillae and palatine bones. It presents in front a large triangular notch, which receives the cartilage of the septum, and behind the free edge of the vomer. Its surface is marked by numerous furrows for vessels and nerves and by the grooves for the nasopalatine nerve, and is traversed by sutures connecting the bones of which it is formed. The lateral wall is formed in front by the frontal process of the maxilla and by the lacrimal bone, in the middle by the ethmoid maxilla and inferior nasal concha, behind by the vertical plate of the palatine bone and the medial pterygoid plate of the sphenoid. On this wall are three irregular antero-posterior passages, termed the superior, middle, and inferior meatuses of the nose. The superior meatus, the smallest of the three, occupies the middle third of the lateral wall. It lies between the superior and middle nasal conchae, the sphenopalatine foramen opens into it behind, and the posterior ethmoidal cells in front. The sphenoidal sinus opens into a recess, the sphenoethmoidal recess, which is placed above and behind the superior concha. The middle meatus is situated between the middle and inferior conchae and extends from the anterior to the posterior end of the latter. The lateral wall of this meatus can be satisfactorily studied only after the removal of the middle concha. On it is a curved fissure, the hiatus semilunaris, limited below by the edge of the uncinate process of the ethmoid and above by an elevation named the bulla ethmoidalis. The middle ethmoidal cells are contained within this bulla and open on or near it. Throughout the hiatus semilunaris the meatus communicates with a curved passage termed the infundibulum, which communicates in front with the anterior ethmoidal cells, and in rather more than 50% of skulls is continued upward as the frontonasal duct into the frontal air sinus. When this continuity fails, the frontonasal duct opens directly into the anterior part of the meatus. Below the bulla ethmoidalis, and hidden by the uncinate process of the ethmoid, is the opening of the maxillary sinus, ostium maxillare. An accessory opening is frequently present above the posterior part of the inferior nasal concha. The inferior meatus, the largest of the three, is the space between the inferior concha and the floor of the nasal cavity. It extends almost the entire length of the lateral wall of the nose, is broader in front than behind, and presents anteriorly the lower orifice of the nasolacrimal canal. The anterior nasal aperture is a heart-shaped or piriform opening, whose long axis is vertical, and narrow end upward. In the recent state it is much contracted by the lateral and alar cartilages of the nose. It is bounded above by the inferior borders of the nasal bones. Laterally by the thin sharp margins which separate the anterior from the nasal surfaces of the maxillae, and below by the same borders, where they curve medialward to join each other at the anterior nasal spine. The coni are each bounded above by the under surface of the body of the sphenoid and alar of the vomer, below by the posterior border of the horizontal part of the palatine bone, laterally by the medial pterygoid plate. They are separated from each other by the posterior border of the vomer. The interior of the skull, Part 2. Differences in the skull due to age. At birth. The skull is large in proportion to the other parts of the skeleton, but its facial portion is small, and equals only about 1/8 of the bulk of the cranium as compared with 1/2 in the adult. The frontal and parietal eminences are prominent, and the greatest width of the skull is at the level of the latter. On the other hand, the glabella, superciliary arches, and mastoid processes are not developed. Ossification of the skull bones is not completed, and many of them, e.g. the occipital, temporals, sphenoid, frontal, and mandible, consist of more than one piece. Unossified membranous intervals, termed fontanelles, are seen at the angles of the parietal bones. These fontanelles are 6 in number. 2, an anterior and a posterior, are situated in the middle line, and 2, an anterolateral and a posterolateral, on either side. The anterior or bregmatic fontanelle is the largest, and is placed at the junction of the sagittal, coronal, and frontal sutures. It is lozenge-shaped, and measures about 4 cm in its anteroposterior and 2.5 cm in its transverse diameter. The posterior fontanelle is triangular in form and is situated at the junction of the sagittal and lambdoidal sutures. The lateral fontanelles are small, irregular in shape, and correspond respectively with the sphenoidal and mastoidal angles of the parietal bones. An additional fontanelle is sometimes seen in the sagittal suture at the region of the obelion. The fontanelles are usually closed by the growth and extension of the bones which surround them, but sometimes they are the sites of separate ossific centres which develop into sutural bones. The posterior and lateral fontanelles are obliterated within a month or 2 after birth, but the anterior is not completely closed until about the middle of the 2nd year. The smallness of the face at birth is mainly accounted for by the rudimentary condition of the maxillae and mandible, the non-eruption of the teeth, and the small size of the maxillary, air sinuses, and nasal cavities. At birth the nasal cavities lie almost entirely between the orbits, and the lower border of the anterior nasal aperture is only a little below the level of the orbital floor. With the eruption of the deciduous teeth there is an enlargement of the face and jaws, and these changes are still more marked after the 2nd dentition. The skull grows rapidly from birth to the 7th year, by which time the foramen magnum and petrous parts of the temporals have reached their full size, and the orbital cavities are only a little smaller than those of the adult. Growth is slow from the 7th year until the approach of puberty, when a second period of activity occurs. This results in an increase in all directions, but it is especially marked in the frontal and facial regions, where it is associated with the development of the air sinuses. Obliteration of the sutures of the vault of the skull takes place as age advances. This process may commence between the ages of 30 and 40, and is first seen on the inner surface, and some 10 years later on the outer surface of the skull. The dates given are, however, only approximate, as it is impossible to state with anything like accuracy the time at which the sutures are closed. Obliteration usually occurs first in the posterior part of the sagittal suture, next in the coronal, and then in the lambdoidal. In old age the skull generally becomes thinner and lighter, but in a small proportion of cases it increases in thickness and weight, owing to an hypertrophy of the inner table. The most striking feature of the old skull is the diminution in the size of the maxillae and mandible consequent on the loss of the teeth and the absorption of the alveolar processes. This is associated with a marked reduction in the vertical measurement of the face and with an alteration in the angles of the mandible. Sexual differences in the skull. Until the age of puberty there is little difference between the skull of the female and that of the male. The skull of an adult female is, as a rule, lighter and smaller, and its cranial capacity about 10% less than that of the male. Its walls are thinner and its muscular ridges less strongly marked. The glabellar, superciliary arches, and mastoid processes are less prominent, and the corresponding air sinuses are small or rudimentary. The upper margin of the orbit is sharp, the forehead vertical, the frontal and parietal eminences prominent, and the vault somewhat flattened. The contour of the face is more rounded, the facial bones are smoother, and the maxillae and mandible and their contained teeth smaller. From what has been said it will be seen that more of the infantile characteristics are retained in the skull of the adult female than in that of the adult male. A well-marked male or female skull can easily be recognized as such, but in some cases the respective characteristics are so indistinct that the determination of the sex may be difficult or impossible. Craniology. Skulls vary in size and shape, and the term craniology is applied to the study of these variations. The capacity of the cranial cavity constitutes a good index of the size of the brain which it contained, and is most conveniently arrived at by filling the cavity with shot and measuring the contents in a graduated vessel. Skulls may be classified according to their capacities as follows. 1. Microcephalic, with a capacity of less than 1,350 cubic centimetres, e.g. those of native Australians and Andaman Islanders. 2. Mesocephalic, with a capacity of from 1,350 cubic centimetres to 1,450 cubic centimetres, e.g. those of African Negroes and Chinese. 3. Megacephalic, with a capacity of over 1,450 cubic centimetres, e.g. those of Europeans, Japanese, and Eskimos. In comparing the shape of one skull with that of another it is necessary to adopt some definite position in which the skulls should be placed during the process of examination. They should be so placed that a line carried through the lower margin of the orbit and upper margin of the external acoustic meatus is in the horizontal plane. The normi of one skull can then be compared with those of another, and the differences in contour and surface form noted. Further, it is necessary that the various linear measurements used to determine the shape of the skull should be made between definite and easily localized points on its surface. The principal points may be divided into two groups: 1. Those in the median plane, and 2. Those on either side of it. The points in the median plane are the mental point, the most prominent point of the chin; alveolar point or prostheon, the central point of the anterior margin of the upper alveolar arch; subnasal point, the middle of the lower border of the anterior nasal aperture at the base of the anterior nasal spine; nasion, the central point of the frontonasal suture; glabella, the point in the middle line at the level of the superciliary arches; ophryon, the point in the middle line of the forehead at the level where the temporal lines most nearly approach each other; bregma, the meeting point of the coronal and sagittal sutures. Orbelion, a point in the sagittal suture on a level with the parietal foramina. Lambda, the point of junction of the sagittal and lambdoidal sutures. Occipital point, the point in the middle line of the occipital bone farthest from the glabella. Inion, the external occipital protuberance. Opistheon, the midpoint of the posterior margin of the foramen magnum. Bastion, the midpoint of the anterior margin of the foramen magnum. The points on either side of the median plane are the gonion, the outer margin of the angle of the mandible. Dacrion, the point of union of the antero-superior angle of the lacrimal with the frontal bone and the frontal process of the maxilla. Stephanion, the point where the temporal line intersects the coronal suture. Pterion, the point where the great wing of the sphenoid joins the sphenoidal angle of the parietal. Auricular point, the centre of the orifice of the external acoustic meatus. Asterion, the point of meeting of the lambdoidal, masto-occipital and masto-parietal sutures. The horizontal circumference of the cranium is measured in a plane passing through the glabella, terna, of the ophryon, flower, in front, and the occipital point behind. It averages about 50 cm in the female and 52.5 cm in the male. The occipitofrontal or longitudinal arc is measured from the nasion over the middle line of the vertex to the opistheon, while the basinasal length is the distance between the bastion and the nasion. These two measurements, plus the antero-posterior diameter of the foramen magnum, represent the vertical circumference of the cranium. The length is measured from the glabella to the occipital point, while the breadth or greatest transverse diameter is usually found near the external acoustic meatus. The proportion of breadth to length, breadth times 100, divided by length, is termed the cephalic index, or index of breadth. The height is usually measured from the bastion to the bregma, and the proportion of height to length, height times 100, divided by length, constitutes the vertical or height index. In studying the face, the principal points to be noted are the proportion of its length and breadth, the shape of the orbits and of the anterior nasal aperture, and the degree of projection of the jaws. The length of the face may be measured from the ophryon or nasion to the chin, or, if the mandible be wanting, to the alveolar point, while its width is represented by the distance between the zygomatic arches. By comparing the length with the width of the face, skulls may be divided into two groups: dolichofacial or leptoprosope, long-faced, and brachyfacial or chemoprosope, short-faced. The orbital index signifies the proportion which the orbital height bears to the orbital width, thus: orbital height times 100 divided by orbital width. The nasal index expresses the proportion which the width of the anterior nasal aperture bears to the height of the nose, the latter being measured from the nasion to the lower margin of the nasal aperture, thus: nasal width times 100 divided by nasal height. The degree of projection of the jaws is determined by the gnathic or alveolar index, which represents the proportion between the basialveolar and basinasal lengths, thus: basialveolar length times 100 divided by basinasal length. The following table, modified from that given by Duckworth, illustrates how these different indices may be utilized in the classification of skulls. Index 1. Cephalic, classification, below 75, nomenclature, dolichocephalic, examples, Kaffirs and native Australians. Index 1. Cephalic, classification, between 75 and 80, nomenclature, mesoticephalic, examples, Europeans and Chinese. Index 1. Cephalic, classification, above 80, nomenclature, brachycephalic, examples, Mongolians and Andamans. Index 2. Orbital, classification, below 84, nomenclature, microceme, examples, Tasmanians and native Australians. Index 2. Orbital, classification, between 84 and 89, nomenclature, mesoceme, examples, Europeans. Index 2. Orbital, classification, above 89, nomenclature, megaceme, examples, Chinese and Polynesians. Index 3. Nasal, classification, below 48, nomenclature, leptorhine, examples, Europeans. Index 3. Nasal. Classification between 48 and 53. Nomenclature. Mesorhine. Examples: Japanese and Chinese. Index 3. Nasal. Classification above 53. Nomenclature. Platyrrhine. Examples: Negroes and native Australians. Index 4. Gnathic. Classification below 98. Nomenclature. Orthognathus. Examples: Europeans. Index 4. Gnathic. Classification between 98 and 103. Nomenclature. Mesognathus. Examples: Chinese and Japanese. Index 4. Gnathic. Classification above 103. Nomenclature. Prognathus. Examples: native Australians. The chief function of the skull is to protect the brain, and therefore those portions of the skull which are most exposed to external violence are thicker than those which are shielded from injury by overlying muscles. Thus the skull cap is thick and dense, whereas the temporal squamae, being protected by the temporalis muscles and the inferior occipital fossae being shielded by the muscles at the back of the neck, are thin and fragile. Fracture of the skull is further prevented by its elasticity, its rounded shape, and its construction of a number of secondary elastic arches, each made up of a single bone. The manner in which vibrations are transmitted through the bones of the skull is also of importance as regards its protective mechanism, at all events as far as the base is concerned. In the vault, the bones being of a fairly equal thickness and density, vibrations are transmitted in a uniform manner in all directions, but in the base, owing to the varying thickness and density of the bones, this is not so, and therefore in this situation there are special buttresses which serve to carry the vibrations in certain definite directions. At the front of the skull, on either side, is the ridge which separates the anterior from the middle fossa of the base, and behind the ridge or buttress which separates the middle from the posterior fossa, and if any violence is applied to the vault, the vibrations would be carried along these buttresses to the sella turcica, where they meet. This part has been termed the centre of resistance, and here there is a special protective mechanism to guard the brain. The subarachnoid cavity at the base of the brain is dilated, and the cerebrospinal fluid which fills it acts as a water cushion to shield the brain from injury. In like manner, when violence is applied to the base of the skull, as in falls upon the feet, the vibrations are carried backwards through the occipital crest and forward through the basilar part of the occipital and body of the sphenoid to the vault of the skull. In connection with the bones of the face, a common malfunction is cleft palate. The cleft usually starts posteriorly, and its most elementary form is a bifid uvula, or the cleft may extend through the soft palate, or the posterior part of the whole of the hard palate may be involved, the cleft extending as far forward as the incisive foramen. In the severest form, the cleft extends through the alveolus and passes between the incisive or premaxillary bone and the rest of the maxilla, that is to say, between the lateral incisor and canine teeth. In some instances, the cleft runs between the central and lateral incisor teeth, and this has induced some anatomists to believe the premaxillary bone is developed from two centres and not from one, as was stated on page 163. The medial segment, bearing a central incisor, is called an endognathion. The lateral segment, bearing the lateral incisor, is called a mesognathion. The cleft may affect one or both sides. If the latter, the central part is frequently displaced forward and remains united to the septum of the nose, the deficiency in the alveolus being complicated with the cleft in the lip, hare lip. On examining a cleft palate in which the alveolus is not implicated, the cleft will generally appear to be in the median line, but occasionally is unilateral and in some cases bilateral. To understand this, it must be borne in mind that three processes are concerned in the formation of the palate: the palatine processes of the two maxillae, which grow in horizontally and unite in the middle line, and the ethmovomerine process, which grows downward from the base of the skull, and frontonasal process to unite with the palatine processes in the middle line. In those cases where the palatine processes fail to unite with each other and with the medial process, the cleft of the palate is median. Where one palatine process unites with the medial septum, the other failing to do so, the cleft in the palate is unilateral. In some cases where the palatine processes fail to meet in the middle, the ethmovomerine process goes downward between them and thus produces a bilateral cleft. Occasionally there may be a hole in the middle line of the hard palate, the anterior part of the hard and soft palate being perfect. This is rare, because as a rule the union of the various processes progresses from before backward, and therefore the posterior part of the palate is more frequently defective than the anterior.
The extremities. The bones by which the upper and lower limbs are attached to the trunk constitute respectively the shoulder and pelvic girdles. The shoulder girdle, or girdle of the superior extremity, is formed by the scapula and clavicles, and is imperfect in front and behind. In front, however, it is completed by the upper end of the sternum, with which the medial ends of the clavicle articulate. Behind, it is widely imperfect, the scapula being connected to the trunk by muscles only. The pelvic girdle, or girdle of the inferior extremity, is formed by the hip bones, which articulate with each other in front at the symphysis pubis. It is imperfect behind, but the gap is filled in by the upper part of the sacrum. The pelvic girdle with the sacrum is a complete ring, massive and comparatively rigid, in marked contrast to the lightness and mobility of the shoulder girdle. The clavicle. The clavicle forms the anterior portion of the shoulder girdle. It is a long bone, curved somewhat like the italic letter F, and placed nearly horizontally at the upper and anterior part of the thorax, immediately above the first rib. It articulates medially with the manubrium sterni, and laterally with the acromion of the scapula. Footnote: the clavicle acts especially as a fulcrum to enable the muscles to give lateral motion to the arm. It is accordingly absent in those animals whose forelimbs are used only for progression, but is present for the most part in animals whose anterior extremities are clawed and used for prehension, though in some of them, as, for instance, in a large number of carnivora, it is merely a rudimentary bone suspended among the muscles and not articulating with either the scapula or sternum. End of footnote. It presents a double curvature, the convexity being directed toward the sternal end, and the concavity at the scapular end. Its lateral third is flattened from above downward, while its medial two-thirds is of a rounded or prismatic form. Lateral third. The lateral third has two surfaces, an upper and a lower, and two borders, an anterior and a posterior. Surface. The upper surface is flat, rough, and marked by impressions for the attachments of the deltoideus in front and the trapezius behind. Between these impressions, a small portion of the bone is subcutaneous. The undersurface is flat. At its posterior border, near the point where the prismatic joins with the flattened portion, is a rough eminence, the coracoid tuberosity. This, in the natural position of the bone, surmounts the coracoid process of the scapula and gives attachment to the conoid ligament. From this tuberosity, an oblique ridge, the oblique or trapezoid ridge, runs forward and lateralward and afford attachment to the trapezoid ligament. Borders. The anterior border is concave, thin, and rough, and gives attachments to the deltoideus. The posterior border is convex, rough, thicker than the anterior, and gives attachment to the trapezius. Medial two-thirds. The medial two-thirds constitute the prismatic portion of the bone, which is curved so as to be convex in front, concave behind, and is marked by three borders separating three surfaces. Borders. The anterior border is contiguous with the anterior margin of the flat portion. Its lateral part is smooth, and corresponds to the interval between the attachments of the pectoralis major and deltoideus. Its medial part forms the lower boundary of an elliptical surface for the attachment of the clavicular portion of the pectoralis major, and approaches the posterior border of the bone. The superior border is continuous with the posterior margin of the flat portion, and separates the anterior from the posterior surface. Smooth and rounded laterally, It becomes rough towards the medial third for the attachment of the sternocleidomastoideus, and ends at the upper angle of the sternal extremity. The posterior or subclavian border separates the posterior from the inferior surface, and extends from the coracoid tuberosity to the costal tuberosity. It forms the posterior boundary of the groove for the subclavius, and gives attachment to the layer of cervical fascia which envelops the omohyoideus. Surfaces. The anterior surface is included between the superior and anterior borders. Its lateral part looks upward and is continuous with the superior surface of the flattened portion. It is smooth, convex, and nearly subcutaneous, being covered only by the platysma. Medially, it is divided by a narrow subcutaneous area into two parts, a lower elliptical in form, and directed forward for the attachment of the pectoralis major, and an upper for the attachment of the sternocleidomastoideus. The posterior or cervical surface is smooth, and looks backward toward the root of the neck. It is limited above by the superior border, below by the subclavian border, medially by the margin of the sternal extremity, and laterally by the coracoid tuberosity. It is concave mediolaterally, and is in relation, by its lower part, with the transverse scapular vessels. This surface, at the junction of the curves of the bone, is also in relation to the brachial plexus of nerves and the subclavian vessels. It gives attachment near the sternal extremity to part of the sternohyoideus, and presents, near the middle, an oblique foramen directed lateralward, which transmits the chief nutrient artery of the bone. Sometimes there are two foramini on the posterior surface, or one on the posterior and another on the inferior surface. The inferior or subclavian surface is bounded in front by the anterior border, behind by the subclavian border. It is narrowed medially, but gradually increases in width laterally, and is continuous with the upper surface of the flat portion. On its medial part is a broad, rough surface, the costal tuberosity, rather more than 2 centimeters in length, for the attachment of the costoclavicular ligament. The rest of this surface is occupied by a groove, which gives attachment to the subclavius. The coracoclavicular fascia, which splits to enclose the muscle, is attached to the margins of the groove. Not infrequently this groove is subdivided longitudinally by a line which gives attachment to the intramuscular septum of the subclavius. The sternal extremity. Extremitas sternalis. Internal extremity. The sternal extremity of the clavicle is triangular in form, directed medialward and a little downward and forward. It presents an articular facet, concave from before backward, convex from above downward, and articulates with the manubrium sterni through the intervention of an articular disc. The lower part of the facet is continued on to the inferior surface of the bone as a small semi-oval area for articulation with the cartilage of the first rib. The circumference of the articular surface is rough for the attachment of numerous ligaments. The upper angle gives attachment to the articular disc. The acromial extremity. Extremitas acromialis. Outer extremity. The acromial extremity presents a small, flattened, oval surface directed obliquely downward for articulation with the acromion of the scapula. The circumference of the articular facet is rough, especially above, for the attachment of the acromioclavicular ligaments. In the female, the clavicle is generally shorter, thinner, less curved, and smoother than in the male. In those persons who perform considerable manual labor, it becomes thicker and more curved, and its ridges for muscular attachment are prominently marked. Structure. The clavicle consists of cancellous tissue, enveloped by a compact layer, which is much thicker in the intermediate part than at the extremities of the bone. Ossification. The clavicle begins to ossify before any other bone in the body. It is ossified from 3 centers. 2 primary centers, a medial and lateral, for the body, which appears during the 5th or 6th week of fetal life, and a secondary center for the sternal end, which appears about the 18th or 20th year and unites with the rest of the bone about the 25th year. The scapula. The scapula forms the posterior part of the shoulder girdle. It is a flat triangular bone with 2 surfaces, 3 borders, and 3 angles. Surfaces. The costal or ventral surface presents a broad concavity, the suprascapular fossa. The medial 2/3 of the fossa are marked by several oblique ridges, which run lateralward and upward. The ridges give attachment to the tendinous insertions, and the surfaces between them to the fleshy fibers of the subscapularis. The lateral third of the fossa is smooth and covered by the fibers of this muscle. The fossa is separated from the vertebral border by smooth triangular areas at the medial and inferior angles, and in the interval between these by a narrow ridge which is often deficient. These triangular areas and the intervening ridge afford attachment to the serratus anterior. At the upper part of the fossa is a transverse depression, where the bone appears to be bent on itself along a line at right angles to and passing through the center of the glenoid cavity, forming the considerable angle called the subscapular angle. This gives greater strength to the body of the bone by its arched form, while the summit of the arch serves to support the spine and acromion. The dorsal surface is arched from above downward, and is subdivided into 2 unequal parts by the spine. The portion above the spine is called the supraspinatus fossa, and that below it is the infraspinatus fossa. The supraspinatus fossa, the smaller of the 2, is concave, smooth, and broader at its vertebral than at its humeral end. Its medial 2/3 is origin to the supraspinatus. The infraspinatus fossa is much larger than the preceding. Toward its vertebral margin, a shallow concavity is seen at its upper part. Its center presents a prominent convexity, while near the axillary border is a deep groove which runs from the upper towards the lower part. The medial 2/3 of the fossa give origin to the infraspinatus. The lateral third is covered by this muscle. The dorsal surface is marked near the axillary border by an elevated ridge, which runs from the lower part of the glenoid cavity downward and backward to the vertebral border about 2.5 centimeters above the inferior angle. The ridge serves for the attachment of a fibrous septum, which separates the infraspinatus from the teres major and teres minor. The surface between the ridge and the axillary border is narrow in the upper 2/3 of its extent, and is crossed near the center by a groove for the passage of the scapular circumflex vessels. It affords attachment to the teres minor. Its lower third presents a broader, somewhat triangular surface, which gives origin to the teres major, and over which the latissimus dorsi glides. Frequently the latter muscle takes origin by a few fibers from this part. The broad and narrow portions above alluded to are separated by an oblique line, which runs from the axillary border, downward and backward, to meet the elevated ridge. To it is attached a fibrous septum which separates the teres muscle from each other. The spine, spina scapulae. The spine is a prominent plate of bone, which crosses obliquely the medial 4/5 of the dorsal surface of the scapula at its upper part, and separates the supra from the infraspinatus fossa. It begins at the vertical border by a smooth, triangular area over which the tendon of insertion of the lower part of the trapezius glides, and, gradually becoming more elevated, ends in the acromion, which overhangs the shoulder joint. The spine is triangular and flattened from above downward, its apex being directed towards the vertebral border. It presents two surfaces and three borders. Its superior surface is concave. It assists in forming the supraspinatus fossa and gives origin to part of the supraspinatus. Its inferior surface forms part of the infraspinatus fossa, gives origin to a portion of the infraspinatus, and presents near its center the orifice of a nutrient canal. Of the three borders, the anterior is attached to the dorsal surface of the bone. The posterior, or crest of the spine, is broad and presents two lips and an intervening rough interval. The trapezius is attached to the superior lip, and a rough tubercle is generally seen on the portion of the spine which receives the tendon of the insertion of the lower part of this muscle. The deltoideus is attached to the whole length of the inferior lip. The interval between the lips is subcutaneous and partly covered by the tendinous fibers of these muscles. The lateral border, or base, the shortest of the three, is slightly concave. Its edge is thick and round, is continuous above within the under surface of the acromion, below the neck of the scapula. It forms the medial boundary of the great scapular notch, which serves to connect the supra and infraspinatus fossa. The acromion. The acromion forms the summit of the shoulder and is a large, somewhat triangular or oblong process, flattened from behind forward, projecting at first lateralward, and then curving upward and forward, so as to overhang the glenoid cavity. Its superior surface, directed upward, backward, and lateralward, is convex, rough, and gives attachment to some fibers of the deltoideus, and in the rest of its extent is subcutaneous. Its inferior surface is smooth and concave. Its lateral border is thick and irregular, and presents 3 or 4 tubercles for the tendinous origins of the deltoideus. Its medial border, shorter than the lateral, is concave, gives attachment to a portion of the trapezius, and presents about its center a small oval surface for articulation with the acromial end of the clavicle. Borders. Of the 3 borders of the scapula, the superior is the shortest and thinnest. It is concave and extends from the medial angle to the base of the coracoid process. At its lateral part is a deep semicircular notch, the scapular notch, formed partly by the base of the coracoid process. This notch is converted into a foramen by the superior transverse ligament, and serves for the passage of the suprascapular nerve. Sometimes the ligament is ossified. The adjacent part of the superior border affords attachment to the omohyoideus. The axillary border is the thickest of the 3. It begins above at the lower margin of the glenoid cavity, and inclines obliquely downward and backward to the inferior angle. Immediately below the glenoid cavity is the rough impression, the infraglenoid tuberosity, about 2.5 centimeters in length, which gives origin to the long head of the triceps brachii. In front of this is a longitudinal groove, which extends as far as the lower third of this border, and affords origin to part of the subscapularis. The inferior third is thin and sharp, and serves for the attachment of a few fibers of the teres major behind, and of the subscapularis in front. The vertebral border is the longest of the 3, and extends from the medial to the inferior angle. It is arched, intermediate in thickness, between the superior and axillary borders, and the portion of it above the spine forms an obtuse angle with the part below. This border presents an anterior and a posterior lip, and an intermediate narrow area. The anterior lip affords attachment to the serratus anterior, the posterior lip to the supraspinatus above the spine, the infraspinatus below. The area between the 2 lips, to the levator scapulae above the triangular surface at the commencement of the spine, to the rhomboideus minor on the edge of that surface, and to the rhomboideus major below it. This last is attached by means of the fibrous arch, connected above to the lower part of the triangular surface at the base of the spine, and below to the lower part of the border. Angles. Of the three angles, the medial, formed by the junction of the superior and vertebral borders, is thin, smooth, rounded, inclined somewhat lateralward, and gives attachment to a few fibers of the levator scapulae. The inferior angle, thick and rough, is formed by the union of the vertebral and axillary borders. Its dorsal surface affords attachment to the teres major, and frequently to a few fibers of the latissimus dorsi. The lateral angle is the thickest part of the bone, and is sometimes called the head of the scapula. On it is a shallow piriform articular surface, the glenoid cavity, which is directed lateralward and forward, and articulates with the head of the humerus. It is broader below than above, and its vertical diameter is the longest. The surface is covered with cartilage in a fresh state, and its margins, raised slightly, give attachment to a fibrocartilaginous structure, the glenoid labrum, which deepens the cavity. At its apex is a slight elevation, the supraglenoid tuberosity, to which the long head of the biceps brachii is attached. The neck of the scapula is the slightly constricted portion, which surrounds the head and is more distinct below and behind than above and in front. The coracoid process, processus coracoideus. The coracoid process is a thick curved process attached by a broad base to that upper part of the neck of the scapula. It runs at first upward and medialward, then, becoming smaller, it changes its direction and projects forward and lateralward. The ascending portion, flattened from before backwards, presents in front a smooth concave surface, across which the suprascapularis passes. The horizontal portion is flattened from above downward. Its upper surface is convex and irregular, and gives attachment to the pectoralis minor. Under its surface is smooth. Its medial and lateral borders are rough. The former gives attachment to the pectoralis minor, and the latter to the coracochromial ligament. The apex is embraced by the conjoined tendon of origin of the coracobrachialis and the short head of the biceps brachii, and gives attachment to the coracoclavicular fascia. On the medial part of the root of the coracoid process is a rough impression for the attachment of the conoid ligament, and running from it obliquely forward and lateralward, onto the upper surface of the horizontal portion, is an elevated ridge for the attachment of the trapezoid ligament. Structure. The head, processes, and the thickened part of the bone contains cancellous tissue. The rest consist of a thin layer of compact tissue. The central part of the supraspinatus fossa and the upper part of the infraspinatus fossa, but especially the former, are usually so thin as to be semi-transparent. Occasionally the bone is found wanting in this situation, and the adjacent muscles are separated only by fibrous tissue. Ossification. The scapula is ossified from 7 or more centers, 1 for the body, 2 for the coracoid process, 2 for the acromion, 1 for the vertebral border, and 1 for the inferior angle. Ossification of the body begins about the 2nd month of fetal life by the formation of an irregular quadrilateral plate of bone immediately behind the glenoid cavity. This plate extends so as to form the chief part of the bone, and spine growing up from its dorsal surface about the 3rd month. At birth, a large part of the scapula is osseous, but the glenoid cavity, the coracoid process, the acromion, the vertebral border, and the inferior angle are cartilaginous. From the 15th through the 18th month after birth, ossification takes place in the middle of the coracoid process, which as a rule becomes joined with the rest of the bone about the 15th year. Between the 14th and 20th years, ossification of the remaining parts take place in quick succession, and usually in the following order. First, in the root of the coracoid process, in the form of a broad scale. Secondly, near the base of the acromion. Thirdly, in the inferior angle and contiguous part of the vertebral border. Fourthly, near the extremity of the acromion. Fifthly, in the vertebral border. The base of the acromion is formed by an extension from the spine. The two separate nuclei of the acromion unite, and then join with the extension from the spine. The upper third of the glenoid cavity is ossified from a separate center, subcoracoid, which makes its appearance between the 10th and 11th years, and joins between the 16th and the 18th. Further, an epiphyseal plate appears for the lower part of the glenoid cavity, while the tip of the coracoid process frequently presents a separate nucleus. These various epiphyses are joined to the bone by the 25th year. Failure of the bony union between the acromion and spine sometimes occurs, the junction being affected by fibrous tissue or by an imperfect articulation. In some cases of supposed fracture of the acromion with ligamentous union, it is probable that the detached segment was never united to the rest of the bone. Section 37. The humerus. The humerus is the longest and largest bone of the upper extremity. It is divisible into a body and two extremities. Upper extremity. The upper extremity consists of a large rounded head joined to the body by a constricted portion called the neck, and two eminences, the greater and lesser tubercles. The head, or caput humeri. The head, nearly hemispherical in form. Footnote: though the head is nearly hemispherical in form, its margins, as Humphrey has shown, is by no means a true circle. Its greatest diameter is from the top of the intertubercular groove in a direction downward, medialward, and backward. Hence it follows that the greatest elevation of the arm can be obtained by rolling the articular surface in this direction, that is to say, obliquely upward, lateralward, and forward. End of footnote. Is directed upward, medialward, and a little backward, and articulates with the glenoid cavity of the scapula. The circumference of its articular surface is slightly constricted, and is termed the anatomical neck, in contradistinction to a constriction below the tubercles called the surgical neck, which is frequently the seat of fracture. Fracture of the anatomical neck rarely occurs. The anatomical neck, or column anatomicum, is obliquely directed, forming an obtuse angle with the body. It is best marked in the lower half of its circumference. In the upper half it is represented by a narrow groove separating the head from the tubercles. It affords attachment to the articular capsule of the shoulder joint, and is perforated by numerous vascular foraminae. The greater tubercle, or tuberculum majus, or greater tuberosity. The greater tubercle is situated lateral to the head and lesser tubercle. Its upper surface is rounded and marked by three flat impressions. The highest of these gives insertion to the supraspinatus, the middle to the infraspinatus, the lowest one, and the body of the bone for about 2.5 centimeters below it, to the teres minor. The lateral surface of the greater tubercle is convex, rough, and continuous with the lateral surface of the body. The lesser tubercle, or tuberculum minus, or lesser tuberosity. The lesser tubercle, although smaller, is more prominent than the greater. It is situated in front, and is directed medialward and forward. Above and in front it presents an impression for the insertion of the tendon of the subscapularis. The tubercles are separated from each other by a deep groove, the intertubercular groove or bicipital groove, which lodges the long tendon of the biceps brachii and transmits a branch of the anterior humeral circumflex artery to the shoulder joint. It runs obliquely downward and ends near the junction of the upper with the middle third of the bone. In the fresh state, its upper part is covered with a thin layer of cartilage, lined by a prolongation of the synovial membrane of the shoulder joint. Its lower portion gives insertion to the tendon of the latissimus dorsi. It is deep and narrow above, and becomes shallow and a little broader as it descends. Its lips are called, respectively, the crest of the greater and lesser tubercles, or bicipital ridges, and forms the upper part of the anterior and medial borders of the body of the bone. The body or shaft, or corpus humeri. The body is almost cylindrical in the upper half of its extent, prismatic and flattened below, and has 3 borders and 3 surfaces. Borders. The anterior border runs from the front of the greater tubercle above to the coronoid fossa below, separating the anterior medial from the anterolateral surface. Its upper part is a prominent ridge, the crest of the greater tubercle. It serves for the insertion of the tendon of the pectoralis major. About its center it forms the anterior boundary of the deltoid tuberosity. Below, it is smooth and rounded, affording the attachment to the brachialis. The lateral border runs from the back part of the greater tubercle to the lateral epicondyle, and separates the anterolateral from the posterior surface. Its upper half is rounded and indistinctly marked, serving for the attachment of the lower part of the insertion of the teres minor, and below this giving origin to the lateral head of the triceps brachii. Its center is traversed by a broad but shallow oblique depression, the radial sulcus or musculospiral groove. Its lower part forms a prominent, rough margin, a little curved from behind forward, the lateral supracondylar ridge, which presents an anterior lip for the origin of the brachioradialis above, the extensor carpi radialis longus below, a posterior lip for the triceps brachii, and an intermediate ridge for the attachment of the lateral intermuscular septum. The medial border extends from the lesser tubercle to the medial epicondyle. Its upper third consists of a prominent ridge, the crest of the lesser tubercle, which gives insertion to the tendon of the teres major. About its center is a slight impression for the insertion of the coracobrachialis, and just below this is the entrance of the nutrient canal, directed downward. Sometimes there is a second nutrient canal at the commencement of the radial sulcus. The inferior third of this border is raised into a slight ridge, the medial supracondylar ridge, which becomes very prominent below. It presents an anterior lip for the origins of the brachialis and pronator teres, a posterior lip for the medial head of the triceps brachii, and an intermediate ridge for the attachment of the medial intramuscular septum. Surfaces. The anterolateral surface is directed lateralward above, where it is smooth, rounded, and covered by the deltoideus. Forward and lateralward below, where it is slightly concave from above downward, and gives origin to part of the brachialis. About the middle of this surface is a rough triangular elevation, the deltoid tuberosity, for the insertion of the deltoideus. Below this is the radial sulcus, directed obliquely from behind, forward, and downward, and transmitting the radial nerve and profunda artery. The anteromedial surface, less extensive than the anterolateral, is directed medialward above, forward and medialward below. Its upper part is narrow and forms the floor of the intertubercular groove, which gives insertion to the tendon of the latissimus dorsi. Its middle part is slightly rough for the attachment of some of the fibers of the tendon of insertion of the coracobrachialis. Its lower part is smooth, concave, from above downward, and gives origin to the brachialis. Footnote: a small hook-shaped process of bone, the supracondylar process, varying from 2 to 20 millimeters in length, is not infrequently found projecting from the anteromedial surface of the body of the humerus 5 centimeters above the medial epicondyle. It is curved downward and forward, and its pointed end is connected to the medial border just above the medial epicondyle by a fibrous band which gives origin to a portion of the pronator teres. Through the arch completed by this fibrous band, the median nerve and brachial artery pass, when these structures deviate from their usual course. Sometimes the nerve alone is transmitted through it, or the nerve may be accompanied by the ulnar artery, in cases of high division of the brachial. A well-marked groove is usually found behind the process, in which the nerve and artery are lodged. This arch is the homologue of the supracondylar foramen found in many animals, and probably serves in them to protect the nerve and artery from compression during the contraction of the muscles in this region. End of footnote. The posterior surface appears somewhat twisted, so that its upper part is directed a little medialward, its lower part backward, and a little lateralward. Nearly the whole of this surface is covered by the lateral and medial heads of the triceps brachii, the former arising above and the latter below the radial sulcus. The lower extremity. The lower extremity is flattened from before backward and curves slightly forward. It ends below in a broad articular surface which is divided into 2 parts by a slight ridge, projecting on either side of the lateral and medial epicondyles. The articular surface extends a little lower than the epicondyles and is curved slightly forward. Its medial extremity occupies a lower level than the lateral. The lateral portion of this surface consists of a smooth, rounded eminence named the capitellum of the humerus. It articulates with the cup-shaped depression on the head of the radius, and is limited to the front and lower part of the bone. On the medial side of this eminence is a shallow groove, in which is received the medial margin of the head of the radius. Above the part of the capitellum is a slight depression, the radial fossa, which receives the anterior border of the head of the radius when the forearm is flexed. The medial portion of the articular surface is named the trochlea, and presents a deep depression between the 2 well-marked borders. It is convex from before backward, concave from side to side, and occupies an anterior, lower, and posterior parts of the extremity. The lateral border separates it from the groove, which articulates with the margin of the head of the radius. The medial border is thicker, of greater length, and consequently more prominent than the lateral. The grooved portion of the articular surface fits accurately within the semilunar notch of the ulna. It is broader and deeper on the posterior than on the anterior aspect of the bone, and is inclined obliquely downward and forward towards the medial side. Above the front part of the trochlea is a small depression, the coronoid fossa, which receives the coronoid process of the ulna during flexion of the forearm. Above the back part of the trochlea is a deep triangular depression, the olecranon fossa, in which the summit of the olecranon is received in extension of the forearm. These fossa are separated from one another by a thin, transparent lamina of bone, which is sometimes perforated by the supratrochlear foramen. They are lined in the fresh state by the synovial membrane of the elbow joint, and their margins afford attachment to the anterior and posterior ligaments of this articulation. The lateral epicondyle is a small, tuberculated eminence, curved a little forward, and giving attachment to the radial-collateral ligament of the elbow joint, and to a tendon common to the origin of the supinator and some of the extensor muscles. The medial epicondyle, larger and more prominent than the lateral, is directed a little backward. It gives attachment to the ulnar collateral ligament of the elbow joint, to the pronator teres, and to a common tendon of origin of some of the flexor muscles of the forearm, The ulnar nerve runs in a groove on the back of this epicondyle. The epicondyles are continuous above with the supracondylar ridges. Structure. The extremities consist of cancellous tissue covered with a thin, compact layer. The body is composed of a cylinder of compact tissue, thicker at the center than towards the extremities, and contains a large medullary canal which extends along its whole length. Ossification. The humerus is ossified from 8 centers, one for each of the following parts: the body, the head, the greater tubercle, the lesser tubercle, the capitellum, the trochlea, and one for each epicondyle. The center for the body appears near the middle of the bone in the 8th week of fetal life, and soon extends towards the extremities. At birth, the humerus is ossified in nearly its whole length, only the extremities remaining cartilaginous. During the first year, sometimes before birth, ossification commences in the head of the bone, and during the 3rd year, the center for the greater tubercle, and during the 5th, that for the lesser tubercle, making their appearance. By the 6th year, the centers for the head and tubercles have joined, so as to form a single large epiphysis, which fuses with the body about the 20th year. The lower end of the humerus is ossified as follows. At the end of the 2nd year, ossification begins in the capitellum, and extends medialward to form the chief part of the articular end of the bone. The center for the medial part of the trochlea appears about the age of 12. Ossification begins in the medial epicondyle about the 5th year, and in the lateral about the 13th or 14th year. About the 16th or 17th year, the lateral epicondyle and both portions of the articulating surface, having already joined, unite with the body, and at the 18th year the medial epicondyle becomes joined to it.
Section 38. The ulna, elbow bone. Footnote. In the anatomical position, the forearm is placed in extension and supination, with the palm looking forward and the thumb on the outer side. End footnote. The ulna is a long bone, prismatic in form, placed at the medial side of the forearm parallel with the radius. It is divisible into a body and 2 extremities. Its upper extremity, of great thickness and strength, forms a large part of the elbow joint. The bone diminishes in size from above downward, its lower extremity being very small and excluded from the wrist joint by the interposition of an articular disc. The upper extremity, proximal extremity. The upper extremity presents 2 curved processes, the olecranon and the coronoid process, and 2 concave articular cavities, the semilunar and radial notches. The olecranon process. The olecranon is a large, thick, curved eminence situated at the upper and back part of the ulna. It is bent forward at the summit so as to present a prominent lip which is received into the olecranon fossa of the humerus in extension of the forearm. Its base is contracted where it joins the body and the narrowest part of the upper end of the ulna. Its posterior surface, directed backward, is triangular, smooth, subcutaneous, and covered by a bursa. Its superior surface is of quadrilateral form, marked behind by a rough impression for the insertion of the triceps brachii, and in front, near the margin, by a slight transverse groove for the attachment of part of the posterior ligament of the elbow joint. Its anterior surface is smooth, concave, and forms the upper part of the semilunar notch. Its borders present continuations of the groove on the margin of the superior surface. They serve for the attachment of ligaments, that is, the back part of the ulnar collateral ligament medially, and the posterior ligament laterally. From the medial border a part of the flexor carpi ulnaris arises, while to the lateral border the anconeus is attached. The coronoid process, processus coronoideus. The coronoid process is a triangular eminence projecting forward from the upper and front part of the ulna. Its base is continuous with the body of the bone and of considerable strength. Its apex is pointed, slightly curved upward, and in flexion of the forearm is received into the coronoid fossa of the humerus. Its upper surface is smooth, concave, and forms the lower part of the semilunar notch. Its antero-inferior surface is concave, and marked by a rough impression for the insertion of the brachialis. At the junction of this surface with the front of the body is a rough eminence, the tuberosity of the ulna, which gives insertion to a part of the brachialis. To the lateral border of this tuberosity the oblique cord is attached. Its lateral surface presents a narrow, oblong articular depression, the radial notch. Its medial surface, by its prominent free margin, serves for the attachment of part of the ulnar collateral ligament. At the front part of this surface is a small rounded eminence for the origin of one head of the flexor digitorum sublimis. Behind the eminence is a depression for part of the origin of the flexor digitorum profundus. Descending from the eminence is a ridge which gives origin to one head of the pronator teres. Frequently the flexor pollicis longus arises from the lower part of the carnoid process by a rounded bundle of muscular fibers. The semilunar notch, incisura semilunaris, greater sigmoid cavity. The semilunar notch is a large depression formed by the olecranon and the carnoid process, and serving for articulation with the trochlea of the humerus. About the middle of either side of this notch is an indentation which contracts it somewhat, and indicates the junction of the olecranon and the carnoid process. The notch is concave from above downward, and divided into a medial and a lateral portion by a smooth ridge running from the summit of the olecranon to the tip of the carnoid process. The medial portion is the larger, and is slightly concave transversely. The lateral is convex above, slightly concave below. The radial notch, incisura radialis, lesser sigmoid cavity. The radial notch is a narrow, oblong, articular depression on the lateral side of the carnoid process. It receives the circumferential articular surface of the head of the radius. It is concave from before backward, and its prominent extremities serve for the attachment of the annular ligament. The body or shaft, corpus ulnae. The body at its upper part is prismatic in form, and curved so as to be convex behind and lateralward. Its central part is straight, its lower part is rounded, smooth, and bent a little lateralward. It tapers gradually from above downward, and has 3 borders and 3 surfaces. Borders. The volar border, margo volaris, anterior border, begins above at the prominent medial angle of the carnoid process, and ends below in front of the styloid process. Its upper part, well defined, and its middle portion, smooth and rounded, give origin to the flexor digitorum profundus. Its lower fourth serves for the origin of the pronator quadratus. This border separates the volar from the medial surface. The dorsal border, margo dorsalis, posterior border, begins above at the apex of the triangular subcutaneous surface at the back part of the olecranon, and ends below at the back of the styloid process. It is well marked in the upper 3/4, and gives attachment to an aponeurosis which affords a common origin to the flexor carpi ulnaris, the extensor carpi ulnaris, and the flexor digitorum profundus. Its lower fourth is smooth and rounded. This border separates the medial from the dorsal surface. The interosseous crest, crista interossea, external or interosseous border, begins above by the union of 2 lines, which converge from the extremities of the radial notch, and enclose between them a triangular space for the origin of part of the supinator. It ends below at the head of the ulna. Its upper part is sharp, its lower fourth smooth and rounded. This crest gives attachment to the interosseous membrane, and separates the volar from the dorsal surface. Surfaces. The volar surface, facies volaris, anterior surface, much broader above than below, is concave in its upper 3/4, and gives origin to the flexor digitorum profundus. Its lower fourth, also concave, is covered by the pronator quadratus. The lower fourth is separated from the remaining portion by a ridge directed obliquely downward and medialward, which marks the extent of origin of the pronator quadratus. At the junction of the upper with the middle third of the bone is the nutrient canal, directed obliquely upward. The dorsal surface, facies dorsalis, posterior surface, directed backward and lateralward, is broad and concave above, convex and somewhat narrower in the middle, narrow, smooth, and rounded below. On its upper part is an oblique ridge which runs from the dorsal end of the radial notch downward to the dorsal border. The triangular surface above this ridge receives the insertion of the anconeus, while the upper part of the ridge affords attachment to the supinator. Below this the surface is subdivided by a longitudinal ridge, sometimes called the perpendicular line, into two parts. The medial part is smooth and covered by the extensor carpi ulnaris. The lateral portion, wider and rougher, gives origin from above downward to the supinator, the abductor pollicis longus, the extensor pollicis longus, and the extensor indicis proprius. The medial surface, facies medialis, internal surface, is broad and concave above, narrow and convex below. Its upper 3/4 give origin to the flexor digitorum profundus. Its lower fourth is subcutaneous. The lower extremity, distal extremity. The lower extremity of the ulna is small and presents two eminences. The lateral and larger is a rounded articular eminence, termed the head of the ulna. The medial, narrower and more projecting, is a non-articular eminence, the styloid process. The head presents an articular surface, part of which, of an oval or semilunar form, is directed downward, and articulates with the upper surface of the triangular articular disc which separates it from the wrist joint. The remaining portion, directed lateralward, is narrow, convex, and received into the ulnar notch of the radius. The styloid process projects from the medial and back part of the bone. It descends a little lower than the head, and its rounded end affords attachment to the ulnar collateral ligament of the wrist joint. The head is separated from the styloid process by a depression for the attachment of the apex of the triangular articular disc, and behind by a shallow groove for the tendon of the extensor carpi ulnaris. Structure. The long, narrow medullary cavity is enclosed in a strong wall of compact tissue, which is thickest along the interosseous border and dorsal surface. At the extremities the compact layer thins. The compact layer is continued onto the back of the olecranon as a plate of close, spongy bone with lamellae parallel. From the inner surface of this plate and the compact layer below it, trabeculi arch forward toward the olecranon and coronoid, and cross other trabeculi, passing backward over the medullary cavity from the upper part of the shaft below the coronoid. Below the coronoid process there is a small area of compact bone from which trabeculi curve upward to end obliquely to the surface of the semilunar notch, which is coated with a thin layer of compact bone. The trabeculi at the lower end have a more longitudinal direction. Ossification. The ulna is ossified from three centers, one each for the body, the inferior extremity, and the top of the olecranon. Ossification begins near the middle of the body, about the 8th week of fetal life, and soon extends through the greater part of the bone. At birth the ends are cartilaginous. About the 4th year, a center appears in the middle of the head, and soon extends into the styloid process. About the 10th year, a center appears in the olecranon near its extremity, the chief part of this process being formed by an upward extension of the body. The upper epiphysis joins the body about the 16th, the lower about the 20th year. Articulations. The ulna articulates with the humerus and radius. The radius. The radius is situated on the lateral side of the ulna, which exceeds it in length and size. Its upper end is small and forms only a small part of the elbow joint, but its lower end is large and forms the chief part of the wrist joint. It is a long bone, prismatic in form and slightly curved longitudinally. It has a body and two extremities. The upper extremity, proximal extremity. The upper extremity presents a head, neck, and tuberosity. The head is of a cylindrical form, and on its upper surface is a shallow cup or fovea for articulation with the capitulum of the humerus. The circumference of the head is smooth. It is broad medially where it articulates with the radial notch of the ulna, narrow in the rest of its extent, which is embraced by the annular ligament. The head is supported on a round, smooth, and constricted portion called the neck, on the back of which is a slight ridge for the insertion of part of the supinator. Beneath the neck, on the medial side, is an eminence, the radial tuberosity. Its surface is divided into a posterior rough portion, for the insertion of the tendon of the biceps brachii, and an anterior, smooth portion on which a bursa is interposed between the tendon and the bone. The body or shaft, corpus radii. The body is prismoid in form, narrower above than below, and slightly curved so as to be convex lateralward. It presents 3 borders and 3 surfaces. Borders. The volar border, margo volaris, anterior border, extends from the lower part of the tuberosity above to the anterior part of the base of the styloid process below, and separates the volar from the lateral surface. Its upper third is prominent, and from its oblique direction has received the name of the oblique line of the radius. It gives origin to the flexor digitorum sublimis and flexor pollicis longus. The surface above the line gives insertion to part of the supinator. The middle third of the volar border is indistinct and rounded. The lower fourth is prominent, and gives insertion to the pronator quadratus and attachment to the dorsal carpal ligament. It ends in a small tubercle into which the tendon of the brachioradialis is inserted. The dorsal border, margo dorsalis, posterior border, begins above at the back of the neck, and ends below at the posterior part of the base of the styloid process. It separates the posterior from the lateral surface. It is indistinct above and below, but well marked in the middle third of the bone. The interosseous crest, crista interossea, internal or interosseous border, begins above at the back part of the tuberosity, and its upper part is rounded and indistinct. It becomes sharp and prominent as it descends, and at its lower part divides the 2 ridges which are continued to the anterior and posterior margins of the ulnar notch. To the posterior of the 2 ridges, the lower part of the interosseous membrane is attached, while the triangular surface between the ridges gives insertion to part of the pronator quadratus. This crest separates the volar from the dorsal surface, and gives attachment to the interosseous membrane. Surface. The volar surface, facies volaris, anterior surface, is concave in its upper 3/4, and gives origin to the flexor pollicis longus. It is broad and flat in its lower fourth, and affords insertion to the pronator quadratus. A prominent ridge limits the insertion of the pronator quadratus below, and between this and the inferior border is a triangular rough surface for the attachment of the volar radiocarpal ligament. At the junction of the upper and middle thirds of the volar surface is the nutrient foramen, which is directed obliquely upward. The dorsal surface, facies dorsalis, posterior surface, is convex and smooth in the upper third of its extent, and covered by the supinator. Its middle third is broad, slightly concave, and gives origin to the abductor pollicis longus above, and the extensor pollicis brevis below. Its lower third is broad, convex, and covered by the tendons of the muscles which subsequently run in the grooves on the lower end of the bone. The lateral surface, facies lateralis, external surface, is convex throughout its entire extent. Its upper third gives insertion to the supinator. About its center is a rough ridge for the insertion of the pronator teres. Its lower part is narrow and covered by the tendons of the abductor pollicis longus and extensor pollicis brevis. The lower extremity. The lower extremity is large, of quadrilateral form, and provided with 2 articular surfaces, one below for the carpus, and another at the medial side for the ulna. The carpal articular surface is triangular, concave, smooth, and divided by a slight antero-posterior ridge into 2 parts. Of these, the lateral triangular articulates with the navicular bone, the medial quadrilateral with the lunate bone. The articular surface for the ulna is called the ulnar notch, sigmoid cavity, of the radius. It is narrow, concave, smooth, and articulates with the head of the ulna. These 2 articular surfaces are separated by a prominent ridge, to which the base of the triangular articular disc is attached. This disc separates the wrist joint from the distal radio-ulnar articulation. This end of the bone has 3 non-articular surfaces, volar, dorsal, and lateral. The volar surface, rough and irregular, affords attachment to the volar radiocarpal ligament. The dorsal surface is convex, affords attachment to the dorsal radiocarpal ligament, and is marked by 3 grooves. Enumerated from the lateral side, the first groove is broad but shallow, and subdivided into 2 by a slight ridge. The lateral of these 2 transmits the tendon of the extensor carpi radialis longus, the medial the tendon of the extensor carpi radialis brevis. The second is deep but narrow, and bounded laterally by a sharply defined ridge. It is directed obliquely from above downward and lateralward, and transmits the tendon of the extensor pollicis longus. The third is broad for the passage of the tendons of the extensor indicis proprius and extensor digitorum communis. The lateral surface is prolonged obliquely downward into a strong conical projection, the styloid process, which gives attachment by its base to the tendon of the brachioradialis, and by its apex to the radial collateral ligament of the wrist joint. The lateral surface of this process is marked by a flat groove for the tendons of the abductor pollicis longus and extensor pollicis brevis. Structure. The long, narrow medullary cavity is enclosed in a strong wall of compact tissue which is thickest along the interosseous border, and thinnest at the extremities, except over the cup-shaped articular surface, fovea, of the head, where it is thickened. The trabeculi of the spongy tissue are somewhat arched at the upper end, and pass upward from the compact layer of the shaft to the fovea capituli. They are crossed by others parallel to the surface of the fovea. The arrangement at the lower end is somewhat similar. Ossification. The radius is ossified from 3 centers, one for the body and one for either extremity. That for the body makes its appearance near the center of the bone during the 8th week of fetal life. About the end of the 2nd year, ossification commences in the lower end, and at the 5th year in the upper end. The upper epiphysis fuses with the body at the age of 17 or 18 years, the lower about the age of 20. An additional center sometimes found in the radial tuberosity appears about the 14th or 15th year.
6b. The hand. Part 1. The carpus. The skeleton of the hand is subdivided into 3 segments: the carpus or wrist bones, the metacarpus or bones of the palm, and the phalanges or bones of the digits. The carpus. Ossa carpi. The carpal bones, 8 in number, are arranged in 2 rows. Those of the proximal row, from the radial to the ulnar side, are named the navicular, lunate, triangular, and pisiform. Those of the distal row, in the same order, are named the greater multangular, lesser multangular, capitate, and hamate. Common characteristics of the carpal bones. Each bone, excepting the pisiform, presents 6 surfaces. Of these, the volar or anterior and the dorsal or posterior surfaces are rough for ligamentous attachment, the dorsal surfaces being the broader, except in the navicular and lunate. The superior or proximal and inferior or distal surfaces are articular, the superior generally convex, the inferior concave. The medial and lateral surfaces are also articular where they are in contact with the contiguous bones, otherwise they are rough and tuberculated. The structure in all is similar, viz., cancellous tissue enclosed in a layer of compact bone. Bones of the proximal row, upper row. The navicular bone, os navicular manus, scaphoid bone. The navicular bone is the largest bone of the proximal row and has received its name from its fancied resemblance to a boat. It is situated at the radial side of the carpus, its long axis being from above downward, lateralward, and forward. The superior surface is convex, smooth, of triangular shape, and articulates with the lower end of the radius. The inferior surface, directed downward, lateralward, and backward, is also smooth, convex, and triangular, and is divided by a slight ridge into 2 parts, the lateral articulating with the greater multangular, the medial with the lesser multangular. On the dorsal surface is a narrow, rough groove which runs the entire length of the bone and serves for the attachment of ligaments. The volar surface is concave above and elevated at its lower and lateral part into a rounded projection, the tubercle, which is directed forward and gives attachment to the transverse carpal ligament and sometimes origin to a few fibers of the abductor pollicis brevis. The lateral surface is rough and narrow and gives attachment to the radial collateral ligament of the wrist. The medial surface presents two articular facets. Of these, the superior or smaller is flattened of semilunar form and articulates with the lunate bone. The inferior or larger is concave, forming with the lunate a concavity for the head of the capitate bone. Articulations. The navicular articulates with 5 bones, the radius proximally, greater and lesser multangulars distally, and capitate and lunate medially. The lunate bone, os lunatum, semilunar bone. The lunate bone may be distinguished by its deep concavity and crescentic outline. It is situated in the center of the proximal row of the carpus, between the navicular and triangular. The superior surface, convex and smooth, articulates with the radius. The inferior surface is deeply concave and of greater extent from before backward than transversely. It articulates with the head of the capitate and by a long narrow facet, separated by a ridge from the general surface, with the hamate. The dorsal and volar surfaces are rough for the attachment of ligaments, the former being the broader and of a somewhat rounded form. The lateral surface presents a narrow, flattened, semilunar facet for articulation with the navicular. The medial surface is marked by a smooth, quadrilateral facet for articulation with the triangular. Articulations. The lunate articulates with 5 bones, the radius proximally, capitate and hamate distally, navicular laterally, and triangular medially. The triangular bone, os tricatum, cuneiform bone. The triangular bone may be distinguished by its pyramidal shape and by an oval isolated facet for articulation with the pisiform bone. It is situated at the upper and ulnar side of the carpus. The superior surface presents a medial, rough, non-articular portion, and a lateral convex articular portion which articulates with the triangular articular disc of the wrist. The inferior surface, directed lateralward, is concave, sinuously curved, and smooth for articulation with the hamate. The dorsal surface is rough for the attachment of ligaments. The volar surface presents, on its medial part, an oval facet for articulation with the pisiform. Its lateral part is rough for ligamentous attachment. The lateral surface, the base of the pyramid, is marked by a flat, quadrilateral facet for articulation with the lunate. The medial surface, the summit of the pyramid, is pointed and roughened for the attachment of the ulnar collateral ligament of the wrist. Articulations. The triangular articulates with 3 bones, the lunate laterally, the pisiform in front, the hamate distally, and with the triangular articular disc which separates it from the lower end of the ulna. The pisiform bone, os pisiform. The pisiform bone may be known by its small size and by its presenting a single articular facet. It is situated on a plane anterior to the other carpal bones and is spheroidal in form. Its dorsal surface presents a smooth, oval facet for articulation with the triangular. This facet approaches the superior but not the inferior border of the bone. The volar surface is rounded and rough and gives attachment to the transverse carpal ligament and to the flexor carpi ulnaris and abductor digiti quinti. The lateral and medial surfaces are also rough, the former being concave, the latter usually convex. Articulation. The pisiform articulates with 1 bone, the triangular. Bones of the distal row, lower row. The greater multangular bone, Os multangulum majus. Trapezium. The greater multangular bone may be distinguished by a deep groove on its volar surface. It is situated at the radial side of the carpus, between the navicular and the first metacarpal bone. The superior surface is directed upward and medialward. Medially it is smooth, and articulates with the navicular. Laterally it is rough, and continuous with the lateral surface. The inferior surface is oval, concave from side to side, convex from before backward, so as to form a saddle-shaped surface for articulation with the base of the first metacarpal bone. The dorsal surface is rough, the volar surface is narrow and rough. At its upper part is a deep groove, running from above obliquely downward and medialward. It transmits the tendon of the flexor carpi radialis, and is bounded laterally by an oblique ridge. This surface gives origin to the opponens pollicis and to the abductor and flexor pollicis brevis. It also affords attachment to the transverse carpal ligament. The lateral surface is broad and rough for the attachment of ligaments. The medial surface presents two facets. The upper, large and concave, articulates with the lesser multangular. The lower, small and oval, with the base of the second metacarpal. Articulations. The greater multangular articulates with four bones: the navicular proximally, the first metacarpal distally, and the lesser multangular and second metacarpal medially. The lesser multangular bone, os multangulum minus, trapezoid bone. The lesser multangular is the smallest bone in the distal row. It may be known by its wedge-shaped form, the broad end of the wedge constituting the dorsal, the narrow end the volar surface, and by its having four articular facets touching each other and separated by sharp edges. The superior surface, quadrilateral, smooth, and slightly concave, articulates with the navicular. The inferior surface articulates with the proximal end of the second metacarpal bone. It is convex from side to side, concave from before backward, and subdivided by an elevated ridge into two unequal facets. The dorsal and volar surfaces are rough for the attachment of ligaments, the former being the larger of the two. The lateral surface, convex and smooth, articulates with the greater multangular. The medial surface is concave and smooth in front for articulation with the capitate, rough behind for the attachment of an interosseous ligament. Articulations. The lesser multangular articulates with four bones: the navicular proximally, second metacarpal distally, greater multangular laterally, and capitate medially. The capitate bone, os capitatum, os magnum. The capitate bone is the largest of the carpal bones, and occupies the center of the wrist. It presents above a rounded portion or head, which is received into the concavity formed by the navicular and lunate. A constricted portion or neck, and below this the body. The superior surface is round, smooth, and articulates with the lunate. The inferior surface is divided by two ridges into three facets, for articulation with the second, third, and fourth metacarpal bones, that for the third being the largest. The dorsal surface is broad and rough. The volar surface is narrow, rounded, and rough, for the attachment of ligaments, and a part of the adductor pollicis obliquus. The lateral surface articulates with the lesser multangular by a small facet at its anterior-inferior angle, behind which is a rough depression for the attachment of an interosseous ligament. Above this is a deep, rough groove forming part of the neck and serving for the attachment of ligaments. It is bounded superiorly by a smooth, convex surface for articulation with the navicular. The medial surface articulates with the hamate by a smooth, concave, oblong facet, which occupies its posterior and superior parts. It is rough in front for the attachment of an interosseous ligament. Articulations. The capitate articulates with seven bones: the navicular and lunate proximally, the second, third, and fourth metacarpals distally, the lesser multangular on the radial side, and the hamate on the ulnar side. The hamate bone, os hamatum, unciform bone. The hamate bone may be readily distinguished by its wedge-shaped form and the hook-like process which projects from its volar surface. It is situated at the medial and lower angle of the carpus, with its base downward, resting on the fourth and fifth metacarpal bones, and its apex directed upward and lateralward. The superior surface, the apex of the wedge, is narrow, convex, smooth, and articulates with the lunate. The inferior surface articulates with the fourth and fifth metacarpal bones by concave facets which are separated by a ridge. The dorsal surface is triangular and rough for ligamentous attachment. The volar surface presents, at its lower and ulnar side, a curved, hook-like process, the hamulus, directed forward and lateralward. This process gives attachment by its apex to the transverse carpal ligament and the flexor carpi ulnaris, by its medial surface to the flexor brevis and opponens digiti quinti. Its lateral side is grooved for the passage of the flexor tendons into the palm of the hand. It is one of the four eminences on the front of the carpus to which the transverse carpal ligament of the wrist is attached, the others being the pisiform medially, the oblique ridge of the greater multangular, and the tubercle of the navicular laterally. The medial surface articulates with the triangular bone by an oblong facet, cut obliquely from above, downward and medialward. The lateral surface articulates with the capitate by its upper and posterior part, the remaining portion being rough for the attachment of ligaments. Articulations. The hamate articulates with 5 bones, the lunate proximally, the fourth and fifth metacarpals distally, the triangular medially, the capitate laterally. 6b, Part 2, the metacarpus. The metacarpus consists of 5 cylindrical bones which are numbered from the lateral side, ossa metacarpalia 1 through 5. Each consists of a body and 2 extremities. Common characteristics of the metacarpal bones. The body, corpus, shaft. The body is prismoid in form and curved so as to be convex in the longitudinal direction behind, concave in front. It presents 3 surfaces, medial, lateral, and dorsal. The medial and lateral surfaces are concave for the attachment of the interossei and separated from one another by a prominent anterior ridge. The dorsal surface presents in its distal 2/3 a smooth, triangular, flattened area which is covered in the fresh state by the tendons of the extensor muscles. This surface is bounded by 2 lines which commence in small tubercles situated on either side of the digital extremity and passing upward converge and meet some distance above the center of the bone and form a ridge which runs along the rest of the dorsal surface to the carpal extremity. This ridge separates 2 sloping surfaces for the attachment of the interossei dorsalis. To the tubercles on the digital extremities are attached the collateral ligaments of the metacarpophalangeal joints. The base or carpal extremity, basis, is of a cuboidal form and broader behind than in front. It articulates with the carpus and with the adjoining metacarpal bones. Its dorsal and volar surfaces are rough for the attachment of ligaments. The head or digital extremity, capitulum, presents an oblong surface markedly convex from before backward, less so transversely, and flattened from side to side. It articulates with the proximal phalanx. It is broader and extends farther upward on the volar than on the dorsal aspect, and is longer in the antero-posterior than in the transverse diameter. On either side of the head is a tubercle for the attachment of the collateral ligament of the metacarpophalangeal joint. The dorsal surface, broad and flat, supports the extensor tendons. The volar surface is grooved in the middle line for the passage of the flexor tendons, and marked on either side by an articular eminence continuous with the terminal articular surface. Characteristics of the individual metacarpal bones. The first metacarpal bone, os metacarpale 1, metacarpal bone of the thumb, is shorter and stouter than the others, diverges to a greater degree from the carpus, and its volar surface is directed toward the palm. The body is flattened and broad on its dorsal surface, and does not present the ridge which is found on the other metacarpal bones. Its volar surface is concave from above downward. On its radial border is inserted the Apennine pollicis. Its ulnar border gives origin to the lateral head of the first interosseous dorsalis. The base presents a concavo-convex surface for articulation with the greater multangulum. It has no facets on its sides, but on its radial side is a tubercle for the insertion of the abductor pollicis longus. The head is less convex than those of the other metacarpal bones, and is broader from side to side than from before backward. On its volar surface are two articular eminences, of which the lateral is the larger, for the two sesamoid bones and the tendons of the flexor pollicis brevis. The second metacarpal bone, os metacarpale 2, metacarpal bone of the index finger, is the longest, and its base the largest of the four remaining bones. Its base is prolonged upward and medialward, forming a prominent ridge. It presents four articular facets, three on the upper surface and one on the ulnar side. Of the facets on the upper surface, the intermediate is the largest and is concave from side to side, convex from before backward for articulation with the lesser multangulum. The lateral is small, flat, and oval for articulation with the greater multangulum. The medial, on the summit of the ridge, is long and narrow for articulation with the capitate. The facet on the ulnar side articulates with a third metacarpal. The extensor carpi radialis longus is inserted on the dorsal surface and the flexor carpi radialis on the volar surface of the base. The third metacarpal bone, os metacarpale 3, metacarpal bone of the middle finger, is a little smaller than the second. The dorsal aspect of its base presents on its radial side a pyramidal eminence, the styloid process, which extends upward behind the capitate. Immediately distal to this is a rough surface for the attachment of the extensor carpi radialis brevis. The carpal articular facet is concave behind, flat in front, and articulates with the capitate. On the radial side is a smooth concave facet for articulation with the second metacarpal, and on the ulnar side two small oval facets for the fourth metacarpal. The fourth metacarpal bone, os metacarpale 4, metacarpal bone of the ring finger, is shorter and smaller than the third. The base is small and quadrilateral. Its superior surface presents two facets, a large one medially for articulation with the hamate, and a small one laterally for the capitate. On the radial side are two oval facets for articulation with the third metacarpal, and on the ulnar side a single concave facet for the fifth metacarpal. The fifth metacarpal bone, os metacarpale 5, metacarpal bone of the little finger, presents on its base one facet on its superior surface, which is concavo-convex and articulates with the hamate, and one on its radial side, which articulates with the fourth metacarpal. On its ulnar side is a prominent tubercle for the insertion of the tendon of the extensor carpi ulnaris. The dorsal surface of the body is divided by an oblique ridge, which extends from near the ulnar side of the base to the radial side of the head. The lateral part of this surface serves for the attachment of the fourth interosseous dorsalis. The medial part is smooth, triangular, and covered by the extensor tendons of the little finger. Articulations. Besides their phalangeal articulations, the metacarpal bones articulate as follows: the first with the greater multangulum, the second with the greater multangulum, lesser multangulum, capitate and third metacarpal, the third with the capitate and second and fourth metacarpals, the fourth with the capitate, hamate, and third and fifth metacarpals, and the fifth with the hamate and fourth metacarpal.
The phalanges of the hand. Phalanges digitorum manus. The phalanges are 14 in number, 3 for each finger and 2 for the thumb. Each consists of a body and 2 extremities. The body tapers from above downward, is convex posteriorly, concave in front from above downward, flat from side to side. Its sides are marked by ruff, which give attachment to the fibrous sheaths of the flexor tendons. The proximal extremities of the bones of the first row present oval, concave articular surfaces, broader from side to side than from before backward. The proximal extremity of each of the bones of the second and third rows present a double concavity separated by a median ridge. The distal extremities are smaller than the proximal, and each ends in two condyles separated by a shallow groove. The articular surface extends farther on the volar than on the dorsal surface, a condition best marked in the bones of the first row. The ungual phalanges are convex on their dorsal and flat on their volar surfaces. They are recognized by their small size, and by a roughened, elevated surface of a horseshoe form on the volar surface of the distal extremity of each, which serves to support the sensitive pulp of the finger. Articulations. In the forefingers, the phalanges of the first row articulate with those of the second row, and with the metacarpals. The phalanges of the second row with those of the first and third rows, and the ungual phalanges with those of the second row. In the thumb, which has only two phalanges, the first phalanx articulates by its proximal extremity with the metacarpal bone, and by its distal with the ungual phalanx. Ossification of the bones of the hand. The carpal bones are each ossified from a single center, and ossification proceeds in the following order. In the capitate and hamate during the first year, the former preceding the latter. In the triangular during the third year. In the lunate and greater multangular during the fifth year, the former preceding the latter. In the navicular during the sixth year. In the lesser multangular during the eighth year, and in the pisiform about the 12th year. Occasionally an additional bone, the os central, is found on the back of the carpus, lying between the navicular, less multangular, and capitate. During the second month of fetal life it is represented by a small cartilaginous nodule, which usually fuses with the cartilaginous navicular. Sometimes the styloid process of the third metacarpal is detached and forms an additional ossicle. The metacarpal bones are each ossified from two centers, one for the body and one for the distal extremity of each of the second, third, fourth, and fifth bones, one for the body and one for the base of the first metacarpal bone. The first metacarpal bone is therefore ossified in the same manner as the phalanges, and this has led some anatomists to regard the thumb as being made up of three phalanges, and not of a metacarpal bone and two phalanges. Ossification commences in the middle of the body about the 8th or 9th week of fetal life, the centers for the second and third metacarpals being the first, and that for the first metacarpal the last to appear. About the third year, the distal extremities of the metacarpals of the fingers and the base of the metacarpal of the thumb begins to ossify. They unite with the bodies about the 20th year. The phalanges are each ossified from two centers, one for the body and one for the proximal extremity. Ossification begins in the body about the 8th week of fetal life. Ossification of the proximal extremity commences in the bones of the first row between the third and fourth years, and a year later in those of the second and third rows. The two centers become united in each row between the 18th and 20th years. In the ungual phalanges, the centers of the body appear in the distal extremities of the phalanges, instead of the middle of the bodies, as in the other phalanges. Moreover, of all the bones in the hand, the ungual phalanges are the first to ossify. Footnote: Alan Thompson demonstrated the fact that the first metacarpal bone is often developed from three centers. That is to say, there is a separate nucleus for the distal end, forming a distinct epiphysis visible at the age of 7 or 8 years. He also stated that there are traces of a proximal epiphysis in the second metacarpal bone. End footnote.
Chapter 6C. The Bones of the Lower Extremity. Section 1. The Hip Bone. Ossa extremitatis inferioris and os coxae innominate bone. The hip bone is a large, flattened, irregularly shaped bone, constricted in the center and expanded above and below. It meets its fellow on the opposite side in the middle line in front, and together they form the sides and anterior wall of the pelvic cavity. It consists of three parts, the ilium, ischium, and pubis, which are distinct from each other in the young subject, but are fused in the adult. The union of the three parts takes place in and around a large, cuff-shaped articular cavity, the acetabulum, which is situated near the middle of the outer surface of the bone. The ilium, so called because it supports the flank, is the superior, broad, and expanded portion which extends upward from the acetabulum. The ischium is the lowest and strongest portion of the bone. It proceeds downward from the acetabulum, expands into a large tuberosity, and then, curving forward, forms with the pubis a large aperture, the obturator foramen. The pubis extends medialward and downward from the acetabulum, and articulates in the middle line with the bone of the opposite side. It forms the front of the pelvis and supports the external organs of generation. The ilium, os ilii. The ilium is divisible into two parts, the body and the ala. The separation is indicated on the internal surface by a curved line, the arcuate line, and on the external surface by the margin of the acetabulum. The body, corpus os ilii. The body enters into the formation of the acetabulum, of which it forms rather less than 2/5. Its external surface is partly articular, partly non-articular. The articular segment forms part of the lunate surface of the acetabulum. The non-articular portion contributes to the acetabular fossa. The internal surface of the body is part of the wall of the lesser pelvis and gives origin to some fibers of the obturator internus. Below it is continuous with the pelvic surfaces of the ischium and pubis, only a faint line indicating the place of union. The ala os ilii. The ala is the large expanded portion which bounds the greater pelvis laterally. It presents for examination two surfaces, an external and an internal, a crest and two borders, an anterior and a posterior. The external surface, known as the dorsum ilii, is directed backward and lateralward behind, and downward and lateralward in front. It is smooth, convex in front, deeply concave behind, bounded above by the crest, below by the upper border of the acetabulum, in front and behind by the anterior and posterior borders. This surface is crossed in an arched direction by three lines, the posterior, anterior, and inferior gluteal lines. The posterior gluteal line, superior curved line, the shortest of the three, begins at the crest about 5 centimeters in front of its posterior extremity. It is at first distinctly marked, but as it passes downward to the upper part of the greater sciatic notch where it ends, it becomes less distinct and is often altogether lost. Behind this line is a narrow semilunar surface, the upper part of which is rough and gives origin to a portion of the gluteus maximus. The lower part is smooth and has no muscular fibers attached to it. The anterior gluteal line, the middle curved line, the longest of the three, begins at the crest about 4 centimeters behind its anterior extremity, and, taking a curved direction downward and backward, ends at the upper part of the greater sciatic notch. The space between the anterior and posterior gluteal lines and the crest is concave, and gives origin to the gluteus medius. Near the middle of this line a nutrient foramen is often seen. The inferior gluteal line, inferior curved line, the least distinct of the three, begins in front at the notch on the anterior border, and curving backward and downward, ends near the middle of the greater sciatic notch. The surface of bone included between the anterior and inferior gluteal lines is concave from above downward, convex from before backward, and gives origin to the gluteus minimus. Between the inferior gluteal line and the upper part of the acetabulum is a rough, shallow groove, from which the reflected tendon of the rectus femoris arises. The internal surface of the ala is bounded above by the crest, below by the arcuate line, in front and behind by the anterior and posterior borders. It presents a large, smooth, concave surface called the iliac fossa, which gives origin to the iliacus and is perforated at its inner part by a nutrient canal, and below this a smooth, rounded border, the arcuate line, which runs downward, forward, and medialward. Behind the iliac fossa is a rough surface divided into two portions, an anterior and a posterior. The anterior surface, auricular surface, so called from its resemblance in shape to the ear, is coated with cartilage in the fresh state, and articulates with a similar surface on the side of the sacrum. The posterior portion, known as the iliac tuberosity, is elevated and rough for the attachment of the posterior sacroiliac ligaments and for the origins of the sacrospinalis and multifidus. Below and in front of the auricular surface is the preauricular sulcus, more commonly present and better marked in the female than in the male. To it is attached the pelvic portion of the anterior sacroiliac ligament. The crest of the ilium is convex in its general outline, but is sinuously curved, being concave inward in front, concave outward behind. It is thinner at the center than at the extremities, and ends in the anterior and posterior superior iliac spines. The surface of the crest is broad and divided into external and internal lips, and an intermediate line. About 5 centimeters behind the anterior superior iliac spine, there is a prominent tubercle on the outer lip. To the external lip are attached the tensor fasciae latae, obliquus externus abdominis, and latissimus dorsi, and along its whole length the fascia lata. To the intermediate line the obliquus internus abdominis, to the internal lip the fascia iliaca, the transverse abdominis, quadratus lumborum, sacrospinalis, and iliacus. The anterior border of the ala is concave. It presents two projections separated by a notch. Of these, the uppermost, situated at the junction of the crest and anterior border, is called the anterior superior iliac spine. Its outer border gives attachment to the fascia lata and the tensor fasciae latae, its inner border to the iliacus, while its extremity affords attachment to the inguinal ligament and gives origin to the sartorius. Beneath this eminence is a notch from which the sartorius takes origin and across which the lateral femoral cutaneous nerve passes. Below the notch is the anterior inferior iliac spine, which ends in the upper lip of the acetabulum. It gives attachment to the straight tendon of the rectus femoris and to the iliofemoral ligament of the hip joint. Medial to the anterior inferior spine is a broad, shallow groove over which the iliacus and psoas major pass. This groove is bounded medially by an eminence, the iliopectineal eminence, which marks the point of union of the ilium and pubis. The posterior border of the ala, shorter than the anterior, also presents two projections separated by a notch, the posterior superior iliac spine and the posterior inferior iliac spine. The former serves for the attachment of the oblique portion of the posterior sacroiliac ligaments and the multifidus. The latter corresponds with the posterior extremity of the auricular surface. Below the posterior inferior spine is a deep notch, the greater sciatic notch. The ischium, os ischii. The ischium forms the lower and back part of the hip bone. It is divisible into 3 portions, a body and 2 rami. The body, corpus os ischii. The body enters into and constitutes a little more than 2/5 of the acetabulum. Its external surface forms part of the lunate surface of the acetabulum and a portion of the acetabular fossa. Its internal surface is part of the wall of the lesser pelvis. It gives origin to some fibers of the obturator internus. Its anterior border projects as the posterior obturator tubercle. From its posterior border there extends backward a thin and pointed triangular eminence, the ischial spine, more or less elongated in different subjects. The external surface of the spine gives attachment to the gemellus superior, its internal surface to the coccygeus, levator ani, and the pelvic fascia, while to the pointed extremity the sacrospinous ligament is attached. Above the spine is a large notch, the greater sciatic notch. Converted into a foramen by the sacrospinous ligament, it transmits the piriformis, the superior and inferior gluteal vessels and nerves, the sciatic and posterior femoral cutaneous nerves, the internal pudendal vessels and nerve, and the nerves to the obturator internus and quadratus femoris. Of these, the superior gluteal vessels and nerve pass out above the piriformis, other structures below it. Below the spine is a smaller notch, the lesser sciatic notch. It is smooth, coated in the recent state with cartilage, the surface of which presents 2 or 3 ridges corresponding to the subdivisions of the tendon of the obturator internus, which winds over it. It is converted into a foramen by the sacrotuberous and sacrospinous ligaments, and transmits the tendon of the obturator internus, the nerve which supplies that muscle, and the internal pudendal vessels and nerve. The superior ramus, ramus superior ossetii, descending ramus. The superior ramus projects downward and backward from the body, and presents for examination 3 surfaces: external, internal, and posterior. The external surface is quadrilateral in shape. It is bounded above by a groove which lodges the tendon of the obturator externus. Below is continuous with the inferior ramus. In front it is limited by the posterior margin of the obturator foramen. Behind, a prominent margin separates it from the posterior surface. In front of this margin, the surface gives origin to the quadratus femoris, and anterior to this, to some of the fibers of origin of the obturator externus. The lower part of the surface gives origin to part of the adductor magnus. The internal surface forms part of the bony wall of the lesser pelvis. In front it is limited by the posterior margin of the obturator foramen. Below it is bounded by a sharp ridge which gives attachment to a falciform prolongation of the sacrotuberous ligament, and, more anteriorly, gives origin to the transversus perenni and ischiocavernosus. Posteriorly, the ramus forms a large swelling, the tuberosity of the ischium, which is divided into 2 portions: a lower, rough, somewhat triangular part, and an upper, smooth, quadrilateral portion. The lower portion is subdivided by a prominent longitudinal ridge, passing from base to apex, into 2 parts. The outer gives attachment to the adductor magnus, the inner to the sacrotuberous ligament. The upper portion is subdivided into 2 areas by an oblique ridge, which runs downward and outward. From the upper and outer area, the semimembranosus arises. From the lower and inner, the long head of the biceps femoris and the semitendinosus. The inferior ramus, ramus inferior ossetii, ascending ramus. The inferior ramus is the thin, flattened part of the ischium, which ascends from the superior ramus and joins the inferior ramus of the pubis, the junction being indicated in the adult by a raised line. The outer surface is uneven for the origin of the obturator externus and some of the fibers of the adductor magnus. Its inner surface forms part of the anterior wall of the pelvis. Its medial border is thick, rough, slightly everted, forms part of the outlet of the pelvis, and presents 2 ridges and an intervening space. The ridges are continuous with similar ones on the inferior ramus of the pubis. To the outer is attached the deep layer of the superficial perineal fascia, fascia of Colles, and to the inner the inferior fascia of the urogenital diaphragm. If these 2 ridges be traced downward, they will be found to join with each other just behind the point of origin of the transversus perenni. Here the 2 layers of the fascia are continuous behind the posterior border of the muscle. To the intervening space, just in front of the point of origin of the ridges, the transversus perenni is attached, and in front of this a portion of the crus penis vel clitoridis and the ischiocavernosus. Its lateral border is thin and sharp, and forms part of the medial margin of the obturator foramen. The pubis, os pubis. The pubis, the anterior part of the hip bone, is divisible into a body, a superior, and an inferior ramus. The body, Corpus os pubis. The body forms 1/5 of the acetabulum, contributing by its external surface both to the lunate surface and the acetabular fossa. Its internal surface enters into the formation of the wall of the lesser pelvis and gives origin to a portion of the obturator internus. The superior ramus, ramus superior os pubis, ascending ramus. The superior ramus extends from the body to the median plane where it articulates with its fellow of the opposite side. It is conveniently described in two portions, viz. A medial flattened part and a narrow lateral prismoidal portion. The medial portion of the superior ramus, formally described as the body of the pubis, is somewhat quadrilateral in shape, and presents for examination 2 surfaces and 3 borders. The anterior surface is rough, directed downward and outward, and serves for the origin of various muscles. The adductor longus arises from the upper and medial angle, immediately below the crest. Lower down, the obturator externus, the adductor brevis, and the upper part of the gracilis take origin. The posterior surface, convex from above downward, concave from side to side, is smooth and forms part of the anterior wall of the pelvis. It gives origin to the levator ani and obturator internus, and attachment to the puboprostatic ligaments and to a few muscular fibers prolonged from the bladder. The upper border presents a prominent tubercle, the pubic tubercle, pubic spine, which projects forward. The inferior crus of the subcutaneous inguinal ring, external abdominal ring, and the inguinal ligament, Poupart's ligament, are attached to it. Passing upward and lateralward from the pubic tubercle is a well-defined ridge, forming a part of the pectineal line, which marks the brim of the lesser pelvis. To it are attached a portion of the inguinal falx, conjoined tendon of obliquus internus and transversus, the lacunar ligament, Gimbernaut's ligament, and the reflected inguinal ligament, triangular fascia. Medial to the pubic tubercle is the crest, which extends from this process to the medial end of the bone. It affords attachment to the inguinal falx and to the rectus abdominis and pyramidalis. The point of junction of the crest with the medial border of the bone is called the angle. To it, as well as to the symphysis, the superior crus of the subcutaneous inguinal ring is attached. The medial border is articular. It is oval and is marked by 8 or 9 transverse ridges, or a series of nipple-like processes arranged in rows, separated by grooves. They serve for the attachment of a thin layer of cartilage, which intervenes between it and the interpubic fibrocartilaginous lamina. The lateral border presents a sharp margin, the obturator crest, which forms part of the circumference of the obturator foramen, and affords attachment to the obturator membrane. The lateral portion of the ascending ramus has 3 surfaces, superior, inferior, and posterior. The superior surface presents a continuation of the pectineal line, already mentioned as commencing at the pubic tubercle. In front of this line, the surface of bone is triangular in form, wider laterally than medially, and is covered by the pectineus. The surface is bounded laterally by a rough eminence, the iliopectineal eminence, which serves to indicate the point of junction of the ilium and pubis, and below by a prominent ridge which extends from the acetabular notch to the pubic tubercle. The inferior surface forms the upper boundary of the obturator foramen and presents laterally a broad and deep oblique groove for the passage of the obturator vessels and nerve, and medially a sharp margin, the obturator crest, forming part of the circumference of the obturator foramen and giving attachment to the obturator membrane. The posterior surface constitutes part of the anterior boundary of the lesser pelvis. It is smooth, convex from above downward, and affords origin to some fibers of the obturator internus. The inferior ramus, ramus inferior os pubis, descending ramus. The inferior ramus is thin and flattened. It passes lateralward and downward from the medial end of the superior ramus. It becomes narrower as it descends and joins with the inferior ramus of the ischium below the obturator foramen. Its anterior angle is rough for the origin of muscles. The gracilis along its medial border, a portion of the obturator externus where it enters into the formation of the obturator foramen, and between these two, the adductoris brevis and magnus, the former being the more medial. The posterior surface is smooth and gives origin to the obturator internus, and, close to the medial margin, to the constrictor urethrae. The medial border is thick, rough, and everted, especially in females. It presents two ridges, separated by an intervening space. The ridges extend downward and are continuous with similar ridges on the inferior ramus of the ischium. To the external, it is attached to the fascia of Colles, and to the internal, the inferior fascia of the urogenital diaphragm. The lateral border is thin and sharp, forms part of the circumference of the obturator foramen, and gives attachment to the obturator membrane. The acetabulum, cotyloid cavity. The acetabulum is a deep, cup-shaped, hemispherical depression directed downward, lateralward, and forward. It is formed medially by the pubis, above by the ilium, laterally and below by the ischium. A little less than 2/5 is contributed by the ilium, a little more than 2/5 by the ischium, and the remaining fifth by the pubis. It is bounded by a prominent uneven rim, which is thick and strong above, and serves for the attachment of the glenoidal labrum, cotyloid ligament, which contracts its orifice and deepens the surface for articulation. It presents below a deep notch, the acetabular notch, which is continuous with a circular, non-articular depression, the acetabular fossa, at the bottom of the cavity. This depression is perforated by numerous apertures and lodges a mass of fat. The notch is converted into a foramen by the transverse ligament. Through the foramen, nutrient vessels and nerves enter the joint. The margins of the notch serve for the attachment of the ligamentum teres. The rest of the acetabulum is formed by a curved articular surface, the lunate surface, for articulation with the head of the femur. The obturator foramen, foramen obturatum, thyroid foramen. The obturator foramen is a large aperture situated between the ischium and pubis. In the male it is large and of an oval form, its longest diameter slanting obliquely from before backward. In the female it is smaller and more triangular. It is bounded by a thin, uneven margin to which a strong membrane is attached, and presents superiorly a deep groove, the obturator groove, which runs from the pelvis obliquely medialward and downward. This groove is converted into a canal by a ligamentous band, a specialized part of the obturator membrane, attached to two tubercles. One, the posterior obturator tubercle, on the medial border of the ischium, just in front of the acetabular notch. The other, the anterior obturator tubercle, on the obturator crest in the superior ramus of the pubis. Through the canal, the obturator vessels and nerve pass out of the pelvis. Structure. The thicker parts of the bone consist of cancellous tissue, enclosed between two layers of compact tissue. The thinner parts, as at the bottom of the acetabulum and the center of the iliac fossa, are usually semi-transparent and composed entirely of compact tissue. Ossification. The hip bone is ossified from 8 centers, 3 primary, 1 each for the ilium, ischium, and pubis, and 5 secondary, 1 each for the crest of the ilium, the anterior inferior spine, said to occur more frequently in the male than in the female, the tuberosity of the ischium, the pubic symphysis, more frequent in the female than in the male, and 1 or more for the Y-shaped piece at the bottom of the acetabulum. The centers appear in the following order. In the lower part of the ilium, immediately above the greater sciatic notch, about the 8th or 9th week of fetal life. In the superior ramus of the ischium, about the 3rd month. In the superior ramus of the pubis, between the 4th and 5th months. At birth, the 3 primary centers are quite separate, the crest, the bottom of the acetabulum, and the ischial tuberosity, and the inferior rami of the ischium and pubis still being cartilaginous. By the 7th or 8th year, the inferior rami of the pubis and ischium are almost completely united by bone. About the 13th or 14th year, the three primary centers have extended their growth into the bottom of the acetabulum, and are there separated from each other by a Y-shaped portion of cartilage, which now presents traces of ossification, often by two or more centers. One of these, the os acetabuli, appears about the age of 12 between the ilium and pubis, and fuses with them about the age of 18. It forms the pubic part of the acetabulum. The ilium and ischium then become joined, and lastly the pubis and ischium, through the intervention of this Y-shaped portion. At about the age of puberty, ossification takes place in each of the remaining portions, and they join with the rest of the bone between the 20th and 25th years. Separate centers are frequently found for the pubic tubercle and the ischial spine, and for the crest and angle of the pubis. Articulations. The hip bone articulates with its fellow of the opposite side and with the sacrum and femur.
The pelvis. The pelvis, so called from its resemblance to a basin, is a bony ring interposed between the movable vertebrae of the vertebral column which it supports and the lower limbs upon which it rests. It is longer and more massively constructed than the wall of the cranial or thoracic cavities, and is composed of four bones, the two hip bones laterally and in front, and the sacrum and coccyx behind. The greater or false pelvis. The greater pelvis is the expanded portion of the cavity situated above and in front of the pelvic brim. It is bounded on either side by the ilium. In front it is incomplete, presenting a wide interval between the anterior borders of the ilia, which is filled up in the fresh state by the parietes of the abdomen. Behind is a deep notch on either side between the ilium and the base of the sacrum. It supports the intestines and transmits part of their weight to the anterior wall of the abdomen. The true or lesser pelvis. The lesser pelvis is that part of the pelvic cavity which is situated below and behind the pelvic brim. Its bony walls are more complete than those of the greater pelvis. For convenience of description, it is divided into an inlet bounded by the superior circumference, and outlet bounded by the inferior circumference, and a cavity. The superior circumference. The superior circumference forms the brim of the pelvis, an included space being called the superior aperture or inlet. It is formed laterally by the pectineal and arcuate lines, in front of the crest of the pubes, and behind by the anterior margin of the base of the sacrum and sacrovertebral angle. The superior aperture is somewhat heart-shaped, obtusely pointed in front, diverging on either side, and encroached upon behind by the projection forward of the promontory of the sacrum. It has three principal diameters: antero to posterior, transverse, and oblique. The anterior-posterior or conjugate diameter extends from the sacrovertebral angle to the symphysis pubis. Its average measurement is about 110 millimeters in the female. The transverse diameter extends from the greatest width of the superior aperture, from the middle of the brim on one side to the same point on the opposite. Its average measurement is about 135 millimeters in the female. The oblique diameter extends from the iliopectineal eminence of one side to the sacroiliac articulation of the opposite side. Its average measurement is about 125 millimeters in the female. The cavity of the lesser pelvis is bounded in front and below by the symphysis pubis, and the superior rami of the pubes, above and behind, by the pelvic surfaces of the sacrum and coccyx, which, curving forward above and below, contract the superior and inferior apertures of the cavity, laterally by a broad, smooth, quadrilangular area of bone, corresponding to the inner surfaces of the body and superior ramus of the ischium, and that part of the ilium which is below the arcuate line. From this description it will be seen that the cavity of the lesser pelvis is a short, curved canal, considerably deeper on its posterior than on its anterior wall. It contains, in the fresh subject, the pelvic colon, rectum, bladder, and some of the organs of generation. The rectum is placed at the back of the pelvis, in the curve of the sacrum and coccyx. The bladder is in front, behind the pubic symphysis. In the female, the uterus and vagina occupy the interval between these viscera. The lower circumference. The lower circumference of the pelvis is very irregular. The space enclosed by it is named the inferior aperture or outlet, and is bounded behind by the point of the coccyx, and laterally by the ischial tuberosities. These eminences are separated by 3 notches, one in front, the pubic arch, formed by the convergence of the inferior rami of the ischium and the pubis on either side. The other notches, one on either side, are formed by the sacrum and coccyx behind, the ischium in front, and the ilium above. They are called the sciatic notches. In the natural state they are converted into foramina by the sacrotuberous and sacrospinous ligaments. When the ligaments are in situ, the inferior aperture of the pelvis is lozenge-shaped, bounded in front by the pubic arcuate ligament and the inferior rami of the pubes and ischia, laterally by the ischial tuberosities, and behind by the sacrotuberous ligaments and the tip of the coccyx. The diameters of the outlet of the pelvis are 2, anterior-posterior, and transverse. The antero-posterior diameter extends from the tip of the coccyx to the lower part of the pubic symphysis. Its measurement is from 90 to 115 millimeters in the female. It varies with the length of the coccyx and is capable of increase or diminution on account of the mobility of that bone. The transverse diameter, measured from the posterior parts of the ischial tuberosities, is about 115 millimeters in the female. Footnote: the measurements of the pelvis given above are fairly accurate, but different figures are given by various authors, no doubt due mainly to differences in the physique and stature of the population from whom the measurements have been taken. End of footnote. Axes. A line at right angles to the plane of the superior aperture at its center would, if prolonged, pass through the umbilicus above and the middle of the coccyx below. The axis of the superior aperture is therefore directed downward and backward. The axis of the inferior aperture, produced upward, would touch the base of the sacrum, and is also directed downward and slightly backward. The axis of the cavity, i.e., an axis at right angles to a series of planes between those of the superior and inferior apertures, is curved like the cavity itself. This curve corresponds to the concavity of the sacrum and coccyx, the extremities being indicated by the central points of the superior and inferior apertures. A knowledge of the direction of these axes serves to explain the course of the fetus in the passage through the pelvis during parturition. Position of the pelvis. In the erect posture, the pelvis is placed obliquely with regard to the trunk. The plane of the superior aperture forms an angle of from 50 degrees to 60 degrees, and that of the inferior aperture one of about 15 degrees with the horizontal plane. The pelvic surface of the symphysis pubis looks upward and backward, the concavity of the sacrum and coccyx downward and forward. The position of the pelvis in the erect posture may be indicated by holding it so that the anterior superior iliac spines and the front of the top of the symphysis pubis are in the same vertical plane. Differences between the male and female pelvis. The female pelvis is distinguished from that of the male by its bones being more delicate and its depth less. The whole pelvis is less massive, and its muscular impressions are slightly marked. The ilia are less sloped, and the anterior iliac spines more widely separated, hence the greater lateral prominence of the hips. The preauricular sulcus is more commonly present and better marked. The superior aperture of the lesser pelvis is larger in the female than in the male. It is more nearly circular, and its obliquity is greater. The cavity is shallower and wider. The sacrum is shorter, wider, and its upper part is less curved. The obturator foramen are triangular in shape and smaller in size than in the male. The superior aperture is larger and the coccyx more movable. The sciatic notches are wider and shallower, and the spines of the ischia project less inward. The acetabula are smaller and look more distinctly forward. The ischial tuberosities and the acetabula are wider apart, and the former are more everted. The pubic symphysis is less deep, and the pubic arch is wider and more rounded than in the male, where it is an angle rather than an arch. The size of the pelvis varies not only in the two sexes, but also in different members of the same sex, and does not appear to be influenced in any way by the height of the individual. Women of shorter stature, as a rule, have broad pelvis. Occasionally the pelvis is equally contracted in all its dimensions, so much so that all its diameters measure 12.5 millimeters, less than the average, and this even in well-formed women of average height. The principal divergence, however, are found at the superior aperture and affect the relation of the anterior-posterior to the transverse diameter. Thus, the superior aperture may be elliptical either in a transverse or an antero-posterior direction, the transverse diameter in the former and the antero-posterior in the latter, greatly exceeding the other diameters. In other instances, it is almost circular. In the fetus, and for several years after birth, the pelvis is smaller in proportion than in the adult, and the projection of the sacrovertebral angle less marked. The characteristic differences between the male and female pelvis are distinctly indicated as early as the 4th month of fetal life. Abnormalities. There is arrest of development in the bones of the pelvis in cases of extraversion of the bladder. The anterior part of the pelvic girdle is deficient, the superior rami of the pubes are imperfectly developed, and the symphysis is absent. The pubic bones are separated to the extent of from 2 to 4 inches, the superior rami shortened and directed forward, and the obturator foramen diminished in size, narrowed, and turned outward. The iliac bones are straightened out more than normal. The sacrum is very peculiar. The lateral curve, instead of being concave, is flattened out or even convex, with the iliosacral facets turned more outward than normal, while the vertical curve is straightened.
The femur, Part 1. The femur, the longest and strongest bone in the skeleton, is almost perfectly cylindrical in the greater part of its extent. In the erect posture it is not vertical, being separated above from its fellow by a considerable interval, which corresponds to the breadth of the pelvis, but inclining gradually downward and medialward, so as to approach its fellow toward its lower part, for the purpose of bringing the knee joint near the line of gravity of the body. The degree of this inclination varies in different persons, and is greater in the female than in the male, on account of the greater breadth of the pelvis. The femur, like other long bones, is divisible into a body and 2 extremities. The upper extremity, or proximal extremity. The upper extremity presents for examination a head, a neck, a greater and lesser trochanter. The head, or caput femoris. The head, which is globular and forms rather more than a hemisphere, is directed upward, medialward, and a little forward, the greater part of its convexity being above and in front. Its surface is smooth, coated with cartilage in the fresh state, except over an ovoid depression, the fovea capitis femoris, which is situated a little below and behind the center of the head, and gives attachment to the ligamentum teres. The neck, or column femoris. The neck is a flattened pyramidal process of bone, connecting the head with the body, and forming with the latter a wide angle opening medialward. The angle is widest in infancy and becomes lessened during growth, so that at puberty it forms a gentle curve from the axis of the body of the bone. In the adult, the neck forms an angle of about 125 degrees with the body, but this varies in inverse proportion to the development of the pelvis and its stature. In the female, in consequence of the increased width of the pelvis, the neck of the femur forms more nearly a right angle with the body than it does in the male. The angle decreases during the period of growth, but after full growth has been attained it does not usually undergo any change. Even in old age, it varies considerably in different persons of the same age. It is smaller in short than in long bones, and when the pelvis is wide. In addition to projecting upward and medialward from the body of the femur, the neck also projects somewhat forward. The amount of this forward projection is extremely variable, but on an average is from 12 degrees to 14 degrees. The neck is flattened from before backward, contracted in the middle, and broader laterally than medially. The vertical diameter of the lateral half is increased by the obliquity of the lower edge, which slopes downward to join the body at the level of the lesser trochanter, so that it measures 1/3 more than the antero-posterior diameter. The medial half is smaller and of a more circular shape. The anterior surface of the neck is perforated by numerous vascular foramina. Along the upper part of the line of junction of the anterior surface with the head is a shallow groove, best marked in elderly subjects. This groove lodges the orbicular fibers of the capsule of the hip joint. The posterior surface is smooth and is broader and more concave than the anterior. The posterior part of the capsule of the hip joint is attached to it about 1 centimeter above the intertrochanteric crest. The superior border is short and thick and ends laterally at the greater trochanter. Its surface is perforated by large foramina. The inferior border, long and narrow, curves a little backward to end at the lesser trochanter. The trochanters. The trochanters are prominent processes which afford leverage to the muscles that rotate the thigh on its axis. They are 2 in number, the greater and the lesser. The greater trochanter, or trochanter major, great trochanter, is a large, irregular, quadrilateral eminence situated at the junction of the neck with the upper part of the body. It is directed a little lateralward and backward, and, in the adult, is about 1 centimeter lower than the head. It has 2 surfaces and 4 borders. The lateral surface, quadrilateral in form, is broad, rough, convex, and marked by a diagonal impression, which extends from the posterior superior to the anterior inferior angle, and serves for the insertion of the tendon of the gluteus medius. Above the impression is a triangular surface, sometimes rough for part of the tendon of the same muscle, sometimes smooth through the interposition of a bursa between the tendon and the bone. Below and behind the diagonal impression is a smooth triangular surface, over which the tendon of the gluteus maximus plays, a bursa being interposed. The medial surface, of much less extent than the lateral, presents at its base a deep depression, the trochanteric fossa or digital fossa, for the insertion of the tendon of the obturator externus, and above and in front of this an impression for the insertion of the obturator internus and gemelli. The superior border is free. It is thick and irregular and marked near the center by an impression for the insertion of the piriformis. The inferior border corresponds to the line of junction of the base of the trochanter with the lateral surface of the body. It is marked by a rough, prominent, slightly curved ridge, which gives origin to the upper part of the vastus lateralis. The anterior border is prominent and somewhat irregular. It affords insertion at its lateral part to the gluteus minimus. The posterior border is very prominent and appears as a free, rounded edge, which bounds the back part of the trochanteric fossa. The lesser trochanter, or trochanter minor, small trochanter, is a conical eminence, which varies in size in different subjects. It projects from the lower and back part of the base of the neck. From its apex 3 well-marked borders extend. 2 of these are above, a medial continuous with the lower border of the neck, a lateral with the intertrochanteric crest. The inferior border is continuous with the middle division of the linea aspera. The summit of the trochanter is rough, and gives insertion to the tendon of the psoas major. A prominence of variable size occurs at the junction of the upper part of the neck with the greater trochanter, and is called the tubercle of the femur. It is the point of meeting of 5 muscles, the gluteus minimus laterally, the vastus lateralis below, and the tendon of the obturator internus 2 gemelli above. Running obliquely downward and medialward from the tubercle is the intertrochanteric line, or spiral line of the femur. It winds around the medial side of the body of the bone, below the lesser trochanter, and ends about 5 centimeters below this eminence in the linea aspera. Its upper half is rough, and affords attachment to the iliofemoral ligament of the hip joint. Its lower half is less prominent, and gives origin to the upper part of the vastus medialis. Running obliquely downward and medialward from the summit of the greater trochanter on the posterior surface of the neck is a prominent ridge, the intertrochanteric crest. Its upper half forms the posterior border of the great trochanter, and its lower half runs downward and medialward to the lesser trochanter. A slight ridge is sometimes seen commencing about the middle of the intertrochanteric crest and reaching vertically downward for about 5 cm along the back part of the body. It is called a linea quadrata and gives attachment to the quadratus femoris and a few fibers of the adductor magnus. Generally, there is merely a slight thickening about the middle of the intertrochanteric crest, marking the attachment of the upper part of the quadratus femoris. The body or shaft, or corpus femoris. The body, almost cylindrical in form, is a little broader above than in the center, broadest and somewhat flattened from before backward below. It is slightly arched so as to be convex in front and concave behind, where it is strengthened by a prominent longitudinal ridge, the linea aspera. It presents for examination 3 borders, separating 3 surfaces. Of the borders, one, the linea aspera, is posterior, one is medial, and the other lateral. The linea aspera is a prominent longitudinal ridge or crest on the middle third of the bone, presenting a medial and a lateral lip, and a narrow, rough, intermediate line. Above, the linea aspera is prolonged by 3 ridges. The lateral ridge is very rough, and runs almost vertically upward to the base of the greater trochanter. It is termed the gluteal tuberosity, and gives attachment to part of the gluteus maximus. Its upper part is often elongated into a roughened crest, on which a more or less well-marked, rounded tubercle, the third trochanter, is occasionally developed. The intermediate ridge or pectineal line is continued to the base of the lesser trochanter and gives attachment to the pectineus. The medial ridge is lost in the intertrochanteric line. Between these two, a portion of the iliacus is inserted. Below, the linea aspera is prolonged into 2 ridges, enclosing between them a triangular area, the popliteal surface, upon which the popliteal artery rests. Of these 2 ridges, the lateral is the more prominent, and descends to the summit of the lateral condyle. The medial is less marked, especially at its upper part, where it is crossed by the femoral artery. It ends below at the summit of the medial condyle in a small tubercle, the adductor tubercle, which affords insertion to the tendon of the adductor magnus. From the medial lip of the linea aspera and its prolongations above and below, the vastus medialis arises, and from the lateral lip and its upward prolongation, the vastus lateralis takes origin. The adductor magnus is inserted into the linea aspera, and to its lateral prolongation above and its medial prolongation below. Between the vastus lateralis and the adductor magnus 2 muscles are attached, via the gluteus maximus inserted above, and the short head of the biceps femoris arising below. Between the adductor magnus and the vastus medialis 4 muscles are inserted, the iliacus and the pectineus above, the adductor brevis and the adductor longus below. The linea aspera is perforated a little below its center by the nutrient canal, which is directed obliquely upward. The other 2 borders of the femur are only slightly marked. The lateral border extends from the anterior inferior angle of the greater trochanter to the anterior extremity of the lateral condyle. The medial border from the intertrochanteric line, at a point opposite the lesser trochanter, to the anterior extremity of the medial condyle. The anterior surface includes that portion of the shaft which is situated between the lateral and medial borders. It is smooth, convex, broader above and below than in the center. From the upper 3/4 of the surface the vastus intermedius arises. The lower fourth is separated from the muscle by the intervention of the synovial membrane of the knee joint and a bursa. From this upper part of it the articularis genu takes origin. The lateral surface includes the portion between the lateral border and the linea aspera. It is continuous above with the corresponding surface of the greater trochanter, below with that of the lateral condyle. From its upper 3/4 the vastus intermedius takes origin. The medial surface includes the portion between the medial border and the linea aspera. It is continuous above with the lower border of the neck, below with the medial side of the medial condyle. It is covered by the vastus medialis. The lower extremity, or distal extremity. The lower extremity, larger than the upper, is somewhat cuboid in form, but its transverse diameter is greater than antero-posterior. It consists of two oblong eminences known as the condyles. In front, the condyles are but slightly prominent, and are separated from one another by a smooth, shallow articular depression called the patellar surface. Behind, they project considerably, and the interval between them forms a deep notch, the intercondyloid fossa. The lateral condyle is the more prominent and is the broader both in the antero-posterior and transverse diameters. The medial condyle is the longer and, when the femur is held with its body perpendicular, projects to a lower level. When, however, the femur is in its natural oblique position, the lower surfaces of the two condyles lie practically in the same horizontal plane. The condyles are not quite parallel with one another. The long axis of the lateral is almost directly antero-posterior, but that of the medial runs backward and medialward. Their opposed surfaces are small, rough, and concave, and form the walls of the intercondyloid fossa. This fossa is limited above by a ridge, the intercondyloid line, and below by the central part of the posterior margin of the patellar surface. The posterior cruciate ligament of the knee joint is attached to the lower and front part of the medial wall of the fossa, and the anterior cruciate ligament to an impression on the upper and back part of its lateral wall. Each condyle is surmounted by an elevation, the epicondyle. The medial epicondyle is a large convex eminence to which the tibial collateral ligament of the knee joint is attached. At its upper part is the adductor tubercle, often referred to, and behind it is a rough impression which gives origin to the medial head of the gastrocnemius. The lateral epicondyle, smaller and less prominent than the medial, gives attachment to the fibular collateral ligament of the knee joint. Directly below it is a small depression from which a smooth, well-marked groove curves obliquely upward and backward to the posterior extremity of the condyle. This groove is separated from the articular surface of the condyle by a prominent lip across which a second, shallower groove runs vertically downward from the depression. In the fresh state, these grooves are covered with cartilage. Popliteus arises from this depression. Its tendons lie in the oblique groove when the knee is flexed, and in the vertical groove when the knee is extended. Above and behind the lateral epicondyle is an area for the origin of the lateral head of the gastrocnemius, above and to the medial side of which the plantaris arises. The articular surface of the lower end of the femur occupies the anterior, inferior, and posterior surfaces of the condyle. Its front part is named the patellar surface and articulates with the patella. It presents a median groove which extends downward to the intercondyloid fossa and two convexities, the lateral of which is broader, more prominent, and extends farther upward than the medial. The lower and posterior parts of the articular surface constitute the tibial surface for articulation with the corresponding condyles of the tibia and menisci. These surfaces are separated from one another by the intercondyloid fossa and from the patellar surface by faint grooves which extend obliquely across the condyles. The lateral groove is the better marked. It runs lateralward and forward from the front part of the intercondyloid fossa and expands to form a triangular depression. When the knee joint is fully extended, the triangular depression rests upon the anterior portion of the lateral meniscus, and the medial part of the groove comes into contact with the medial margin of the lateral articular surface of the tibia in front of the lateral tubercle of the tibial intercondyloid eminence. The medial groove is less distinct than the lateral. It does not reach as far as the intercondyloid fossa, and therefore exists only on the medial part of the condyle. It receives the anterior edge of the medial meniscus when the knee joint is extended. Where the groove ceases laterally, the patellar surface is seen to be continued backward as a semilunar area close to the anterior part of the intercondyloid fossa. This semilunar area articulates with the medial vertical facet of the patella in forced flexion of the knee joint. The tibial surfaces of the condyles are convex from side to side and from before backward. Each presents a double curve, its posterior segment being an arc of a circle, its anterior part of a cycloid.
Footnote. A cycloid is a curve traced by a point in the circumference of a wheel when the wheel is rolled along in a straight line. End of footnote.
The architecture of the femur.
Note. The following paragraphs are taken almost verbatim from Koch's article, in which we have the first correct mathematical analysis of the femur in support of the theory of the functional form of bone proposed by Wolff and by Roux. End of note.
Koch, by mathematical analysis, has shown that in every part of the femur there is a remarkable adaptation of the inner surface of the bone to the mechanical requirements due to the load of the femur head. The various parts of the femur taken together form a single mechanical structure wonderfully well adapted for the efficient economical transmission of the load of the acetabulum to the tibia, a structure in which every element contributes its modicum of strength in the manner required by theoretical mechanics for maximum efficiency. The internal structure is everywhere so formed as to provide in an efficient manner for all the internal stresses which occur due to the load on the femur head. Throughout the femur with a load on the femur head, the bony material is arranged in the paths of the maximum internal stresses which are thereby resisted with the greatest efficiency, and hence with maximum economy of material. The conclusion is inevitable that the inner structure and outer form of the femur are governed by the conditions of maximum stresses to which the bone is subjected normally by the preponderant load of the femur head, that is, by the body weight transmitted to the femur head through the acetabulum. The femur obeys the mechanical laws that govern other elastic bodies under stress. The relation between the computed internal stresses due to the load of the femur head and the internal structure of the different portions of the femur is in very close agreement with the theoretical relations that should exist between stress and structure for maximum economy and efficiency, and therefore it is believed that the following laws of bone structure have been demonstrated for the femur. 1. The internal structure and external form of human bone are closely adapted to the mechanical conditions existing at every point in the bone. 2. The inner architecture of normal bone is determined by definite and exact requirements of mathematical and mechanical laws to produce maximum of strength with a minimum of material.
The femur, Part 2. The inner architecture of the upper femur. The spongy bone of the upper femur, to the lower limit of the lesser trochanter, is composed of two distinct systems of trabeculi arranged in curved paths. One, which has its origin in the medial inner side of the shaft, and curving upward in a fan-like radiation to the opposite side of the bone. The other, having origin in the lateral outer portion of the shaft, and arching upward and medially to end in the upper surface of the greater trochanter, neck and head. These two systems intersect each other at right angles. A medial compressive system of trabeculi. As the compact bone of the medial inner part of the shaft nears the head of the femur, it gradually becomes thinner and finally reaches the articular surface of the head as a very thin layer. From a point at about the lower level of the lesser trochanter, 2½ to 3 inches from the lower limit of the articular surface of the head, the trabeculi branch off from the shaft in smooth curves, spreading radially to cross to the opposite side in two well-defined groups, a lower or secondary group, and an upper or principal group. A. The secondary compressive group. This group of trabeculi leaves the inner border of the shaft beginning at about the level of the lesser trochanter, and for a distance of almost 2 inches along the curving shaft, with which the separate trabeculi make an angle of about 45 degrees. They curve outwardly and upwardly to cross in radiating smooth curves to the opposite side. The lower filaments end in the region of the greater trochanter. The adjacent filaments above these pursue a more nearly vertical course and end in the upper portion of the neck of the femur. The trabeculi of this group are thin and with wide spaces between them. As they traverse the space between the medial and lateral surfaces of the bone, they cross at right angles the system of curved trabeculi which arise from the lateral outer portion of the shaft. B. The principal compressive group. This group of trabeculi springs from the medial portion of the shaft just above the group above described, and spreads upward and in slightly radial smooth curved lines to reach the upper portion of the articular surface of the head of the femur. These trabeculae are placed very closely together, and are the thickest ones seen in the upper femur. They are a prolongation of the shaft from which they spring in straight lines which gradually curve to meet at right angles the articular surface. There is no change as they cross the epiphyseal line. They also intersect at right angles the system of lines which rise from the lateral side of the femur. This system of principal and secondary compressive trabeculae corresponds in position and in curvature with the lines of maximum compressive stress, which were traced out in the mathematical analysis of this portion of the femur. B. Lateral tensile system of trabeculae. As the compact bone of the outer portion of the shaft approaches the greater trochanter, it gradually decreases in thickness. Beginning at a point about 1 inch below the level of the lower portion of the greater trochanter, numerous thin trabeculae are given off from the outer portion of the shaft. These trabeculae lie in 3 distinct groups. C. The greater trochanter group. These trabeculae rise from the outer part of the shaft just below the greater trochanter and rise in thin curving lines to cross the region of the greater trochanter and end in its upper surface. Some of these filaments are poorly defined. This group intersects the trabeculae of group A, which rise from the opposite side. The trabeculae of this group evidently carry small stresses, as is shown by their slenderness. D. The principal tensile group. This group springs from the outer part of the shaft immediately below group C and curves convexly upward and inward in nearly parallel lines across the neck of the femur and ends in the inferior portion of the head. These trabeculae are somewhat thinner and more widely spaced than those of the principal compressive group, B. All the trabeculae of this group cross those of groups A and B at right angles. This group is the most important of the lateral system, tensile, and, as will be shown later, the greatest tensile stresses of the upper femur are carried by the trabeculae of this group. E. The secondary tensile group. This group consists of the trabeculae which spring from the outer side of the shaft and lie below those of the preceding group. They curve upward and medially across the axis of the femur and end more or less irregularly after crossing the midline, but a number of these filaments end in the medial portion of the shaft and neck. They cross at right angles the trabeculae of group A. In general, the trabeculae of the tensile system are lighter in structure than those of the compressive system in corresponding positions. The significance of the difference in thickness of these two systems is that the thickness of the trabeculae varies with the intensity of the stresses at any given point. Comparison of Figure 247 with Figure 251 will show that the trabeculae of the compressive system carry heavier stresses than those of the tensile system in corresponding positions. For example, the maximum tensile stress at section 8 in the outermost fibre is 771 pounds per square inch, and at the corresponding point on the compressive side the compressive stress is 954 pounds per square inch. Similar comparisons may be made at other points, which confirm the conclusion that the thickness and closeness of spacing of the trabeculae varies in proportion to the intensity of the stresses carried by them. It will be seen that the trabeculae lie exactly in the paths of the maximum tensile and compressive stresses, and hence these trabeculae carry these stresses in the most economical manner. This is in accordance with the well-recognized principle of mechanics that the most direct manner of transmitting stress is in the direction in which the stress acts. Figure 249 shows a longitudinal frontal section through the left femur, which is the mate of the right femur on which the mathematical analysis was made. In this midsection the system of tensile trabeculae, which rises from the lateral, outer part of the shaft and crosses over the central area to end in the medial portion of the shaft, neck and head, is clearly shown. This figure also shows the compressive system of trabeculae which rises on the medial portion of the shaft and crosses the central area to end in the head, neck and greater trochanter. By comparing the position of these two systems of trabeculae shown in Figure 249 with the lines of maximum and minimum stresses shown in Figures 248 and 250, it is seen that the tensile system of trabeculae corresponds exactly with the position of the lines of maximum and minimum tensile stresses, which were determined by mathematical analysis. In a similar manner, the compressive system of trabeculae in Figure 249 corresponds exactly with the lines of maximum and minimum compressive stresses computed by the mathematical analysis. The amount of vertical shear varies almost uniformly from a maximum of 90 pounds, 90% of the load on the femur head, midway between sections 4 and 6, to a minimum of 5.7 pounds at section 18. There is a gradual diminution of the spongy bone from section 6 to section 18, parallel with the diminished intensities of the vertical shear. The trabeculae of the upper femur, as shown in frontal sections, are arranged in two general systems, compressive and tensile, which correspond in position with the lines of maximum and minimum stresses in the femur determined by the mathematical analysis of the femur as a mechanical structure. 2. The thickness and spacing of the trabeculae vary with the intensity of the maximum stresses at various points in the upper femur, being thickest and most closely spaced in the regions where the greatest stresses occur. 3. The amount of bony material in the spongy bone of the upper femur varies in proportion to the intensity of the shearing force at the various sections. 4. The arrangement of the trabeculae in the positions of maximum stresses is such that the greatest strength is secured with a minimum of material. Significance of the inner architecture of the shaft. 1. Economy for resisting shear. The shearing stresses are at a minimum in the shaft. It is clear that a minimum amount of material will be required to resist the shearing stresses. As horizontal and vertical shearing stresses are most efficiently resisted by material placed near the neutral plane, in this region a minimum amount of material will be needed near the neutral axis. In the shaft there is very little if any material in the central space, practically the only material near the neutral plane being in the compact bone, but lying at a distance from the neutral axis. This confirms the requirement of mechanics for economy, as a minimum of material is provided for resisting shearing stresses where these stresses are a minimum. 2. Economy for resisting bending moment. The bending moment increases from a minimum at section 4 to a maximum between sections 16 and 18, then gradually decreases almost uniformly to 0 near section 75. To resist bending moment stresses most effectively, the material should be as far from the neutral axis as possible. It is evident that the hollow shaft of the femur is an efficient structure for resisting bending moment stresses, all of the material in the shaft being relatively at a considerable distance from the neutral axis. It is evident that the hollow shaft provides efficiently for resisting bending moment, not only due to the load on the femur head, but from any other loads tending to produce bending in other planes. 3. Economy for resisting axial stress. The inner architecture of the shaft is adapted to resist in the most efficient manner the combined action of the minimal shearing forces and the axial and maximum bending stresses. The structure of the shaft is such as to secure great strength with a relatively small amount of material. The distal portion of the femur. In frontal section, in the distal 6 inches of the femur, there are to be seen two main systems of trabeculae, a longitudinal and a transverse system. The trabeculae of the former rise from the inner wall of the shaft and continue in perfectly straight lines parallel to the axis of the shaft and proceed to the epiphyseal line, whence they continue in more or less curved lines to meet the articular surface of the knee joint at right angles at every point. Near the centre there are a few thin, delicate, longitudinal trabeculae, which spring from the longitudinal trabeculae just described, to which they are joined by fine transverse filaments that lie in planes parallel to the sagittal plane. The trabeculae of the transverse system are somewhat lighter in structure than those of the longitudinal system, and consist of numerous trabeculae at right angles to the latter. As the distal end of the femur is approached, the shaft gradually becomes thinner until the articular surface is reached, where there remains only a thin shell of compact bone. With the gradual thinning of the compact bone of the shaft, there is a simultaneous increase in the amount of the spongy bone, and a gradual flaring of the femur, which gives this portion of the bone a gradually increasing gross area of cross-section. There is a marked thickening of the shell of bone in the region of the intercondyloid fossa, where the anterior and posterior crucial ligaments are attached. The thickened area is about 0.4 inch in diameter and consists of compact bone from which a number of thick trabeculae pass at right angles to the main longitudinal system. The inner structure of the bone is here evidently adapted to the efficient distribution of the stresses arising from this ligamentary attachment. Near the distal end of the femur, the longitudinal trabeculae gradually assume curved paths and end perpendicularly to the articular surface at every point. Such a structure is in accordance with the principles of mechanics, as stresses can be communicated through a frictionless joint only in a direction perpendicular to the joint surface at every point. With practically no increase in the amount of bony material used, there is a greatly increased stability produced by the expansion of the lower femur from a hollow shaft of compact bone to a structure of much larger cross-section almost entirely composed of spongy bone. Significance of the inner architecture of the distal part of the femur. The function of the lower end of the femur is to transmit through a hinged joint the loads carried by the femur. For stability, the width of the bearing on which the hinge action occurs should be relatively large. For economy of material, the expansion of the end bearing should be as lightly constructed as is consistent with proper strength. In accordance with the principles of mechanics, the most efficient manner in which stresses are transmitted is by the arrangement of the resisting material in lines parallel to the direction in which the stresses occur and in the paths taken by the stresses. Theoretically, the most efficient manner to attain these objects would be to prolong the innermost filaments of the bone as straight lines parallel to the longitudinal axis of the bone, and gradually to flare the outer shell of compact bone outward, and continuing to give off filaments of bone parallel to the longitudinal axis as the distal end of the femur is approached. These filaments should be well braced transversely, and each should carry its proportionate part of the total load parallel to the longitudinal axis, transmitting it eventually to the articular surface and in a direction perpendicular to that surface. Referring to Figure 249, it is seen that the large expansion of the bone is produced by the gradual transition of the hollow shaft of compact bone to cancellated bone, resulting in the production of a much larger volume. The trabeculi are given off from the shaft in lines parallel to the longitudinal axis, and are braced transversely by two series of trabeculi at right angles to each other, in the same manner as required theoretically for economy. Although the action of the muscles exerts an appreciable effect on the stresses in the femur, it is relatively small and very complex to analyze, and has not been considered in the above analysis. Ossification. The femur is ossified from 5 centres, one for the body, one for the head, one for each trochanter, and one for the lower extremity. Of all the long bones except the clavicle, it is the first to show traces of ossification. This commences in the middle of the body at about the 7th week of fetal life, and rapidly extends upward and downward. The centres in the epiphyses appear in the following order. In the lower end of the bone, at the 9th month of fetal life, from this centre the condyles and epicondyles are formed, in the head. At the end of the 1st year after birth, in the greater trochanter, during the 4th year, and in the lesser trochanter between the 13th and 14th years. The order in which the epiphyses are joined to the body is the reverse of that of their appearance. They are not united until after puberty, the lesser trochanter being first joined, then the greater, then the head, and lastly the inferior extremity, which is not united until the 20th year.
The patella. The patella is a flat, triangular bone situated on the front of the knee joint. It is usually regarded as a sesamoid bone, developed in the tendon of the quadriceps femoris, and resembles these bones 1. In being developed in a tendon, 2. In its center of ossification presenting a knotty or tuberculated outline, 3. In being composed mainly of dense cancellous tissue. It serves to protect the front of the joint, and increases the leverage of the quadriceps femoris by making it act at a greater angle. It has an anterior and a posterior surface, 3 borders, and an apex. Surfaces. The anterior surface is convex, perforated by small apertures for a passage of nutrient vessels, and marked by numerous rough longitudinal striae. This surface is covered, in the recent state, by an expansion from the tendon of the quadriceps femoris, which is continuous below with the superficial fibers of the ligamentum patella. It is separated from the integument by a bursa. The posterior surface presents above a smooth, oval, articular area divided into 2 facets by a vertical ridge. The ridge corresponds to the groove on the patellar surface of the femur, and the facets to the medial and lateral parts of the same surface. The lateral facet is the broader and deeper. Below the articular surface is a rough, convex, non-articular area, the lower half of which gives attachment to the ligamentum patellae. The upper half is separated from the head of the tibia by adipose tissue. Borders. The base or superior border is thick and sloped from behind, downward and forward. It gives attachment to that portion of the quadriceps femoris which is derived from the rectus femoris and vastus intermedius. The medial and lateral borders are thinner and converge below. They give attachment to those portions of the quadriceps femoris which are derived from the vasti lateralis and medialis. Apex. The apex is pointed and gives attachment to the ligamentum patella. Structure. The patella consists of nearly uniform, dense cancellous tissue covered by a thin, compact lamina. The cancellii immediately beneath the anterior surface are arranged parallel with it. In the rest of the bone, they radiate from the articular surface toward the other parts of the bone. Ossification. The patella is ossified from a single center, which usually makes its appearance in the 2nd or 3rd year, but may be delayed until the 6th year. More rarely, the bone is developed by 2 centers placed side by side. Ossification is completed about the age of puberty. Articulation. The patella articulates with the femur. The tibia. The tibia is situated at the medial side of the leg, and excepting the femur, is the longest bone of the skeleton. It is prismoid in form, expanded above, where it enters into the knee joint, contracted in the lower third, and again enlarged, but to a lesser extent below. In the male, its direction is vertical, and parallel with the bone of the opposite side, but in the female, it has a slightly oblique direction downward and lateralward, to compensate for the greater obliquity of the femur. It has a body and 2 extremities. The upper extremity is large and expanded into 2 eminences, the medial and lateral condyles. The superior articular surface presents 2 smooth articular facets. The medial facet, oval in shape, is slightly concave from side to side and from before backward. The lateral, nearly circular, is concave from side to side, but slightly convex from before backward, especially at its posterior part, where it is prolonged onto the posterior surface for a short distance. The central portions of these facets articulate with the condyles of the femur, while their peripheral portions support the menisci of the knee joint, which here intervene between the 2 bones. Between the articular facets, but nearer the posterior than the anterior aspect of the bone, is the intercondyloid eminence, or spine of tibia, surmounted on either side by a prominent tubercle, onto the sides of which the articular facets are prolonged. In front of and behind the intercondylar eminence are rough depressions for the attachment of the anterior and posterior cruciate ligaments and the menisci. The anterior surfaces of the condyle are continuous with one another, forming a large, somewhat flattened area. This area is triangular, broad above, and perforated by large vascular foramina, narrow below where it ends in a large oblong elevation, the tuberosity of the tibia, which gives attachment to the ligamentous patella. A bursa intervenes between the deep surface of the ligament and the part of the bone immediately above the tuberosity. Posteriorly, the condyles are separated from each other by a shallow depression, the posterior intercondylar fossa, which gives attachment to part of the posterior cruciate ligament of the knee joint. The medial condyle presents posteriorly a deep transverse groove for the insertion of the tendon of the semimembranosus. Its medial surface is convex, rough, and prominent. It gives attachment to the tibial collateral ligament. The lateral condyle presents posteriorly a flat articular facet, nearly circular in form, directed downward, backward, and lateralward for articulation with the head of the fibula. Its lateral surface is convex, rough, and prominent in front. On it is an eminence situated on a level with the upper border of the tuberosity, and at the junction of its anterior and lateral surfaces, for the attachment of the iliotibial band. Just below this is a part of the extensor digitorum longus takes off, and a slip from the tendon of the biceps femoris is inserted. The body or shaft. The body has 3 borders and 3 surfaces. Borders. The anterior crest or border, the most prominent of the 3, commences above at the tuberosity and ends below at the anterior margin of the medial malleolus. It's sinuous and prominent in the upper 2/3 of its extent, but smooth and rounded below. It gives attachment to the deep fascia of the leg. The medial border is smooth and rounded above and below, but more prominent in the center. It begins at the back part of the medial condyle and ends at the posterior border of the medial malleolus. Its upper part gives attachment to the tibial collateral ligament of the knee joint to the extent of about 5 centimeters, and insertion to some fibers of the popliteus. From the middle third of some fibers of the soleus and flexor digitorum longus take origin. The interosseous crest, or lateral border, is thin and prominent, especially its central part, and gives attachment to the interosseous membrane. It commences above in front of the fibular articular facet and bifurcates below to form the boundaries of a triangular rough surface for the attachment of the interosseous ligament connecting the tibia and fibula. Surfaces. The medial surface is smooth, convex, and broader above than below. Its upper third, directed forward and medialward, is covered by the aponeurosis derived from the tendon of the sartorius, and by the tendons of the gracilis and semitendinosus, all of which are inserted nearly as far forward as the anterior crest. In the rest of its extent it is subcutaneous. The lateral surface is narrower than the medial. Its upper 2/3 present a shallow groove for the origin of the tibialis anterior. Its lower third is smooth, convex, curves gradually forward to the anterior aspect of the bone, and is covered by the tendons of the tibialis anterior, extensor hallucis longus, and the extensor digitorum longus, arranged in this order from the medial side. The posterior surface presents, at its upper part, a prominent ridge, the popliteal line, which extends obliquely downward from the back part of the articular facet for the fibula to the medial border. At the junction of its upper and middle thirds, it marks the lower limit of the insertion of the popliteus, serves for the attachment of the fascia covering this muscle, and gives origin to part of the soleus, flexor digitorum longus, and tibialis posterior. The triangular area above this line gives insertion to the popliteus. The middle third of the posterior surface is divided by a vertical ridge into 2 parts. The ridge begins at the popliteal line and is well marked above, but indistinct below. The medial and broader portion gives origin to the flexor digitorum longus, the lateral and narrower to part of the tibialis posterior. The remaining part of the posterior surface is smooth and covered by the tibialis posterior, flexor digitorum longus, and the flexor hallucis longus. Immediately below the popliteal line is the nutrient foramen, which is large and directed obliquely downward. The lower extremity. The lower extremity is much smaller than the upper, presents 5 surfaces. It is prolonged downward on the medial side as a strong process, the medial malleolus. Surfaces. The inferior articular surface is quadrilateral and smooth for articulation with the talus. It is concave from before backward, broader in front than behind, and traversed from before backward by a slight elevation separating 2 depressions. It is continuous with that on the medial malleolus. The anterior surface of the lower extremity is smooth and rounded above, and covered by the tendons of the extensor muscles. Its lower margin presents a rough transverse depression for the attachment of the articular capsule of the ankle joint. The posterior surface is traversed by a shallow groove directed obliquely downward and medialward, continuous with a similar groove on the posterior surface of the talus, and serving for the passage of the tendon of the flexor hallucis longus. The lateral surface presents a triangular rough depression for the attachment of the inferior interosseous ligament connecting it with the fibula. The lower part of this depression is smooth, covered with cartilage in the fresh state, and articulates with the fibula. The surface is bounded by two prominent borders, continuous above with the interosseous crest. They afford attachment to the anterior and posterior ligaments of the lateral malleolus. The medial surface is prolonged downward to form a strong pyramidal process, flattened from without inward, the medial malleolus. The medial surface of this process is convex and subcutaneous. Its lateral or articular surface is smooth and slightly concave, and articulates with the talus. Its anterior border is rough for the attachment of the anterior fibers of the deltoid ligament of the ankle joint. Its posterior border presents a broad groove, the malleolar sulcus, directed obliquely downward and medialward, and occasionally double. This sulcus lodges the tendons of the tibialis posterior and flexor digitorum longus. The summit of the medial malleolus is marked by a rough depression behind for the attachment of the deltoid ligament. Structure. The structure of the tibia is like that of the other long bones. The compact wall of the body is thickest at the junction of the middle and lower thirds of the bone. Ossification. The tibia is ossified from three centers, one for the body and one for either extremity. Ossification begins in the center of the body about the 7th week of fetal life and gradually extends towards the extremities. The center for the upper epiphysis appears before or shortly after birth. It is flattened in form and has a thin tongue-shaped process in front, which forms the tuberosity. That for the lower epiphysis appears in the 2nd year. The lower epiphysis joins the body at about the 18th, and the upper one joins about the 20th year. Two additional centers occasionally exist, one for the tongue-shaped process of the upper epiphysis, which forms the tuberosity, and one for the medial malleolus.
The fibula, calf bone. The fibula is placed on the lateral side of the tibia, with which it is connected above and below. It is the smaller of the two bones, and in proportion to its length, the most slender of all the long bones. Its upper extremity is small, placed toward the back of the head of the tibia, below the level of the knee joint, and excluded from the formation of this joint. Its lower extremity inclines a little forward, so as to be on a plane anterior to that of the upper end. It projects below the tibia and forms the lateral part of the ankle joint. The bone has a body and two extremities. The upper extremity or head, capitulum fibulae, proximal extremity. The upper extremity is of an irregular quadrate form, presenting above a flattened articular surface directed upward, forward, and medialward for articulation with a corresponding surface on the lateral condyle of the tibia. On the lateral side is the thick and rough prominence continued behind into a pointed eminence, the apex styloid process, which projects upward from the posterior part of the head. The prominence at its upper and lateral part gives attachment to the tendon of the biceps femoris and to the fibular collateral ligament of the knee joint, the ligament dividing the tendon into two parts. The remaining part of the circumference of the head is rough for the attachment of muscles and ligaments. It presents in front a tubercle for the origin of the upper and anterior fibers of the peroneus longus, and a surface for the attachment of the anterior ligament of the head, and behind another tubercle for the attachment of the posterior ligament of the head and the origin of the upper fibers of the soleus. The body or shaft, corpus fibulae. The body presents four borders: the anterolateral, the anteromedial, the posterolateral, and the posteromedial, and four surfaces: anterior, posterior, medial, and lateral. Borders. The anterolateral border begins above in front of the head, runs vertically downward to a little below the middle of the bone, and then curving somewhat lateralward, bifurcates so as to embrace a triangular subcutaneous surface immediately above the lateral malleolus. This border gives attachment to an intermuscular septum which separates the extensor muscles on the anterior surface of the leg from the peronei longus and brevis on the lateral surface. The anteromedial border, or interosseous crest, is situated close to the medial side of the preceding, and runs nearly parallel with it in the upper third of its extent, but diverges from it in the lower two-thirds. It begins above, just beneath the head of the bone. Sometimes it is quite indistinct for about 2.5 centimeters below the head, and ends at the apex of a rough triangular surface immediately above the articular facet of the lateral malleolus. It serves for the attachment of the interosseous membrane which separates the extensor muscles in front from the flexor muscles behind. The posterolateral border is prominent. It begins above at the apex and ends below in the posterior border of the lateral malleolus. It is directed lateralward above, backward in the middle of its course, backward and a little medialward below, and gives attachment to an aponeurosis, which separates the peronei on the lateral surface from the flexor muscles on the posterior surface. The posteromedial border, sometimes called the oblique line, begins above at the medial side of the head, and ends by becoming continuous with the interosseous crest at the lower fourth of the bone. It is well marked and prominent at the upper and middle parts of the bone. It gives attachment to an aponeurosis, which separates the tibialis posterior from the soleus and flexor hallucis longus. Surfaces. The anterior surface is the interval between the anterolateral and anteromedial borders. It is extremely narrow and flat in the upper third of its extent. Broader and grooved longitudinally in its lower third, it serves for the origin of three muscles: the extensor digitorum longus, extensor hallucis longus, and peroneus tertius. The posterior surface is the space included below the posterolateral and the posteromedial borders. It is continuous below with the triangular area above the articular surface of the lateral malleolus. It is directed backward above, backward and medialward at its middle, directly medialward below. Its upper third is rough for the origin of the soleus. Its lower part presents a triangular surface, connected to the tibia by a strong interosseous ligament. The intervening part of the surface is covered by the fibers of origin of the flexor hallucis longus. Near the middle of this surface is the nutrient foramen, which is directed downward. The medial surface is the interval included between the anteromedial and the posteromedial borders. It is grooved for the origin of the tibialis posterior. The lateral surface is the space between the anterolateral and posterolateral borders. It is broad and often deeply grooved. It is directed lateralward in the upper two-thirds of its course, backward in the lower third, where it is continuous with the posterior border of the lateral malleolus. This surface gives origin to the peronei longus and brevis. The lower extremity or lateral malleolus, malleolus lateralis, distal extremity, external malleolus. The lower extremity is of a pyramidal form and somewhat flattened from side to side. It descends to a lower level than the medial malleolus. The lateral surface is convex, subcutaneous, and continuous with the triangular, subcutaneous surface on the lateral side of the body. The medial surface presents in front a smooth triangular surface, convex from above downward, which articulates with a corresponding surface on the lateral side of the talus. Behind and beneath the articular surface is a rough depression which gives attachment to the posterior talofibular ligament. The anterior border is thick and rough and marked below by a depression for the attachment of the anterior talofibular ligament. The posterior border is broad and presents the shallow malleolar sulcus for the passage of the tendons of the perioni longus and brevis. The summit is rounded and give attachment to the calcaneofibular ligament. Ossification. The fibula is ossified from 3 centers, one for the body and one for either end. Ossification begins in the body about the 8th week of fetal life and extends toward the extremities. At birth, the ends are cartilaginous. Ossification commences in the lower end in the 2nd year and in the upper about the 4th year. The lower epiphysis, the first to ossify, unites with the body about the 20th year. The upper epiphysis joins about the 25th year.
Tarsus. The skeleton of the foot consists of 3 parts: the tarsus, the metatarsus, and phalanges. The tarsus. Ossa tarsi. The tarsal bones are 7 in number, namely the calcaneus, talsus, cuboid, navicular, and the 1st, 2nd, and 3rd cuneiforms. The calcaneus, os calcis. The calcaneus is the largest of the tarsal bones. It is situated at the lower and back part of the foot, serving to transmit the weight of the body to the ground, and forming a strong lever for the muscles of the calf. It is irregularly cuboidal in form, having its long axis directed forward and lateralward. It presents, for examination, 6 surfaces. Surfaces. The superior surface extends behind onto that part of the bone which projects backward to form the heel. This varies in length in different individuals, is convex from side to side, concave from before backward, and supports a mass of fat placed in front of the tendo calcaneus. In front of this area is a large, usually somewhat oval-shaped facet, the posterior articular surface, which looks upward and forward. It is convex from behind forward, and articulates with the posterior calcaneal facet on the under surface of the talus. It is bounded anteriorly by a deep depression, which is continued backward and medialward in the form of a groove, calcaneal sulcus. In the articulated foot, this sulcus lies below a similar one on the under surface of the talus, and the two form a canal, sinus tarsi, for the lodgment of the interosseous talocalcaneal ligament. In front and to the medial side of this groove is an elongated facet, concave from behind forward, and with its long axis directed forward and lateralward. This facet is frequently divided into 2 by a notch. Of the 2, the posterior and larger is termed the middle articular surface. It is supported on a projecting process of bone, the sustentaculum tali, and articulates in the middle calcaneal facet on the under surface of the talus. The anterior articular surface is placed on the anterior part of the body and articulates with the anterior calcaneal facet on the talus. The upper surface, anterior and lateral to the facets, is rough for the attachment of ligaments and for the origin of the extensor digitorum brevis. The inferior or plantar surface is uneven, wider behind than in front, and convex from side to side. It is bounded posteriorly by a transverse elevation, the calcaneal tuberosity, which is depressed in the middle and prolonged at either end into a process, the lateral process, small, prominent, and rounded, gives origin to part of the abductor digiti quinti, the medial process, broader and larger, gives attachment, by its prominent medial margin, to the abductor hallucis, and in front of the flexor digitorum brevis and the plantar aponeurosis. The depression behind the processes gives origin to the abductor digiti quinti. The rough surface in front of the processes gives attachment to the long plantar ligament, and to the lateral head of the quadratus plantae, to a prominent tubercle, nearer the anterior part of the surface, as well as to a transverse groove in front of the tubercle, is attached the plantar calcaneocuboid ligament. The lateral surface is broad behind and narrow in front, flat and almost subcutaneous. Near its center is a tubercle for the attachment of the calcaneofibular ligament. At its upper and anterior part, this surface gives attachment to the lateral talocalcaneal ligament, and in front of the tubercle it presents a narrow surface marked by two oblique grooves. The grooves are separated by an elevated ridge or tubercle, the trochlear process, peroneal tubercle, which varies much in size in different bones. The superior groove transmits the tendon of the peroneus brevis, the inferior groove that of the peroneus longus. The medial surface is deeply concave. It is directed obliquely downward and forward, and serves for the transmission of the plantar vessels and nerves into the sole of the foot. It affords origin to part of the quadratus plantae. At its upper and forepart is a horizontal eminence, the sustentaculum tali, which gives attachment to a slip of the tendon of the tibialis posterior. This eminence is concave above and articulates with the middle calcaneal-articular surface of the talus. Below it is grooved with the tendon of the flexor hallucis longus. Its anterior margin gives attachment to the plantar calcaneonavicular ligament, and its medial to a part of the deltoid ligament of the ankle joint. The anterior or cuboid articular surface is of a somewhat triangular form. It is concave from above downward and lateralward, and convex in a direction at right angles to this. Its medial border gives attachment to the plantar calcaneonavicular ligament. The posterior surface is prominent, convex, wider below than above, and divisible into three areas. The lowest of these is rough and covered by the fatty and fibrous tissue of the heel. The middle, also rough, gives insertion to the tendo calcaneus and the plantaris, while the highest is smooth and is covered by a bursa, which intervenes between it and the tendo calcaneus. Articulations. The calcaneus articulates with two bones, the talus and cuboid. The talus, astragalus, ankle bone. The talus is the second largest of the tarsal bones. It occupies the middle and upper part of the tarsus, supporting the tibia above, resting upon the calcaneus below, articulating on either side with the malleoli, and in front with the navicular. It consists of a body, a neck, and a head. The body, corpus tali. The superior surface of the body presents, behind, a smooth trochlear surface, the trochlea, for articulation with the tibia. The trochlea is broader in front than behind, convex from before backward, slightly concave from side to side. In front it is continuous with the upper surface of the neck of the bone. The inferior surface presents two articular areas, the posterior and middle calcaneal surfaces, separated from one another by a deep groove, the sulcus tali. The groove runs obliquely forward and lateralward, becoming gradually broader and deeper in front. In the articulated foot it lies above a similar groove upon the upper surface of the calcaneus, and forms with it a canal, sinus tarsi, filled up in the fresh state by the interosseous talocalcaneal ligament. The posterior calcaneal articular surface is large and of an oval or oblong form. It articulates with a corresponding facet on the upper surface of the calcaneus. Footnote. Sewell, Journal of Anatomy and Physiology, Volume 38, pointed out that in about 10% of bones a small triangular facet, continuous with the posterior calcaneal facet is present at the junction of the lateral surface of the body with the posterior wall of the sulcus tali.] and is deeply concave in the direction of its long axis, which runs forward and lateralward at an angle of about 45 degrees with the median plane of the body. The middle calcaneal articular surface is small, oval in form and slightly convex. It articulates with the upper surface of the substenticulum tali of the calcaneus. The medial surface presents at its upper part a pear-shaped articular facet for the medial malleolus, continuous above with the trochlea. Below the articular surface is a rough depression for the attachment of the deep portion of the deltoid ligament of the ankle joint. The lateral surface carries a large triangular facet, concave from above downward, for articulation with the lateral malleolus. Its anterior half is continuous above with the trochlea, and in front of it is a rough depression for the attachment of the anterior talofibular ligament. Between the posterior half of the lateral border of the trochlea and the posterior part of the base of the fibular articular surface is a triangular facet, which comes into contact with the transverse inferior tibiofibular ligament during flexion of the ankle joint. Below the base of this facet is a groove which affords attachment to the posterior talofibular ligament. The posterior surface is narrow and transversed by a groove running obliquely downward and medialward and transmitting the tendon of the flexor hallucis longus. Lateral to the groove is a prominent tubercle, the posterior process, to which the posterior talofibular ligament is attached. This process is sometimes separated from the rest of the talus, and is then known as the os trigonum. Medial to the groove is a second smaller tubercle. The neck, column tali. The neck is directed forward and medialward, and comprises the constricted portion of the bone between the body and the oval head. Its upper and medial surfaces are rough for the attachments of ligaments. Its lateral surface is concave and is continuous below with a deep groove for the interosseous talocalcaneal ligament. The head, caput tali. The head looks forward and medialward. Its anterior articular or navicular surface is large, oval, and convex. Its inferior surface has two facets, which are best seen in the fresh condition. The medial, situated in front of the middle calcaneal facet, is convex, triangular, or semi-oval in shape, and rests on the plantar calcaneo-navicular ligament. The lateral, named the anterior calcaneal articular surface, is somewhat flattened and articulates with the facet on the upper surface of the anterior part of the calcaneus. Articulations. The talus articulates with four bones: tibia, fibula, calcaneus, and navicular. The cuboid bone, os cuboideum. The cuboid bone is placed on the lateral side of the foot, in front of the calcaneus, and behind the fourth and fifth metatarsal bones. It is of a pyramidal shape, its base being directed medialward. Surfaces. The dorsal surface, directed upward and lateralward, is rough for the attachment of ligaments. The plantar surface presents in front a deep groove, the perineal sulcus, which runs obliquely forward and medialward. It lodges the tendon of the perineus longus, and is bounded behind by a prominent ridge, to which the long plantar ligament is attached. The ridge ends laterally in an eminence, the tuberosity, the surface of which presents an oval facet. On this facet glides sesamoid bone or cartilage frequently found in the tendon of the perineus longus. The surface of bone behind the groove is rough. For the attachment of the plantar calcaneocuboid ligament, a few fibers of the flexor hallucis brevis and a fasciculus from the tendon of the tibialis posterior. The lateral surface presents a deep notch formed by the commencement of the perineal sulcus. The posterior surface is smooth, triangular, and concavo-convex. For articulation with the anterior surface of the calcaneus, its infero-medial angle projects backward as a process which underlies and supports the anterior end of the calcaneus. The anterior surface, of smaller size, but also irregularly triangular, is divided by a vertical ridge into two facets. The medial, quadrilateral in form, articulates with the fourth metatarsal. The lateral, larger and more triangular, articulates with the fifth. The medial surface is broad, irregularly quadrilateral, and presents at its middle and upper part a smooth oval facet for articulation with a third cuneiform, and behind this occasionally a smaller facet for articulation with the navicular. It is rough in the rest of its extent for the attachment of strong interosseous ligaments. Articulations. The cuboid articulates with four bones, the calcaneus, third cuneiform, and fourth and fifth metatarsals, occasionally with a fifth, the navicular. The navicular bone. Os naviculare pedis. Scaphoid bone. The navicular bone is situated at the medial side of the tarsus, between the talus, behind, and the cuneiform bones in front. Surfaces. The anterior surface is convex from side to side, and subdivided by two ridges into three facets, for articulation with the three cuneiform bones. The posterior surface is oval, concave, broader laterally than medially, and articulates with the rounded head of the talus. The dorsal surface is convex from side to side, and rough for the attachment of ligaments. The plantar surface is irregular, and also rough for the attachment of ligaments. The medial surface presents a rounded tuberosity, the lower part of which gives attachment to part of the tendon of the tibialis posterior. The lateral surface is rough and irregular for the attachment of ligaments, and occasionally presents a small facet for articulation with the cuboid bone. Articulations. The navicular articulates with four bones, the talus and three cuneiforms, occasionally with a fifth, the cuboid. The first cuneiform bone. Os cuneiform primum. Internal cuneiform. The first cuneiform bone is the largest of the three cuneiforms. It is situated at the medial side of the foot, between the navicular behind and the base of the first metatarsal in front. Surfaces. The medial surface is subcutaneous, broad and quadrilateral, and its anterior plantar angle is a smooth oval impression, into which part of the tendon of the tibialis anterior is inserted. In the rest of its extent it is rough for the attachment of ligaments. The lateral surface is concave, presenting along its superior and posterior borders a narrow L-shaped surface. The vertical limb and posterior part of the horizontal limb of which articulate with the second cuneiform, while the anterior part of the horizontal limb articulates with the second metatarsal bone. The rest of this surface is rough for the attachment of ligaments and part of the tendon of the perineus longus. The anterior surface, kidney-shaped and much larger than the posterior, articulates with the first metatarsal bone. The posterior surface is triangular, concave, and articulates with the most medial and largest of the three facets of the anterior surface of the navicular. The plantar surface is rough and forms the base of the wedge. At its back part is a tuberosity with the insertion of part of the tendon of the tibialis posterior. It also gives insertion in front to part of the tendon of the tibialis anterior. The dorsal surface is the narrow end of the wedge, and is directed upward and lateralward. It is rough for the attachment of ligaments. Articulations. The first cuneiform articulates with four bones, the navicular, second cuneiform, and first and second metatarsals. The second cuneiform bone. Os cuneiforme secundum. Middle cuneiform. The second cuneiform bone is smallest of the three, is of very regular wedge-like form, the thin end being directed downward. It is situated between the other two cuneiforms, and articulates with the navicular behind, and the second metatarsal in front. Surfaces. The anterior surface, triangular in form, and narrower than the posterior, articulates with the base of the second metatarsal bone. The posterior surface, also triangular, articulates with the intermediate facet on the anterior surface of the navicular. The medial surface carries an L-shaped articular facet running along the superior and posterior borders for articulation with the first cuneiform, and is rough in the rest of its extent for the attachment of ligaments. The lateral surface presents posteriorly a smooth facet for articulation with the third cuneiform bone. The dorsal surface forms the base of the wedge. It is quadrilateral and rough for the attachment of ligaments. The plantar surface, sharp and tuberculated, is also rough for the attachment of ligaments, and for the insertion of a slip from the tendon of the tibialis posterior. Articulations. The second cuneiform articulates with four bones: the navicular, first and third cuneiforms, and second metatarsal. The third cuneiform bone. Os cuneiforme tertium. External cuneiform. The third cuneiform bone, intermediate in size between the two preceding, is wedge-shaped, the base being uppermost. It occupies the center of the front row of the tarsal bones, between the second cuneiform medially and cuboid laterally, the navicular behind, and the third metatarsal in front. Surfaces. The anterior surface, triangular in form, articulates with the third metatarsal bone. The posterior surface articulates with a lateral facet on the anterior surface of the navicular, and is rough below for the attachment of ligamentous fibers. The medial surface presents an anterior and posterior articular facet, separated by a rough depression. The anterior, sometimes divided, articulates with the lateral side of the base of the second metatarsal bone. The posterior skirts the posterior border and articulates with the second cuneiform. The rough depression gives attachment to an interosseo ligament. The lateral surface also presents two articular facets, separated by a rough non-articular area, the anterior facet, situated at the superior angle of the bone, is small and semi-oval in shape, and articulates with the medial side of the base of the fourth metatarsal bone. The posterior and larger one is triangular or oval, and articulates with the cuboid. The rough, non-articular area serves for the attachment of the interosseous ligament. The three facets for articulation with the three metatarsal bone are continuous with one another. Those for articulation with the second cuneiform and navicular are also continuous, but that for articulation with the cuboid is usually separate. The dorsal surface is of an oblong form, its posterior-lateral angle being prolonged backward. The plantar surface is a rounded margin, and serves for the attachment of part of the tendon of the tibialis posterior, part of the flexor hallucis brevis, and ligaments. Articulations. The third cuneiform articulates with 6 bones: the navicular, the second cuneiform, cuboid, and second, third, and fourth metatarsals.
The metatarsus. The metatarsus consists of 5 bones which are numbered from the medial side, ossa metatarsalia 1 through 5. Each presents for examination a body and 2 extremities. Common characteristics of the metatarsal bones. The body is prismoid in form, tapers gradually from the tarsal to the phalangeal extremity, and is curved longitudinally, so as to be concave below, slightly convex above. The base or posterior extremity is wedge-shaped, articulating proximally with the tarsal bones, and by its sides with the contiguous metatarsal bones. Its dorsal and plantar surfaces are rough for the attachments of ligaments. The head, or anterior extremity, presents a convex articular surface, oblong from above downward, and extending farther backward below than above. Its sides are flattened, and on each is a depression, surmounted by a tubercle, for ligamentous attachment. Its plantar surface is grooved antero-posteriorly for the passage of the flexor tendons, and marked on either side by an articular eminence continuous with the terminal articular surface. Characteristics of the individual metatarsal bones. The first metatarsal bone, os metatarsali 1, metatarsal bone of the great toe. The first metatarsal bone is remarkable for its great thickness, and is the shortest of the metatarsal bones. The body is strong and of well-marked prismoid form. The base presents, as a rule, no articular facets on its sides, but occasionally, on the lateral side, there is an oval facet, by which it articulates with the second metatarsal. Its proximal articular surface is of large size and kidney-shaped. Its circumference is grooved for the tarso-metatarsal ligaments, and medially gives insertion to part of the tendon of the tibialis anterior. Its plantar angle presents a rough oval prominence for the insertion of the tendon of the peroneus longus. The head is large. On its plantar surface are two grooved facets on which glide sesamoid bones. The facets are separated by a smooth elevation. The second metatarsal bone, os metatarsali 2. The second metatarsal bone is the longest of the metatarsal bones, being prolonged backward into the recess formed by the three cuneiform bones. Its base is broad above, narrow and rough below. It presents four articular surfaces, one behind of a triangular form for articulation with the second cuneiform, one at the upper part of its medial surface for articulation with the first cuneiform, and two on its lateral surface, an upper and lower, separated by a rough non-articular interval. Each of these lateral articular surfaces is divided into two by a vertical ridge. The two anterior facets articulate with the third metatarsal. The two posterior, sometimes continuous, with the third cuneiform. A fifth facet is occasionally present for articulation with the first metatarsal. It is oval in shape, and is situated on the medial side of the body, near the base. The third metatarsal bone, os metatarsali 3. The third metatarsal bone articulates proximally, by means of a triangular smooth surface, with the third cuneiform, medially by two facets, with the second metatarsal, and laterally by a single facet with the fourth metatarsal. This last facet is situated at the dorsal angle of the base. The fourth metatarsal bone, os metatarsali 4. The fourth metatarsal bone is smaller in size than the preceding. Its base presents an oblique quadrilateral surface for articulation with the cuboid, a smooth facet on the medial side, divided by a ridge into an anterior portion for articulation with the third metatarsal, and a posterior portion for articulation with the third cuneiform. On the lateral side, a single facet for articulation with the fifth metatarsal. The fifth metatarsal bone, os metatarsali 5. The fifth metatarsal bone is recognized by a rough eminence, the tuberosity, on the lateral side of its base. The base articulates behind by a triangular surface cut obliquely in a transverse direction with the cuboid, and medially with the fourth metatarsal. On the medial part of its dorsal surface is inserted the tendon of the peroneus tertius, and on the dorsal surface of the tuberosity, that of the peroneus brevis. A strong band of the plantar aponeurosis connects the projecting part of the tuberosity with the lateral process of the tuberosity of the calcaneus. The plantar surface of the base is grooved for the tendon of the abductor digiti quinti, and gives origin to the flexor digiti quinti brevis. Articulations. The base of each metatarsal bone articulates with one or more of the tarsal bones, and the head with one of the first row of phalanges. The first metatarsal articulates with the first cuneiform, the second with all three cuneiforms, the third with the third cuneiform, the fourth with the third cuneiform and the cuboid, and the fifth with the cuboid. The phalanges of the foot, phalanges digitorum pedis. The phalanges of the foot correspond, in number and general arrangement, with those of the hand. There are 2 in the great toe, and 3 in each of the other toes. They differ from them, however, in their size, the bodies being much reduced in length, and especially in the first row, laterally compressed. First row. The body of each is compressed from side to side, convex above, concave below. The base is concave, and the head presents a trochlear surface for articulation with the second phalanx. Second row. The phalanges of the second row are remarkably small and short, but rather broader than those of the first row. The ungual phalanges, in form, resemble those of the fingers, but they are smaller and are flattened from above downward. Each presents a broad base for articulation with the corresponding bone of the second row, and an expanded distal extremity for the support of the nail and end of the toe. Articulations. In the second, third, fourth, and fifth toes, the phalanges of the first row articulate behind with the metatarsal bones, and in front with the second phalanges, which in their turn articulate with the first and third. The ungual phalanges articulate with the second. Ossification of the bones of the foot. The tarsal bones are each ossified from a single center, excepting the calcaneus, which has an epiphysis for its posterior extremity. The centers make their appearance in the following order: calcaneus at the 6th month of fetal life, talus about the 7th month, cuboid at the 9th month, third cuneiform during the 1st year, first cuneiform in the 3rd year, second cuneiform and navicular in the 4th year. The epiphysis for the posterior extremity of the calcaneus appears at the 10th year, and unites with the rest of the bone soon after puberty. The posterior process of the talus is sometimes ossified from a separate center, and may remain distinct from the main mass of the bone, when it is named the os trigonum. The metatarsal bones are each ossified from 2 centers, one for the body and one for the head of the 2nd, 3rd, 4th, and 5th metatarsals, one for the body and one for the base of the 1st metatarsal. Footnote. As was noted in the 1st metacarpal, so in the 1st metatarsal there is often a second epiphysis for its head. End footnote. Ossification commences in the center of the body about the 9th week, and extends toward either extremity. The center for the base of the 1st metatarsal appears about the 3rd year, the centers for the heads of the other bones between the 5th and 8th years. They join the bodies between the 18th and 20th years. The phalanges are each ossified from 2 centers, one for the body and one for the base. The center for the body appears about the 10th week, that for the base between the 4th and 10th years. It joins the body about the 18th year. Section 51. Comparison of the bones of the hand and foot. The hand and foot are constructed on somewhat similar principles, each consisting of a proximal part, the carpus or the tarsus, a middle portion, the metacarpus or the metatarsus, and a terminal portion, the phalanges. The proximal part consists of a series of more or less cubical bones, which allow a slight amount of gliding on one another, and are chiefly concerned in distributing forces transmitted to or from the bones of the arm or leg. The middle part is made up of slightly movable long bones, which assist the carpus or tarsus in distributing forces, and also give greater breadth for the reception of such forces. The separation of the individual bones from one another allows of the attachments of the interossei, and protects the dorsipalmar and dorsiplantar vascular anastomoses. The terminal portion is the most movable, and its separate elements enjoy a varied range of movements, the chief of which are flexion and extension. The function of the hand and foot are, however, very different, and the general similarity between them is greatly modified to meet these requirements. Thus the foot forms a firm basis of support for the body in the erect posture, and is therefore more solidly built up, and its component parts are less movable on each other than those of the hand. In the case of the phalanges, the difference is readily noticeable. Those of the foot are smaller, and their movements are more limited than those of the hand. Very much more marked is the difference between the metacarpal bone of the thumb and the metatarsal bone of the great toe. The metacarpal bone of the thumb is constructed to permit of great mobility, is directed at an acute angle from that of the index finger, and is capable of a considerable range of movements at its articulation with the carpus. The metatarsal bone of the great toe assists in supporting the weight of the body, is constructed with great solidity, lies parallel with the other metatarsals, and has a very limited degree of mobility. The carpus is small in proportion to the rest of the hand, is placed in a line with the forearm, and forms a transverse arch, the concavity of which constitutes a bed for the flexor tendons and the palmar vessels and nerves. The tarsus forms a considerable part of the foot, and is placed at right angles to the leg, a position which is almost peculiar to man, and has relation to his erect posture. In order to allow of their supporting the weight of the body with the least expenditure of material, the tarsus and a part of the metatarsus are constructed in a series of arches, the disposition of which will be considered after the articulations of the foot have been described. The sesamoid bones, ossa sesamoidea. Sesamoid bones are small, more or less rounded masses embedded in certain tendons, and usually related to joint surfaces. Their functions probably are to modify pressure, to diminish friction, and occasionally to alter the direction of a muscle pull. That they are not developed to meet certain physical requirements in the adult is evidenced by the fact that they are present as cartilaginous nodules in the fetus, and in greater numbers than in the adult. They must be regarded, according to Thelenius, as integral parts of the skeleton phylogenetically inherited. Physical necessities probably come into play in selecting and in regulating the degree of development of the original cartilaginous nodules. Nevertheless, irregular nodules of bone may appear as the result of intermittent pressure in certain regions, for example, the rider's bone, which is occasionally developed in the adductor muscles of the thigh. Sesamoid bones are invested by the fibrous tissue of the tendons, except on the surfaces in contact with the parts over which they glide, where they present smooth articular facets. In the upper extremity, the sesamoid bones of the joints are found only on the palmar surface of the hand. Two, of which the medial is the larger, are constant at the metacarpophalangeal joint of the thumb. One is frequently present in the corresponding joint of the little finger, and one or two in the same joint of the index finger. Sesamoid bones are also found occasionally at the metacarpophalangeal joints of the middle and ring fingers, at the interphalangeal joint of the thumb, and at the distal interphalangeal joint of the index finger. In the lower extremity, the largest sesamoid bone of the joints is the patella, developed in the tendon of the quadriceps femoris. On the plantar aspect of the foot, two, of which the medial is the larger, are always present at the metatarsophalangeal joint of the great toe, one sometimes at the metatarsophalangeal joints of the second and fifth toes, one occasionally at the corresponding joint of the third and fourth toes, and one at the interphalangeal joint of the great toe. Sesamoid bones apart from joints are seldom found in the tendons of the upper limb. One is sometimes seen in the tendon of the biceps brachii opposite the radial tuberosity. They are, however, present in several of the tendons of the lower limb, for example, one in the tendon of the peroneus longus, where it glides on the cuboid, one appearing late in life in the tendon of the tibialis anterior, opposite the smooth facet of the first cuneiform bone, one in the tendon of the tibialis posterior, opposite the medial side of the head of the talus, one in the lateral head of the gastrocnemius, behind the lateral condyle of the femur, and one in the tendon of the psoas major, where it glides over the pubis. Sesamoid bones are found occasionally in the tendon of the gluteus maximus, as it passes over the greater trochanter, and in the tendons which wind around the medial and lateral malleoli. End of Anatomy of the Human Body, Part 1, by Henry Gray. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org.